Welcome you all to the third day of this international e-conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. We had 2 days of brainstorming sessions on 5 different themes, and today we have again 3 different themes with diverse lectures from distinguished scientists all over the world. I once again welcome you all on behalf of our organizing committee for this international e-conference. Today we have 3 different sessions. The first session is on one of the contemporary research field, that is next generation sequencing and omics platforms in crop improvement. So I welcome you all for this session, and the chairman of this session is our beloved professor Dr. Shailaja Ittalmani madam, and co-chairman of this session is Dr. Chandrayappa sir, former professor. And we are privileged that the keynote address today will be given by one of the eminent scientists, distinguished personality, Dr. N.K. Singh sir. Sir, we welcome you, sir, for this international e-conference. Madam, Shailaja madam, yes. We are finding words to express about madam. Madam, our professor, a distinguished scientist who has got a niche for herself in the field of molecular genetics. She will be chairing this session. Madam, to say about madam, so she has served at various levels in the university and as well as at the national level, project scientist at International Rice Research Institute, Philippines, visiting fellow there, and she is member of several committees, editorial committee of Verizon. She is a member of Asian Rice Biotechnology Network, and she is a DBT nominee for Indam Seeds, and she is the recipient of several awards, and she is honored with several awards. To name a few are ICR Young Women Agriculture Scientist, IRRI Young Scientist Award, Dr. C.V. Raman Research Award, US Best Alumni Award, and many others. Madam, we welcome you for this conference, and you have to chair the session. It's also a privilege to welcome the co-chairman of this session, Dr. Professor Chandrayappa sir, the professor and head biotechnology and genetics at MS Ramaya College of Arts and Science and Commerce. A very dedicated professor who has authored many books which every student wishes to refer for biotechnology, and is a recipient of many awards. We welcome you, sir. Madam?
Thank you. Thank you.
Here is yours, madam.
Yes. Thank you. Thank you, Lakshmi Devamma.
Thank you, madam. Thank you. Please.
Shall we start?
Yes, madam. We shall start.
Yes. I welcome, first of all, thank you to all the organizers. And I welcome my co-chairman, Dr. Chandrayappa, who is my colleague at University of Agriculture Sciences, Bangalore. And we will coordinate this program. We have one keynote address by Dr. N.K. Singh, a prominent scientist, not only in India and all over the world. He's done very good research work, path-breaking research. At present, he is the national professor under the ICR National Research Center of Biotechnology. Can I continue?
Yes, madam. Just there was slight disturbance. Sorry for inconvenience.
Okay.
Madam?
You are muted, Shailaja. Please unmute yourself.
Didn't mute. They said the organizers muted.
Okay.
No, sorry, madam.
It's okay.
Hello.
Hello.
Yeah, madam. Continue, madam, please.
Okay. I would like to give a brief introduction of Dr. N.K. Singh, who is at present the national professor at ICR under the P.P. Pal chair. He is known not only for his research, but also the various administrative and academic levels he has worked in. Again, it's muted.
No, we can hear. Please go ahead.
Yeah, Dr. Singh has worked worldwide with researchers to improve various traits in crops of both cereals and pulses and vegetables like tomato. He has been part of many national and international research projects. He's worked on several sequencing of crops and plant genomics, particularly his contribution in decoding of rice, tomato, and pigeon pea genomes, and in understanding the wheat seed storage proteins to improve their quality. Genome sequencing annotation of crops and microbes are important work that he has undertaken, which are useful for plant breeders for molecular and precise development of varieties. He's made a significant advances in comparative analysis of rice and wheat, and also in salt tolerance and basmati rice improvement. Awards and honors, if we go on listing, I think whole day we will be mentioning them, but still I would like to mention some of his recognitions he got for his work, dedicated work. He, as a student also, he has been a very bright student and meritorious with gold medals, both at MSc and BSc at BHU. Then he was a postdoctoral fellow at the CSIRO Australia. Then he was a national research fellow at Government of Australia. He's been the fellow of several academies. To name a few is Indian Academy of Sciences, National Academy of Sciences, NAS, the National Agricultural Academy, New Delhi, and several others, the Plant Breeding Academy. And he's won the prestigious ICR Rafi Ahmed Kidwai Award, among several other awards that he has been recognized with, along with Norman Borlaug Award. He was the secretary of the National Academy of Agriculture Sciences earlier in 2011, and now he is the chairperson. These are very brief introduction of Dr. N.K. Singh, whom I was associated for a long time in our rice research projects. And he's still very active in research, and he's doing very path-breaking research. Now, I would invite Dr. N.K. Singh to give a talk on the topic on gene discovery and genomics assisted plant breeding. Welcome, Dr. N.K. Singh, to our conference.
Thank you very much, Dr. Shailaja, for nice introduction.
That was a brief one. You please start the talk.
Yes,
we have
I will
about half an hour time for you.
Yeah.
Yeah.
So first of all, let me Dr. Fakhruddin, and actually I know him for a long time since NP Genomics program in the US. And I'm really thankful to him and to all the organizers. I can see it's from internationally and nationally, an overview of gene discovery and genomics assisted plant breeding. When I change my slide, yeah.
Hello, sir.
There's slight disturbance. There's slight disturbance. Can you set it right?
Hello, madam.
Yeah.
Madam, nimdu back, like your back camera is on, madam. You just on the front camera, your Galaxy phones. You are not visible.
I think it's on.
It's on, madam, but the camera is back camera.
Rear end is, it's showing rear end, madam.
Okay.
Once again, you set as early.
Now it's fine.
Madam, put on your camera, madam, in this setting.
Yeah.
No, madam, still it's showing same thing. Tap on the camera, madam. It will be there a direction like moving. Two-way direction will be. You need to tap on that.
Hello, madam. Can you double tap on the camera, madam?
Yes, I'm coming.
Yeah, madam.
Is it?
No, ma'am.
Ma'am,
Ma'am,
double
double
tap
tap on the camera, ma'am.
Wait.
Double tap on the camera, madam.
Yeah, I'm doing it, but it's not coming.
just once you do, ma'am. Just once like that. I think it'll turn back.
Wait. Yeah.
Yes, ma'am. It's perfect.
Yes. ಅದು ತಾನೇ ಹೋಗ್ತಾ ಇದೆ ಫಕ್ರುದ್ದೀನ್
ಹಾ ಮ್ಯಾಮ್
ತಾನೇ ಹೋಗಿ ಬರೋದು ಮಾಡ್ತಾ ಇದೆ
ಸರಿ ಆಗುತ್ತೆ ಮ್ಯಾಮ್ ಸರಿ
ಓಕೆ
ಗೋ ಹೆಡ್ ಮ್ಯಾಮ್ ಪ್ಲೀಸ್
Is it okay?
Yes, madam. Now it's perfect.
Is
Perfect.
Dr. Singh speaking or you're doing?
Some network disturbance he got, it seems.
Okay.
Joining back, madam.
Okay.
Thank you. Now it's perfect, madam.
Okay.
Hello, can you hear me?
Yes, sir.
Yes, we can hear you. Please go on.
There was some issue with the connection, I think. So anyway, I will resume. So I will be talking mostly about the work done by me at the National Institute of Plant Biotechnology. In the beginning, I would like to acknowledge the funding support from the DBT and ICR and a large number of scientists and students who have been collaborating with me. I have been working directly under my supervision. So I'd like to thank all of them because mostly I'm presenting it is their work. So this is the outline of my presentation. After a brief introduction about the decoding of the why we need to decode the genomes and what is being done at NIPB. Then what are the major problems with the green revolution varieties and emerging threats due to global climate change. And then one of the major projects which we have been doing from QTL to variety for infusing climate resilience into high yielding variety. Then a significant part of my presentation will be on the wild rice, searching for new genes in the wild rice. So mostly my presentation is focused on rice, but I'll be also talking about little bit about the other works which we have been doing. So as you know, I think most of you will know that the genome is defined as the complete set of genetic information present in a single haploid cell of an organism, including its protoplasm, that is chloroplast and mitochondria. And we need to sequence the genomes because it is very useful at all levels, starting from the very basic. When we have the complete physical map, that is genome sequence of different species, then we can understand the evolutionary and phylogenetic relationship between different species. And now there are many studies have constructed the tree of life, which shows that how each and every species on Earth is related to each other based on the DNA sequence information. So we can trace back our common origin of all living things and how far or how close they are from each other. So this is a very basic curiosity every human being has to know about. So this genome information, the complete physical map answers this very beautifully and very precisely. Then if you come to the more practical level, once we have the complete genome information, then we can create very high density fingerprints of our germplasm resources. We know that the India and many of the developing countries, they are very rich in biodiversity, which has been recognized as the property internationally through biodiversity treaty. But unless you are able to fingerprint it, unless you are able to define it, that what is your material, you will not be able to claim that right. So it helps you to fingerprint your material and precisely define it, that which material belongs to whom. Then having genome information is very, very useful for the gene discovery. I will show you the examples. We can identify and clone the genes based on the genome information. If you don't have the genome information, it will be just like going without a GPS. Now everybody is familiar how useful the GPS is to find any particular place or person uh, when you are driving or searching. Similarly, having the genome information, it is a GPS of the living word. You can find the genes, which chromosome, which particular place the gene is there. So gene discovery and cloning makes it easier. And finally, actual the breeding process itself. Breeding when breeders trying to select for a desirable type from the existing variation or the variation which he has created. So DNA marker assisted breeding is very much, you can call it now genome assisted breeding, because you can select the foreground, the background, and the linkage drag, all those things you can select once you have the complete knowledge. Even the genome editing, which is now coming days, very, very promising technology. You can call it genome editing also, some people call gene editing also, but it is the same thing. You edit the gene, and when you edit the gene, you also edit the genome. So it is possible when you have the complete genome information, because you have to work out that there is no off target in the whole genome, and that is only possible when you have the genome information. So it's a very, very useful to have the genome. Now, and that's why, people spend a lot of money world over trying to sequence genomes of as many crop plants or animal species as possible, because it definitely has utility there. So this is the gene discovery and utilization process flow and timeline. The very first thing we need to do, that we have the germ plant collections and gene banks around the world, and we know that the genes are there in the germ plant. Nobody has created any gene so far, despite so much advancement in science. Even the synthetic genes or synthetic cell which we talk about, it is nothing but the copy of the already existing information about the cells and the genes. I have no information anybody has created a de novo, a new gene, which is not a copy of a existing one. So the genes are there. Then how we have to discover the gene? First, we have to discover the trait. We have to discover the germ plasm which have particular trait value. Maybe it may be salt tolerance or resistance to a particular disease, or any stress, or maybe yield or quality. so many traits. So for that, we have to do is screening. We have to take the germ plasm and do high throughput screening and identify some material which have that particular trait value. So through reverse engineering process, this is in this forward genetics it is called. There is also reverse genetics, which is a bit more tedious because you have to do every gene and then find which gene can affect a particular trait. So it's better to go this way. Start from the germ plant, identify a germ plant which is bearing a particular trait. And really the challenge for the breeder is that all the good traits are not in a single genotype. They are spread around different. Some varieties may have very high yielding but may not have good quality resistance. Some may have resistance but not high yielding. So all the good traits are spread out in different genotypes, and we have to bring them together into one genotype and try to make a super kind of variety which will have higher yield also, resistance also, high quality also. So to achieve that, we need to identify the genes. So first we identify the genotype bearing the trait, and then we can identify the genes. For that, we need to create a mapping population, a segregating population. Preferably the biparental population is much better, but we can do it through association studies also, uh, which is another kind of a segregating population. And for that, we can use a series of functional genomics tools like gene expression profiling and so many other approaches are there. Knock down, you have to validate the function by knocking down the gene by overexpression or underexpression. And once we identify the genes, then we can go back to the germ plasm and see that how many different alleles are there for that particular gene. There may be several alleles. The Mendelian genetics described only two alleles of each gene, dominant, recessive in the beginning, but then multiple allele theory came also. But now we know that at molecular level, at sequence level, almost every gene has got several alleles. Some of these differences may not have functional significance, but there are many SNPs, indels between different alleles of any gene. So we can go back and do resequencing of these different alleles of a gene and see how they are associated, which allele is the desirable one, which is the undesirable one. Quite often, they are the two sides of the same coin. Then the breeding process is actually, we have a large population. Quite often it is biparental. They cross elite with the elite, but sometimes it could be elite with the some distant relatives or agronomically weak material. Sometimes they're multi-parental also, but you have to cross two at a time in any case. And then we can use marker-assisted selection because we know the markers linked to the genes. So directly, if you have the functional markers, it's very good. Or something like genomic selection, where each marker has a breeding value, each gene. So these are the things which are coming up, but at present, I will just describe you what has been successfully used. And this finds the place to the society, which is the ultimate goal of any science, to serve the society. So in the conventional breeding, this process used to take 10 to 15 years, I think at least 15 years. But now with the help of genomics assisted breeding, this time duration is halved, maybe 5 to 7 years, sometimes even 3-4 years one can uh, release a new variety. Then the genome sequencing of genomes is very challenging, mainly because of the size of the genomes. You can see the most microbial genomes are about 5 MB in size, and then Arabidopsis was 125, rice was 390 MB. But many of the crop plants, they are polyploids like barley, maize, wheat. They have enormous genome size, and it is very challenging to assemble the genome because a large amount of repetitive DNA is there. So when you try to assemble the chromosomes, there is a huge challenge because any sequence will go and match to many different places due to redundancy of the genome repeats. It is very challenging. That's why, despite so much, in the last 20 years, genome sequencing is being done, but you can count on finger the genomes which have been sequenced of the higher plants to the higher quality level. There are many drafts are available, but high quality reference sequences are you can still count on the finger. So India started its journey through the RISE Genome Project. I will not go into much detail, but it was in 2000 project funded by the DBT, where our institute and the Delhi University South Campus we sequenced chromosome 11 uprise, that is long arm, and it took 2 years to generate the draft, and then it was launched in Tokyo. Then another 3 years it took to finish the genome to reference quality level. So in 2000, this paper was published in 2005, and this was the first reference genome of a crop plants, and it has been used very extensively since then for gene discovery and molecular bidding in rice. We at our institute, we developed a chip, 50K SNP chip, and this chip has been patented because it has certain unique features. It is based on the single copy genes, and it's totally genic chip. Most of the chips are having SNPs which are intergenic. Their genomic SNPs are there, but this chip has got entirely genic chip based on the single copy genes and some of the cloned agronomically important genes. Another important thing is that it has got multiple SNPs from genes. So on an average, it will have about 6 SNPs per gene, 5, 6 SNPs. Some of the genes may have to 10 SNP. So that helps us to find out the haplotype information. That means alleles of a gene. Say, for example, GBSS gene, we have 8 SNPs and 9 haplotypes. So normally with the SNP chip, you can only score 2 alleles, because SNPs are biallelic in nature. So every SNP can give you only 2 alleles. Here also, each SNP will give you 2 alleles, but when you have number of SNPs per gene, then you can get a number of haplotypes. So truly you can get multiple alleles per any given gene. And that is the unique feature of this chip. I will show you the application of that thing. Similarly, after that, we were involved in the tomato genome sequencing, and we sequenced the chromosome 5 of tomato. Delhi University South Campus and our team again, we did this thing. Then subsequently wheat genome, which I showed you is a very large genome. So it took several years to sequence the draft genome, and India was given the responsibility of sequencing chromosome 2A of wheat. It has 21 chromosomes and it's very large repetitive elements. Then it took another 4 years to reach the reference point. So 2018, the reference genome was published with more than 90% coverage. But still this reference itself, it has got many gaps in the sequence. You know, we have the chromosome based, because of the wheat has got certain anuplied stocks, which facilitates the chromosome based sequencing, assembly of the chromosomes. But because it was based on largely on the Illumina sequencing, there are a lot of EONs are there, and more than 2% EONs are there, which is actually very, very important, because the many genes annotations will be missing because they will be interrupted by EONs. So the efforts are still on to chromosome by chromosome improve this wheat genome sequence. Then one more, which is very unique thing, is that this is the pigeon pea genome, which we did the first genome which is sequenced completely in India, and that's why Dr. Fakhruddin was also one of the partner. And in 2011, we sequenced the first pulse crop genome in the world and the first plant genome in India. This was a draft genome, and we predicted this was about 62% coverage. 47,000 genes were predicted from this. Then this information was used to create the SNP arrays. This time it's a Illumina array, and it was used to map the plant type genes in pigeon pea. You can see the dwarf pigeon pea is very important, because nowadays shortage of labor, we want the short stature pigeon pea which can be mechanically harvested. And this one variety has already been released, Pusa Arhar 16, which is a dwarf variety and early variety. Most of the pigeon pea variety are still medium variety and they are taller in nature. So there's a need to breed short duration and shorter height pigeon pea variety. And now breeding program is geared towards that. So the here importance is that having genome information, we could very quickly map, and now there are many other things are also mapped that they work at ICRISAT also, and also IAPR in Kanpur, and PAU. A lot of good work is going on pigeon pea, which has been accelerated by the availability of the genome sequence. Now you can see here there were two major loci, pleiotrichic loci, on chromosome 4 and chromosome 5, which actually controlling this plant type behavior, height of the plant, branching, maturity. So that means just manipulating one or two gene, one can completely change the plant architecture, which will be suitable for new varieties. Now, very similar to the rice chip, we have made a chip for pigeon pea also. This is also totally genic chip. It has got 62,000 SNP, and it also includes 747 here disease resistance genes. So when we have this information about the genome, we know how many disease resistant type genes are there. So pigeon pea has about nearly 1500 we have actually annotated. Out of that, 747 are included in this chip. And you can see that on an average, about 9.9 SNPs per gene. So every gene, how many alleles are there, that information also we can get by use of this chip, which is very, very useful. So this chip, this was published actually earlier this year. Now it is available for anybody who wants to use it for pigeon pea genetics and breeding. This is very neatly you get, this one allele, this is another allele, and the heterozygote. So pigeon pea has little bit heterozygote, but mostly it is homozygous. And you can see these chips gives nearly 100% success rate each time because this is single copy gene based, there is no cross hybridization. So we have run 4 of these chips and all the samples were successful. This is just a repeat slide. Similarly, the jute genome was also a draft genome with more than 90% coverage was published in collaboration with the CRIJF at Barakpur by our institute, and the for jute fiber quality, a major QTL was mapped here on chromosome linkage group 1. Now something may be interesting to the horticulture community. We have been working last few years on the mango genome, and mango genome is very, very challenging. Even though genome size is small, about 400 MB, but the challenge is that it is highly heterozygous. So far all the genomes which have been published, whether it's a plant or animal or whatever, it is assumed that they are haploid in nature. That means we have no account of the maternal and the paternal chromosome. So either heterozygosity is very low as far as many of the animal, including human, or in the plant cases, we either sequence the inbred or the double haploid, so that we are giving a haploid genome information. Even if you see any of the genome browser, you have a single thread of chromosome running. But we know that every organism which reproduces sexually, it has 2 chromosomes, one for coming from maternal and one from paternal side. And these 2 chromosomes are not identical in most cases. So we need to have information on both the chromosome. So this kind of approach, now tools are becoming available for the last couple of years. There's an approach called trio-binning approach. First, I will highlight you what is the problem. Whenever we try to assemble such genomes using the conventional method of assembly, the genome size we assemble is bigger than the actual genome size, because some portion of the genome where heterozygosity is too large, the maternal and the paternal sequence, they don't collapse into one, they assemble separately. And where the homozygosity is more, heterozygosity is less, they will assemble together. So this is really very, very mixing of the assembly. So this is the problem we face quite some time. Even Australia and USA people also wanted to sequence. They started. All of us face this problem. And in China also, they have done some draft sequencing. Everybody faced this problem. But we were lucky in some way that we have both the parents of this variety which we chose, Amrapali. We know the parents are actual parents are there in the Pusa orchard. That is Neelam and Dasari. Dasari was the female parent and Neelam, South Indian variety, was the male parent. So original plants are there, and the Amrapali, the progeny, is also there. So there's an approach called trio binning approach, where we can sequence the genome of the progeny with the help of the sequence of the two parents. So even though both the parents are also heterozygote, I will not go into detail. Now there is a trio binning approach where one can separate the maternal and paternal chromosome reads of the hybrid. Using this one, we have been able to assemble the genome. To cut the long story short, we know that mango has thousands of variety and it has originated in India. So this kind of resources will be very helpful for gene discovery and improvement of mango. Similar to rice and pigeon pea, we have developed developed a chip of mango also. This time it has got 82,000 SNP at the same cost, because you can go from 0 to 90,000 elements you can put at the same cost. So you design the chip and it will be manufactured by Epimetrics. So here also we have this mostly based on the single copy genes for the wide genome coverage. Plus it has got the cloned horticulturally important genes and disease resistance genes. They are all part of the thing. And once again, the success rate is 100%. And the lot of heterozygosity in most mango variety. When we have genotype 384 variety, and we know the level of heterozygosity each variety has. And we have used this for association mapping of fruit number of traits. I'm not going to show all of you here. These are unpublished work also. So fruit acidity work was phenotyping done at IHR Bangalore. This is a multi-institutional project actually. CISH Lucknow, IHR Bangalore, IRI, and our institute. So we have got several important QTLs here. And also for the pulp percentage, what percentage will be pulp and what percentage will be stone. So that was in very briefly that the genome sequence which we have generated and its implications. Now let's come back to the rice thing for varietal improvement. We know that the most of the green revolution variety, they are dwarf in nature. They were bred for high input condition. That means water and fertilizer, they were considered not a limiting thing. We were only selected for the very high yield. And as a result, we have got the yield, but we lost so many other traits, including many times the quality of the variety and also stress tolerance, particularly abiotic stress tolerance. We lost it because simply we did not select for it. We did not lose deliberately. And as a result of global warming, now this is coming as a big threat. Flooding, drought, salinity, and also new diseases. These are coming as a real threat. So now there's a need to rediscover those genes and put it back in the high yielding background. So the traditional land races of rice and wild relatives of rice, they are rich source of genes of all kinds. All the genes are out there. Just we have to search them and put it back in the high yielding background. So mega varieties of rice, so these are the green revolution varieties here. BR11, CR1009, IR64, Swarna, Samba, Masuri. They are cultivated in millions of hectares by the farmers and they are very popular. And despite their susceptibility, farmers want to cultivate this one because they give higher yield. Whereas traditional varieties, they have very high level of stress tolerance and good quality, but farmers are reluctant to grow them because they have very poor yield. So many people say that we have lost all the varieties. We haven't lost any variety. They are all there in the NBPGR gene bank, but nobody will cultivate them because they are very poor yielding. So we have to combine the high yield with this all climate resilience, disease resistance. And in a molecular breeding, there are a number of strategies are there. You can have a pedigree breeding, stateway marker assisted selection by crossing two variety. Here we need large population to get the desired one. Particularly if we have a poor elite versus poor class, then you need a bigger population. So mostly breeders prefer elite versus elite, so they will have very few undesirable types. So the population size is the real question here. Then marker-assisted recurrent selection applies mostly in the cross-pollinated crops. There are some examples in maize and wheat. Then genomic selection is just a coming up technique. Really, we don't have demonstrated success in plants. There's some success in animals. It is there for milk production and other thing. But in plants, the population size has to be really big to find the desirable thing. So genomic selection is still being worked on. Marker-assisted backcross breeding is the one which has been very successful, because very quickly through backcrossing, one can eliminate the undesirable type. Because when you cross any two varieties, for example, rice, you cross two varieties, then if they differ only for 24 genes, that means one on each telomere, so they will generate 16 million combinations, and only one combination is like the original parent. So this way we generate so many undesirable type that backcrossing is the only way to improve the thing. So this one example here is the Swarna sub-1. The donor was identified in 1970, and the breeding lines and mapping was done in 1990. And after the availability of rice genome sequence, the 2006, the sub-1 gene was cloned using genomic assisted cloning. And then within 3 years, the first variety was released, Swarna sub-1, which was Swarna basically, but this one gene sub-1 was introduced through background selection, foreground selection, and recombinant selection. So this work was very, very important. Nirja was involved from our Rice Institute at ERI. And this variety was released in India and has become very popular because it survives 2 weeks of complete flooding. Now this gene has been transferred in a number of backgrounds, and it behaves in each of these backgrounds, because there is a ethylene response factor which makes the rice dormant during the flooding. So just like the frog, goes into hibernation and then wakes up again when the water comes. Similarly, these rice varieties having that gene, under submergence, they go into sleeping state, but they don't die. And once the water is gone, they will again regenerate and bear the fruit and give the yield. This particular chip was used to again analyze the background of the rice variety developed at ERI in Swarna, IR64, Savitri, and Samba Masuri. And you can see that Swarna was the best one because it has 97% genome recovery, whereas the Samba Masuri has only 79% genome recovery. So there was about more than 15% gap in the genome recovery, which was analyzed through the low density SSR marker, but with the high density genome chip, background can be analyzed very nicely. Here at our institute, we have identified a gene for QTL GN 4.1. There is a kinase gene is involved there. And when we transfer this into different background, we get very high grain number. Almost this is a trait for the super rice, which was described by Dr. Khush. We have fewer tillers, not very few, just slightly less, but most of them are productive tillers. And again, you can see the plants are slightly taller in nature, the leaves are wider, and the panicule is bigger in size. Because of time cited, I'm giving you just one example here. In the Samba Masuri background, it has tremendous effect. In fact, now we have 3 years data, and the grain number is almost double. See, the lower 4 panicles are from the original Samba Masuri, and the upper 4 panicles are this QTL added with the 97% genome recovery. And here you can see the number of grains in the original Samba Masuri and more than double in the, just by the addition of one gene. They are very well filled grain. Panicule exertion is also good. Using this chip, we can do the background analysis here. This is example. The blue is the background has been recovered except in one places here, and chromosome 4 is this QTL. Now, based on these successes of the Swarna sub-1, other thing, we launched a major program here in India with the DBT support with 15 institutions to transfer these QTLs. There are 7 QTLs for drought tolerance, 1 for salt tolerance, and 1 for submergence tolerance. And using this limited backcross breeding in 2 to 3 backcrosses, using the foreground selection for the genes, or if the gene is not known, the QTL peak marker and the flanking marker. and then background selection using the chip. And this was standardized and shared between the partners, where the background selection was done in at an IPB because it involves the chip work. Then the QTL DTY 1.1 was fine mapped. Here there was a negative linkage, in the repulsion phase between the drought tolerance and the dwarfing gene. So most of the dwarf variety, they have lost this drought tolerance gene because very tightly linked with this thing. And this linkage we have broken. Now we have the positive alleles for drought tolerance in combined with the dwarfness. This is the process by which one has gone through zooming in, clone that particular segment. We don't know exactly what is the gene behind it, but we have narrowed down the region and separated it from SD1. And you can see here on the right side is the original Swarna, the left side, which is the green one, is the recombined Swarna, which is dwarf but is still drought tolerant. Now the last part of my project is that I'm searching for new genes and alleles from the wild rice, because the wild rice has faced, it is there for millions of years without any help from anybody. It has at least one order of the magnitude more number of genes for stress tolerance. So in my national professional project, I started this one. Many of you will be aware that the Radwing at the Arizona University, he has sequenced 13 different wild rice species, but many of them are not crossable with the cultivated rice. So I focus mainly on the Oryza rufipogon and Nivara, and collected from all over India. And these are the typical habitats. These can be very easily crossed with the cultivated rice. So in the roadside here, you can see in the Kaimur in Bihar, then here in UP, near a shallow pond, Tal, which we call the large area with lowland area. Then again a shallow pond, a small pond. These are all wild rice in the river basin in Assam, near Brahmaputra basin. Then also we can, that were the abundant actually habitat. Then there are rare habitats that are there in mostly in the upland, even in Himalayan, Dharmsala, near Dharmsala in Himachal Pradesh, yeah, Dhamtari in Chhattisgarh. Then upland here in the Jharkhand, Ranchi or Hubballi district are collected 2 years back from Western Ghat. Some area is still to be collected. Then the most important one here is that the rice is adapted to different stress condition. Like this is the sodic soil, it is growing there. This is the coastal sea water, it is growing. Then drought situation also, everything else is dead, but this is surviving. And the deep water, of course, also it is growing. So these are being used to identify the genes. Now the last one is the threatened habitat. Wherever this wild rice was growing, it is getting lost very rapidly because of the, you can see the building are being made. This is for housing, and then here is for the school, here is for the widening of a road, and then here is the large industrial park is being developed. Once this is developed, this rice will be lost, and this roadside is getting lost here, and then this is getting depleted very rapidly because nobody wants it. For the farmers, it is a weed, and for the developmental agency, it is a free area land where something else can be built. And we have characterized more than 800 of these. We have grown them and made a database. I will not go into too much detail, but we have screened them for different taste, drought tolerance. We find lines which are extremely sensitive to extremely tolerant. Similarly, gene allele mining we have done for the DREP gene and found association. Some alleles are associated with higher level of tolerance. In anaerobic germination, we find lines which are more than 90% germination under 15 centimeter of water, whereas the positive control like Kalonji, they have only 60% germination. And the high yielding variety like IR64, they have zero germination. So these are important for the direct seeded rice. These have been mapped also. Then we find new sources of submergence tolerance in some of them. You can see here, this is a new one. Then salt tolerance, we have found lines which are better than the Pokali. Pokali has a score of 3, they have a score of 1. And then BLB resistance, very, very highly resistant lines have been identified by my colleagues, collaborators. Sheath blight also, we have found the lines and it is getting mapped. Then SNP haplotypes of the known genes, like HKT family of genes which are involved in the salt tolerance. we have done the allele mining and we found the positive values in couple of genes. Now, in one of the cases, I will give you one example where KSWR173 collected from Himachal Pradesh has the score of 1. We have mapped it and classed it, and back class 2 is now presently there. But in BC1, we have mapped it. In BC1 F2 family, we did the phenotyping. BC1 F1 family, we did the genotyping. And you can see this segregation here for the ons and the hull color. Then for this particular seedling stage salt tolerance SCS score, we get from 1, and also we get 9 score. And then using the standard techniques, this is the chip was used for genotyping, and it worked very well. Out of so many markers, we selected those about 1,000 markers which are segregating expected 1:1 ratio in the backcross population, and created a map and identified the major QTLs are chromosome 1, 3, and 12. Now here it is very interesting that IR64 itself has QTLs for salt tolerance, which we do not know because we never classed it with the sensitive one. So here for that particular locus, this wild rice is sensitive, but it had additional loci which are very, very important. So all the red ones are from the IR64 and the blue one from the wild rice donor. So having this complete genome information, high density chip and marker, it helps you to dissect out any trait and see how many major genes are there contributed by which parent, so that you can follow them into the backcross breeding process. And underlying genes also, there are a number of candidate genes we are in the process of validating. Again, having the complete information tells you that what is the region and how many genes are there, and then you can start validation experiments on that. 4 varieties have already been released through this program of this. These are mostly from the line disease of rice. DRR Dhan 50 here is very, very successful for last 3 years. Its demand is growing. It has got salt tolerance, it has got drought tolerance, having 2 QTLs and also submergence tolerance. So this is the first variety in the world, marker assisted breeding for both dual drought and submergence tolerance, these 2 QTLs. And we have demonstrated in the target regions, and farmers are very, very happy accepting this variety. Now, future challenges. My last slide is there, probably last 1 or 2 slides. So we need to choose the plant because of the high cost of the genome sequencing and the complexity of the genome. We need to choose the plants of the high economic importance for genome decoding, and it is very useful once we do that. And at presently, we can use a combination of single molecule sequencing like the PacBio long read sequencing, and in combination with optical fingerprinting to assemble the genome. If it is heterozygosity, then we need to have the 2 parents also. Then RNA-seq, ISO-seq is you can do the full transcript using PacBio ISO-seq methodology. You can have the alternative splicing information. Then high density genetic map is needed. So we should move from the SSR based map to SNP based. So very quickly, you can do both polymorphism survey and mapping in one go. Then biology of the plant is very important. Unless you understand the biology, do proper phenotyping, spatial and temporal expression of the genes, RNA, protein, and the trait level, it is very, very important. In fact, this is now the most challenging thing, the phenotyping. Then we need to characterize our land races and wild relatives of the crop species to identify the new genes, and then use this genome information, just like I showed you some example for marker assisted genome association study or biparental QTL mapping or actual breeding itself. So I have taken a little longer, but I try to give an overview of the work which I have been doing. So thank you very much for your kind attention.
Thank you. Thank you, Dr. N.K. Singh, for your very exhaustive and informative talk. Thank you very much for your exhaustive and very informative and very exhaustive that carried out right from basics to applied and to the product development in plant breeding, that is varieties. I'm sure the PhD students and the young scientists will get very good information. And now I ask Dr. Chandrayappa, co-chair, if you have any comments on his talk.
I have one question to ask to the speaker. How much information from transcriptome proteomics you take along with the primary gene sequencing for a given any character? Because just directly you cannot rely the nucleotide sequence, genome sequence alone. So do you consider information from transcriptome proteomics also?
Yes, it is very, very important. We have used it extensively for our salt tolerance work because we identify the QTLs, and then within the QTL, the number of genes are there. So we particularly combine the transcriptome information. What are the genes which are differentially expressed, as well as they are co-located in the QTL region. The same thing we do at the proteome level also. So like this, we have identified a couple of genes, particularly say serine hydroxymethyltransferase, SHMT gene in rice, which is giving a very good level of salt tolerance. So this was identified by combining the transcriptome, proteome, and the genome information. So this actually accelerates the process of the gene discovery.
Exactly. Thank you, sir. There's not much time is there to ask some more questions.
Thank you so much, and I was pleased to be here. Thank you so much.
Thank you, Dr. Singh.
So go ahead with the next speaker.
Can we thank you, Dr. N.K. Singh, for a very, very informative talk and all the new discoveries you told us in your research. Can we go to the next speaker, Dr. Chandrayappa? Or are there any more questions?
I think we will skip the questions because
Okay,
already we have taken more time.
Can we invite the next speaker?
Yeah, please go ahead. Invite the next speaker.
Okay, we'll invite the next speaker, Dr. Nataraj Karba, professor at US Bangalore. Dr. Nataraj Karba is a professor at the Department of Crop Physiology, and he has worked exhaustively on the genomics and the targeted crop improvement and in various crops. But today he'll be talking on the discovery of novel genes in mulberry. I welcome Dr. Nataraj Karba, who is also a recipient of several awards for his research and recognitions for his research. Dr. Karba, please come in.
Yeah, thank you very much, ma'am. Good morning. I'm very pleased that you are chairing our session, and I'm also happy that Professor Chandrayappa is also co-chairing this session. And I'll be thrilled to, be part of this program. And I'll go through quickly some of the activities what we are doing in our lab with specific reference to transcriptomics in mulberry, but I will also pick up on other issues related to transcriptome work, what we are doing in our lab. And basically, this particular theme is NGS and omics platforms in crop improvement. From that context, I thought I will give other activities in addition to mulberry. So, ma'am, how much time do I have?
You have 20 minutes, 25 minutes.
All right.
You have 25 minutes, Raj.
Thank you very much. Am I audible? Is that okay? My voice is fine?
Yes. Yeah, it's fine. You go ahead.
Yeah, thank you. Thank you very much. So my talk would be, 5 different aspects or issues I would touch upon. One is introducing the omics, basically pitching on the transcriptomic part of our work. And second, trait-specific gene identification using transcriptomic approaches. So probably I'll give few examples, not only mulberry, also the finger millet which we are doing, just to give overall activity what we have been doing in our lab. And the third aspect would be the analysis what we do transcriptome to identify the novel pathways and genes, whatever the lead what we have, I just want to highlight. And the fourth one, to manipulate the trait, these days we started using endophytes. Certain plant physiological processes are modulated by the organisms associated within the plant. So I will be talking a small issues, transcriptome based approach, what we use to understand the plant endophyte interaction, the fourth issue. And at last, I will conclude my talk by saying that the transcriptome what we have generated or people are generating can also be used for multiple other purposes, mainly to identify the complexity in post-transcriptional level, basically alternative splicing event. So this is how I would like to conclude in 20 minutes quickly, without any details. I'll be very happy to share the details at the end if required. So what we have been working in our center at US Bangalore GKVK, the Department of Crop Physiology, is to understand the physiological process which are associated with the abiotic stress in general, drought in particular. You know that plant growth and development is in fact regulated by several environmental factors, and major one in a modern climate change scenario is water and nutrient availability and of course productivity. So to understand that, as our first speaker, lead speaker just now mentioned, people are using genomic approaches. People have also attempted epigenomics, transcriptomics, epitranscriptomics, and of course multiomics like proteomics and metabolomics and things like that. These are the tools and techniques which are available to us to understand the complex interaction of the plant with the environment, so that you can try to understand the complex metabolic processes at micro level, whole plant level, as well as plant to plant interaction or plant to microbe interaction in the macro level, and evolve suitable strategy to manage or to improve the crop. So that is the idea. But today I will not be going to those details. The task would be to tell us what we have been doing in the area of transcriptomics. Although I mentioned as mulberry, but it will be other system as well. So what I would start now from the beginning itself is the mulberry system, because why we picked on mulberry? Mulberry is one of the hardy system, and it has tremendous growth potential and known for multiple applications both in India and China and other places. And we also have now sufficient amount of genome as well as omics data being generated, and we feel that this is one of the kind of tree system which has features to treat as a potential perennial model system, popular and other system what people are arguing. So we have now some kind of information transcriptome data generated, and the traits which are governing for drought tolerance is being identified. So therefore we have generated some kind of additional information to understand the complex biological process in mulberry. Most of the molecular biologists generate transcriptome data by imposing rapid stress or rapid desiccation. And we are not following, as a physiologist, we understand that the plant is a system, plant is a society, multiple cells coordinate each other, therefore they modulate transcriptome or the abundance of the metabolites. From that context, we try to grow them in a big container, try to impose stress, in our case, drought stress, monitor the physiological process, use those kind of tissue samples for transcriptome analysis. Similarly, since mulberry has a high rate of bud break and development, we also trying to chase the bud development process and harvest the tissue for transcriptome and see what are the features which contribute for rapid bud break in mulberry. And we use different platforms, including Illumina to ISO-seq platforms to harvest the information in the form of transcriptome data. And we have more than 13 transcriptome data generated in one go, and they have been submitted to the database for the benefit of others. And we are also exploiting that information for our benefit as well. So the data what we have is complex, as everybody knows who are working on transcriptome. You get large amount of data. We try to compare between the one treatment versus another. We have a control versus droughts, and we have certain known genes getting altered, certain unknown genes, and we are interested in some kind of genes which are contributing for overall cell tolerance mechanism, belong to upstream or downstream regulatory pathways. So we are trying to assimilate, and we have no sufficient information captured and gathered and used for our further understanding of the complex interlinking networks operating in complex systems like mulberry. So what I want to say is that we are interested in upstream regulatory genes, and they are the one probably they govern multiple pathways at cellular level, and they govern the cell tolerance mechanisms. So we have now several transcription factors identified, shortlisted, and also confirmed the stress response nature across different types of stresses, not only drought, although we identified from drought-specific transcriptome library, these genes are also activated under different stresses. So in other words, our goal seems to be, our main aim is to identify the genes which are contributing for cell tolerance mechanisms. We have one of the major mechanisms, water relations, other one is cell tolerance mechanisms. So cell tolerance is again a complex character. You need to have efficient proteins to maintain the membrane integrity. Say, for example, protein turnover, cell cycle regulation, scavenging reactive oxygen species, many more like that. So we are looking for certain key genes which cover some of these cell tolerant traits so that we can manipulate or alter the plant phenotype if required as per our will. So we have shortlisted many genes. I don't have time to explain all of them. Say one of the genes called APTL2 family transcription factor, which is expressed under diverse stresses, as I mentioned. When we overexpress them in model system, tobacco, the cells, that is, I would say the cell lines overexpressing these proteins are able to survive better than the vector control or the wild type under several stresses. In this example, what I'm showing is NaCl stress. They're able to maintain cell division and cell metabolic process better than the wild type or untransformed or transformed vector control system. Similarly, we have other transcription factors like BHLH, basic helix loop like transcription factors, one of the genes which we identified, highly represented in our library and also validated. And it is also able to sustain growth and in fact induce multiple shoots even under stressful situations. What does it mean? We have no novel genes which are able to maintain cellular tolerance mechanisms or cell tolerance under stressful conditions. Similarly, just to switch the gear into the other hardy system, Professor Shailaja is an expert in finger millet. You know the finger millet is a hardy system. It is a C4 plant capable of growing under limited water conditions. We also have a large amount of transcriptome data in one of our DBT projects. And we found very interesting trait here that the tolerance is associated with more expression of basal regulatory genes or housekeeping genes. In fact, TBP associated factors, say for example, TABs are highly represented in the stress transcriptome, indicating that these are the good information. If the system is capable of maintaining housekeeping activity, probably it can maintain the stress response in a better way. So this way, this transcriptome data is giving us a lot of leads, and we have the housekeeping genes validation also being done. Say for example, one of the genes BTF3 identified in peanut, we actually, when we were expressed in tobacco system, we were able to maintain the cellular metabolism better way. Same way, another housekeeping genes like TEA data box binding protein CAF6 in finger millet is also able to contribute cell level tolerance. In other words, what we have found out from our transcriptome lead is that housekeeping genes are also very important. Don't think only about stress response. The important point I want to pick up on is that all the system what we are playing with have large amount of proteins or the genes unknown function. We call them as PUFs, proteins of unknown function, which are not characterized. You don't have any information, even in Arabidopsis model system. So we have around 30% approximately in most of these, like 33 to 40%, I would say, in these libraries of finger millet as well as mulberry. So our interest is to see what are these unique things there, why are they produced, and they are, some of them are highly upregulated. To another that, to understand their relevance, in collaboration with NCBS Professor Sodamini, we were able to develop a pipeline we called PUFAS, proteins of unknown function annotator server. Through this, we were able to actually extract some information about relevance of some of these PUFs or the proteins of unknown function. In fact, these PUFs fall into different categories, whatever we have analyzed, and we have some interesting data. Some of them we took forward for understanding the relevance. So for example, when we overexpress one of the PUFs like USP like, it's like, because we are not sure unless we validate fully and see what it does at the cellular level. So the overexpressed lines are able to maintain desiccation or manifold stress tolerance. They were able to survive better under heat stress. In fact, these seedlings are much better, and callus is in fact able to grow under stressful situations and a little bit better than the wild type or the vector control system. So what is that I'm saying? Again, they're indirectly contributing for cell tolerance mechanism. Probably we picked up on one of the interesting things. See, when you go at the system level, whole plant level also, we could get a similar phenotype in a tobacco system. we withhold irrigation and ask the plant to recover, the recovery is perfect when these USP-like genes are overexpressed in tobacco. So we actually use the transcriptome data, we use the PUFA server, and we landed in identifying probably a novel gene. Similarly, we have few more examples. In this case, USP, what I just now mentioned, not only doing something to the eukaryotic system, even in the prokaryotic system like E. coli, we were able to show that, say, for example, these red lines, what I'm showing indicate the transgenic or transformed bacterial cell were able to grow under stressful condition better than the non-transformed bacterial cells. So, in other words, it is imparting cellular level tolerance. So, I have many more examples. Since my time is limited, I just want to pick up on the other thing called RRM, that is RNA recognition motif protein, which we actually initially identified as PUFs. When we convert it into our PUFA server annotation, we could get that it is like RRM-like gene, and this gene also having some relevance with respect to stress tolerance, what I call it a cellular level tolerance. What it does internally, we need to understand further. There is a long way to go to dissect the complex interactions. But you know that RNA metabolism is very crucial, especially post-translational modifications and stabilization of RNA is very crucial. Probably this can also give tolerance to the wide array of stressors. So prospecting novel gene is the goal. Transcriptome is helping us. In fact, there are certain interesting pups where we call it a DUFs. They carry domains of unknown function, not only in our mulberry and finger system, what I'm talking about, it is also there in Arabidopsis. These are called unknown domains, undefined domains that make the protein unique, but we do not know. We actually featured upon some of those DUFs now. We are trying to understand the relevance. Say, for example, when we impose the plant at whole plant level drought stress, 40% soil fill capacity, 60% fill capacity. Some of these depths, what we numbered here in one of the graphs, what I'm showing here, you can see that they are upregulated. There is fold increase over control. That means they are relevant. What do they do? What is their function? A long way to go. We are in the process of understanding that. I just want to impress you with this data that when you silence one of the depths, we do not know what it does. We found the homologs in the model system, the question of Benthamiana. We try to silence them transiently using virus-induced gene silencing assay. You know that this assay is widely used. This is a PDS silence plant. When you silence a PDS, phyton desaturase, plant pro white phenotype. Same way, when we silence one of the depths 1068, I could see, or we could see elongation growth, the increase or vector control system. Long way to go. This is just a lead. We feel that some of these dubs and pubs are having long, the relevance in modulating not only stress tolerance, but also plant growth and development. And some of these genes have been identified from mulberry. So we feel that we will have a nice data or genomic resources to play with. So that's one part of the story. I mentioned as a fourth component, transcriptome to understand the plant-endophyte interactions. We have been working with the endophytes to trigger certain physiological process, especially specific traits contributing for drought or sunlight stress tolerance. And we have certain endophytes under ICR-funded program identified. They trigger certain process, and in case of certain crops like rice, as well as in tomato and chili. I'm not going to those. So we were asking the question, what is happening? What is the communication between the endophyte and the plant cell? And we used in one of our studies transcriptome-based approach, and we were able to understand at least to some level what is happening between the endophytes and the plant cell with respect to their communication. Part of the information is available now. We need to exploit more these kind of tools, that is especially metabolome, transcriptome, to understand further and pitch on the interactive mechanism. So this is one of the leads I would like to mention, that transcriptome is highly useful for such studies as well. In our experiments, whenever we do a stress experiment, sometimes in our expression studies, we come across splice variation. We did find in two cases. Say, for example, in rice, one of the genes called DBF1 is alternatively spliced. We shall not get into details of this why and how. Say, for example, Rasi and Telehamsa, when we took, we could find differential expression of these splice variants. And one of these splice variant has some other features like premature termination. We shall not discuss those aspects, but we do find splice variants. Similarly, we found similar type of splice variation in our mulberry system we have BHLH A and B version, and these two seems to be associated with stress response. You see the whole plant level stress versus fold change. If you see the variation in the expression of these splice variants. And when we analyze these splice variants, we are actually getting more and more information. One of the splice variant is missing miRNA binding, site. That means there is other modification, regulation possibilities. What is the message I want to throw? Splice variation is another important event associated with developmental process as well as stress response. So we can use the transcriptome analysis to identify the alternative splicing, as our speaker just now in the lead talk mentioned. Transcriptome to analyze alternative splicing is an option. So we have different types of alternative splicing. We actually made an attempt to identify the alternative splicing events when there is endophyte. In fact, endophyte fungus reduces the alternative splicing events in rice in one of our preliminary studies, what we reported. And similarly, we have now large volume of data of drought-specific transcriptome in mulberry. We're trying to identify the splice variation, and we have around 30 to 70% of the plant genes undergo alternative splicing. That literature information is available. Based on that lead, we are trying to identify the events or alternative splice events and try to see their relevance in imparting cellular level tolerance and growth. So this is what we have been doing. What is that? To conclude, I just want to tell you that omics, especially transcriptomics, is helping us a lot, biologists and physiologists like me, to understand the cellular level physiological process. A plant is capable of growing better than others because of the regulation, genetic level as well as the transcript level regulations. So we are using transcriptome to generate genomic resources. Even marker can be developed. You can have a large amount of genes which might be useful for targeted manipulation of traits of our choice. And novel pathways and gene discovery is possible, as I just now given some example like doves and fox. Unknown pathways can be unearthed. Regulatory genes can be identified to make sure that you can have a master regulator control a particular trait or a process. Another important issue would be that plant is not growing in isolation, as I mentioned. Physiological traits are triggered by the associated organisms like endophytes. So you have the endophyte genome as well as plant genome plays an important role. So the whole genome interaction can be studied. That is the transcriptome-based approach can be applied. So you have a meta-transcriptome being evolved. That means you can see what is happening overall when these two cells, plant cell as well as endophyte cells interact. You can actually harvest meta-transcriptomics and try to understand the complex interaction between the organisms. So in addition to that, you can also use them to find out the post-transcription regulation, basically understanding alternative splice events. So that's how I feel that transcriptome approach is to be used, and it is highly useful for our crop improvement program. So just to conclude, I must thank our students team, Lakshmi, Chaitra, Sanjeevan, Pooja, Jeevan, Tarun, Tinu, Ashwini, and Shivsharan. These are our team members who are contributing for mulberry work. They have significantly contributed for our transcriptome data. And I also would like to thank my mentor as well as collaborator, Professor Umashankar, Paramjit Purana, Sodamini, and head of the department who also collaborate with us on finger millet, Professor Nanjareddy. And the entire work is possible because of active collaboration with the University of Delhi South Campus, Center Sericulture Research Institute, Mysore, Groningen University, Netherlands, University of Saskatchewan, NCBS Bangalore, and FS University, INA, Germany. I'm very much thankful to NHTP, ICR for funding, as well as Department of Biotechnology for sustained support in the area of transcriptomics and gene discovery. With this, I would like to thank the organizer, mainly RC Jagdisha and Dr. B. Fakhruddin for giving me an opportunity to share our work to this august audience. Thank you very much. Back to you, ma'am.
Okay, thank you, Natraj. Hello.
Hello.
Yeah.
Thank you, Natraj, for a very good information on mulberry and other crop genomics, and this basic research is very useful for trade-based development. And I'm sure the PhD students, MSc students, and the young scientists got very good information from your talk. Thank you.
Thank you, madam. Thank you very much. Professor Chandrayappa, sir, thank you so much.
Oh, you are welcome. I have one question for you.
Please, sir.
Yeah. So it's related to stress. You told the endophytes are very, very important other than, the genotype, okay? But rhizosphere microorganisms, they play a very, very important role in, overcoming the different kinds of stresses. So have you considered that? Because no, some of the rhizosphere bacteria, say for example, even they have a, ability to control stomatal opening during the drought conditions. So if that is the case, you should have been included some aspects related to rhizosphere microorganisms.
Very valid question, sir. That's very correct. In fact, some of the hormones, say ethylene, is produced by these organisms which are associated with the soil system. We are not, going out of the plant body, sir. We are working within the plant.
Okay.
Yeah, thank you.
Yeah. One more small question. I don't
Yes,
want
sir.
to dominate asking too many questions. It's related to mulberry. For
Yes,
a long
sir.
time, I was thinking, compared to any other crops, mulberry is highly resistant to insects. Okay.
Correct.
So why? What could be the reason? Whether it's any metabolites are there or nature of the plant itself? I was wondering. Can you please highlight on it?
Yes, sir. We have a project. Professor Umashankar is leading that on sugar mimicking alkaloid, which in fact prevent the digestive system or activity of other insects, which is not the case in mulberry. So there is a nice storyline on that, sir. We can definitely share more information on that. Yes.
Okay.
Yeah.
Thank you.
Thank you so much, sir.
You are welcome. Madam,
Thank you, Natraj.
any audience can ask the question? Is it allowed? If there is a time.
Since Natraj has given some time,
Okay.
I think we can go for two questions. Two questions.
Yeah. Anyone from audience
If anybody
would like to ask question?
Sir, excuse me, am I audible?
Yes.
Yes.
Professor Natraj, sir, it was an excellent presentation. I just wanted to know, when you analyze alternate splicing in the host upon exposure to endophytes, did you look at the alternate splicing variations in the endophyte itself in response to hosts?
Yeah, a very good question. That's right. We don't have that combination of analysis, right? That's right. You may have independent alternative splicing happening even in the endophytes. So this I just shared small information, and some of the additional information, if you want, you can also harvest from the paper. That's right. That's a very important thing, and I don't have answer for that. What is happening there? That's right. Thank you.
Dr. Nataraj Karaba, can you?
Yes, sir.
Audible? Yeah, Faiz here. Yeah, wonderful presentation. I would like to know something about the endophytes. Are you focusing on unculturable or culturable or as a whole holobiome?
Hello. Yeah. So we are only bothered about culturable endophytes, Professor Ayas. Thank you so much for the question. Yeah, we are bothered now at this point of time, culturable endophytes. That's right.
Yeah.
You are right. There are so many unculturable, and you are an expert. You know better than anyone else here. And more complex interaction happens. They cannot be harvested out of the plant body. That's right.
Yeah, because then how do you link? So it would be, are you defining the organisms clearly?
I missed some part of your query, Ayas.
Are you able to define the organisms when you do the transcriptome analysis?
Right. We know what organism it is. We identify, and we know what definite organism we are playing with. Yes.
Okay.
Yeah.
Good.
Yeah. Thank you, Payas. Yeah.
Thank you.
Madam, you can invite the next speaker.
Thank you very much, Chairman and Co-Chair. Thank you so much.
You
Thanks
are
for the
welcome.
opportunity.
You are welcome. Thank you.
We'll move to the next speaker, Dr. M. Ravindran, Professor at CMB, TNAU, Coimbatore. He will speak on accelerating stress gene recovery through genomic and bioinformatics. Dr. Ravindran is eminent scientist in TNAU, and he is one of the pioneer workers in the functional genomics. And today he will tell about his novel work and discovery of novel genes. Ravindran, welcome.
Yes, madam. Very good morning, and thank you for the opportunity given. Am I audible, madam?
Yes, you are. Please go ahead.
My slides are visible?
Yes, you are audible.
Okay. Thank you very much, and good morning, everyone. And thanks for the opportunity given to share some of my experiences on using various genomics and bioinformatic tools in accelerating gene discovery for crop improvement program. So
make full screen of your slide. You can make full screen of. Yeah, thank you. Go ahead.
Right, madam. So coming to the introduction, to meet the growing demand due to increasing population, we are in a position, or we are in need of doubling our food production by 2050. And especially with regard to rice, when you look into the growth in demand, so we need to reach around 160 million tons from the current level of around 100 million tons. But the current yield trends are not satisfactory or not sufficient enough to meet the growth in demand. So when we look into the prevailing growth trend and the required growth in demand, so we need to grow at the rate of around 2.4% per year in terms of yield, but our current trends are around 1% growth. So this clearly indicates that prevailing yield trends are not sufficient enough to meet the growth in demand. And we need to further increase the rice yield potential or to improve the potential yield. So we need to overcome challenges like yield plateau. So when you look into the yield trends in rice yields have undergone two major leaps right from 1950s. So first during 1960s, through the introduction of this green revolution gene or the SG1 gene, we were able to improve the harvest index. And secondly, through the introduction of hybrids during 80s. So with these two technologies, we were able to increase the rice yields to 3 to 4-fold. But when you look into the yield trends during the past 15-20 years, it's plateaued around 6 to 7 tons. Under average field conditions, our productivity is stagnating around 4 to 5 tons per hectare. The second major limitation is that declining land, water, and labor resources. So this is fast becoming a major factor limiting rice productivity. And thirdly, due to increase in the occurrence of several abiotic and biotic stresses due to climate change, so the yield levels are reduced so much. So year by year, we are seeing increase in the gap between the potential yield and the realized yield. So how to improve the yield potential or how to increase the food production or double the food production by 2050, there are 3-4 strategies suggested. So one is to increase the food production by increasing the area under cultivation. That is definitely not possible because of increasing urbanization and all. And crop diversification and reducing the yield losses occurred due to various pest and diseases and abiotic stresses, and finally by reducing the post-harvest losses. So out of several opportunities available, so reducing the yield gap between the potential yield and the realized yield, so this lies with the breeders. So this holds promise in increasing the productivity or increasing the food production. So when you look into the gap between the potential yield and the realized yield, so under if you take rice potential yield of rice, it's around 8 to 10 tons per hectare. So wherever we go, so the newly released varieties are having the potential to perform or yield up to 8 to 10 tons per hectare. But our national average is around 4 to 4.5 tons per hectare. So in most of the areas, so which are very vulnerable to occurrence of abiotic stresses, the yield levels are 1 to 2 tons per hectare. So if we are able to equip our rice varieties So to perform better under these marginal environment, so then there are better chances to increase the food production, so without much struggle. So when we look into the possibility of developing such resilient rice varieties adapted to a marginal environment or stress-prone environment, the progress through conventional breeding is limited. And here the applications of recent innovations like marker-assisted breeding, transgenics, molecular breeding, genomics-assisted breeding, they hold a promise in accelerating the genetic gains. So this has been well demonstrated in case of maize, where so no tool can replace plant breeding, but they can assist the breeding or the breeders in improving their selection efficiency. So they will definitely help in accelerating genetic gains. So the yield advantage or the genetic gain, what you will achieve in 20 years through conventional breeding, this can be very well achieved in less than 10 years by using these tools. So either alone or in combinations. So in case of maize, this is well demonstrated. So the combination of conventional plant breeding with marker-assisted breeding and the transgenics, so this has allowed in the acceleration of genetic gains within 5 to 6 years. So how to move forward? So there are n number of literatures, thousands and thousands of literatures which are dumped in the public domain. But when you look into the practical utility or the deployment of these informations into plant breeding, so the gap is so huge. So especially with regard to rice, so there are more than 5,000 publications with regard to QTL discovery, but hardly 500 are put into use, especially with regard to drought tolerance. When you look into the literature, there are more than 2,000 QTLs published in the literature, drought tolerance related QTLs. But hardly 5 to 10 QTLs have been put into practical applications or breeding application. So why there is a huge gap between discovery and the deployment or practical applications? So the reasons are like, as far as my understanding or my survey goes, so the QTLs for component traits are very limited. So especially with regard to abiotic stress tolerance, so they have mapped QTLs for yield under stress or yield per se. So the QTLs for component traits are very limited. So this makes the deployment very difficult. So it will vary from environment to environment. And most of the QTLs are large intervals and with very minor effect, less than 10% effect. So because they are all what you call quantitative traits and they will act additively. So each and every QTL will contribute so in an additive manner. So in addition, the QTLs which have been discovered in one genetic background, so it has not been validated across different genetic background. Sporadic attempts have been made. So with these informations and the limitations, how to move forward? So we can plan for fine mapping and understanding the molecular basis of QTL so that it will allow us to generate improved knowledge on the physiological and molecular basis of these QTLs, and then identifying the candidate genes, develop gene-based markers for precise introgression of these traits into the susceptible background through molecular breeding. So far, before some 5 to 10 years or until the advancements in genomics, we were going behind the classical map-based cloning or positional cloning, which was a difficult or tedious process. So this will involve a two-stage research. First one, they will identify the QTLs controlling the trait. And then in the second stage, so they will develop a genomic DNA library and then screen the library using the markers link, identify the back clones harboring the candidate gene or the markers, so linked to the trait, and then identifying the candidates. So using this approach, classical approach, so several landmark discoveries have been made. So the first example is DEP1, dense erect panicle architecture, which was cloned successfully during 2010. And then SUB1, a major milestone for the successful demonstration of marker-assisted backcross breeding in croplands. So, and then thirdly, very recent innovation on DRO1, so deep rooting architecture. So several such innovations have been made in several croplands, agriculturally important plant, using this approach. But when you look into the time taken, so it's a long journey. So especially when you take submergence tolerance in rice as an example, so which is one of the well demonstrated case for marker assisted breeding. So the research started around 1950 when Dr. Meckel was doing his PhD at Erie, and he identified the donor, and finally this was put into breeding applications, and the first submergent tolerant variety Swarna Sub-1 was developed through marker assisted backcross breeding and commercialized during 2010. It took almost 60 years so at Erie to from the discovery of the donor till the release of commercial variety using this finding. So we cannot afford for those many years for each and every trait or identifying candidate genes controlling different traits. So advancements in genomics and bioinformatics, so they have allowed us to develop so a set of protocols. So for measuring the gene activity or for surveying the sequence polymorphism or combining both. So how to correlate the expression with sequence polymorphism and the linkage map. So genetical genomics procedures have been developed. So these tools, genomics combined with analyzing sequence polymorphism, so they have allowed us to move faster in identifying candidate genes controlling such quantitative traits. So these advancements, so they have allowed us to generate improved knowledge or to make faster progress in terms of gene discovery. When you look into the recent review, so there are more than 5,000 genes which are functionally validated with reference to rice. So we have also made some attempts, 2-3 attempts for using these genomic tools so in accelerating gene discovery. So that I will discuss with you now. So especially with regard to drought tolerance in rice. So rice is endowed with huge amount of genetic diversity for various drought tolerance related traits. So we went for using microarray based measurement of gene activity in a set of contrasting rice genotype differing in their drought tolerance, IR64 and above. So IR64 is a mega variety, but susceptible to drought, and Apo is a drought tolerant. You can Apo is popularly known as El Nino variety, highly suitable or adapted to stress-prone environments. So we analyzed or we profiled the drought responsive gene expression by using microarrays. And when you look into the drought responsive changes in gene expression between control and drought stressed situations. So both IR64 and APO are indica varieties, but they differ in their stress perception and signal amplification and signal transduction and activation of downstream genes. So there were around 330 genes that were upregulated in both the varieties during drought. But when you look into the commonality between the two genotypes, hardly around 10 genes that were found to be common between the two varieties. So both are rice varieties, but you can see the molecular diversity. So genomics has allowed us to what you call capture the differences at molecular level in such a precise manner. Similarly, with reference to downregulated genes during drought, out of around 260 genes, only 2 genes were found to be common between them. Remaining are specific to both the varieties. When you look into the number of genes or magnitude of amplification during stress condition, so the APO was able to activate or suppress so twofold number of genes when compared to the susceptible IR64. So APO it has a powerful mechanism to perceive the signal stress early and amplify the signal and activate more number of genes during stress condition, so to make it adapted to the stress environment. So when those genes were mapped onto metabolic pathways, so we were able to identify so genes specific to asthma protection, carbohydrate metabolism, cell wall biosynthesis, and these many transcription factors, they were all found to be upregulated in APO. See, this is log-10 ratio. So all are upregulated by more than 10-fold. But when you look into their expression in IR64, so almost all of them are negatively regulated, down-regulated. So why there is a huge difference in terms of the activation of these transcription factor, which has the ability to activate hundreds of downstream genes. This may underlie the enhanced drought tolerance, so exhibited by APO. So the advancements in bioinformatics, so this has allowed us to measure the rare phenomena called co-expression. So co-expression is what you call ability of a genome to activate a set of genes involved in the simultaneously, involved in the same pathway or which are located nearby in the genome. So it's one of the well understood processes in eukaryotic genome. So when the plants, they germinate, they activate hundreds of genes. When the plants, so they enter into reproductive stage from vegetative stage, they will shut down hundreds of genes, and they will activate a new set of hundreds of genes. So this advancements in bioinformatics, so our generation of physical map in rice, so this has allowed us to generate or identify such co-expression clusters. The drought tolerant APO, so this has around 14 clusters showing co-expression, whereas this susceptible IR64 had only 5. When such co-expression clusters were mapped onto rice genetic map, so very interestingly, many of the co-expression clusters are co-localized with, so the QTLs associated with osmotic adjustment, cell membrane stability, drought tolerance, yield under stress, etc. So then we went for identifying the genes located near the QTLs or markers reported. We ended up with some set of genes. And then we did one more analysis called genotype versus phenotype analysis. Microarray identified around 200 upregulated genes. So we mapped all those genes onto rice physical map and then identified the SSRs nearby those candidate gene, mutative candidate gene, and surveyed the polymorphism between IR64 and APO. We identified 22 markers or 22 SSR markers exhibiting polymorphism as well as co-localized with these drought responsive genes. And then we associated the polymorphism with the drought tolerance ability of the RILs developed between IR64 and APO. So we were able to narrow down the around 15 to 20 different genes from the set of around 250 genes. All are having more than 10% effect on drought tolerance. So these are our candidates now. So we have started validating these 15 genes one by one. So I will just show you one example how precisely this microarray combined with bioinformatic analysis identified a candidate gene. This osmotin-like protein, so co-localized with the QTL on 1.1. So when we cloned this osmotin from both IR64 and APO, there were 3 SNPs between these two alleles. And when we modeled and docked these two alleles against the potential ligand beta-1,3-glucan and 1,4-glucan, the APO allele was found to have more number of binding sites to beta-1,4-glucan, a predominant component of monocot cell walls, whereas the susceptible IR64 was not having any binding site towards this beta-1,4-glucan. So this was again validated through docking studies. And then we wanted to validate this bioinformatic prediction through wet lab experiment. We developed tobacco transgenic plants overexpressed with IR64 and APO alleles of osmotin. We confirmed the presence of transgenes, the contrasting alleles of the transgenes through PCR. And then when the transgenic plants were subjected to stringent phenotyping, so the tobacco transgenic plants overexpressed with APO allele of osmotin, so they were found to exhibit enhanced level of tolerance against polyethylene glycol stress as well as sodium chloride stress when compared to the transgenic plants overexpressed with IR64 allele of osmotin as well as the non-transgenic plants. So the DAP staining, diaminobenzene staining. So this clearly indicated that the plants overexpressed with apoallele of aspartame, they did not undergo the severe intensity of stress when compared to the non-transgenics, and the transgenic plants overexpressed with IR64 allele overexpressing plant. Again, the same study, the same results were observed when the plants were subjected to drought stress during reproductive stage. Here also you can see this is non-transgenic plant. 30 days after imposing drought stress, you can see visible reduction in the growth. Whereas the apoallele overexpressed plants, they were able to exhibit on par performance with the control plants, non-stressed plants, and they exhibited fantastic recovery during rewatering. So all of them, they were subjected to equal intensity of stress. When they were rewatered, the apoallele overexpressed plants, they exhibited 100% recovery when compared to the non-transgenics, which exhibited only 15% recovery. This susceptible allele overexpressed plant, so they expressed around 50% recovery. So this clearly indicates that the combination of your gene expression profiling and bioinformatics, they allowed us to pinpoint or narrow down candidate genes in a precise manner. And the second example of using another genomics tools called proteomics. So as we all know, root system architecture, they play a major role in drought avoidance. So they help the plant to extract more water from deeper layers of the soil so that the plants can withstand drought much better than the shallow rooted ones. So we selected one such landrace called Nutripathu. So this one, and we developed a mapping population between a shallow rooted IR20 and the real population was phenotyped thoroughly for root system characters or root characters. So root penetration ability, root length, number of roots, etc. So seedling level screening was done. So we identified rills exhibiting contrasting root growth pattern like that of parents. And those four categories, so the shallow rooted IR20, deep rooted Nutripathu, and the rills having root growth characters similar to IR20 and similar to Nutripathu were selected, and 5 rills from each deep rooted category and shallow rooted category were taken, and they were bulked. And both the roots isolated from proteins isolated from the roots of contrasting parents and the contrasting bulked rills, they were subjected to proteomic 2D page analysis. So when the expression pattern of the proteins, they were compared between the 4 categories, we were able to identify proteins exhibiting co-segregation or co-inheritance along with the root growth behavior. So we identified somewhere around 30 proteins which were either upregulated along with Nutripathu root growth behavior or downregulated along with IR20 root growth behavior. So then those proteins were identified through mass spectrometry, and we mapped all those 30 proteins against rice genetic map, QTLs, associated with root growth behavior, and then identified the proteins which are co-localized with root growth QTLs, and we shortlisted somewhere around 3 proteins which are having very high level of co-localization, exhibiting high level of co-localization with the root growth related QTLs. So one such gene is acylreductone dioxygenase involved in ethylene biosynthesis and lignin biosynthesis. So lignin, as you all know, so they have a major role in the cell wall morphogenesis. So when this gene was overexpressed in a shallow rooted ASD16, we isolated this gene from the deep rooted Nutripathu and overexpressed it in a shallow rooted ASD16, our popular variety. You can see the induction of roots here. So 5 days after transferring into rooting media, so these transgenic plantlets, they were found to induce the proliferation of roots. So in much faster manner when compared to the non-transgenic calli or non-transgenic plantlet. So 8 days after putting into the rooting media, so these transgenic plants, they were found to show proliferation of roots much better than the non-transgenic plants. So when the plants were advanced to T3 generation, so you can see the significant influence of the PCR positive transgenic plant when compared to the non-transgenics and PCR negative plant. They were found to have significant influence on the root growth behavior. When precision phenotyping was carried out, so by using imaging and software prediction, you can see this overexpression of acid-reductase dioxygenase was found to have significant influence on initiation and proliferation of lateral roots when compared to the primary roots. So the overexpression of this acid-reductase dioxygenase allowed the transgenic plants to initiate and elongate the lateral roots in a much faster manner when compared to the non-transgenic plants. And the third example is how the RNA sequencing allowed us to identify novel candidate genes controlling salinity tolerance in uncharacterized genomes like finger millet. So now, your university, UAS Bangalore, they have unraveled the genome complexity of finger millet during 2018-20. During 14, this genome was not studied well. So, but phenomic studies, clearly indicated that the finger millet exhibits much better tolerance against salinity when compared to rice. It can withstand even up to 300 millimolar salinity. So we went for analyzing the transcriptome of finger millet by using the novel genomic tool called RNA sequencing. When RNA-seq data was mapped against rice and Setaria, so rice was found to have, mapping against rice allowed us to generate quality data when compared to Setaria italica. So we identified the differentially expressed genes, by using rice as a reference map. And then when these genes were annotated, we were able to identify 14 different genes which are not reported in the literature so far, related to salinity tolerance. So we have started validating them one by one. So using the race PCR, we were able to clone 2 different genes. One is NAC67, another one is ER of ethylene response factor. We validated them through functional genomics, genetic transformation. We overexpressed this ragi gene or finger millet gene in rice, and then we were able to see significant influence of this gene on salinity tolerance in rice. So overexpression of this gene had around 15 to 20% influence on rice yield under salinity. And the last example is how the genomics allowed us to develop novel genetic stocks and the characterization of these novel genetic stocks. So as we all know, mutagenesis is one of the powerful tools in functional genomics. Mutagenesis by using either physical mutagen or chemical mutagen or molecular tools like tDNA insertion or now genome editing. So they will allow us to generate gain of function mutants or loss of function mutant, and the characterization of those mutants will allow us to identify the novel genes. So here we used such tools in developing a rice variety which will allow us to go for labor and water saving rice cultivation. So as I said, declining water and labor resources, so it necessitates us to develop a method of rice cultivation requiring less labor and less water. So direct seeded rice cultivation is one of the potential alternatives, but non-availability of, there are several limitations. One of the major limitations is weed management in rice. We don't have efficient post-emergence weed control in rice using chemicals. So here we generated thousands of 1 lakh mutants in a national network project involving 6 different institutes. And at TNAU, so Dr. Robin was leading this project and I was part of that project. So we identified, when we screened 1 lakh mutants against a broad spectrum non-selective herbicide imazathioper. So this is a very famous technology in USA named as Clearfield technology. So you have Clearfield technology in all crops in USA: rice, maize, groundnut, sunflower, wheat, canola, etc. So here we use this herbicide in pulses ecosystem in India. So we do not use this in rice because rice is sensitive. So we developed a mutant, so exhibiting resistance against this chemical. So screening, why I am telling is this. So mutagenesis is one of the powerful tool in creating novel traits or introducing novel traits. Out of 1 lakh plants, one plant survived. And when this was validated in the subsequent generations, and you can see the efficacy of this mutant trait or novel trait in chemical weed management in rice. So rice was sown under direct seeded sowing, and this was allowed, the weeds were allowed to grow. So after 1 month, you can see there was no difference between rice and wheat. The weeds were completely masking the rice crop. And then when this chemical was sprayed, this is 20 days after spray. So all those weeds, they started drying, yellowing and drying. So this was after 1 month. So only the resistant mutant progenies, they were surviving. And then with the help of our partners, we identified markers linked to the traits. Here, bioinformatics played a major role in accelerating the discovery of the marker. There were 3 homologs in this gene family, AHAS or acetolactate synthase. So we developed a simple F2 population between Co51 and the herbicide tolerant mutant. And then this F2 leaf samples were collected on one side and herbicide spray was given on the other side. And then the phenotype versus genotype analysis identified. So this particular homolog on chromosome 2, this was found to be linked to the trait. And then we identified a tightly linked SSR marker linked to the particular trait. So now we have cloned this gene. Bioinformatics prediction, so this has allowed us to narrow down the SNPs, so which may form the basis for this trait. So we are in the process of validating the SNP, which forms the basis for this novel trait in rice. On the other side, we have started using SSR marker for product development. You can see the efficacy of this trait on chemical weed control. So almost 90% efficiency in terms of weed control using this chemical. So we are making collaborative efforts. We 6 institutes, the lead center is IRI, then NRCPB, then UAS Bangalore, Dr. Seshashayi, and then TNAU, IIRR, and NRRI. So we are in the process of introgressing these novel traits into mega varieties of India to develop herbicide tolerant rice, a new technology which will help us to optimize or standardize direct seeded rice cultivation in rice. So this domestication of rice started with the spreading type. So domestication of rice was mainly through selecting for the loss of function mutants. So from the spreading type, we selected the prostate type, and then from the prostate, we selected the semi-dwarf trait, and then we developed the super. It's all were through the knockout mutations or the loss of function mutations. But the development of improved genotype or the elite resilient genotype, it depends on discovery of novel alleles of candidate gene and assembling them in the suitable genetic background using genomics approaches. So this needs the applications or the deployment of genomic tools like gene expression profiling, resequencing, allele mining, haplotype diversity analysis, and then innovating mating designs assisted by genomics like genomic selection or magic. So which will assemble, which will allow us to assemble even hundreds of superior alleles in a single mating design. So such a powerful technologies are available. So combination of these tools, this will allow us to develop resilient rice varieties in a faster, in an accelerated manner. So we should not hesitate. So to combine these tools with plant breeding, so definitely we will be able to reach the goal in much faster time than by using the conventional breeding alone. So thank you for the opportunity given. I'll be happy to answer any questions. So finally, I would like to thank my alma mater TNAU, all my lab members, my collaborators funding agencies, especially DBT New Delhi, and thanks for the organizers to give me an opportunity. Thank you very much.
Hello, madam. Yeah. So,
Hello, madam.
madam, can you hear? Hello. ಅವರು ರೆಸ್ಪಾಂಡ್ ಮಾಡ್ತಾರೆ ಮೇಡಮ್.
Hello.
Hello.
Sir, am I audible? This is Ravindran.
Can I? Hello.
Yes.
You know, you gave a lot of emphasis on drought resistance. But you know what? One thing that we should know is always there will be some trade-off. If you make plant resistant
Can you
to
be a little bit louder?
Yeah. So you know, when you make a plant resistant to some kind of stressor, then always there will be some kind of a trade-off. That means there will be a drastic effect on the yield. Okay, particularly in case of.
Okay.
Can you hear me? Can you hear me?
Sir.
Yeah. Particularly when you are making plant to produce more roots.
Sir, can you be little bit louder? Sir, can you be little bit louder? Your voice is very feeble.
Again, I'm repeating the question. So when you make a plant to become a resistance for various factors like biotic and abiotic factors, then always there will be some trade-off on the yield. For example, in your case,
yes,
you are developing varieties for long roots. So, there will be redistribution of nutrients, carbohydrates or whatever it may be, photosynthesis more for root development. How much it is going to affect on yield? Do you have any idea? Hello? No response.
Hello?
Hello, Doctor. Yeah.
Sir, can you hear me?
Yeah.
So as you said, all these stresses, abiotic stresses, they will have significant influence on the yield. But there are favorable loci in that tolerant varieties, which will allow us to reduce the yield loss. So that is our aim.
Okay.
Our aim is to reduce the yield gap, not to make a rice plant which will not be affected by drought. That is impossible.
Because keeping a plant alive is not a needed character. At the same time, overall yield, production also important. So, do you take care of those?
Sir, actually, I will tell you an example. So, when you subject the plants to drought or any dehydration stress, the plants will tend to lose water. Okay. So, the susceptible varieties, they will lose water rapidly. They will reach very minimum level of relative water content, whereas the tolerant varieties, they will allow the plant or they will lose water very slowly, so that they will maintain enough moisture or water inside the plant. The effect will not be that much significant. That's it.
No, that is okay. But I'm talking about increasing the root length. So the biomass towards the increase of root, that can affect on the yield because the photosynthesis will be diverted more for root development. Because
Yes,
that is
sir.
a major area when you are working. I was a little bit concerned about that. How much increase of root biomass is going to affect on the overall yield?
Sir, that's what. When you talk about this partitioning or the conversion of or mobilization of photosynthetics for development of roots, so your argument or your claim will be right in case of tall varieties, right? Now we have systems or we have tools to manipulate the plant architecture. So the plants, you will be able to break the linkage between height and drought tolerance. There are successful examples, so made by Arvind Kumar. So the root development will not be continuing during the stress condition, but constitutively they will have longer roots. So the deep root ones, they will have longer roots under normal condition. During stress condition, they will help the plant to extract water from deeper layers of the soil when compared to the shallow rooted one. That's it.
Okay.
Your argument will be right in case of genotypes which will maintain their roots during the stress condition. The partitioning will be diverted.
Okay. Yeah, thank you, sir. Madam, any audience can ask one or two questions before we move on?
May I ask Dr. Ravindran one question, please?
Yes, sir.
May I ask? May I ask?
Yeah, yes, sir. Sure, please.
Yeah, this is Dr. Soumitra Paul from University of Calcutta. So, my question is to Dr. Ravindran that you have used osmotin gene from IR64 and from the Apo variety. And you have shown that there is a difference in the gene expression of this osmotin gene during drought.
Yes, sir.
But yeah, right. So did you check, you have checked the SNP mutation on the gene, but generally we know that the gene regulation depends on the promoter activity, generally.
It's in the promoter, yes, sir.
Yeah. So
we are in the process of validating it. We have
Yeah.
observed interesting differences in the promoter also. Okay.
Okay. So did you check any SNP mutation in the promoter region?
Yes, sir. Yes, sir. There are. One small indel is there.
Yeah.
Okay.
Okay.
We are in the process of validating that. I haven't shown as we are in the process of validating it.
Okay. 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 Thank you. Thank you, Dr. Ravindran. And thank you for your nice presentation.
Thank you.
Yeah, thank you. Madam, Dr. Shailaja, she is not there.
Sir, some network.
You ask Dr. Shivprasad. You call upon Dr. Shivprasad.
Yeah. So with the permission of chairman, shall I go ahead to announce a small change in the order of the speakers?
Yes, sir. Yes, sir.
Yeah. Now there is a small changes. Now Dr. P.V. Shivprasad, TA for NCBS, is going to give a talk instead of Dr. Amit C.R. Mitra from ICR, because he has some important work in his institution. So I request Dr. Amit to give later. So at the same time, I am requesting the Shivprasad to go ahead and give a talk.
Okay. Can you all see my slides? Yeah.
So again,
yeah. Thank you for the kind introduction. I'm sorry, actually, I don't work here. I had to come here because of some other reason. That's why there is a small change in the program. I apologize for barging in, not in my schedule. Thank you for the introduction. So I'm Shivprasad. I work at NCBS, close by, not more than 5 kilometers from here. So I'm here like a guest sitting in the hub where all this is being done. So my lab is around 7 years old in NCBS. So we are under TFR, and TFR comes under Department of Atomic Energy. In my lab, we work with all these model systems. Some of them you have already seen in a couple of days. You have seen pictures of those. Most of the world looks at Arabidopsis because it's an excellent model system. We purposely look at other plants because they are more interesting. Now, for my talk, I need to introduce two different proteins. Both are nicely named, so you won't forget them. The first one is called dicer or dicer-like protein. So just like a name suggests, this is a protein which can cut long double-strand RNA bits into uniform size bits, just like a very good cook. So if the cook is good, all these bits will be of uniform size. These proteins are extremely robust. They know how to cut. They cut either 21 or 22 or 24 nucleotide small bits from these RNA substrates. The name of the idea for the name came from a student. She was cooking and she thought that just like if she can cook nicely, cut nicely, this protein must be cutting nicely. She was working with flies. The protein is universal. It's present in all walks of life. In plants, the number of dicers are more in number compared to animals. So at least there are 4 dicers in plants. The second key protein is called argonaut. This is an interesting protein. Again, the name is because the mutant in Arabidopsis has this thin, long, elongated leaf. And somebody, we still don't know who the person is, they thought this is resemblant to octopus called Argonaut argo and named it as argonaut. This is a key protein because it can pick up the small RNA made by dicer, the protein which I described previously, and look for Watson-Crick complementarity in the target region, both DNA and RNA. And once it finds in the complementarity in that short region, it can go and regulate those RNAs. Just like dicers, there are many more argonauts in plants, sometimes up to 20 in monocots. Many of those we don't know what they do. Now, together, these two proteins, they make what we call it as silencing pathway or RNA silencing. This is a major signaling pathway regulating many coding regions as well as non-coding regions. Typically, RNA is RNA reduction, RNA-like structure. This is cut by the dicer, the one which cuts to make small RNAs. These get loaded into argonaut. In the argonaut, one strand gets degraded, and now it is ready to go and make the pairing. The pairing can be with the mRNA. Typically, when it pairs with the mRNA, the result is a drastic, it's a cut, which is called a splicing because not these double-strand RNA gets cut, only the target RNA gets cut. And if a small RNA targets the RNA, the target RNA will be completely removed, and then there will be hardly production of protein. This is how most of the small RNAs in plants work. In animals, though, the targeting is mostly through translational repression, which we don't see much in plants, and we don't know the reason for this. But the most interesting reason part of this pathway is that certain argonauts with certain sized small RNAs can induce what we call it as epigenetics at the level of transcriptional silencing, and this is brought about by induction of DNA methylation and histone modifications. So this can be seen only in plants and Pombe so far, and it is called RNA-directed DNA methylation. And it is special because if you induce such modification, the epigenetic modification, the change can be inherited almost in a Lamarckian fashion. So Lamarck should have probably not used giraffe as example, he should have picked plants as example. So this change can help plants to cope with diverse changes, including adaptation to newer conditions. The whole pathway, it regulates most of the transcription factors and many other part of the genome. So in our lab, we try to see how these small RNAs are made, what are the rules for them, what kind of dicers are involved, what are the complexes involved. We also try to see what kind of argonauts are present in rice, which are different from Arabidopsis. Arabidopsis ones are well studied. We try to see what's the result of this pathway, finally ending up targeting RNAs or other part of the genome. We also try to see molecular rules for induction and maintenance of epigenetic changes in plants. So one of the pathway here is called microRNA pathway. So I'm giving you a little bit more introduction here because it's relevant for this talk. MicroRNAs are coded by the genome, just like any other mRNA coding region, but they are short, they are made by pol 2, and this RNA gets a foldback structure. From this long foldback structure, one small RNA is made by one of the dicer, and this can typically go and target the RNA. Here, the confirmation for targeting the RNA comes by doing a sequencing called degradome. In this degradome sequencing, this bit of the RNA is cloned from a given tissue, from a given plant, and the cut is very precise between position 10 and 11. And so if you find these cut bits in the cell, then you know that's the result of a specific microRNA cleavage. Now, one of the long-standing question is why from long structure, this microRNA precursor structure, only one microRNA is generated. You may have up to 1 kb of this long stem loop structure. So what we found is that the length of this loop is absolutely important. It's around 50 base pair. And recently in the lab, Anushree has found that this region where microRNA or its complementary region are made, they should have a very specific G or C. So the reason I am mentioning here without much details is because this is quite important for many reasons. One of the reason is for functional studies across plants where you don't want to do a knockout, the best strategy is artificial microRNA. This is a system developed in Detlef Weigel's lab. And the problem with this tool is that it will give many candidates, but very few will be really useful. Very few of them are going to knock down a given RNA. The reason is they don't take into consideration the GC preference that we have found. And with various tools, we show that we can, if you incorporate the GC signature that we have found, slightly elevated than other regions in the genome, then one will be able to make a very good targeting. Like we show here, the optimum level GC will knock down the gene completely. Here the assay is like this. In a gene, this gene can induce anthocyanin production in tobacco. On top, we design artificial microRNAs of various types. And the best ones are the ones which have around 50% GC in that 21 nucleotide microRNA. With the right signature, it should be in specific places G or C, then you get the maximum knockdown. So I'm just very briefly mentioning this because those of you who are interested in functional genomic studies might use this tool, and this is a very robust tool. Not many people can do CRISPR-Cas targeting, and because Cas9 stays in the system, it can lead to many undesirable events that you can simply avoid by having a artificial microRNA. So here you can do specific knockdown. So the most of the talk I'm going to focus on this. So we all continue to depend on plants, and it has not changed since we evolved. So whether it's for food, clothing, medicine, or shelter, we just want plants to produce more and more of what they are already doing without realizing that plants are already overburdened. It's like a two-wheeler carrying 1 ton of weight. We are already making them do it. And on top, we just want now climate resilient crops that will still produce yield irrespective of whatever the new condition is. And on top, we want them all to be in a sustainable manner. So the goal is to improve yield in no matter what condition we bring to the plants. So it is very well known that incremental knowledge is probably not enough. So we need to really think out of the box. So first we have to see why plants are making all this. Are they making because they need clothing or medicine? No. So they make because they are in partnership with animals. So for a long time, around 100 million years ago, that's really a long time of dinosaurs. Dinosaurs didn't enjoy colorful flowers and fruits because angiosperms, flowering plants, evolved roughly around 125 million years ago. So this is when plants tricked animals to spread their genes by bribing with colorful flowers as well as nutritious fruits or seeds so that they can be dispersed everywhere. And this led to the real evolution of flowering plants. Even Darwin, who had answer for everything, he thought this is an abominable mystery. And in this partnership, it not just allowed plants to evolve rapidly in sort of no time, but also helped evolution of animal forms, which were not seen previously, including, for example, us. So this partnership was not just direct, not very simple, because the animals put money on two things, one on locomotion and another one on thinking, consciousness. And whereas plants were stationary, they had to put money on chemistry, and they ended up as the best chemists in the world. So they not just make carbohydrates and protein, thousands of fold more than what they really need, but they also have many different chemicals to counter animals. Nicotine can induce damage to the collagen, the muscle is gone in the animal. Caffeine stops hunger in various organisms. Scopolamine is a medicine for Parkinson's at low doses, but it induces madness at high doses. Cyanides, cyanides can stop life cycle of many animals. Flavonols are interesting. They can alter the taste buds. Like in gooseberry, it tastes bitter. After that, whatever you eat is sweet. You can also have flavonols behaving the other way. There are photosensitizers which can induce massive macromolecular damage in systems. Then there are photoprotectants, just like the carbohydrates and proteins. These are also bribes. They can help to protect macromolecules. Then very specific molecules, THC, acin, cannabis, these are supposed to induce changes in our consciousness. So animals, including us, we are all fond of this. If you think about any thinker, starting from Aristotle or anybody, they were all under the influence of this. And you keep reading about this in the newspaper. So in this collaboration, plants really mastered, they started making many more things just because they have to interact with the animals who helped them with spread. That was a watershed moment in the history of Earth. The second watershed moment came much later. That's somewhat around 10,000 years ago. Here, plants did not target everybody. They target one organism, that's us. We could change the face of the earth. We destroyed or replaced all the forms to pamper few plants, and these turned out to be cereals because they can make plenty of grains with very little inputs and very few competitors. So in this coevolution bargain, there is more grain for us, but dominance in numbers and pampering goes to crops. Plants such as Brassicaceae members, they need to be pampered. If we don't go near them, there is no fruit to harvest. So among these are the champions. Among them, rice is really the champion because it's one of the most abundant macroorganisms on Earth. Now, how did rice evolve to this form? So the wild species of rice, which Professor Singh in the morning he was mentioning, they are semi-aquatic in nature. They may have very few leaves, very few seeds, and seeds fall off. Most of the time, they do only sexual reproduction. Whereas in the cultivated lines, we have the plants which are stronger. They need much less water. We still complain about them, but they can have robust stems, huge panicles, sometimes a foot-long panicle holding a lot of grains which stick in the plant. So this modification is basically domestication of rice. Now, this slide again, Professor Singh has shown. Indica rice has evolved from Oryza nivara and Oryza rubipogan, mostly from Oryza nivara, whereas Japonica and Javanica rice mostly came from rubipogan. So these plants are still found in the edge of lakes, sometimes in a python-like prostrate form. So you can have huge variation in the wild species, and they are prone to hybridize with the cultivated line. So you get a huge variation, and it needs an expert taxonomist to identify the wild species. Now we go back to them and started breeding them to reintroduce key traits to cultivated lines. Now, one aspect of this, why exactly this change happened or how exactly this change has been very fascinating aspect of plant research. And obviously people wanted to look at the genomic changes because this should have the archive of every change that we have introduced in it. And surprisingly, very few genetic changes have been associated with domestication have been identified. There are few, but not that much. Mayer and Purganandan, these are key people working on domestication of crops. They found that genetic changes are not proportional to phenotypic diversity in crops. So just to give an example, so again, Professor Singh has mentioned this, we have around 200 QTLs for rice yield, and we know identity of only 8 genes so far. So it's like, we don't know what else is there. So we thought maybe there is a role for small RNA and epigenetics in rice domestication. So to study this, the one slide is missed here. We picked wild species and cultivated species and land races from throughout the country. And we did a very painful phenotyping, and we took 12 lines, wild species, cultivated species, and land races for whole genome analysis that included small RNAs, long RNAs, histone modifications, both active and inactive marks, as well as DNA methylation. And the one small aspect of this work I am presenting today, that's basically from Shweta Chenna, a really fantastic PhD student. So in any plant, if you extract small RNAs and do a profiling to see what size of small RNAs can be found and at what depth, this is the profile you see. You get lot of 21 and lot of 24. These two peaks are prominent. Now we tend to think about peak means something else, but here you have to concentrate on small RNA peaks. Surprisingly, in wild species, Oryza nivara, you get a bigger peak of 22, which has not been observed in any other plant so far. And please remember, most of the plants we are working are either plants like Arabidopsis or crops. So this was very surprising, because if you look at here, Oryza nivara has very high abundance of 22s, Rufipogan to some extent, nothing in the cultivated lines or land races. So we thought, what is the source of this 22 nucleated small RNAs? So we mapped back these small RNAs to the rice genome. It has 12 chromosomes, and these are differentially expressing loci in log scale. So you can see this one region accounted for almost all the change we saw, unusual abundance we saw in wild species of rice. And this region fell back between certain markers, and we found a new microRNA. This microRNA is extremely abundant in wild species, somewhere very low in land races, nothing in cultivated species. So this is a new microRNA we found with many other assays that I don't want to go to the details. Now, when we looked into the abundance of this microRNA across tissues, we got a very interesting correlation. Wild species very high, especially nivara. Land races intermediate, cultivated species hardly any expression of this microRNA. And you all saw Professor Singh's slide. Wild species, they need a support to stay straight. They are prostrate, hardly making any seeds. And high yielders, of course, short, but they can make a lot of strong stems and seeds. So we thought, what is the target of this microRNA? It's a new microRNA, not identified previously, so we wanted to find wow. So we did many different analysis and found out that this targets lacases. So lacases are required to convert these monomers of alcohol into polymers of lignin, and lignin provides mechanical strength, especially when it is binding to certain tissues. So this all starts with phenylpropanate pathway. You all are familiar with this pathway. It starts with phenylalanine. Humans make dopamine for our addiction from phenylalanine, whereas plants make many fantastic molecules such as lignins, anthocyanin, flavonols, and suberins, and so on. So lacases are the targets. So then we wanted to know what kind of lacases are targets. Rice has 24 lacases. Most of them, almost all of them, were not characterized previously. What we found is that many lacases are targeted by single microRNA. Their mRNAs get cut in position 10 and 11, as we can see with the degradome analysis. And though the lacases were falling into different clades, they were all targeted by microRNA whose sequence is identical, was because the target region in RNA was similar. This is the target region. It codes for a peptide called enal, and this peptide is not found anywhere else other than among lacases. Now, the microRNA we found is not 21 but 22. Most of the microRNAs present in plants are 21. And usually, if the microRNAs are 21, they cut the RNA into 2, the pathway stops. But if the microRNA is 22, this bit becomes a substrate for RNA-dependent RNA polymerase, a plant-specific enzyme, to induce what we call as a robust secondary silencing. So what we found is that the lacases loci in wild species like Novara made such cascade regions from different lacases. Basically, to completely shut down the expression of multiple lacases, they need not be directly targeted by microRNA, but still ended up in reducing the abundance of those mRNAs. Now, I'll skip this. So what we find is that basically in wild species, we have very high abundance of microRNA, and correspondingly, what do you expect with the abundance of mRNA? mRNA is chopped, so it should be low. That's what we find here. You can see in wild species, abundance of this specific lacase is very low. Land races have intermediate levels, whereas cultivated lines, this is Pusa basmati 1, very high abundance of this mRNA. Correspondingly, when there is less of lacase, you have less lignin in tissues, more lignin accumulation in cultivated species. In stem sections, less lignin in wild species, more in cultivated species. We also ended up purifying these lacases and with the concanaline beads, these are glycoproteins. And again, we found that there are more lacases in cultivated species compared to wild species. So basically, this is the correlation we find. During domestication, we have very high abundance of microRNA which reduced during domestication. Correspondingly, lignification has increased in specific tissues and there is increase in yield. So this is what happened in wild species. MicroRNA is very high, lacase are cut, hardly any production of lacase, so you have low lignin, plants are prostrate, they have closed panicles, and there is sheath shattering, which is common for almost all plants. But during domestication, in the promoter of microRNA, there is a small change now. MicroRNA doesn't express, lacase expresses a lot, and because lacase expresses a lot, there is more lignification. Now it can support panicles, grain filling, and shatterproofing. So all these change has taken place. So now we have to prove this. How do we prove this? So we took this high abundant microRNA, put into the cultivated lines to see whether this microRNA now alters the phenotype backwards towards wild species. So we can do indica rice transformation in multiple rice lines. Not many people can do it, so we are very proud of that. So we can make transgenic plants and we can prove them with the junction fragment southern analysis. When we generated plants which can express a lot of microRNA, the plants were sick, they are not stable, mechanically strong, they made total of 0 seeds. Whereas if you now express, so this is sort of like the wild species. If you now express one of the lacase in a microRNA mutant site mutated so that it cannot be regulated by the microRNA anymore, the plants are robust, they also make more yield. So now we thought, how come people must missed such a key finding? So we went back to those 200 QTLs that I mentioned previously and started seeing, so these QTLs we extracted from various places. There is at least 1-2 QTLs from GQK. So then we try to see the unknown QTLs, which nobody has mapped to specific genes. Do they have microRNA and the lacases? What we find is that around 20 are already known. 26 of the remaining unknown QTLs mapped to microRNA and lacases. So as you can see here, some of those are really having only lacases. So this is the yield-related QTLs known, and this is what we added. So we got very good comments, good coverage, and our paper was in Plant Cell for 2.5 months as the most rated. Now, it's not just one microRNA is changing, so our lab is involved in many similar aspects of domestication research. We find that some of the argonauts, which I mentioned, are present only in monochores but not in dichores. We looked at 2 of them recently, and AGO17, it improves yield if you overexpress them. And this, again, expression of this gene is linked to domestication. AGO18, which is a very special, it makes a unique clade of argonauts. It is required for inducing male sterility. So if you knock down, there will be male sterility. If you have more expression, then there will be more fertility. So when in an institute like ours, where most of the people are working with animals, obviously people will ask, how does that work in animals? Because we and most other animals were also domesticated. It seems like there is a smaller RNA connection there as well. Myostatin mutant enables microRNA targeting. It's responsible for double muscle phenotype, as in all these animals. And this has been identified in eels, and it has been patented. Now one can easily alter the expression of myostatin. And if you see now a bodybuilder, you don't have to be really surprised whether they really put the effort or just reduced myostatin levels. Now, the last few minutes, I want to talk about another aspect of domestication. Do I have a couple of minutes? Or I should stop?
Hello, I think you can take a few more minutes.
Okay, I take not more than 4 minutes.
Yeah,
so the flowers, yeah, thank you. So flowers and fruits, and there is also a promise of food. Not just women, but men are also fond of flowers, because if you see flowers, you know there will be a fruit to collect after a few months. So this has been a major bribe by plants for us and other animals, such as in birds. So we all like, if you keep 20 fruits and we will pick the most colorful one. You just have to see the apple place where apples are kept, you pick the most colorful one. So for plants, this helps with attracting pollinators and for attracting animals for dispersal. So we have smaller RNA data sets from around 100 plants now, and we were simply comparing how small RNAs look, specific microRNAs look in different clades of plants. And we found that one microRNA which should not be expressing is suddenly very highly expressed in grapes. So we took all these grape varieties, and they have either very high levels of anthocyanin or flavonol. So we made small RNA, RNA-seq, metabolite, and proteome analysis in these, and found that you need specific microRNA to regulate the coloration in fruits. So the targets are MIB transcription factors. So these are enriched in plants which we have selected during domestication for more color. There are hundreds of copies. And when they express, they make colorful fruits. But if microRNAs regulate what is called as the repressors, which are another class of MIBs, which are sort of result of our many times appears to be our domestication associated genes. If they are cut by microRNA, of course you get color. But if MIB repressor allowed to express, they induce the flavonol branch of phenylpropanoid pathway to make flavonols. So the molecular signatures are true for grapes, and some signatures are true for other fruit-bearing, colorful fruit-bearing plants. So basically, all those plants which have colorful fruits, you have the high abundance of specific microRNA, and compared to all those fruits which don't have. So basically, now we can actually induce the color very easily by introducing the right microRNA or the target. So we have sort of a handle for making the specific metabolites, at least in tobacco. We can't transform grape yet, but we are trying to do the editing. So I'd like to thank my lab. They want to call themselves as Epipepis. Shweta is the one who works on rice. Varsha works on grape. Many others have interesting projects. My source of inspiration are David Balcom, who is the person who found out small RNAs, and Professor Velthambi, my PhD supervisor. Very good and fantastic collaborators, people who shared materials, our own sequencing facility, colleagues in NCBS. I was a Ramanujam fellow, so support came from DST and grants from DBT. And thank you for listening to me. I will be happy to take any questions.
So I have one question that is related to the size of microRNA. The dicer, why it cuts the DNA exactly between 21 and 24? Whether it's because of an enzyme or because that length is going to stabilize after complementation, complementary binding target.
So it's a very interesting question. So the dicer recognizes the length of specific, but it is, it can take mistakes of 1 or 2 base pair. But the real stabilization also comes when it binds to organoid protein. So both are involved. Cut is done by dicer. It has very precise cutting ability, either to cut 21 or 24. But the organoid, so they are made hundreds, but only the ones which are associated with the organoids, we find them more abundant.
Okay. So that means the enzyme is evolved first. Then the length is later it is stabilized. Do you think that way?
Many people have looked into why it should be, for example, 21, why 22, why 24. So there is a researcher in Japan who found that 21 gives the right ability, very high specificity, but at the same time, it will not be, see, if it is long, there will be, switches will be very few. So these are like switches, right? Small RNA.
Yeah, yeah.
But if you make smaller, there will be too many non-specific targeting, which no system can afford to have. So 21 and 24 appears to be the right size in
right
these
size.
genomes with around 45, 50,000 gene and huge non-coding region which are under the control of smaller. So even the predictions, physicists' predictions also showed this is the right range. Now, among them, one class makes DNA methylation. Why? We don't know. That's something we try to study in the lab. But 21 is very specific. It goes to the right organoid, and that organoid 1, cannot pick up 24. And its ability to go and cut the RNA into 2. So that's how probably that pathway evolved.
One or two questions from the audience, real quick.
Am I audible,
We are
sir?
running shortage of time.
Am I audible?
Yeah, it's audible.
Yeah, Dr. Shivaprasad, thank you for a very inspiring talk. This question is related to evolutionary domain. What do you think, whether mRNA shaped the target genes or target genes first evolved and shaped mRNA?
Thank you. I think, so they evolved, we may sometimes make jokes, just like every successful man, there is somebody, or women, there is somebody. Just like every successful transcription factor, there is usually a microRNA. Okay, so every most abundant transcription factors, they are under the control of microRNA because they don't want the transcription factor to express in different tissue. If you think about it, this gives that ability for the plant to have different layers of tissues making different things. And that's absent, let's say, in mosses. You have less abundant transcription factors, only sometimes like MADS and MIPS are present, but not all the classes. So that regulation, fine-tuning, is brought about by microRNA. So if you remove any microRNA which targets transcription factor, most of the time these are embryolethal, all you get a plant which looks completely, different from the wild type. So I would say they went hand in hand. Maybe this is sort of the people think this is how plants evolve to different forms. And there are people who hypothesize that even different clades of plants, families, they have signatures of specific transcription factors and microRNAs.
So any more? Otherwise, one or two questions from the chat box. Well, no, they are all appreciated. No. They are all appreciated. They are not asked. We thank the speaker for giving a very, exciting talk. It was very, good information. Now I request the next speaker, Dr. Amita.
Sir, he needs to unshare his screen, sir.
What?
First, Dr. Shivaprasad needs to unshare his screen. It will be difficult to share. It's okay. Yeah, very good morning to all of you. I am Dr. Amita Mitra. Am I audible?
Yes, you are audible to me.
Yeah, my first slide has some problem in which the title is written. So I have straight away moved it to the outline of my presentation. Before commencing my presentation, first I would like to do the Thanksgiving because in the last minute there is no time for that generally. By virtue of being posted at National Institute of Plant Biotechnology and working with Dr. T. Mahapatra and Dr. N.K. Singh, and by my training as geneticist, I have largely worked on DNA genotyping, especially the SNP genotyping. So I'll be talking about that. And I also here place on record my sincere thanks to DBT and NAIP for funding our research. And also Dr. Kishore Gaikwad for referring me to Dr. Fakhruddin and giving me an opportunity to speak before the August gathering. And thank you so much. Now I have planned my talk in few subtitles. One is in simple words try to introduce talk about the various genotyping systems and how automated capillary electrophoresis can help in medium throughput genotyping. Then various methods of SNP genotyping in brief way. And then what is the status of genotyping in horticultural crops? This is case studies from across the researchers, not from our lab. And then about what are the achievements of our laboratory in terms of SNP genotyping. So commencing with the RFLPs, which is the first molecular marker by Boston group. This is the first generation marker, which is a hybridization based, and the markers really took up very well after discovery of PCR by Kerry Mullis and Williams RAPD, followed by the other RAPD like techniques. And then SSRs, AFLPs, and which more often than not use either restriction enzymes or PCR based or combine them. And we then culminated in the SNPs around 2000. And now for 20 years, single nucleotide polymorphisms, which are otherwise also mostly the basis of all other polymorphisms detected by other marker techniques, still ruling. Of course, along with SSRs, which are important for their own codominant and multiple allelic nature. And even this marker technology has moved into like structural variations, copy number variations, and methylations, but still the SNPs are at the base. So now, so we have mostly we do the mostly we have gel-based techniques for using these markers. I think there is some problem if we use animations, I think. So that's why the slides are giving slight trouble. So this slide is full of different marker systems, genotype shows that. And then it talks about the use of capillary electrophoresis in genotyping different sets of markers, especially the SSRs and AFLPs. Can you see my slides?
No, madam, it's blank.
Yeah, it's empty.
Yeah, even I am
The
having.
slide is there, but it's empty.
Yeah, now you can see.
Now you can see, no, but even then the content is not fully coming. Here I have tried to talk about the using capillary electrophoresis and combining with the fluorescent labels and trying to use for medium throughput of genotyping of SSR markers, and also the same can be applied to AFLP markers. This is just to inform that we can use not only SNP genotyping but also other standard markers like SSRs, AFLP systems for medium to high throughput genotyping. And in our lab, we have used these systems to show can I stop sharing once and sharing once again?
It's on a blank slide.
Yeah. I'll share once again. स्लाइड्स नहीं दिख रहे हैं अमोल, पता नहीं क्यों. Can you see this slide now?
No, madam.
Your
No,
name.
not yet.
Okay. Just a minute. Give me a few minutes. I'll log in from another system. Just give me 3 minutes. Yeah, you open my Google.
Chandrayappa, can you hear me?
Yeah, I can hear.
Okay. So I was off in between. There was no internet. Now it has come back.
I realized that there is a problem with the signal.
Actually, it's raining here.
Yeah. No, yeah. Heavy rains outside.
Yeah.
Now
rains,
you are back, madam.
There are rains and sometimes. Now I'm back. Yeah.
Fine. Go ahead.
I think for the next session, no, we have to be strict with time. Only 10 minutes.
Yeah. It should not be
That
more
is
than
10 minutes each, no? Oral. Yeah. Otherwise, it will affect the next session.
That's right.
Yeah. So we will tell them by 10 minutes we'll finish. Because this has prolonged a little bit.
Almost an hour.
Yeah.
Hello?
Yeah.
Yeah, wait for 2 minutes. She's joining from another.
Yeah. She told we are waiting in between. I and Chandrayappa were making some conversation.
So actually what Dr. Shiv Prasad gave the talk is highly relevant to the horticulture department. I hope someone will take up research projects in that area.
Yes.
Yeah.
The fruits, vegetables, and especially flowers, colors.
Even in fruits, making changes in the color as well as they can also control on sugars, probably making the fruits more likable by increasing the fructose and other things. That will be very interesting area.
Yeah. I was there for his talk.
Excuse
what
me.
I missed was Ravindran's little bit. Yeah.
Am I audible now?
Are you back? Yes. You are audible. Please go ahead.
Yeah, thank you. Yeah, so I think the screen is perfect.
Yeah.
I think you can, now the screen is visible, ma'am?
Yes. You can come
Yes,
to
ma'am.
your slide. Yeah. Okay. Screen and you both are visible.
Okay. Thank you.
Yeah.
Yeah, band kar do. So these are all the different marker systems we were talking about, and this is the we can use the automated capillary electrophoresis, and we can do the at least the medium throughput genotyping using SSR markers, and this DNA fragment analyzers can be used for SSR multiplexing, AFLP, single strand conformational polymorphisms, back DNA, that is bacterial artificial chromosome, fingerprinting, and other than the regular Sanger sequencing applications. So only thing we need to take care of here is that we are using multicolored fluorescently labeled probes with each of the SSRs, and we combine them in right proportions so that the wavelength will be different for different ones, and there's obviously energy. So accordingly we have to do it. So then we can create projects like this, and using internal size standards and other things, we can use the So we can use it to genotype this. Using such applications, we have identified the mini core set from the complete 7,000 germplasm of rice, and also we have used it to characterize the Nagina 22 mutants in our laboratory. And as we talked about, the single nucleotide polymorphisms are the basis of all polymorphisms, at least 90%. The only thing we need to take care of is that we always talk about in reference to the homologous chromosomes, and so that the positions are defined, and we don't consider the spurious ones, so that in a population at least 1% of the population should be defined by this particular SNP, so that they are meaningful. And the moment we talk about SNPs, since chromosomes are linear molecules and they get inherited, it is the haplotype that makes more sense, because it is a set of closely linked genetic marker. In this small example, we can see that if we talk in terms of seed coat color, and singly if we pick up the SNP, the relationship is little obscure. But the moment we take the haplotype and talk about it, like AC dash, that is an indel or GT, then the relationship is very clear between the phenotype and the genotype. So the power of the technology comes from using a large number of markers. So now moving on to the different SNP genotyping methods. Basically, there are two major approaches. One is the genotyping the known SNPs, and second is the discovering and genotyping, which where both go hand in hand. And all discovery and genotyping are sequencing based approaches, like resequencing the whole genomes, reduced representation like libraries like RAD sequencing, or genotyping by sequencing approaches, or we move on to targeted sequencing approaches. Whether they are, most of them are probe based, some are PCR based also, and these probe based and PCR based approaches are always combined with the NGS technology to get into the SNPs. In case of known SNPs, we sometimes use PCR based approaches, like where either a TACMAN assay or a CASP assay is being used, or we use a microarray based, that is a DNA chip based approach, where Infinium and Affymetrix are the major players in the globe. So these are all the different techniques. I will simply very slightly touch upon them, and then before moving on to their achievements. CASPAS is a BCR based chemistry. This has become very popular mainly because of this reason. This has a master mix which is always the same. Only the locus specific primer, which is a reverse primer, and the allele specific primer, which are responsible for the SNPs, the biallelic SNP we are talking about. They are the ones we keep to change. So we have a allele 1 specific primer and allele 2 specific primer, and they are always have a specific tails, and these tail sequences in the master mix are added to the fluorescent labels, which are always attached to the their complementary molecule is DNA molecule is attached to the quencher. So this makes the genotyping very easy possible. This is basically a uniplex assay wherein the PCR for each SNP is done separately, and then it is combined, but we can do a PCR even for 15-30 SNPs at a time. So that is where the advantage comes. But otherwise, this is not a multiplex assay. This is just a uniplex assay. So in the PCR round 1, the appropriate allele specific primer will bind to it, and in the PCR 2, it will get extended, creating, while doing this extension, we create the complementary sequence for this tail. So in the third PCR round, the quencher, this will get separated because the quencher will come here. So because of that, this is the real genotyping step in this assay. So whenever we have a few markers, but a large number of samples, like we are working in a seed industry, or let us say we are running a diagnostic laboratory, these kinds of markers are very, very useful. Otherwise, even in mapping populations, we can use them, but only the cost effectiveness is compromised. Otherwise, they are very extremely easy markers to, good marker system to use. And then coming to the array based or chip based genotyping, Illumina, if we talk about Illumina, almost the same thing happens with Affymetrix assay also. Both the cases, we do a whole genome amplification. This is isothermal amplification. Then we do a robust N fragmentation and precipitate the DNA, and then make it single stranded and hybridize to the bead array chip, where each array has a in the well only one particular probe is present. So to this, this is the probe, and to which our blue color template is added, and depending upon the probe and template combination, they are there is a single base extension is done. If you go back to here, you will see this is the template where the SNP is present. And then when the single base extension is done in the probe, there is an extension happening. This extension is done always with the labeled probes, which gives us the genotyping. So since in a single, in a very few nanometers, we have so many probes present, we enhance the signal of the cell by amplify the signal of the cell using the antigen antibody approach, and then we protect the port, and then we can visualize them or laser scan them so that the entire data can come up. So we have to run a very small program for this. So the Axiom workflow also works in the same. The only difference is that instead of DDNTPs, we have labeled like oligonucleotides here. So these oligonucleotides are going to be, instead of single base extension, there is a ligation step happening here. But the data handling is completely the same for here. Dr. N.K. Singh showed you in the morning here a very clear clusters. So these clusters can be, if they are slightly beyond they can be manually curated also. There are provisions available if we with proper gen-trained scores. And this is one thing is that in across crop species and animal species, such DNA chips are readily available. But can we design this chip? Yes, if depending upon our need and our crop, and we can custom design this chip. In the morning, Dr. N.K. Singh sir also talked about like a 62K SNP chip, 82K SNP chip, 50K SNP chip. This is all custom designed. That's why they all have different sizes. But there is a range, like up to 5,000 SNPs, you will have a certain price range, and up to 50K, and up to 1 lakh, and up to million. Like this, there are slabs are available. According to our budget and our need, we can do this. Whatever data is available in the public domain, are using the resequencing data which is available, or resequencing the founder species, we can generate such data and use the robust we are getting for designing such assays. All we would need is the some nearly 60 to 100 base pair sequences flanking the particular SNP. So then we can, this is the about the DNA chip. Moving on to other type of markers, that is because in DNA chip we have selected the markers, but instead of we going and selecting the markers, we can simply use the restrictions enzyme based approach, where we need not do anything. We don't need any prior information like a resequencing information or EST pools or anything like that. We can simply use a restriction digestion. Let us say we have two samples, S1 and S2, and then they will certainly, if they are very have a different pedigree, different lineage, they are supposed to have large number of SNPs. So we can use a restriction enzymes to digest these samples separately, and the restriction site specific linkers can be added here, and they can be sheared and purified and then hybridized. So like this, we can develop the restriction site associated markers, and once we have a large number of such markers, we can also create a specific micro array for that by placing the PCR amplified rat tag clones on a chip, and then print them and then use them for hybridizing what the entire mapping population or the samples. So this technology was first tested in Drosophila, and recombination breakpoint was used by using two different restriction enzymes, EcoR1 as well as ZO1, and the identical results were obtained. But whatever, like this, since this involves a lot of work, people immediately next year itself, this paper came in 2007. In 2008 itself, the science progressed to using GRAD but by sequencing. So in case of sequencing, like we have the genomic DNA, we digest with a restriction enzyme, add a P1 adapter, which is the adapters always have sites for the annealing to the restriction site, as well as they have sites for the Illumina sequencing, those primer complementary sites. And after ligating them, we always ligate then to a barcode and then to a second adapter. Second adapter is designed in such a way that it is a divergent adapter. It will function only if the first adapter is already ligated to it. So because of this, we get a complete like rad tags where both the adapters have ligated, and now when these two adapter ligated pools go in for NGS sequencing, we get the complete sequence information across samples. So the improvement over this technology is the GBS. In case of GBS, we can use different enzymes, though APEK1 is more popular compared to PST1 or ECO enzymes, mainly because it has a CWG recognition site. W is the adenine thymine 1, so this is a very popular and most used recognition restriction enzyme for GBS technology. But it is we can always try GBS with other enzymes also. And the adapters are kept simple, not like divergent adapters. They are straightforward adapters. Very simple protocol of having the GBS libraries. We have to have DNA samples in hardly 10 nanogram concentration, and then we have to barcode them, then ligate them to adapter pairs, and then cut them with the restriction enzymes. And then once the adapters are now ligated to the restriction enzyme, we need to inactivate and then pool the samples and do the size exclusion. And once the right sizes are obtained, then we can go for simply sequencing them. So this is the barcode sequences available. These are generally 4 to 8 nucleotide long, which distinguish one sample from the other sample. And the adapters, as I told earlier also, they always have the sequences for the PCR to allow PCR, Illumina PCR to happen. So this is about GBS, which is a very popular technique. And then we can have targeted sequencing also. Again, for targeted sequencing, AmpliSeq and Fluidum based techniques and transcriptome based we have ion torrent based technologies are available. This like Fluidum is specifically meant for genotyping, but these kinds of ion torrent can do multiple things. Apart from targeted sequencing, it can do transcriptomics also, bacterial genome sequencing. It is useful for doing metagenomics. It is useful for multiple things. Basically, all the such targeted sequencing techniques are either PCR based. We have to have the genes of interest are amplified, PCR amplified from across genotypes. They are ligated to barcoded adapters, the PCR products, and then pooled, size selected, and sequenced. In case of there are lots of probe-based approaches also, like AmpliSeq, where we first design the probe complementary to the genes of interest, and then we either RNA or DNA specific probes can be designed, and then we shear the biotin-related DNA, bind it to the probes, then we enrich our samples for the target DNA and barcode them and sequence them. So this is with respect to the targeted sequencing technologies available. So now moving on to the horticultural crops, I chose as such we don't work that much on horticultural crops. Only we do support them in our lab, but still I chose that because honoring the organizers who are University of Horticultural Sciences, and also the kind of tremendous improvement made in horticultural crops in application of these technologies. Because most of the marker technologies either evolved with agriculture species like Arabidopsis, maize or rice, and are sometimes microbes, or sometimes with respect to animal species. But generally, horticultural species have picked up later. But I would like to here highlight the different achievements that have made across the globe by across crops and horticultural species. For example, in cabbage, we have a 674K assay available, which is developed based on NGS of 10 parents. And also we have different like 11.48K, 130K, and 32K assays. Across crops, we have assays available, and they have used either NGS of accessions or GBS. And more importantly, they have applied this for diversity analysis, mapping of black rot resistance in cabbage, and discerning the linkage disequilibrium and genetic architecture of the crops. And in case of carrot, the purple pigmentation loci have been mapped using the Illumina assay and the CASP assay, and the gene is being used for a marker assisted selection. In case of eggplant, initially it was done for fingerprinting. Later, now we have assays in which either the complete eggplant genetic diversity has been worked out, which I will talk about in a few minutes. Then this is from a peach consortium, wherein Spain, Italy, and USA labs have combined, where more than 1 million SNPs were identified across 56 peach accessions by whole genome resequencing, from which 8K SNP array was defined, and of which 7K markers were polymorphic, which were by testing over 709 accessions. And this is a very clean SNP set now being used extensively for peach improvement. And then in case of using rose, because rose has a large genetic diversity. There are tetraploid, diploid, octaploid, pentaploid, a large number of ploidy levels are available. But despite rose is a very important commercial crop, we don't have neither have a rose sequence resources, nor have very good genetic maps available in rose, hampering the crop improvement in rose improvement. So this paper came in 2018. What they have tried to do is that they did a GBS of rose, and then they used the strawberry sequence as a reference, and used some using 3 different mapping populations of diploid roses. They could develop a very good genetic linkage map for rose, which can serve as an excellent resource for mapping a large number of important genes, the volatiles and aromatics and other things that rose gives us. So this is an excellent resource for rose. And then in grape wine, one of the important problem is nematode. So recently this paper has come where the SNP markers have been identified, which are tightly linked to root knot nematode. For this, they have used both GBS approach of a F1 population, and then this F1, but they had certain difficulties because they could not map by simple direct approach. So they used association mapping. They combined then independently sequencing the parentals of these F1, and by combining multiple methods, and then taking some 200 SNPs, and then using them for sequenome mastery validation, which is nothing but a MALDI-TOF for SNP genotyping, MALDI-TOF assay. So they finally landed on the a very small region, which is giving nematode resistance, and this is again being now used in marker assisted selection. And in case of apple, there is a consortium called Apple International Breeders Consortium, which maintains a large number of apple genotypes, and which includes the elite varieties and the land races, and also some populations for which the parents as well as the entire population is maintained by them. So they used a Thermo Fisher based open array platform to genotype 125 SNPs. SNPs. All these SNPs are functional SNPs in the sense that they are linked to either a quality trait or pest and disease resistance. In case of quality, firmness of the fruit or the sweetness of the fruit, those quality and the color of the fruit are included. So all these SNPs, nearly 128 SNPs were used, and an assay was developed, and then this was used in this elite breeding material, and finally they identified the 33 most important markers for use in breeding, and these 33 important markers can be used in any way, like singly or in combination using any assay, and now this 33 markers are completely used by this group and also the entire Apple International Breeding Consortium for marker assisted breeding. And in case of cherry also, there was initially a 6K array available. Now they have this using a resequencing data and EST pool data and GBS data available. This was improved to 9K 6 plus 9, that is 15K SNP array. Now this is because cherry is a very complex genome. Like it has a sweet cherry, one is sour cherry, one is a diploid and another is a tetraploid. They have subgenomes available. So it's a very complex. Most of the fruit crops are horticultural crops by way of their genetic nature. They are not many times they are not seed propagated. They are clonally propagated, and they have heterozygous balance. And because of that, they have a lot of polyploidy and complex genomic arrangements in them. So it is comparatively more difficult also to work with these crops. So such efforts like representing the sweet genome, the sweet cherry genome and sour cherry genome and the other related wild relatives, they have very developed this assay and also very clearly demarcated the fruit firmness QTL. Similarly, in case of eggplant, I was talking about the genome-wide perfect SNPs have been used to do the genetic architecture of the species, and which gave an important finding that mostly it is oval fruits and round fruits populations are the main fruit population. Like the genetic architecture is largely determined by fruit shape in case of eggplant, and in case of oval and round fruits there is a very huge genetic erosion. This is my last slide with respect to horticultural crops. Here the very interesting thing, as a few minutes ago I told that mostly the developments come from other species, the landmark developments, maybe after Inothera lamarckiana or something like that, where classical genetics is helped by horticultural species. We have here the cucumber genotyping platform. In cucumber, a new target SNP-seq assay has been developed, and this is a very, very good assay because it is a very comparatively a cheaper one. One thing is that it requires 3 days like any other SNP genotyping assay. The most important is that for one sample, for 100 to 2000 SNP, it is only $7. So this is the, and then the robust SNPs, the call rate is very high, nearly 100% genotyping call rate, and the SNP quality and depth. What happens in most of the SNP genotyping platforms is that they are very good. You don't need any prior information to do a NGS based approach. Of course, this is target sequencing. We would need a prior information. But what happens in after NGS data comes to our hand is that we don't get uniform data points across the samples. This is one of the main problems in any sequencing based approach as compared to DNA microarray or chip based approaches. So in this case, what happens is that this more than 100x coverage is obtained in this technology, and because of that you get 100% call rate, and there is no missing data point in this. So this is the strength of this assay. So this was developed for cucumber, and this was developed and tested in cucumber, and this has enabled them to do genetic analysis. And in coming to our institute, we have some of the points have been already highlighted by Dr. N.K. Singh in his work. So here I would like to talk about the Illumina platform first came to our institute, where we used it for maize 55K assay, and then we used it for wheat 9K assay, genotyping. This is all in 2012-2013 period. And then we, before around 2010, we developed a rice 384 SNP assay, specifically from stress responsive genes. From morning we can listen to a lot of stress related talks, and this 5K, following the success of this 384 SNP assay, we developed a 5K SNP assay, which is again exclusively based on abiotic stress responsive genes and the genic positions, and this has been successfully used for mapping salinity tolerance and heat tolerance in rice. And then this was the first, before the 62K SNP assay Dr. N.K. Singh sir talked about. The initial one which we used was the 1536, that is 1.5K assay, which was a ready-made Golden Gate assay available. And then later we used the Pigeon P 60K assay, or the let us say 62K assay, and the high density intraspecific SNP linkage map could be done. And this high density intraspecific linkage map was also dependent on a lot of GBS data. And then Dr. N.K. Singh has already talked about these three assays, so I will skip them. So this is the 384K assay I was talking about, which is where 9 SNPs could discriminate accessions from upland and lowland rice, and the following which this 5K SNP was used for it has more than 200 citations, and this has been used for mapping the reproductive stage QTLs for salinity tolerance. And this it also allowed us to map the QTLs for heat tolerance and high resolution. Like by a high resolution, what I mean is that less than 400 kilobase pair, the QTLs could be narrowed down using approximately a population size of 300 and RILs, and also like by very precision phenotyping under heat stress facility available at Karnal. And these regions hardly have 50 genes, which we are trying to validate now. We have already validated and narrowed down to 4-5 candidate genes, which we are trying to clone and validate. And this 50K SNP assay has been already talked about, so I will skip this. This is my last slide. I would like to here just emphasize upon how moving away from 1536 SNP assay to 62K SNP assay helped us. Like from 1536 SNP assay, we got 267 SNPs, and which has helped us a lot in mapping, which Dr. N.K. Singh sir told you, like in linkage group 4 and linkage group 5, we saw the QTLs in co-localized. And then if you, this is all the 11 linkage groups of Pigeon P. Now I have here shown you only a few linkage groups of Pigeon P, and we can have a look at the resolution, how well the maps are resolved when we talk about, when we move away from 267 SNPs obtained from 1536 assay and 2000 SNPs polymorphic SNPs obtained from 62K assay. So this is the power of the technology. So using this and then combining them with the transcriptomes and the proteomes and other approaches, we can land on the candidate genes and or even the QTLs, and in a very narrow interval, they can be directly used for marker assisted selection. In that way, this dwarf stature QTLs have been already used in marker assisted selection in case of Pigeon P. So I have tried to highlight the various options available and the success stories available in case of horticultural crops, along with some of the work done at our lab. I thank the organizers for the opportunity, and I also thank Dr. Mahapatra sir, Dr. N.K. Singh sir, and the NAP, DBT and ICR and our institute, as well as all the students, RAs and SRFs working in our group. Thank you once again, ma'am. I'm done.
Thank you, Dr. Amita.
Yeah, can I have one question?
Yes, sir.
Madam, how much we can rely on SNPs? Because highest polymorphism is because of SNPs. That itself, it tells, genome is not that stable. So if you develop a variety, how much we can rely on the SNP based breeding?
SNP based, sir, as long as the SNPs are linked to the QTLs, then generally for breeding, sir, we use SNPs to identify the QTL regions. Once the QTL regions are identified, we generally do a translational work of developing allele specific assays, either using SSR markers or using some other markers. For marker assisted selection, we rarely directly use the SNP assay, sir. We use it only for mapping the traits or tagging the traits. Once that thing is done, and we know the linked markers, or once we know the functional markers, after that we try to go on to some gel based assays which easily breeders can use for marker assisted selection. This translational step we always do, sir.
No, that's fine. Methodology is fine. My question is, SNPs are how strong they are. Do they stable with the generations?
They are stable, sir. They are stable with the generations. They said generally we for this, what we do is that I can understand you feel like 10⁻⁶ is the mutation. One is that is a germline mutation, and mostly, but we sample from somatic tissues. That is where you are coming up to. But sir, if we are whenever we genotype, the samples are never collected from a single plant. They are always collected from a represent. Like let us say we are sampling IR64. We sample from like 5 to 10 plants for IR64. Then we see to that if it is, let us say, sequence based, then it is a complete perfect SNP. Like it has a very high quality. There is no heterozygosity seen, and we use that kind. And for that is, this is for with respect to using the SNP. And then coming to the power of the technology, it is little different, sir, because we are going to have genome-wide scans, and it is like a moving average kind of approach. So we are going to like the data analyzes it in a different way. We don't take like just one SNP into account. We are always going to walk the genome based on the moving average, how the SNPs are moving along the chromosome in the progeny. So that should not be a problem, sir.
Because even a single SNP change can, have an effect on the phenotype.
Yes, sir. And one more. Yeah, and one more point I would like to clarify here, sir. There is genic SNP versus intergenic SNP. When we talk about the genic SNP, they always have extremely high validation rate. And even those papers I have cited, there also, if you see, wherever the genic SNPs are picked up, the validation rate will be almost 100%, at least more than 80%. But when we go to intergenic SNPs, it will be hardly 33%. So these problems do exist. So that is why we always prefer genic SNPs.
Okay. Thank you, madam.
Thank you, sir.
So, few more questions from the audience. In the chat box, there are two.
Okay.
I
I think we have finished in my talks.
Yeah.
Shall we move to the oral presentations now?
Yes.
This is for only 10 minutes. Please stick to the time. We are already 1 hour behind. Now we'll start with oral presentation. First speaker is Nimisha Sharma from IRI. I request Nimisha Sharma to start her talk.
Thank you, ma'am. Am I audible, ma'am?
10 minutes. Yes, you are. Please go ahead.
Okay, thank you, ma'am. Thank you. A very good afternoon to all of you. So, myself, Dr. Nimisha Sharma, scientist at Division of Fruits and Horticulture Technology, IRI, and I am going to talk on the topic explore the differential flowering behavior in mango and development of genic SSR marker database using transcriptome analysis. So we all know that mango tree is widely known for its strong tendency for alternate bearing, which severely affects the fruit yield from year to year. Generally, South Indian cultivars are regular bearer, but North Indian popular cultivars like the Dashari, Langda, Chosa, they are irregular bearer in nature. So earlier hypothesis was given that some flowering site limitation, then nutrition point of view, some hormones are responsible for this irregular bearing phenomena. So in this study, we are using differential gene expression analysis from mango samples, one from regular bearer variety, another from irregular bearer varieties, so that we can understand the molecular mechanism behind this important phenomena. So this diagram represents that many factors are responsible for this irregular bearer nature. So in terms of genetic factor, cultivar genetic makeup, in addition to that, some genes are responsible for this phenomena. And when we see the other factors, then in terms of physiological factors, source-sink relationship is very much important. And moreover, environmental condition also influence this phenomena, like temperature, photoperiod, vernalization, and some of the enzymes are also responsible for this important phenomena. So the present talk I am delivering with the following objectives. First one is the generation and validation of genic SSR marker database using transcriptome analysis, then differential gene expression studies in regular and irregular mango varieties. So this is the diagrammatic representation. We are taking one regular variety is Neelam, another irregular variety is Dashari, and we all know that Amrapali is regular bearer nature. So Dashari and Neelam generates the Amrapali variety. So it is very important to study the parents of this variety, how this regularity feature comes in Amrapali varieties. So simply I am not going in detail because I am going to highlight here SSR marker database. So first we have generated the transcript set, and after that we can go for the MISA software for microsatellite identification, and then go for batch primer 3 for generation of the primers. So simply here I am presenting that Dashari and Neelam, we have generated so many reads, and these reads further generated the different transcript, like 27,528 in case of Neelam and 20,771 transcripts from Dashari. And we combine them so that redundancy can be discarded. And after that, we have identified SSR from 1363 context, di, tri, tetra, penta, and hexa we have identified, and mononucleotide we have excluded. And after that, 6 to 7 primer pairs we have generated through batch primer 3, and out of that 100 primers we have validated. 38 were of expected size, 1 is smaller than expected size, 6 larger than expected size, 32 produces the multiple band in case of mango varieties, and however 23 didn't amplify. So this is the all summary about the SSR marker generation from this Neelam and Dashari transcript. And this diagram show the wet lab validation for the in silico design primer, and we have tested the polymorphism in 8 mango varieties further. Now coming to the second objective, differential gene expression studies in regular and irregular mango varieties, we have utilized this transcript data for generating this important bearing phenomena, and we have identified many genes and annotated different pathway, and further these genes were validated using RT-qPCR. So this is the summary of differential analysis statistics of these varieties. Total 42,397 genes were identified, and out of that 12519 genes were differentially expressed at log 2 fold value, and even it follow the criteria significance, that is value less than 0.05. And out of that 6459 genes were upregulated and 6104 genes were downregulated. I am here would like to mention that in case of Neelam and Dashari, some common set of genes are also found and some unigenes are also found. So it is important to see the unigenes which affects the regularity in case of Neelam, and genes which are unique to Dashari which may be responsible for irregular bearing nature in case of Dashari. So we have annotated a different pathway, and according to the previous hypothesis, we have identified several pathways: cellular, flowering, hormone, meristem, metabolic, oxidative stress, oxidoreductase, torsion, alternate bearing. Maximum number of genes found under the case of metabolic process and minimum in the case of flowering, and 26 alternate bearing genes were identified. Further, we have identified some important pathways like alternate bearing, hormonal, metabolic, because these pathways mainly responsible for this alternate bearing phenomena. I am not going to detail for every pathway due to slide limitation. In case of alternate bearing, here we can see that in Neelam, some of the gene like SAD3 and SAD1 overexpressed. And when we see the hormonal pathway, GA hormone basically upregulated in case of Dashari, that is irregular bearing nature, and previous hypothesis also have shown up. In case of metabolic, some of the genes mainly well expressed in case of Dashari, and some genes, very few genes in case of Neelam. And further, these important pathway like alternate bearing, hormone, and then metabolic, we have utilized this pathway for primer generation. So we have generated large set of primers, and 15 primers we have validated for the confirmation of in silico analysis. So this is the validation of differentially expressed gene by RT-qPCR. We have used beta-actin gene as a reference, and double delta CT analysis we have used to calculate the fold chain value and threshold value. And then we have computational results verified by RT-qPCR. In case of in silico analysis, the summarized that out of 59 genes were upregulated in case of Dashari, whereas only 6 genes were more expressed in case of Neelam. When we did the wet lab validation, then we have found that all of the genes have shown similar behavior in case of wet lab, except one gene, that is LOC 103420644, which was less expressed in case of Dashari, but in silico analysis has proposed that it has to be shown overexpressed in case of this variety. And all hormone and metabolic genes reveal the consistent results. So now I am going to summarize the result. In case of SSR data generation by using these varieties, this marker can be used for diversity analysis and genetic mapping of useful horticulture traits in mango, and we are utilizing this marker at present. And then as per differential gene expression analysis, we have identified a list of differentially expressed genes related to alternate bearing phenomena. 26 genes we have identified, and then in comparison to leaves of bearing trees, the non-bearing ones had elevated expression of genes responsible for the carbohydrate metabolism. In mango, the autonomous GA pathway appeared to be involved in both biennial bearing and flowering control. Earlier, previous researcher also has reported this. And then genes belonging to oxidoreductase pathway and starvation were mainly upregulated in regular variety Neelam. Despite the rapid progress, if we talk about future thrusts, then in flowering transcriptome and genetic studies, a number of mechanisms are still not clear because it is affected by external as well as internal factors. So it needs more clarity so we can combine molecular tool with the possible horticulture intervention. So I would like to acknowledge ICR IRI and PTC funded project and Young Scientist DST sub project for financial assistance, then Director ICR, Director NIPB, Dr. NK Singh, and then co-authors, our head of the department, DDG Horticulture, then Dr. Manish Srivastav, Dr. Jayaprakash, and Dr. Meenakshi Malik, Dr. Neha Sharma, Dr. Rajay Kumar Mahato, and of course, Director NIPB, Dr. NK Singh, sir. Thank you very much. And in last, I would also like to acknowledge organizing committee for this international e-conference, because during this pandemic, it is a wonderful chance that we are going to present our work. Thank you very much.
Thank you, madam.
Okay.
And only one question will be asked. So in your studies, you have concentrated more on hormonal genes, metabolic genes, and other things. But the alternate bearing is directly related to the storage in the bark, how much carbon will be stored. That also determines one year if it gives higher yield, there is exhaustion of nutrients in the bark, whereas second year is going to be less yielding. So what do you say about this?
Sir, as per nutritional control.
Yeah.
Sir, actually we are going to study in Bombay Green and Totapuri this nutritional theory. For Neelam and Dashari, already data we are used for the transcriptome analysis, but the detailed study of this alternate bearing phenomena in the Bombay Green and Totapuri we are going to conduct, because nutritional phenomena is very much related with the alternate bearing phenomena.
Exactly.
So some of the genes are definitely responsible for this phenomena. But in the present study, we have only focused on alternate bearing pathway and hormone and metabolic, carbohydrate metabolism, sir, mainly.
Okay, thank you.
Thank you, sir. Thank you.
Madam, go ahead.
Okay, thank you, Nimisha.
Thank you, madam. Thank you.
Okay, our next speaker is Dr. Vishnu Dev Prasad from Bihar Agricultural University, Sabur. Please, you have 10 minutes. Please stick to the time also. Please go ahead.
There's a problem with audience audio.
Yes. Can you hear me?
Yeah, I can hear you, but not from the speaker.
Yeah, from the speaker there is an audio problem. Yeah, now it's okay.
Okay. Yeah. Good afternoon to all. I would like to thank organizers for giving me an opportunity to speak, present my research work here. I have little cold, so my voice is not very clear, but I will try to deliver my finding to the participants. The topic of my presentation is next generation sequencing for identification of brassinosteroid biosynthetic pathway in the safflower. As you all know that brassinosteroid are a plant steroidal hormone which play an essential role in plant growth and development and transfer tolerant to a wide abiotic and biotic stress. Brassinosteroid biosynthetic pathway is the best understood pathway, and this is the simplified overview of brassinosteroid biosynthetic pathway. And you can see here, top 4 is the rate limiting step. So our thought is, if we make changes in the DOF4, we can able to make the changes in the brassinosteroid level in the plant. So if we increase the brassinosteroid level, then plant will be more resistant towards the abiotic and biotic stress. So previously we have done the overexpression of BR biosynthetic pathway in the brassica, and we have found that in the transgenic line, there is an increase in 30% yield, as well as you can see the yield attributed traits has been increased in the transgenic plants. We have also shown that this brassinosteroid treatment has increased the thermotolerance, as well as it enhanced the drought stress tolerance in the brassica nepus. So keeping these things in mind, we try to identify the brassinosteroid biosynthetic pathway in the safflower. As you all know that safflower is known for its oil quality, and it has the ability to grow in a drought stress condition, but there is no report yet for the presence of or existence of any brassinosteroid biosynthetic pathway in the safflower. So we went for doing the next generation sequencing approach, and we gave the plant with the treatment of brassinosteroid, that is epibrassinosteroid, and we performed the sequencing, next generation sequencing. These are the NGS sequencing data, and we have get approximately 5 GB of the data from control as well as EBR treated sample, and we have got approximately 34,000 transcripts. And this is the workflow how we have done the bioinformatics analysis. We have used the BLAST X for functional notation. Then we have done the pathway analysis. We have done the differential gene expression analysis. We performed the BLAST2 analysis. And we have also identified the various transcription factor as well as transcription factor binding sites. So these are the different transcripts that we have obtained in control as well as EVR treated sample. And most of our transcript fall in between 200 to 500 base pair. Similarly, we have got the very high number of CTS in the range of 200 to 500 base pair. So then we have performed the trait pathway analysis, and we have found various brassinosteroid biosynthetic pathway, and fortunately we have also identified the DWAR4 gene, which is the rate limiting step in the brassinosteroid biosynthetic pathway, and we have identified the full length of DWAR4 gene, and we did the qRT-PCR analysis to confirm this DWAR4 gene and other brassinosteroid biosynthetic pathway. So this was the first report where we have shown this brassinosteroid biosynthetic pathway exists in the safflower. Then we also identified some of the transcription factor binding sites, and you can see here we have identified several transcription factor binding sites which are influenced by the different hormones, and we have also identified some of the transcription factor which is influenced by the brassinosteroid. And these are the genes we performed the performed the analysis, and we have found that several biotic as well as abiotic stress responsive genes has been upregulated and downregulated, and we have also found some of the genes we are upregulated by brassinosteroid treatment. Thank you very much, and I would like to thank DST for the funding. Thank you.
Madam.
Hello.
Hello, madam.
Yeah. Hello.
Hello. He took he took 6 minutes.
Yeah, that's it.
He saved our time.
Yeah, really. Okay. Thank you.
Thank you. Thank you.
Yeah, thank you. The next speaker. We'll go to the next speaker. No?
Yeah.
Next speaker is P. Supriya from ICR, Hyderabad.
Respected Chairman, ma'am, am I audible?
She's from NAM.
Yes,
so you please go ahead.
Yeah.
Supriya.
Yes, ma'am, thank you.
Dr. Supriya.
Respected Chairman, co-chairman of the session, and other distinguished scientists, very good afternoon. I am Dr. Supriya, bioinformatics scientist at ICR NAM, Hyderabad. Today, I would like to present a part of my PhD research work that is on whole genome bisulfite sequencing analysis of sesame. As we know, sesame is an oilseed crop. Whether slides are changing? Hello.
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 Madam, continue.
Actually, my slides are not changing in my system. Is it slides are changing in your?
No, madam, no.
They are not changing. Yeah.
Yes, ma'am, I'm trying, but they're not changing.
Restart it. Restart it.
ನಮ್ದು ಪ್ರಾಬ್ಲಮ್ ಆಯ್ತಲ್ಲ
Click on the next slide and see. Yeah.
Yeah, as we all know that sesame is an oilseed crop and it belongs to family Pedalaceae. As a source of high quality oil, it is valued for its stability, nutritional value, and resistance to rancidity. It also has several nutritional and health benefits, because of which it is considered as queen of oilseeds. However, it is also considered as orphan crop because of limited interest to researchers. As a consequence, it lags behind the other major oilseed crops as concerns genetic improvement. So we all know that to know the structure of genome, whole genome analysis is performed, and to know the expression of genome transcriptome analysis is performed. So it is also important to know the regulation of genome, and one of the mechanisms of regulation, that is methylome, is analyzed in the present study. So now we need to understand what is methylome. It is nothing but entirety of methylated nucleotides in the DNA. So addition of a methyl group converts cytosine into 5-methyl cytosine. Normally, the methylome is highly variable between different species, between organisms of the same species, and between different cell types of the same organism. So these methylated nucleotides can inhibit the transcription, which results in regulation of gene expression. So the motivation for the present study was like recent advances in DNA methylation techniques have enabled to understand correlation between methylation patterns and hybrid vigor in several crop species such as rice, brassica, soybean, etc. However, knowledge about interaction of methylation patterns and gene expression in oilseed crops such as sesame has still remained elusive. So the present study was undertaken with the objectives to generate a genome-wide methylation profile of Shweta variety of sesame crop, and also to develop a comprehensive information system of sesame methylome. Coming to the materials and methods, DNA samples were extracted from leaves of Shweta variety of sesame just before initiation of flowering, and DNA was isolated from the leaves using CTAB method. Then the bisulfite sequencing libraries were amplified, quality tested using bioanalyzer, and then subjected to Illumina HiSec for impaired and more. The poor quality reads and adapters were removed from the raw data using Trimomatic software. Then the high quality reads were mapped to reference genome using Bismarck software, followed by the calculation of DNA methylation levels in different contexts, that is CG, CHG, and CHH contexts. So after adapter removal and removal of low quality sequences, 23 to 25 million clean reads were obtained, and these reads were mapped to reference genome as discussed before using Bismarck software. So further, to calculate the frequency of cytosine conversion, reads were mapped to unmethylated chloroplast genome of sesame, and we obtained a conversion rate of more than 96%, indicating high efficiency of cytosine conversion in our experiment. Then fractional proportion of DNA methylation was calculated for 3 different contexts, and the results showed that 48% of DNA methylation was in CG context, and 26% was in CHG, and 25% of methylation was happening in CHH context. So overall, sesame genome was found to be more enriched with CG methylation, closely followed by CHG methylation. In the figure, we can see different methylation pattern in different chromosomes of the genome. The outermost circle representing the linkage groups towards inside from the outer circle. The 3 circles are showing methylation level in CG, CHG, and CHH context respectively, and the innermost circles are showing the methylation levels in transposons and genic regions. So the red color represents that methylation is in high in these particular regions, in these particular linkage groups. The methylation, CG methylation is higher. Likewise, followed by orange color represents less methylation, and likewise. So results were very evident as expected that the level of methylation was higher in transposons when compared to genes. Since transposons are usually heavily methylated and attenuation in DNA methylation leads to their increased transcription and occasionally transposition. Even though the exact biological role of CG gene body methylation is remained elusive till date, it has been proposed that it might play a role in suppressing promoters activity. Therefore, we analyze the patterns of DNA methylation in 3 different contexts, including 2 kb upstream and 2 kb downstream of the flanking regions of gene body. So results showed that methylation level showed a sharp increase from both the transcription start site and termination site, and it dropped slightly before attaining a stable pattern. The results also showed best in gene bodies, whereas CG methylation is accumulated at higher levels when compared to other 2 methylation context. So to make the best use of analyzed data, we developed an information system that is a SESAM genomic information resource where methylation information is available. And methylation level can be seen here in 3 different contexts, that is CG, CHG and CHH, on different linkage groups along with their genomic coordinates on different linkage groups. So likewise on whole genome also in transposons and CDS also, this different methylation level can be seen, and here we have also provided the methylation profile of SESAM whole genome UG11 that is sequenced by Chinese. So in conclusion, the transposable elements can influence neighboring gene expression, that contribute to the inheritance, and thus represents the most common example of epigenetic inheritance in plants. Consistently, our data also revealed a low expression of genes when compared to transposable elements, suggesting similar roles of transposable elements in SESAM crop. And the data generated and analyzed in the present investigation are made available through SESAM Genomic Information Resource, which is an integrated genomic resource for SESAM crop, and it can be freely accessed at the given URL. So our data will serve as a comprehensive community resource for further understanding of DNA methylation in other oilseeds crops, and in SESAM also it can serve as a reference for other studies. So thank you for the opportunity, and thank you for your kind attention.
Okay, thank you. Thank you, Okay, other
Thank you.
Scientist at Regional Research Station, CISH, West Bengal. You have 10 minutes, please.
Okay, ma'am.
Please do the slideshow, I think.
Good afternoon, everyone. As you know, that India is the largest producer of mango in the world, and mango is the most important fruit crop in India, and it is also known as king of fruits and natural fruit of India. But the productivity of the mango in India is very less compared to the Kenya, where that is 48.8 metric ton per hectare. But in India, it is very less. There are several biotic and abiotic stress that affect the mango production. Out of that, mango malformation is also causing a serious loss to the mango production. After his first report in 1891 in Bihar, it has spread to different parts of the country as well as in different parts of the world, and now it has become a global problem. Mango malformation is mainly of 2 types, that is vegetative malformation and floral malformation. And out of them, floral malformation is causing serious loss because it is directly related to the mango production. More than 120 years of research has been carried out, but the etiology was still confused due to contradictory reports on its etiology, like that it is caused by fungus, virus, mites, and hormones, nutritional aspect. Different research has been carried out, but not a conclusive result has been done how it is happened. So recently, it has been confirmed that it is caused by fugitive species, and different species are responsible in different countries. I have found 2 research gaps related to mango malformation. Till now, no resistance susceptible genes has been found it related to mango malformation. And flowering, since it is a flowering related phenomena, no flowering genes and flowering pathway has been studied in this. So keeping these research gaps in mind, I have took these studies and I have worked on transcriptome and flowering gene and expression analysis in healthy and malformed tissue mango. For this study, I took two objectives. First is to generate transcript data in healthy and malformed tissue. Second is to identify differential express gene, flowering genes and pathways. For this study, I took the mango, highly susceptible cultivar to mango malform, that is Amrapali, and three malform stages and 2 healthy stages were collected. Further, 15 replicates of each stages were collected and RNA was isolated and quality was checked in agrogel. Then further quantification was done by qubit fluorometer. After that, it was submitted. RNA was submitted for sequencing. This I will not go in detail because much has been spoke about in a few days. Now coming to the objective, we have generated this much, near about 5 to 6 GB data per sample we have generated, and plenty of transcript and unigenes and CDS we have predicted. We have done homologous gene sequences among different species, and we have found that citrus sinensis was closely related, followed by clemency and cocoa. We have developed some marker also, mined some markers also in this transcriptome. After that, we have annotated our unigene sequences to different database, and through NR database, we have able to annotate 96 to 97% unigenes. Then we have annotated our unigene sequence through gene ontogene analysis, and we are able to annotate 52% genes. These are the different classes of gene ontology in 2 healthy stages. This I will not go in detail because it is basic things which we for annotations. After that, we have done annotation through COG analysis also, and we are able to annotate through COG analysis 30% genes, and these were mainly classified into 28 categories, and out of that, mostly that green peak and orange peak are showing that most of the unigenes are found in unknown function. This is the 2 healthy stages. Thus, now coming to the most important thing which I have done. We have collected all the flowering genes present in flowering domains, and we have developed the flowering genes database. And we have blasted our samples with that flowering genes database, and we are able to find out more than 710 to 900 flowering genes per sample. Like that, it varies. And after that, what we have done that we want to know that what actual flowering genes are present. So we have removed the repetitions in the flowering genes, and we are able to find that mainly 200 to 204 to 209 genes were present in our 5 samples. After that, we have that which genes are having high frequency for in healthy stage and malformed stage, and we are able to find that TPL, the stopless flowering genes, was present in all the malformed stage, whereas it is less copy number is present in healthy stage. But the beauty is that of this gene is that we have able to find that it is highly upregulated in healthy cases, healthy stages, though it is present in less number in healthy stages. Coming to the second objective, that is differential expression analysis. In gene ontology analysis, we have found drastic changes in some categories of the genes, from birth stage to panicle development stage, and the categories were binding, catalytic activity, cell part, and signal organism process, cellular metabolic process. Then we have found differentially expressed flowering genes, which genes were upregulated and which genes were downregulated. At birth stage, that is HB1 versus MB1, we are able to find that LD, license specific demethylase like 2 genes were highly upregulated, followed by more copy number of topless genes and Burki genes. And at this stage, 17 genes were upregulated and 6 genes were downregulated. At both the stages, HB2 versus MB2 and HB2 versus MB2, we have found that ubiquitin specific protease genes was highly upregulated, whereas MYB30 was downregulated at both the stages. After that, we have found what are the unique genes present in 6 possible combinations, which are upregulated and which are downregulated. We have found that this picture shows that the unique genes and 6 different possible combinations. These are the upregulated genes, and these are the downregulated genes in 6 possible combinations. After that, we have shown the heat map of the different flowering genes, and these are some flowering genes which are present in healthy birth stages compared to 3 different malformed stages. These are the healthy panicle development stage genes, which are expressed in 3 malformed stages. The the green color indicates the upregulation, whereas the red color indicates the downregulation of the genes. After that, we have those flowering genes we have mapped into 10 different flowering related pathways, and we are able to found that upregulation and downregulation of those flowering genes, and we have mapped in this putative model of flowering pathway. And the most upregulated genes were found in autonomous pathway, followed by photoperiodic pathway. After that, slide is not. After that, to confirm our results, we have done RT-PCR analysis, and we have selected 5 genes along with reference genes with the elongation factor 1A. And these are the details of these genes which we have done through RT-PCR analysis. And the RT-PCR analysis has supported our transcriptomic data results, and which indicates that the validation of our results. After that, we have done, since it is a pathogen induced, we have done the Biotech stress pathway studies through Mapman and enrichment analysis, and we are able to find that ERF in transcription factor, ERF and Werke genes. These genes are also responsible for flowering in flowering also, and these genes are highly upregulated. After that, we have studied the plant pathogen interaction pathway, and in that pathway, we are able to find that the number of genes were of Werke and calcium binding protein and disease resistant protein were more in healthy stages compared to malformation. But when we go for differential expression analysis, we are able to find that only Werke transcription factor was high in this healthy stages. Now, coming to the conclusion, this transcriptome assay has narrowed down the genes and pathways which are really useful in mango resistant breeding program. The highly upregulated flowering genes were lysine specific demethylase, Topless, Werke, and UBP12, and whereas downregulated was MYB30. This differential expression and the differential expression analysis and Mapman analysis revealed that Werke genes are involved in defense against malformation. And these results of this study provide a resource for exploring complex molecular mechanisms in flower development and identification of gene targets for functional and applied genomics in mango. This research has been also published in the Springer Journal, that is Journal of Plant Biochemistry and Biotechnology. At last, I would like to thank you for the organizing committee for inviting me, for giving me a chance to present my research, and all those who have directly and indirectly helped me, ICR, IRI, ICR, National Institute of Plant Biotechnology, and DST for providing fellowship. Thank you.
Okay, thank you, Dr. Ashok Yadav. Thank you. We will go to the next speaker. That is Dr. Raghavendra G. Master Professor, College of Horticulture, Bagalkot.
Good afternoon, everyone. Respected Chairman,
He will
Co-Chair.
speak on nutritional profiling. Yes. Yeah.
Yeah. Good afternoon, everyone. Respected Chairman, Co-Chairman, the delegates, and all the participants. Indeed, it's my pleasure to present our ongoing research on an underutilized crop, but a very powerful vegetable crop with respect to breeding as well as nutritional aspects, that is the Mangalore melon. Mangalore melon, commonly called as the Sambar Saute or Mangalore Saute. It is called as Sambar Saute because it is mainly used for preparation of sambar, a kind of vegetable stew, which is commonly prepared dish in case of southern India. And also it is called Mangalore Saute because it is most commonly used in Mangalore and other coastal regions. But it has been cultivated in all the southern states of Karnataka, including Kerala, Karnataka, Tamil Nadu, Andhra Pradesh, and Telangana. And by that time, they are called by different names like Mogekai, Dosakaya, Kanivellari. Botanically, it belongs to a Cucurbitaceae family with the genus Cucumis, Cucumis mello, subspecies agrestis, variety acidulus. Many times, the Mangalore melon has been confused with the Conomon, but acidulus differs from Conomon with respect to the fruit size. The acidulus fruits are bigger compared to the Conomon fruits, and also they have longer shelf. Acidulus fruits have longer shelf life. The origin of Mangalore melon is still debatable, but many molecular studies have shown that the Mangalore melon is mainly domesticated in the southern India. So, but still the confirmation needs to be done. It is a treasure for the breeding of desert melon, that is the musk melon. It's because it has a high long shelf life of more than 6 months. So commonly the farmers follow hanging the fruits to the roof, and they are at the room temperature by tying with the help of banana fiber or the coconut fiber. So this fruits can be stored like this for almost up to an year. And in our research, we collected around 84 different genotypes, accessions from all the 5 southern Indian states, and they have shown high variability for fruit size, fruit shape, and skin color. And they have also shown resistance to diseases, especially cucumber mosaic virus, zucchini yellow mosaic virus, downy mildew, and as well as anthracnose. The unique feature of this Mangalore melon is, so since it has a similar set of homologous chromosomes similar to the desert melon, that is musk melon, so it is readily crossable with the musk melon. So we have obtained an intraspecific hybrid between a Mangalore melon accession and a musk melon variety Haramadu. The intraspecific hybrid has shown that bigger sized fruits than the musk melon, and also they have shown extended shelf life up to 90 days, and the flavor is similar to the musk melon. So that is the added advantage. So like this, we have developed several hybrids. So apart from its breeding value, it also have a lot of nutritional and medicinal values. So it has been reported that the immature fruits, which are commonly used for consumption, have 3.6% protein, 4% fat, and 2.5% carbohydrates. It is also a rich source of several minerals like phosphorus, potassium, iron, zinc. And coming to the medicinal values, it has been used as an Ayurvedic medicine to prevent the macular degeneration in the eyes. It has been used as a functional drink to prevent obesity and diabetes. It has also been reported with antioxidant properties. The uniqueness of this Mangalore melon is the seeds of these fruits contain about 34.68% oil, among which 80.5% of belongs to the polyunsaturated fatty acids, which are required for human nutrition. Although several its medicinal and nutritional values are known, but till date, no specific nutritional analysis has been done. So we know that plants produce over 250,000 secondary metabolites or the biomolecules. Among 250,000, 74,000 has been reported with nutritional values or the medicinal values. So identification or profiling these thousands of metabolites through individual biochemical tests is impossible. Hence, in the era of omics, the metabolomics is a powerful tool to profile the multiple metabolites simultaneously from the fruits or vegetables using either GCMS, LCMS, or NMR. So in this study, we have used the LCMS to profile all the nutrients available in the fruits of Mangalore melon. So for profiling, we selected 3 accessions of Mangalore melon. The MS04 is characterized with a yellow peel or the yellow skin. The MS21 is green colored, whereas MS78 is a striped one with yellow and green color. And whatever the metabolites we got were compared with the musk melon variety Arcajit. Once again, we included 3 tissues. The one is the peel or the skin, the pulp, and the seeds. So the tissues were ground in liquid nitrogen and extracted with 60% aqueous methanol, and they were analyzed on the LCMS machine QTOF LCMS fitted with a Phenomenox C18 column, which is a relatively polar column under negative ionization. The data was processed using MS-Dial as well as XCMS online software for identification of the metabolites. The metabolomic features, that is the mass to charge ratios, were compared with different databases like Metlin, PlantSig, with the accuracy of less than 5 ppm, and also matches with the tandem mass spectra MSMS fragmentation, and they were quantified, their abundance were quantified in comparison with the musk melon variety through pairwise t-test. So the initial total ion chromatogram exhibited that there is lot of variation in the metabolome of melon as well as the Mangalore melon. So we identified around 6,776 metabolomic features. So as you can see, each dot is a metabolomic feature, and the size of the dot indicates their higher accumulation or the higher fold change. So these 6,000 initially to look at the variability in the metabolome, we subjected these 6,776 metabolites for the principal component analysis. The principal component analysis indicate that the metabolome of seeds, pulp, and fruit skin is entirely different. They were entirely grouped different, whereas not much variation was found in the seed metabolome and pulp metabolome. But in the fruit skin metabolome, it was identified that the yellow colored fruit MS4 was totally different from the rest of the fruits. Further, we characterized whether is there any metabolomic variation between among the oxidants. So in all the three tissues, so we could see that there is a significant variation in the metabolome of each of the MS21, MS78, and MS4, different from the AJ, that is the muskmelon variety. The principal components explained almost more than 50% variance. So in case of peel, PC2 could differentiate, PC1 could differentiate, sorry, PC2 could differentiate the melon from the Mangalore melon, whereas in case of pulp, PC1 could differentiate the muskmelon from the Mangalore melon. Similarly, in case of melon seeds also, so PC1 could differentiate the melon from the, that is muskmelon from the Mangalore melon. Further, we identified the differentially accumulated metabolites between different varieties in different tissues. So as I told in the previous slides, the MS4 had accumulated the highest number of metabolites, and it's shown that around 1,027 metabolites were specific to this MS04, whereas in case of pulp and seeds, more than 80% were common to all the three accessions of Mangalore melon, and around 50 to 100 metabolites were specific to each of the varieties. Then further, we subjected these metabolites for identification. Although we got several thousands of metabolites, after removing the isotopic features as well as the adducts, we could identify very few metabolites starting from 39 to 141 in MS21. That is one of the limitation of non-targeted metabolomics. In case of peel, we could find that the majority of the metabolites were belong to the flavonoids, followed by the amino acids and carboxylic acids. So here I present only few metabolites which are highly accumulated. Some of the essential amino acids like threonine, aspartate, and glutamate were highly accumulated to the fold change of 2.5 to almost 3.9 in comparison with the muskmelon. Similarly, it also accumulated high amounts of vitamin B3 and B2. And there was one metabolite, which is a quinone, which is similar to the toluquinol, which is mostly present in the fungal tissues, and which has got an anti-cancerous properties. So among the 26 flavonoids, all the flavonoids belong to flavonols, anthocyanins, and flavones, as well as isoflavones. In case of pulp, we could see that most of the abundant metabolites were the carboxylic acids and carbohydrates. So naturally it was and amino acids also presented the higher accumulation. The amino acids present in the peel were different from the pulp. So we had histidine and arginine instead of threonine and aspartate. In case of pulp, we identified a metabolite called as sanguinarin. The sanguinarin has been reported with an anti-cancerous properties. So we need to further study about the anti-cancerous activity of this metabolite. Similarly, in case of seeds, we found lot of fatty acids and amino acids. So among them, arginine was highly abundant to the extent of 45 times higher than the muskmelon. And also we got vitamin B5 and riboflavin with a higher component. Apart from that, we identified a lot of C18 fatty acids, which are not significant between the melon and the in the seeds of melon and the Mangalore melon. So, but they are present in very high abundance, and those were grouped into fatty acids, glycerophospholipids, sphingolipids, and glycerophospholipids. However, further we need to confirm their identity using MS fragmentation. Apart from fatty acids, we also identified hundreds of peptides. So we need to identify the role of these peptides in human nutrition or any other properties. So with this, so whatever we have done is a preliminary work. Further, we need to develop a nutrient catalog of Mangalore melon with absolute quantification and identification. And the Mangalore melon is usually consumed as a cooked vegetable. So we need to check the bioavailability of nutrients after cooking, what happens. And finally, our objective is to promote this Mangalore melon as a protective food for the next generation. So with this, I thank our funding agency, Premnath Agriculture Science Foundation, for providing the fund facilities, and CAMS Venture Center, National Chemical Laboratory, Pune, for doing the LCMS analysis. And I also thank my research team, especially Dr. Ratnakar Sheth, for collecting the hundreds of accessions of Mangalore melon, Dr. R.C. Jagdish for overall coordination, and my students who are responsible for developing this, conducting this research. Thank you, Anand. I thank the organizers for providing me the opportunity to present my research to the community. Thank you.
Okay.
Thank you. They can cut down the introduction.
Thank you, Dr. Arvind. We'll go to the next speaker, Chandrayappa.
Yes,
madam.
It's time.
Let them cut down the introduction.
We'll go to the next. His introduction was slightly longer, yeah. We'll go to the next speaker. Okay. We'll go to the next speaker, who is Siddhanna Saudi from ICR, Cashew Research Station at Puttur. So let him start. Time now.
Thank you, ma'am. I
Yeah.
would like to thank
Please
the
go ahead.
organizers for giving me this opportunity to present my work on de novo assembly, annotation, and phylogenetic analysis of cashew genome. Are my slides visible, ma'am?
Yes, sir. You go ahead
Yeah,
first.
your slides are visible for 10 minutes.
Okay, ma'am. I'll complete
Maximum of 10 minutes.
Cashew is an edible tree nut crop which is grown over more than 30 countries, and it has got a very important nutritional component which is rich in minerals and polyunsaturated fatty acids. On the first day of lecture series of this conference, Dr. NBPGR director said that we are lacking in the consumption of fruits and nuts, which are rich in minerals. So they play an important role in human health. So in the same way, the cashew is also plays a vital role in nutrition of human beings. It ranks third in the world after almonds, walnuts, and it contributes about 17% to the edible tree nut crops. The worth of cashew nut world trade is about $9.9 million. Here I would like to talk about the status of cashew in India. Cashew came to India as a soil conservation crop, and it has now grown up into a prominent commercial crop, and it is considered to be a dollar earning crop, and it annually produces about 7 to 8 lakh tons of RCNs and stands second in the world. And we are still importing about 50% of our processing capacity. And you can see here the Cashew Export Promotion Council of India's data that almost our production, sometimes it has overtaken, even the import of raw cashew nuts has overtaken. On the same side, you can also see the world map of cashew, where you can find that India is the highest consumer of cashew, which about 30%, followed by US and Europe. So you can see in this graph of here also that there is a widening gap between the cashew kernel export, value of cashew kernel export and raw nut import. And also there is a decrease in the export of the quantity of kernel nuts, suggesting that there is a growing demand in the domestic market for the cashew. Thus, arises the question whether this dollar, which was considered to be dollar earning crop, turning out to be a dollar training crop. So, and also in the future, it has been estimated that about 4 to 5 million tons are required for India alone, which is about 5 times more than the current production The major constraints for cashew production are the low yielding varieties, yield losses, and also the losses due to the pests. Tea mosquito bug and the CSRB are the major pests which cause the losses. And also the crop is very poorly managed. And the one more constraint for breeding of cashew is the low genetic diversity, as it is an introduced crop. However, there is a great increase in the demand for cashew. So there is also a need for increasing the yield substantially. The mode of improvement, genetic improvement would come from conventional breeding, has been coming from conventional breeding. However, it has been very slow and also the results are unpredictable and has a limited success due to the long breeding cycles of cashew, high degree of heterozygosity, and also the large plant size. And the solution is the use of molecular tools, which allow a precise, and also they allow seed at selections even at the seedling stage, and thus they contribute to faster genetic improvement. And it has been suggested that these are highly suitable for perennial tree crops. However, in cashew, the genomic resources are the fuel for application of molecular breeding. So, however, in cashew, we find that there are very limited genomic resources, and there was lack of genomic sequence for a very long time. And we have got very limited molecular markers of 21, and also there is a lack of comprehensive genetic map. Here you can see a few number of studies which are used, mainly the AFLP, about 194 and 11 SSRs to construct the first linkage map in 2000 by Cavalcanti and Grove. And also they have detected about 39 QDLs using the same genotypic data and the same population. There is only one study by an African group which has used the SNPs. Thus, the molecular breeding has been lagging in cashew. So in this study, what we aimed is to develop more genomic resources for cashew. So what you have done is we have assembled a draft genome of cashew. It's done its functional annotation, phylogenetic analysis. And also we have designed novel SSR markers by in silico analysis of draft genome. We also done a comparative transcriptomics between dwarf and giant cashews. We have developed certain mapping populations for development of linkage map. The strategy which we have followed for genome assembly is hybrid de novo assembly, in which we have used both short read sequencing technology as well as long read sequencing technology. The short read, we have used Illumina technology, which produces short reads but accurate sequencing data. Whereas we also use a long nanopore sequencing technology, which produces large read lengths, and it will be helpful to resolve the large complex repeated DNA segments in the genomes, which are very common to the eukaryotic genomes. So they complement each other to generate a good hybrid genome assembly. So this is the workflow which we have followed. We extracted high quality genomic DNA. The phenols are the major problems in case of cashew. So initially we removed them by using the sorbitol buffer, and also we have used a high salt concentration sweetout buffer to remove the salt contaminants. And then we have carried out 120x of Illumina high 4000 sequencing, and also 25x Oxford Nanopore grid ion sequencing. And both the sequences are processed and used for hybrid assembly, scaffolding, and gene annotation and gene ontology. Here are the results of genome assembly. The total cashew genome assembled was about 385 MB. These were arranged in a 4,981 scaffolds and N50 value of 2.55 MB. You can see here 3,575 scaffolds or more than 10 kbp, and about 21 or more than 1 mbp. These are the results of gene prediction and annotations. Over 40,000 protein coding genes were identified, and of which over 32,000 were annotated. And of these annotated genes, they played an important role in biological process, molecular function, and cellular components. We also mined the genome for microsatellite and SSR markers, and we were able to identify about more than 47,000 SSRs, of which 59% were dinucleotide, 24% were trinucleotides, and they were followed by 11% of complex SSRs. We also carried out a phylogenetic analysis with Rhabdopsis thaliana in 6 tree species, and we were able to see that it was very close to Pistachio vera, which is also a member of Anacardiaceae. We also carried out a comparative transcriptomics between a giant type of cashew and a dwarf type of cashew, and we were able to see that over 12,000 genes were overexpressed in case of giant type and over 10,000 in case of dwarf types. Here are a list of genes which have just a few genes which have been overexpressing. And also we identified by in silico analysis the polymorphic genic SSRs, and we were able to identify about 129 polymorphic markers, and out of which 33 were showing more than 10 base pair differences. We also developed mapping populations between Bhaskara, which is a CNSL rich, and then NRC 188, which is a CNSL free. We confirmed their status. Here you can see the CNSL free type, which is very compact, whereas in case of CNSL, CNSL is a cashew nut shell liquid, the liquid which is present in the shell. So you can see honeycomb-like structure filled with oil, whereas here you will find the closed structures, so which is lacking in oil, that is CNSL, and we confirmed it by extracting Soxhless method, and we have also developed another mapping population between dwarf and the Bhaskara. So, and we have planted in the field, and here this is the beginning which we have made, and we'd like to work in the future for designing more and validating the more SSR markers, and also would like to develop a linkage map and go for tagging of genes and QTLs to employ marker selection in cashew, and also would like to identify and characterize genes which are responsible for our agronomic characters in cashew. Here I would like to acknowledge ICR and the acting directors, colleagues, and farm staff who have supported and are continuing to support this work. Thank you, one and all. I would like to thank the organizers as well as the organizing secretary, Dr. Fakhruddin, who showed us the, who has, during our MSc work, sir had taught us the biotechnology course and gave us the interest in biotechnology. Thank you. Thank you, ma'am.
Okay, thank you. Thank you. We will go to the next speaker. Thank you, Dr. Siddhana Saudi.
Thank you.
Thank you. We'll go to the next speaker, and hopefully he's the last speaker also.
Excuse me, boss. You have a text message.
Mr. Sagar Yadav. Unlike all the other 6, he is a PhD scholar from NCL Pune.
Okay.
Mr. Sagar, please start your talk.
Yes, ma'am. My slides are visible?
Yes, visible.
Yeah, you make it a slideshow. You make it in a slideshow.
Okay.
Yeah. Okay, start.
Hello, everyone. I'm Sagar Yadav, PhD research scholar from CSIR National Chemical Lab. So, the title of my topic is Exploring Metabolic Responses in Wheat Bipolaris Sorokina Interactions During Spot Blotch Disease Using High Throughput Omics Approach. So coming to the introduction part, wheat feeds about 35% of the world's population and ranks second in the production, but its production is hampered by the disease called spot blotch, which is caused by Bipolaris sorokiniana. The causal agent is Bipolaris sorokiniana, which is the hemibiotropic fungus, and over 10 million hectares of wheat is under disease in 2013 in India, and annual yield loss is 15 to 20%, and can reach up to 100% under the favorable conditions for the disease. And this is the distribution of Bipolaris sorokiniana worldwide, and it can be seen that it is present in almost all the continents, and in India, it is seen in West Bengal, Bihar, UP, Punjab, and it is spreading towards central and southern part of the India. So research gap is resistance gene pool in wheat for this disease is very limited. Molecular basis of disease is poorly understood, and genome-wide studies on spot blotch have not been reported so far. Based on this background, there's something coming in my slides. Are my slides clearly visible?
You close that. Yeah, you can close that box.
Please drag it away.
Drag it out. Yeah.
Not working. I'm not able to share the drag that.
You can try to move it.
You minimize the slide, then you will see the background, then you remove that. First, you minimize the slide. Minimize.
I think it has gone.
Okay.
Please continue, sir.
Can you click on the next slide? Continue and click on the next slide.
Okay.
Click on the next slide.
Fine.
No, it's
Are my
still
slides
there.
visible now?
Did you click on the next slide?
Yeah.
Yeah, now it has
Changing slides.
gone. You continue. We can see you can continue.
Okay. So the objectives based on this background, the objective design was analysis of this wheat and biopolymeric circular interaction at various time points using the omics approach to identify the putative disease resistance gene. Coming to the methodology part, the greenhouse experiment was designed where the greenhouse experimental setup was done, where the seeds were grown under a confined and controlled condition in greenhouse, maintaining the proper conditions like temperature and humidity, which was favorable for the spot blotch disease. Seeds were grown up to 8 to 10 days, where the two-leaf stage was obtained, and this is the fungal culture, Bipolaris soroginana. The spores were harvested from this culture and was and the spore suspension was prepared, and at the rate of 10 to the power 3 spores per ml, these spores were spread onto the on the leaves of the wheat, and the disease symptoms were observed. So this figure shows the phenotypic effect of this Bipolaris sorokinana on wheat genotypes. So here we have used two genotypes, one is susceptible and one resistant. As this my data is not published, so I'm not disclosing the names of the wheat varieties, the genotypes. So in case of susceptible genotypes, we have seen that on spraying the on spraying the fungal spores, right from the third day, it was seen spots in case of the susceptible genotypes where the disease symptoms increased gradually, and right from the day 7th, we can see the browning of the leaves, and at 10 days post inoculation, complete withering and browning of the leaves was seen. But this case was opposite in case of resistant genotype, where very small spot was seen at 6 dpi, and which shows very little increase up to 10 dpi, but there was no further any increase in the disease symptoms in case of the resistant genotype. So based on that, we calculated the time points of 1, 4, and 6 days post inoculation along with the 0 dpi for further study. This is the workflow of metabolomics approach. Here I have done untargeted metabolomics. So leaf tissues, as I have mentioned, all the 4 time points. So those were taken, and the samples were prepared for the metabolomics, and then metabolites were extracted using the 80% methanol, and further data was given. HR high resolution LCMS was used for data acquisition. Then further data processing and differential analysis was done in using metaboanalyst, and statistical analysis was done in that, and further identification of metabolites and biological significance was done. So this, as I have told, I have used two genotypes, resistant and susceptible. So as shown here in this slide, there are 4 such conditions: resistant control, resistant inoculated genotype, then susceptible control, and susceptible inoculated genotype. So 4 such conditions were there, and four such time points: 0, 1, 4, and 6 days post inoculation. So here PCA, we can completely see that all these 4 conditions were properly separated, where the RC, RI, SC, and SI are properly separated. And this one cluster shows the biological replicates, where it mentions that there were 6 biological replicates in each cluster. So all this proper separation was seen in all the 4 time points. Further, we have done for the PLS-DA, where the VIP score was done. So as seen in this figure, top 15 metabolites were seen in all these 4 time points, and all these top 15 were different in all these 4 time points in all the 4 treatments too. Yeah, which shows the accumulation of metabolites right from high to the low in all these 4 such conditions. Then overall, we have found out 1864, there's 1864 metabolites were detected in all these treatments, and there were many such significant compounds were found in the resistant genotype. So to few the name, flumethazone, vernofluxacide, and salicylicoline was seen accumulated in higher amount in case of only resistant genotype, but which was completely absent in case of the susceptible genotype. And this compound belong to the flavanol, terpenoid, and phenol group. So from this, we can conclude that these metabolites may play an important role in imparting the resistant mechanism in case of the resistant genotype, as this was completely absent in case of the susceptible one. And also in my lab, I am doing the other omics approaches like proteomics and transcriptomics. So by comparing the proteomics, transcriptomics, and metabolomics, that is integrated omics, can be applied to identify the putative disease resistance gene for this spot blotch in case of the wheat. Thank you.
Okay. Okay, thank you, Mr. Sagar Yadav, PhD scholar from NCL.
Okay, thank you, ma'am. Yes, ma'am. Yes, thank you.
I think, okay, thank you. Are you, anybody having question for this Sagar Yadav? No, no. Okay. So with this, we have come to the conclusion of this session. We have lost some time in between also. So I'll just conclude in a second. We had an excellent talk by Dr. N.K. Singh, product oriented, and others also. We had very good talks, and this is the end of this session. My co-chairman, I thank co-chairman and all the organizing committee people, and I request Chandrayappa if he wants to say a few words. He can, please.
I think now the other session people are waiting. I think there were excellent speakers on diverse fields. It was very good. It was very informative. I say thank you.
Okay.
Thank you, madam.
I thank all the people involved. Dr. Chandrayappa, thank you.
Welcome.
Thank you, madam. Thank you. I would like to thank Professor Shailaja Ittalmani, madam, Emeritus Scientist, and Professor Chandrayappa, sir, for being here and for chairing such a very important session where we need a combinatorial approach of integrating these multi-omics platforms for assessing molecular systems to increase productivity. Thank you. Thank you very much on behalf of our organizing committee, madam. And I would also like to thank rapporteurs, Dr. Venkatesh and Dr. Mahantesh B.N. Nayak for reporting the session. Thank you, madam.
Hey, I forgot to thank Dr. Venkatesh and Dr. Mahantesh. Thanks from my side also, Venkatesh and Mahantesh Nayak.
Okay, madam. Thank you. Thank you. Thank you, madam. We'll just move on to the next. Yeah, madam. So we'll move on to the next session. I request our colleague, Dr. Hollis, sir, to continue with the next session. Sir, please.
Second. Hello.
Hello.
Hello. A very warm good morning to all the delegates and the participants. Now we have entered into this third day afternoon session. That is the seventh theme. In vitro culture and micropropagation. Now we have with us two eminent scientists. One, Dr. R. Lokesh, Dean, University of Agricultural Sciences, Raichur, and another eminent scientist, Dr. Payas Thomas, CEO, Thomas Biotech and Saito-backed Center for Biosciences Private Limited, Bengaluru. Now, I just want to introduce our, give introduction about our scientists. I just want to introduce our speaker, our chairman, Dr. R. Lokesh. After his graduation, post-graduation, and doctoral degree program, Dr. Lokesh has entered into University of Agricultural Sciences, Raichur as assistant professor. So during 1992 to 97, after this 2005, he has started working as associate professor. During 2005 to till date, he is working as professor head. After that, 2.5 years, he was worked as dean Agri at College of Agriculture, Bhimrayanagudi, since 2.5 years. Regarding his professional skills, his specialization is in plant tissue culture, biotechnology, and biodiversity. He has contributed a lot in sesame crop. He has guided 25 postgraduate students and 6 PhD doctoral students. He has published more than 180 research papers. He has got several awards. To name few, he has got this Lifetime Achievement Award from IARDO, and he has received an award for professional excellence from different societies. And also he has received Best Teacher Award also. He is a member for this Academic Council, Research Council, Extension Council. Still, he has made a very good biodata. So next, I want to introduce about our co-chairman, Dr. Payas Thomas. So Dr. Payas Thomas did his graduation and post-graduation in Thrissur, Kerala Agriculture University. For his doctoral program, he entered into this Indian Agriculture Research Institutes. After that, he has done his 3 postdoctoral programs in University of Sheffield, United Kingdom, and then University of Michigan, Ann Arbor, USA, and the Flinders University, Adelaide, Australia. He did his 3 PDF programs. Regarding his research experience, he has entered into this ASRB, that is as a scientist. There he had worked nearly more than 20 years. He has worked as a scientist, and then he has worked as principal scientist and head of the plant biotechnology division also. He has got several awards, namely this NAS, National Academy of Agricultural Sciences, New Delhi during 2011, and then a fellow of the National Academy of Biological Sciences, that is NABS Chennai. Like this, for different organization, he has become a fellow. He has guided nearly 5 PhD students and then 4 MSc students. He has handled several
projects. So his research publications, so more than 50 publications he has got in international journal. He has got several publications, among them more than 50 has got in research, international journal repute. This is about our co-chairman. So now I am handing over the session to chairman and co-chairman, sir, Dr. Lokesh sir and Payas Thomas sir, please. I request the chairman, co-chairman, to strictly manage the time, sir, please. We are already lagging behind.
Sure.
1 hour and then 20 minutes.
Okay, fine. Hello, everybody. I'm Dr. Lokesh. First of all, my thanks to Dr. Fakhruddin for giving an opportunity, and also Dr. Halesh for having introduced me. So we have with me the co-chairman, Dr. Payas Thomas. I know him very well. And of course, there is a set of speakers also there. And we're going to have a keynote address by Dr. S. Uma, which is for 30 minutes. And I request all speakers to kindly maintain the timings. Then invited talks, 3 are there with 25 minutes each. Then we have oral presentations followed with 10 minutes each. Okay, so we know that tissue culture is one of the component which is helping in horticulture crops, and not much work in case of crop plants, but however in some cases it can be very, very useful as well. So with this remarks, I request the first speaker, I request Dr. Payas Thomas if you have any point to say. Co-chairman, please.
Thank you, Chairman.
Payas Thomas, please.
Thank you, Chairman. I think we can hear me well. Yeah. I wish to thank Chairman for the introduction, and also Dr. Halesh for giving this opportunity. I think we are lagging behind time, so straight away we can go to the session. Thank you, sir.
Okay. Yeah, I request Dr. S. Uma, the Director ICAR NRC for Banana, Tiruchirappalli, to be the keynote address for this session, and she'll go to speak on next generation tissue culture technology for vegetatively propagated plants using bioreactors. Madam, please. You have 30 minutes, madam.
Thank you, Chair. Thank you, Co-Chair. I also see good number of my friends in the lines going to be the speakers. Nice to be back to US Bangalore and UHS to be part of this, and having this opportunity of presenting this our work for the last couple of years. What we have done, that has been compiled into a presentation called Next Generation Tissue Culture Technology for Vegetatively Propagated Plants Using Bioreactors. As you said, rightly said, tissue culture is the mother of all biotechnology. It started with tissue culture, but still it has its own stake in all the biotechnological research. With this, quality planting material is very much accepted component of agricultural production and development. Worldwide, it is accepted. For seed and planting material, for the availability to small scale farmers, it's always shortage. And globally, there is technical guidelines and standards are available for quality seeds known as quality declared seed, QDS they say, but mostly this is for the seeded crops. But in vegetatively propagated crops, except for potato and banana, none of these crops have received any attention, and for the formal seed quality regulatory systems, especially fruit crops and yams and cassava. These are all the staple foods, but they have been very well neglected. I would rather say it's highly unorganized sector. So in this presentation today, you will realize there is expanded possibilities for improving the accessibility of quality planting material in vegetatively propagated crops. And banana I have taken as a case study and as a success story, and it can also serve as a model for other vegetatively propagated crops. Why banana? It's very important. I don't want to go much in detail, but only thing what I would like to say is there are a lot of constraints in the banana production, mainly the soil-borne diseases where suckers are taken as explants for tissue culture. So whenever you take as tissue culture, yes, in the third or fourth subculture, you always have the secondary contamination and the varieties. I think everybody can mute. I request, please. With the re-emerging and new pests and diseases, especially Fusarium wilt race 4 of 1 and 4, races 1 and 4, which is affecting the whole banana industry in the world. This tissue culture or the planting material has become much more important. And another reason is banana is a polyclonal system where we have not only grand name for which tissue culture technology is very well there, but for multiple crops, multiple varieties, this is not there. And here, organizer you may ask others to mute or you can mute yourself, please. So here, somatic embryogenesis and embryogenic cell suspension technique offer several advantages and
we disconnect.
Dr. Yogesh Bhagat, yes. In banana, the suckers are sold on the roadsides as the planting material. This becomes the source of diseases and many of the pests in the planting. But tissue culture, as we know, very well streamlined. You have the whole process here, starting from suckers until you have the planting and until rooting. The process is very well done. Then, and tissue culture technology is available. You have plenty of tissue culture companies working on it. Thousands of plants are produced. Then what is more required? That was the question. So at national level, if you see, for banana, you need 1,000 million plantlets. I would rather say 1,200 million plantlets are required, even if it is for alternate year of planting, not annual planting. Even if it is alternate planting, you need 1,200 million plantlets. But our capacity right now is only 250 to 280 millions. So there is a gap of 700 million plants approximately. So this, along with soil-borne diseases, we have to look for something which is much more better than tissue culture. That's when the intervention of ICR, NRC, banana came into the picture. The concept of somatic embryogenesis we tried to exploit. So you know what is somatic embryogenesis. I need not go much into detail, but this is derived from a somatic cell, not a gamete. Somatic embryo. And the commercially adopted applications of this somatic embryos and somatic embryogenesis is that it is a form of mass multiplication. Yeah, there's a chance of production of artificial seed production. It's easy, amenable for mutation breeding for any crop improvement program, and it is also good. It's a good explant for genetic manipulation, especially the genetic transformation studies. If you see worldwide, the commercial been an extended number of crops, starting from stevia, but only in few crops. Though I would say the somatic embryogenesis they have exploited in production of planting material, the commercial production has gone only for couple. Some, for example, is trifolium. Coconut, there is a system available, but it has not gone into commercial scale. And you also have other crops like desmodium. Then the conifers is one such crop where somatic embryogenesis is taken for large scale multiplication. You have a couple of number of conifers which have been multiplied, and you see the plantations like this, commercial plantations. The next commercial application has been happening in coffee. You call this as temporary immersion, TIF, the temporary immersion bio. And this has been, see, this is a kind of bioreactors used for coffee. They multiply large scale, and this is the kind of laboratory facilities you have in the using bioreactors and the large scale production of planting material. You see here using the bioreactor here, and the planting material ready for transportation from the tissue culture companies, and it goes wide, far and across from France and grow coffee. They multiply coffee, they send it to Latin American countries. Cuba, all these countries, the plant material. Even if it is the system is available to make that a commercial venture, it really needs some kind of strategies. And application of bioreactor. So somatic embryogenesis produce plants from somatic embryos. Then how bioreactor comes into the picture? These bioreactors, all of us know, they are self-contained, sterile environments. They work mostly on liquid nutrient, and you can monitor it. So you can have a control over microenvironmental conditions. All these, like your oxygen content, carbon dioxide, temperature, humidity, everything can be controlled in a bioreactor, like in any other tissue culture. How do we do this in banana? It's simple, what you see. Here we take the flowering bud as the explant. We don't take the suckers, because suckers, every chance of soil-borne diseases coming along with it. And we use the planting material. We take the very meristem, at the meristematic tissue, at least the 12th flower florets. We take it and we initiate, and they grow into the pro-embryos, embryos, which you transfer them to the glass vessels or the beakers. And from here, the scaling, you take very minute amount of pro-embryos or embryos, that you have to scale it up regularly, so that it comes to the sufficient quantities, and you maintain them in kind of culture racks, and you with agitation or with almost 80 rpm per minute, and you get this kind, whatever the embryos you get here, you make them germinate and you develop into plantlets. This is the normal somatic embryo converting into plantlet. For scaling up, we worked on different bioreactors, and that's where we designed and fabricated different bioreactors with it approach. Many engineers we in the project, and we designed 4 different bioreactors. So one was balloon type bioreactor, the bubble column balloon type, and the balloon type, and this is another type of balloon bubble column. So for scaling up, we finally came to know, we understood that we standardized that a bubble column balloon bioreactor with 2 liters capacity seems to be a very good prototype. This was, we tried initially with grandine, and we also wanted to see it should work for all other crop species, all other banana varieties. So we worked on 3 different varieties, that is Rastali, grandine, and sabri, and it found to be working too good for all these 3 varieties. So from using this this kind of balloon, bubble column balloon type bioreactor, we could increase from 2 ml to almost more than 100 ml within 21 days. We could multiply this embryos. And we also saw that you have on the first 3 days, you have a log phase, and from 3 to 18 days, you have an exponential growth phase. And after that, either it stands still or it goes into the lag phase. So with this this column bioreactor, as I said, you can produce from 2 ml to 100 ml, and from 1 ml of cell suspension. This is the cell suspension we call it. Is from 1 ml of this, you can produce from 4,000 to 40,000 plants, depending on the variety. It's not a small amount. 4,000 to 40,000 plants. So how do we maintain them? We maintain them on this kind of culture racks with agitation at 25 degrees with 70 RPM and in dark condition with periodic viable test and regeneration test. Whenever we do the regeneration test using FDA, it should be any good suspension should have more than 80% regeneration capacity. That is taken as a good suspension, and we try to develop that suspension as a bank, the suspension bank. We grow it in large quantities and we maintain them. So how do we do it? Well, so once you have somatic embryos here, you put it in the bioreactor, and from this bioreactor, slowly the embryos that germinate. And these germinated embryos, you take, this is the small amount of somatic embryos, you take it here, they grow into these many number of embryos germinating, and the small quantity of germinating embryos, you take them out and put it in our bioreactor. They produce the plantlets to the tune of 500 to 600 plants per vessel. This is how you get. In a mat of plants, you get 500 to 600 plants. This is from one vessel. This is a harvested plants. You can see it here. Excuse me. Then, should we grow only in bioreactor or the glass vessel? No, grow in your own container. We have certain kind of mechanism and attachments. You can grow in your own vessel, open vessel, with your cooking vessel. You can also grow in trays like this. You distribute the somatic embryos in such a way that they grow uniformly, and you harvest a mat like this. So, and as usual, it goes for primary and secondary hardening. So how much time do you need? If somebody will ask me how much time do you need, see, you have, as I said, you should have a suspension bank. You should have suspension of a variety. Once you have sufficient quantity, it will take 30 days. If you want to increase this, if you have only 20 ml of this or 50, it to some 100, 200 ml, this needs 30 days. From here, after you enhance the quantity, from this, you take out for germination or production of plantlets, it will take 60 days. The germination of embryos and development of plantlets will take 60 days. In case, if you don't want to increase the size, you just want to grow some 600,000 plants, you simply take out 1 ml or 2 ml of your somatic embryos, take it directly to this bioreactor, and within 30 days, you can, within 60 days, you can develop plantlets. So either you grow in 60 days, or if you want to increase the quantity, then you hardly need 90 days. So over a period of 120 days to 150 days, your plants are ready with hardening. Ex-agar plants are ready within 90 days. So what is the bioreactor system we have developed at NRC Banana? We have developed a system called somatic embryo regeneration vessel, which we have filed a patent both for the process and the design. So with this, each vessel, it can, as I said, 500 to 600 plants, and each rack can have 18 vessels, and each rack can produce 9,000 to 10,000 plants within a span of 3 to 4 months. So in a room of 10 racks, you can produce 90,000 to 1 lakh plants within a span of 4 months. This is over a period, over 1 year, 3 lakh plants you can produce in a room having 10 racks. This is the capacity of the mechanization or the mass production or the high throughput production using this somatic embryogenesis. Cost economics. The cost is, I'll see the convention, I said, not the tissue culture convention system. The somatic embryos, you put them on the plants, or the NRCB technology using this serve vessel. Approximately, it works out to be the plant, agar plants works out to be 6 rupees. Works out to be only 40 paise. For hardening, this is ex-agar. Component is included. It works out to be 10 rupees depending on components what you use, or depending on biopriming, all that. Excluding all that, you have 10 rupees per plant, and this works out to be less than 5 rupees per plant. Say how true, how are you true about the large number of plantlets, and maximum variation seen is less than 3%. We have observed around, depending on the variety, from 1.8 to 2.8%, we have observed the somatic variation. And have you evaluated in the field? Yes. All these 3 different varieties we have produced on large scale. More than 10,000 plants of each variety have been taken to the field, and they have been evaluated. Grand Nain, Rastali, and Sabri of this is Grand Nain plant crop. This is down, you see Grand Nain plants. Not for annual planting, it's a ratoon crop. Similarly, for Rastali, you have plant crop and the ratoon crop. So everywhere it is shown that the bunch variety is on par with the shoot tip culture plantlets. Similarly, for Rastali, it is ECS derived plantlets have also shown. I would rather say they are compared. Okay, so if this is the technology available, what is the problem with these bioreactors for banana? Banana is not only Grand Nain. Everybody thinks only about Grand Nain, but banana is a crop of more than 1,000 varieties. But commercial varieties, I would rather say around 20 varieties are commercial. Can this work for all the commercial varieties? No, it doesn't work. This is because some varieties, they are good for callus production. Some of the diploids, even it produces callus. Grand name, they all produce callus. The next stage, embryogenic callus, not all of them produce embryogenic callus. And after this, if it's going to be ECS production, then again, your some of the AA diploids, they don't respond well. For regeneration, germination, and plant regeneration, even if it is a group, AB is a group of, has a group of varieties like Kundan, Yelaki, all these are, they come under AB, but not all of them. More than a year. And I asked what the longevity of this cell suspension. We have used cell suspension produced even after 15 months, the regeneration capacity and not reduced to less than 8%, always maintained 80%. In somoclonal variation, it has ranged from again 2 to 3%. So the longevity, I would rather say, right now we have tested up to 1.5 years. It is okay. But my recommendation is that never maintain for more than 1 year. Make it as 1 year and go for fresh inoculations. Then, how to overcome these limitations? That means not all the varieties are responding very well. Not all the varieties are amenable to somatic embryogenesis. How do we overcome this? We try to work on their proteomics and transcriptomics. We try to see why some are embryogenic, why some become non-embryogenic. During the developmental stages, why some somatic embryos, they germinate, why they don't germinate. All these we worked out. Different pathways involved in both calla induction, then embryo formation, then germination. These are the three important critical stages. We try to work on them. And we see this is embryogenic, this is non-embryogenic. We try to find why this difference and pathways involved, why this is amenable, why this is not. And proteomics, as I said, and gene ontogeny has been worked out. We have found differentially expressed proteins, and some are uniquely expressed, some are upregulated, some are downregulated. All this data was compiled, and functional classification also we did. Then for the next step, why some embryos are recalcitrant, why they do not germinate. See, some they germinate, some they do not germinate. We try to work by this difference also. For this also, classification of proteins we try to do. And some, as I said, the germination. See, these are germinating embryos, these are non-germinating embryos. So the reason could be very different for depending on the variety. So for all these, when we worked out we have worked out the different pathways, and once we understood the pathways, we tried to validate the differentially expressed proteins with respect to their transcripts. This is some of the proteins at different stages, like early embryogenesis, somatic embryo development, late embryogenesis, and using RT-PCR, and also validation of proteome result of Grand 9, Rastali, and other recalcitrant varieties with their transcripts. And once we understood, we tried to manipulate the callus induction and germination media based on the molecular analysis. So what we did is the question. See, we know that for embryogenic calli, these are the 5 different hormones or ingredients which they really matter. We try to augment the media or reduce or increase or change the concentration of these hormones. For different varieties, a different methodology was tried. Even for germination of embryos, as I said, even the cold response proteins, sour proteins, and calcium related, GA and endogenous auxins, all these were related. And we try to modify based on this. Based on the triggering component, in some cases, we increased the IAA, we increased cytokinins, either like BAP or kinetin. Then sometimes, in some varieties, we increase the calcium chloride. So we try to modify the media so that what makes the what do you say, the problem is analyzed by changing the media. And these are the different varieties we tested. See, the Grandin is the easiest one. Red banana is not so easy. Then Montan, that is cooking banana, or Karibale is not so. Naipuvan is good. See, Naipuvan, many organization, institutions, they have worked on Naipuvan. They say ECS can be produced. Produce for an experimental scale is easy, but producing for a commercial scale is an entirely different issue. So to make it a commercial issue, you really have to understand what makes it. You can produce from 1 ml, you can produce 100 plantlets. That's not our that's not what we want. Our idea is from 1 ml, not less than 10,000 plants should be produced per ml. If that commercial component is not reaching, then it is a researchable issue. So that's why even Naipuvan or the Elaki was taken. Then Karpuravalli is our Budubale. And Sabri is a variety of Tripura, which is very, very important for the northeastern states. We worked on these recalcitrant varieties. So we changed, the media augmentation was done. From the routine media, we augmented the media with different augmentation was done with different ingredients. Then we saw the improved embryogenic cell induction. So you see even more from 200 to 700 times enhanced production of embryogenic calli was observed. In a variety like Sabri, it was very, very recalcitrant. It was never amenable for embryogenic calli. But with this augmentation, with these are not very big interventions. They are very simple interventions, but you should know what exactly is required for this. Then once it is, this embryos are produced, we try to regenerate them using their regeneration capacity or the germinability or their longevity. We also standardized what should be the regeneration protocol. And you see here, even for red banana, which was never produced even somatic embryos, it produced embryos like this, germinating embryos. And once you put them in our bioreactor, this is a test trial. What you see, very small amount we put, you can see more than 80% regeneration plantlets coming out of this cell suspension. So that means now we have a good cell bank for embryogenic cell suspension bank, even for red banana at our institute. So once you produce, we try to see the fidelity. It's a full protocol, definitely not much homoclonal variation. Then what is the success story? I said red banana is one success story we did. And now the plantlets are in field evaluation in Teni. The success story is a variety called Sabri. This variety is very, very important for the state of Tripura. It has high export potential for Bangladesh, but problem is that it is highly, highly susceptible to fusarium wilt. So people are facing almost total crop failure in many of the plantations. They have been, for the last 5 years, they have been struggling to standardize tissue culture protocol. Even we gave technology, we went and established the tissue culture laboratory, but somehow we were finding it difficult. So, but we, as I said, 700% enhanced somatic embryogenic calli we could produce with our technology, and we germinated them. And this was supplied to the stakeholders of Tripura. One MOU signed with the Tripura Biotechnology Council. Seeing this success, DBT has asked us to produce in the project mode to supply 150,000 tissue culture plantlets to Tripura state. You can imagine a scientific organization producing 150,000, because commercial production is not our mandate, research is our mandate. Even with a simple research component, if we have the capacity to produce more than 1 lakh plants, it is definitely a good indication. That means it shows the success of the technology. So what are the other applications of ECS? Definitely in banana, you cannot do breeding. You can develop. You cannot put seeded progenies. You cannot get to develop hybrids. So only one methodology is either you go for mutation or genetic transformation. So this embryogenic cell suspension has been an excellent explant for mutation breeding. It simply goes every day. We produce for different varieties, mainly for our commercial varieties, especially Rasbale or Rastali or Grande, which are highly susceptible to fusarium wilt. We take them, we either in vitro screening using fusaric acid, and we also use gamma irradiation, the facility which we have our institute. And large number of plantlets are being screened every day. So this is possible only because of ECS. If had it been for through tissue culture, definitely it would have taken more than a decade for us to screen the amount of plantlets we have screened in the last 2 years. So with this, we have I would proudly say we have 3 mutants each tolerant to race 1, both in Cavendish and for Rastali. And we have, these are screened in even for race 4 fields in Bihar. We have got excellent leads. These putative transgenic lines, these mutant lines are under for large scale evaluation. And what is the next application? Next application is genetic transformation. Genetic transformation has been made possible in banana only because of embryogenic cell suspension.
Madam, you are exceeding time. Can you sum up?
Yeah. So just give me 5 minutes. I'll sum up in this. This is what we have achieved through embryogenic cell suspension. You can see the biofortified banana, which is 10-fold enhanced PVA and 5-fold enhanced iron. This is the QUT lab which which our Prime Minister has visited, and this project is being monitored on a monthly basis through DBT. So this is what we have seen. The best, the advantage is that the moment you find the best event, it is easy. The more you can, these flower buds can be initiated, the cell suspension can be developed. The moment the regulatory formalities are over, you can produce any number of plantlets of this variety or the event and take it to the large scale multiplication. So this is the beauty of this technology. And the another one is that we have the synthetic seed. It is, of course, this is a common technology, but we are trying to develop the synthetic seeds and which can be taken, the somatic embryos as a synthetic seeds, which can be taken to planting. But the issues which we are addressing is standardizing the mass production protocol, regeneration efficiency, the storage issues, and the transportation logistics we are trying to improve upon. Okay, with all this, what is a new dimension for tissue culture production in the country? How do we centralize the production and supply of quality planting material using this? This is what the proposal to change. This is a proposal to change the banana seed sector from unorganized to organized sector. So what we propose is that have a centralized national facility like 3 or 4 or 5 facilities with the responsibilities like they establish and maintain the certified mother nursery with regular virus indexing and fidelity testing, initiation and establishment of suspension, maintain the suspension, then multiplication and regeneration. But apart from this, what are they planned to do? These centralized facility, the tissue culture, rather I would say to go to this field, this slide, you will understand better. Once you have the centralized facility, the big tissue culture companies can simply access the cell suspension and they can convert them into somatic embryos, grow them into plantlets and harden them. Yeah, otherwise, they don't have to maintain their own mother nursery and it doesn't need as big infrastructure facility as right now they have, the big companies what they have. If it is a smaller company, they will directly take the germinating embryos, they grow or they grow them, they get it rooted and take it for hardening. If it is for FPOs and self-help groups or small entrepreneurs, we can grow them, the ex-agar plants can be given like this and they can take it to their field, they can harden it and take it to their own planting, or they can have their own nursery, they can even sell it and make it a commercial entrepreneurship. The farmer, if he wants, he can take this kind of hardened plants directly from the centralized facilities as mats, he can take it to his field and directly plant them. So this is the kind of ease this technology can provide for the country. This is what we are trying to bring it as a policy paper to the government of India, which is very much interested now to take it on a large scale. So the advantages I said, it can cut down the culture rooms, the savings in terms of overheads and manpower, the ease of handling plantlets, then the production of synthetic seeds will ease the problem of bulk transportation, and these bioreactors can be exploited not only for banana, for any other vegetatively propagated crops if you have the technology to make them to produce a embryogenic cell suspension. It is a small intervention. You make them to develop cell suspension, then somatic embryos, then multiply, regeneration. If you standardize that, the bioreactor system can work for across the vegetatively propagated crops. This centralized mechanism can create entrepreneurs and provide job opportunities for the youth. What is the expected outcome? All of you know now NCS TCP is working, where it is certification is done by NCS TCP. But this bioreactor can make the tissue culture plants cheaper and affordable. And the central national facility eliminates the complexity of accreditation of tissue culture labs. The certification process of tissue culture plants can be made more simple and centralized, and the ease of transportation to the farmers. So this mechanism can take tissue culture plants cheaper and affordable and more accessible to the resource poor farmers, I would say. I would rather say that certification process, which is very, very complex right now, which BCIL is handling with accredited centers, and some being recognized, some being not recognized. And now there is a widespread occurrence of cucumber mosaic virus because of the change in certification agencies. So all these are problems can be overcome if you have a central nationalized facility like this. With this, this is my team. We had developed the bioreactor system. We have the funding supports. We had institute projects for core funding. We had ICR extramural project, and DBT Bioreactor did support us for the as a one component in developing transgenics. That is, for all the centers across the country, we were the responsible institute to give them the embryogenic cell suspension for their transformation work. That's how this whole thing came into a picture, and now it is in the stage of policy paper, and I would rather see in another couple of years this should go as a system of quality planting production in the country. I thank you very much for the organizers for giving me this opportunity. I thank all my team, my SMD and ICR for this research outcome. Thank you so much.
Yeah, congratulations, madam, for wonderful presentation, as well as the way ahead you are thinking into the bioreactors, helping small companies and also big companies. But one thing which I suggest is why can't you think of robotics into the system? There's already Jain irrigation systems that are using robotics into the tissue culture of banana. So you can think of in that line. So thank you for your.
Thank you very much. The Jain irrigation robotics is not for tissue culture; it is for just hardening. But robotics is the next. Is giving us a big project. Probably we are getting into robotics, and we are also working with international.
That's fine. That's wonderful, madam. Thank you so much for your presentation and wonderful information covered. Thank you most. Now I invite, there are totally 3 invited speakers are there with around 25 minutes each. So kindly stick on to the timing again. The first speaker is Dr. Samarendu Mahanti. So Asia Region Director, CAP, the Philippines. So he's going to speak on the economic viability of tissue culture based apical rooted cuttings for decentralizing potato seed system in India. Sir, please.
Thank you very much. Let me share my screen. Can you see my screen?
No, not yet. Okay, go ahead.
Okay, you can see it now? No?
Not yet, sir. Kindly share it, sir.
Okay, let me try.
Not yet, sir. Not yet.
Okay, now let me see. You can?
Sir. Yes, sir.
Okay. Okay, thank you very much.
Yes.
Good afternoon, everybody, and my pleasure to be here in this meeting there. As you know, I am not a scientist, I'm an economist, and it's my pleasure to be here with UHS Bangalore, my alma mater. I graduated from UHS long time ago. So as an economist, I'll give an economic perspective of things, of this apical rooted cutting. As many of you know, the potato seed problem is huge in India. Right now, if you look at our potato season is planting, potato planting is starting right now. The seed price in India right now is close to 80 rupees per kg, because all our seeds are produced in Punjab and moved to 1500 to 2000 kilometers to eastern and southern India there. So I have become a huge fan of tissue culture. I don't know anything about tissue culture, but I'll talk about this particular technology, which I introduced it from Vietnam. This is not a new technology. This is an age-old technology, and it was introduced in India a long time ago, stem cutting, but did not work because of economic regime there. So I'll be talking about this particular technology and what we are doing, including what we are doing in UHS Bangalore about this particular technology there. As you know, the potato seed, we have around more than 2 million hectares of potato acreage in India. We produce 50 million tons, more than 50 million tons of potato. We are second largest potato producer in the world. But if you look at the seed side, the seed replacement rate in India is less than 10%. That means most of the acres, the potato planted, they are not from the formal sector there. Most of the seeds used by the farmers are from the informal sector, and most of the farmers use it, the locally available seed from the market. The seed used in the eastern India, the small farmers in Odisha, Bihar, Assam, they are the leftover seed available in the market, the seeds which are not consumed by the human being. That means these are small size seeds they use, which are leftover, and they use as a planting material. So if you use bad quality seed, you will get bad yield. There is no magic in that. So the whole idea is that why we don't have a high seed replacement in India there. And one of the reason is the most of the formal seed sector is dominated by aeroponics, which is very highly, it's also tissue culture based, but highly capital intensive there. So it is dominated by few companies in Punjab there. They are the one producing the seed. If you want to produce seed to aeroponics, definitely it's a high quality seed, but the cost is becoming a huge factor in terms of the small farmers there. You cannot expect small farmers to give 50 to 60 rupees for 1 kg of seed and use 2.5 tons of seed per hectare. And that's a huge cost for the farmers, knowing they have a huge market base. Once they produce potato, there is no guarantee they will get a good return because the price fluctuation and the lack of infrastructure we have in India. So the price crash after the harvest and they lose money there. So that's the reason this particular technology comes very handy. It is a tissue culture based, same as the aeroponics, but the next step is lot less cost expensive than the aeroponics there. So if you look at the apical rooted cutting, it's very similar to talk to what Dr. Uma said. The nursery grown seedling, it's rooted, then the transplanted there. Cuttings are produced in tissue culture plantlets in the polyhouse rather than the mini tubers there. If you look at aeroponics, they produce mini tubers. But here we produce the tissue culture plantlet and transplant it. We produce a first generation seed there. After rooting, these cuttings are planted in the field to produce the seed tubers there. You can see the 3 stages of this apical rooted cutting, tissue culture, then products of the cuttings, then the products of the seed tubers there. So when we brought this from Vietnam a couple of years ago, our first facility started in Bangalore and Assam. If you are there locally, you can visit the facility and see how this particular technology is doing both technically and also economic wise there. So this particular technology has a huge advantage. It's already proven technology in many countries. It's an age-old technology. For whatever reason, it was not introduced in India a long time ago. It's very simple, very affordable, and can be easily integrated into our production system there. So it's a faster multiplication than the aeroponics there. If you're doing aeroponic seed multiplication, we are looking at the gestation period of 5 to 7 years to be economically viable. So the seed farmers get is G4, G5, G6 there. But in this particular one, the G2 seed, which is the equivalent of second generation seed in aeroponics, become economically viable there. You don't have to multiply so many to be economic viable. Remember that any technology you have, if the economic doesn't make sense, it will not leave the lab. It will stay in the lab. It will not be adopted by the farmers there. And the most importantly, in a country like India, where it is dominated by millions of small farmers, if you want to have a high seed replacement rate, it has to be led by farmers. It cannot be led by multinational and come down to the farmers. So any such technology, it has to be led by farmers. Then only we can have a high seed replacement rate and the decentralization of the seed system there. So it's a huge scope for the scaling up there. I'm just telling you, in 1 year, there are many farmers in Bangalore, Hassan, Haryana, Assam, Odisha. They are now taking up this particular technology because the simplicity and the cost effectiveness of this particular technology. So as you know, it starts with the tissue culture plantlet. I don't have to, you guys are all scientists, you know exactly. So this is talking about you start with the tissue culture plantlets. You start with one tissue culture plantlet, then you start doing cutting both in the tissue culture lab, then you bring it and harden it and you multiply and cut it in the poly house there. So we are looking at from 1 to 256 cuttings in the poly house in a gap of 12 to 15 days period, if you have 100 days of window there, you can end up having 256 cuttings from one tissue culture plantlet there. So this is the pictures you see from the Bangalore and Hassan facility there. You can see one of our team members doing all the cuttings in the poly house from the tissue culture mother plant bed there. So these cuttings are again transplanted and rooted, then taken to the field there. So you can see the cuttings there. So it's a very simple technology there. So you see there the mother bed in Bangalore facility there. You see the cuttings and again planted in the cocoa pit there. Just showing you some of the pictures. Then basically put in the tray. Also the cuttings can be put in the tray and transported to different parts of the things there. So I'm just showing you the pictures of 10 days and 15 days of cuttings. The young cuttings are very vigorous, so they're the one give you the highest number of the things there. So this is the facility in Haryana. We have in Haryana, this is the mother bed. This is how the mother bed looks like. So you can see how quickly this particular technology has spread in different parts of India there. So showing you some of the pictures, showing you how we are transporting from Bangalore to Odisha and Assam, all these places because of the window there. So this is also a picture you see in Hassan. The farmer is transporting the cuttings, the rooted cuttings to his field. So this is where the rooted cuttings, either you can put it in the net house, or also we are trying it in the open field there. If you don't have a lot of aphid population there, you don't have to stick to the net house. Remember, we are only going for one more round of multiplication than we are selling to the farmers as a seed there. So you see the number of tubers here. You see this is in Assam, Morigaon. Last seasons we got an average of 10. Each cuttings we got 10 tubers. The first generation there. The number varies from around 7 to 25, 29 there. So on average you get 10, which becomes economically very viable there. This is the number of tubers we got in Uganda, Africa. 99 tubers we got from one cuttings there. So the variation is huge there. And the economist, that's a matter of concern for me there. The variation can be from 7 to is going to close to 100 there. But as long as your average is 10, then you're okay in terms of scaling it up at the farmers level there. So if you look at the economic business perspective, as I said, if the economic does not make sense, the technology doesn't go anywhere there. So from the business perspective, I can divide into 4 stages. Once you have the tissue culture multiplication of TC lab, then you have the production of cuttings in the poly house, then you produce the first generation either in the net or in the open field, then you have the second generation in the open field there. So either one organization can take up all 4 stages, take it from tissue culture all the way to second generation and sell it in the market to the farmers, or it can be divided into different stages there. Not everybody has to do everything. You can have a tissue culture to cuttings and somebody selling cuttings, then the next enterprises take up the cutting and produce the first and second generation tubers there. So this just to look at the cost and return. If you are, this is from the numbers came from the Bangalore facility there. So you can see the cost of setting up a tissue culture lab is 15 lakhs. So we have a two poly houses, 500 square meters is 20 lakhs. So you have an upfront investment of 35 lakhs there. So if you amortize that, so you're looking at, if it's 10 years, the establishment goes for 10 years. So your amortized cost is 3.5 lakhs per year. Then these are the operating costs below you see. So you start with 20,000 tissue culture plantlets, which is cost around 3 rupees per plantlet, that's 60,000. Then from those things, you produce around 20 lakhs cuttings in one season. We are looking at a matter of window of around 2 to 3 months. If you have the window, we end up producing 20 lakhs cuttings. And the unit cost of the 20 lakhs cutting is 50 paisa. I guess the price kind of goes down if the number increases there. So this price includes the cocoa pet, nutrients, and the tray we use there. So that around 10 lakhs rupees there. The total cost, including the fixed cost upfront, is around 14 lakhs to produce the 20 lakhs cutting. If you sell 20 lakhs cutting, 1 rupees per cutting, which is very conservatively we have put, because farmers are willing to pay more than 1 rupee right now. But I think 1 rupee is a very very sensible price, I would say, the regional price, rational price there. So if you sell those 20 lakhs cuttings, so you end up making 20 lakhs rupees with a profit of around 6 lakhs rupees there. So this is a business opportunity for somebody who starts with the tissue culture plantlet and sell the cutting to the farmers and the self-help group and the farmers group there. It's a good margin in terms of there. So if somebody does not want to invest in the tissue culture, just want to get the plantlets for the tissue culture, then in the kind of a produce the cutting in the poly house, then you reduce your upfront cost. So you are just starting with the one poly house, 10 lakhs rupees with amortized cost of 1 lakh rupees. Then you start with 10,000 plantlets, which is 3 rupees is 30,000. You end up producing 10 lakhs cuttings in the seasons, which is around 5 lakhs. So your total cost is 6 lakhs 30,000, and you end up selling for 10 lakhs with a profit of 370,000 rupees there. So this is another opportunity we're looking at, convincing entrepreneurs, self-help groups to invest in this particular technology there. Then the last one is somebody who takes up the cutting and produce the first generation and the second generation seed and sell it to the farmers there. So if you start with, you buy the cuttings 1 rupee per cutting, and you have a land holding of 1 acre. So that's, you need around 40,000 cuttings per 1 acre there. That's your 40,000 rupees. Your cost of production per 1 acre is 60,000 rupees. If you're doing it open field, that's around 1 lakh rupees. Once you produce the first generation, you need a cold storage, because for the second generation multiplication, that's around 20,000 rupees. So you have 1,20,000 rupees to produce around 4 lakhs first generation tuber, which is equivalent to our mini tubers in the aeroponics, which sells around 6 rupees in the market right now. So once you have these 4 lakhs first generation tubers, you multiply that on the 10 acres next season there, because you need around 40,000 tubers per acres there. So that's cost of production 60,000 per acre. You have a 6 lakhs cost of production. Then you cold storage, you end up producing 80 tons. Very conservatively, 1 acre you end up producing around 8 tons. So 80 tons, that's cold storage cost is around 160. So your total cost after 2 seasons is around 8.8 lakhs. If you sell those 80 tons second generation seed, which is supposed to be very high quality seed, to the farmers at 20 rupees per kg. Remember, 20 rupees per kg also very conservative estimates you have put. You end up having a gross revenue of 16 lakhs there. So we are looking at 16 lakhs gross revenue cost 8.8 lakhs. So in 2 seasons, you end up making around 7 lakhs profit there. So these are the different entrepreneurship we are actually scaling up in different states now. Karnataka, Maharashtra, Odisha, Bihar, Assam, Haryana, all these places there. So this is my short presentation in terms of what we have done in this particular technology there. We had a very high level meeting with Dr. Mahapatra from ICR and Dr. A.K. Singh, DDGR, talk about this particular technology and the certification process in terms of what should be in place to make sure that the farmers get the good quality seed there, and the TC lab certification process there. All these things are happening. So this has to happen very quickly, because this technology is spreading very rapidly among the farmers self-help group, other things there. Eventually, this can be taken up by small farmers all across, from the tissue culture plantlets to producing the second generation seed there. So I'm pretty sure you'll be hearing about this technology in months and few years. Our target is to produce around 10 to 20% of the seed requirement through this technology in India in the next 5 years. Thank you very much. I hope I've stuck to the time.
So thank you very much, sir, for a wonderful presentation and an avenue for our youngsters to take up as an industry. I request you have 1 or 2 questions quickly, so I can allow the audience. Okay, sir, fine. It looks like there are no questions. Maybe they can also chat with you. Thank you very much for your nice presentation and a wonderful technology given to India.
Hello,
so thank you very much, sir.
Hello,
Yeah,
sir.
hello. Sir,
as a part of project, we are working as UHS Bagalkot as collaborating with CIP. It is emerging technology, sir. It is coming up well, actually.
Congratulations. Congratulations. Wonderful.
Thank you, Sam, sir, for presenting nice presentation and technology.
Sir, can I get his number? Can I get his number? Mobile number?
Can you spare your number, please?
Yes, let me just put it the 99371 96744. I can put it in the chat. I'll put in the chat.
Sure.
Thank you.
Thank you so much, sir.
Thank you.
I will now move on to the next speaker, Dr. C. Ashwath, a principal scientist and head division of flower and medicinal crops. So ICR IIHR Bengaluru. So he'll go to speak on the production of secondary metabolites and planting material using bioreactor systems in horticulture. Dr. Ashwath, please.
Yeah, I'm here. Okay.
Hello.
Yeah, I just shared the screen. Am I audible? Am I audible?
Yes. Audible, okay. And also screen is ready.
Okay. Shall I start?
Yes, sir.
Okay.
Sure. 25 minutes.
Okay. Good afternoon, friends. Thank you for the organizers to call me here to give a small talk on bioreactors for mass production of secondary metabolites and planting material. Probably you might have heard about Uma's lecture on bioreactor. Of course, I can say that she is an authority on use of bioreactors in banana. But in my lab, I started some more crops, which I just want to share, and overall the secondary metabolites also, I just want to touch it. Okay. Probably I want to start my presentation with a good material that we are really today, we are self-sufficient, and most of the youngsters are there, probably the students are there. It is not really, it was not a small thing to bring our production to 310 million tons and even horticulture production to 300 million tons. If you go back slightly, I just want to give a small history of our agriculture, because most of you do not know there now, because the food is not a problem. These are the things, like in 1940s, this was the scenario where around 10 million people died in Bengal famine. And we used to get, PL 480 from America. That was a situation. It was a very bad situation. Of course, India was selling books to get the wheat and the rice from the America. And during November 1963, when the PL 480 ship did not arrive to India, and it was a hue and cry for the entire India for a fruit. That time, Indira Gandhi has sent Morarji Desai. He could able to meet the cabinet secretary and requested for the ship to leave America. And in fact, he has asked for sort of 2 pairs of Kolhapur slippers. And when the Morarji Desai bent to take the, what is the size of your the leg, then the next day the paper came that the Indian external minister is touching the feet of the cabinet secretary for PL 480 ship to release. That was the humiliation which our people did, got it. So then, of course, it was a challenge for our beloved Prime Minister Indira Gandhi, and Swaminathan took over it. Borlag helped, and probably nobody knows that Dr. Subramaniam was there, C. Subramaniam was there as agriculture minister. All of them, Lal Bahadur Shastri, they all, put a lot of might, and we could able to make a sort of impact of green revolution. Probably the youngsters should know this, that all our forefathers has did a lot of work, and we have to salute them. And with that keep in mind, we have to now struggle a lot. So as food security increased, we could able to secure our country also through nuclear blast, because we had a courage to do that after once we had a food security. So things went off. Then now don't think that we have no problems. We have a lot of problems. You can see here 4.4% of world's water, 17% of human population you have to feed. So once the water is less, see, this is where I just want to pause my slide. Once the water is less, because water will be the now biggest thing, in future. Probably we have to give water and land to rice, wheat, and followed by oil seeds and sugarcane, then horticulture, fruit. Then the last is, of course, it comes to the medicinal crops. Today I am heading the division of medicinal crops. We have a lot of, this is what our Prime Minister is keep on telling, that now we have to explore. We have a lot of, biodiversity of the plants. You can see here, 7 trillion will be there by 2050. Probably we will not be there by 2050. by you youngsters, there is an ample scope to produce lot of medicinal plants and the secondary metabolites. So this is where, you know, like I just want to take you people. So 30% of drugs depends on plant, and India is the biocenter for most of the plants. And this is where you have to think about it, how to utilize them, how to take out, you know, like secondary metabolites, all those things I'm just going to tell. So probably once if you want to really touch upon this, so there is one aspect where a small aspect, it is the bioreactors, use of bioreactors. Because I stayed in South Korea for 2 years, where they completely, the ginseng which is produced by South Korea is only they are made out of bioreactors. So that means there is a provision to make a complete secondary metabolites like phytochemicals for defense. Normally, the plants produce phytochemicals for defense mechanism, and the same secondary metabolites which we are using it for our, pharmaceutical companies. So you have to now adapt. It is no way that we cannot, we can depend so many things, like we will grow medicinal plants in acres together. It is not possible. So this is one system where a small startup company should come. And that is why I know, in fact, I just want to ignite the younger minds, whether you can really do this. And in addition to this, we have worked a lot on quality planting material. This is another thing which the country is suffering a lot, because we are not giving them the proper quality planting material in many crops, because our production protocols are not proper. And this is where again we can utilize our bioreactors for quality planting material. So the moment you say bioreactor, people think that it is very expensive, millions of rupees, crores of rupees, like that. But probably in our labs, we have a small bioreactors like this. So even a startup companies like this, they can have around 100 small bioreactors of 5 liters. So if it is 5, it will be around 5,000 liters bioreactor. So you need not have, invest like this. This is of course 20,000 liter bioreactors are there, 80,000 are there, 1 lakh liter bioreactors are there, which I have seen. But there is a provision even to multiply your what I said on the secondary metabolites all through this small bioreactors also. There is a possibility. So you can just see here an advantage of bioreactors. So you can monitor everything: temperature, pH, concentration, carbon dioxide, nutrient. So if you take care of this, the plant will give whatever you want. In fact, you can just say you plant to produce this. If you probably give what is the temperature which you require, what is the pH. See, that is the beauty of the plant, where you can redirect the metabolic pathways. You can redirect the metabolic pathways in the way you want. Say, suppose if I want to, if I put a shoot, if I say plant, I want only the roots, I don't want shoots again, you can only get roots. If you say I want only the shoots, it will give only shoots. If you say I want only callus, because callus is the one which produces the metabolites, it will give only callus. But it is left to you. I mean, it is, the protocol is with you. You have to direct the plant, and this is where, like a lot of research has gone into it. Only thing we have to, reshape a little. We have to only give a smart of a cosmetic touch to this. You can just imagine a 2,000 tons or 2 tons, 2,000 liter bioreactor in 1 year. And in the same, if you want to produce 600 hectares of land for 7 years. So you can imagine 2,000 liter bioreactor in 1 year can produce the one which is almost, which can be produced in 600 hectares. So what I say that have to this type of bioreactor leave the 600 hectares to grow rice or wheat or pulses or oil seeds. So let us not occupy the fertile land for multiplication. So let us have a sort of because now India is rich. If there is no problem of power, there is no problem of anything. So it is only now we have to do it. So startup has to commit. Then you can maintain the product quality. See, especially in the medicinal crops, it is the stress which gives the metabolites. So if you don't give stress, the plants will grow, they will never give a metabolites. So what will happen? Say suppose now ashwagandha is grown near Coimbatore, in and around Coimbatore. So it requires some sort of stress. So if the stress is suddenly, say suppose today a cyclone has come, it is rain, it will never give an alkaloid at all. It will never produce an alkaloid. So that is where you can have a sort of product quality can be controlled using this, in this bioreactor, and definitely it will be free of pesticide. So these are all the advantages of bioreactors. But I'm very sorry to say that in India so far, not a single lab as a protocol has been identified by Biotech. Probably first one to say that UMA is for quality planting material and secondary metabolites. There are not many people are working on it. So probably this is the time we should work on secondary metabolites, and we can really export the whatever the material which is required. So you can see here the plant secondary metabolites which are crops which are useful. There are many fruits and vegetables which are giving whatever which we require, like anthocyanin or betalains or capsaicinoids, curcumin. All these are there in the plants and fruits and vegetables. And there are advantageous roots, even the roots. Say these are all done in the labs from 2001 to 2004. If you see, there are people who done a lot of work on extraction of metabolites from the roots, the advantageous roots. But it's all started, it stayed in the labs itself, because we have never taken it into a sort of pilot project or a commercial product. In fact, those were the days only we never thought about it go in a big way. Now probably last 5 years, if you take now under Skill India program or whatever, now a lot of startup companies have come, and this is the time we should utilize these things, and we have to really work it out. So you can see here, androgynous. Now after this COVID, now probably Kalmeg Ashwagandha is now become a darlings of many people, because they say that it will give a sort of immunity to the virus. It will give a immunity. They're like immunity booster. Probably you might have heard that you drink kashaya. I drank kashaya. I didn't get COVID like that. So these are all the medicinal crops. Many medicinal crops are the one which gives the antiviral and anti immunoboosters are there. So we have to produce whatever we require in such a way. Metabolites, say suppose ginseng, we produce it for genocides. Then Panax, we produce it for saponins. Morinda citrifolia, our noni, a miracle plant. Such a nice plant where we have not bothered to explore it. So there are a lot of plants which anti-cancer, antibacterial, the inflammatory. Many are there, but we never bothered much to work. So probably if you see here, berberine, chiconin, paclitaxel, genus, all these are they're all commercialized by the multinational companies. Even Noni, in fact, first started by the multinational company. So they sell, they extract berberine, they extract chiconin from some plants through Korean ginseng. I have seen tons and tons of Korean ginseng is produced in bioreactor, and they sell it back to us. Taxol, which is paclitaxel, Taxol, anti-diabetic drugs. See, these are all which we could have done it. Somebody else has done. Now we are buying from them. So it is the time we should, produce, and vocal is local. Our Prime Minister is saying, and skilled India, vocal is local, Atmanirbhar, all this, really, push, but somebody has to come from it. Probably we have a lab, small lab where I have a small bioreactor. I'll show you a small bioreactor where you can really think about it. So I have a paper where Dr. Peck, one from South Korea, where I worked with him. He has used up to 20,000 liters and almost around 30 plants, 30 to 40 plants he has standardized, bioreactors, advantageous routes. So one of my students worked on Vidania somnifera. It is such a wonderful plant, probably miracle plant. Now everybody wants to work on, drink a small portion of, a root extract of ashwagandha. So you can see here, just using a small 5 liters bioreactors, we could able to get a many number of roots, advantageous roots. And for our surprise, even advantageous roots has got more withanolite content than the which is grown in the field grown plants. So that means that, inside, of course, we have given some stress also inside the bioreactor. You can see here, the amount of roots grown within 1 month. It will, even 10 liters or 20 liters bioreactor, just within 2 months, entire bioreactor will be full of advantageous roots. So this is a small bioreactor. We can attach around 10 bottles. A small, this puncture shop. You might have seen the pump like this in the puncture shops. That type of, a pump. I'll play you the video afterwards. So you can see here, we can get up to 40 to 50 grams within 20 days. Just 1 gram of root, and you can get around 40 to 50 grams of roots within probably 20-25 days or within a month. So it is a very simple thing, and we just also calculated how like initially 1 gram when we put it. So we'll harvest 71 grams within 56 days in 5 liters bioreactor. So the same 71 when you put it into around 50 liters one bioreactor. So you can see here we can almost produce around 24 quintals in 56 days with 500 liters bioreactors in 10 ton. So in a room like this, a small room like this, you can have 500 liters bioreactors, 10 bioreactors, and you can produce around 20 quintals of ashwagandha roots. So this is where a lot of scope is there. Lot of amenities are there now, infrastructure is there, you will get the loans, but somebody has to start it. So you can see here, even the salicylic acid and elicitors treated, almost we got vithanolide content of total vithanolide is 6.2 mg per gram, whereas the field grown roots in the same IHR we got around 5.8. Pardon me? Yeah. So there are many like this secondary metabolites, like as I said, berberine, it is already commercial product has come. You can see here is almost in the bioreactor almost 250 times it has increased. That is how Japan is selling the berberine the product metabolites. So from the Coptis japonica. So this is another one you can see here. So if you do a small cosmetic touch with the research which what they have done, even we may increase up to 300 times from the normal root. So this is the one which grows, outside. So if you just bring one root, then you start producing the advantageous roots. And as I said, Korean ginseng, it is really, terrible, every Korean will take at least 10 to 15 grams of, ginseng, this one every day. And you can see here, 10,000 liters bioreactor with an average production of 45 tons per year. So you can imagine 45 tons per year can be produced. Normally, one person will take around 10 to 15 grams maximum, or 10 grams, 5 to 10 grams. So you can imagine entire thing when they are Koreans, they are growing it in the bioreactor. In ashwagandha also, why don't we do it in the same way? That is my, in fact, dream. If some companies comes, definitely we give a protocol. We have standardized it up to 5 liters. So if somebody, comes, even 50 liters, 100 liters, we just want to start also. So it is a simple bioreactors, as I said, you need not have, big, a 5 liters bioreactor, 10 liters bioreactor. Only thing you have to have some sort of, a system where you can produce tons. This you can see here, the ginseng is all produced in bioreactors. Not a single root has been grown outside. So you can see even the production cost yield. The field cultivated ginseng 523, here it is 30,000 kgs per 0.1 hectare. Whereas the production cost is slightly high because they have taken the production cost of the bioreactors also. Otherwise, you see the yield here and see the yield here. Advantageous root obtained from bioreactor cultivation for 0.1 hectare. If you calculate, it will be around, see there, that's what they have given. 10,000 liters bioreactor for 45 days, operated for 77. You can get this much. Where is 523 and where is 30,000? So this is what, we have to explore it in a proper way. There are some plants, purpurea, the caffeic acid, which is a very, very costly material which is available in market now for a pharmaceutical. You can see here, almost they got around 26.64 mg per gram for dry weight, which is very, very high. They are using the elicitors. So there is one more like hypercium, that's called St. John's wort. We don't get really these days, very difficult to get this type of metabolites in the market. So you can see here almost 50 times it is increased. Either you can go for research also to increase 500 times, to increase to 200 times, 300 times. What elicitors you have to use, how much oxygen you have to increase in the bioreactors. Either you can even take up the research, or you can even with the small labs, you can start multiplying. That is where most of the colleges I visit and I tell the college authorities, kindly do this type of small, like bioreactors. At least the person who comes out of 1 person out of 100, if he picks up, probably India will be rich with so many, metabolites where we can export to the entire world. So Taxol, which is another big story, probably you can see here it's almost required 250 kg just in the US. It's almost, we had to take out the bark of 360,000 trees per year. So which is almost $200,000 Taxol. So you can produce this tactol in tissue culture bark using, like the bioreactors, or you can even the fungus also. There is one small fungus grows, on the tree bark. That also, if you can, grow it in a bioreactor, and alternative species, like as I said, you can just use this alternate species like Taxus baccata, where you can grow this, make the callus, extract the taxol out about it, and imagine, like 1 kg taxol around $200,000. So there is a huge market. There is nothing like that. There is no market. But only thing, people are not taking it. So artemisian now is a big, it is not available in world. It has become, very, very rare because, especially treatment for cerebellar malaria. You can see here, almost 23.02 dry weight artemisinin can accumulation in 1.1 mg, equivalent to this one. So it is very difficult. If you want to take out this from the plant, you have to grow almost 1 year plant with a, very difficult to grow also. So this is another thing you can see here within 28 days, how many roots this artemisian plant has given it in the bioreactor. A small bioreactor, it just started and it will be full of roots. So for our imagination, we never thought that a small advantage roots will have this. Now, in fact, now we have one of my student is working on extraction of lutein product, lutein out of the marigold petals. Otherwise, see, marigold petals, you have to go in 10,000, 20,000 acres to get 1 kg of lutein. So instead of that, why don't we grow, petals inside, you make a callus and extract the lutein out of the marigold petal. So one of my PhD students is working. So there is, there is a wide array of, everything is there. Only thing we have to put our hand. The production of advantageous roots, see here in Gynora procumbens, almost 50 folds it is increased. All are cancer. See, cancer probably, if you see, every alternate research in the world, alternate person who is working on something on research in life sciences is working on cancer. So if we get probably this type of metabolites, you can really, there is no place where there is nothing like we can say that this drug I don't require, because for cancer, everything is possible, to reduce the cancer. So there are many plants. So I have listed a lot of plants, where we can produce the advantageous roots, and using a lot of solicitors, and we can increase from 15, 30 to 250 times more than what the plant produces in the natural environment. So there are a lot of papers where I collected the information. It is really mind-boggling that whether it is possible, like a person can start, exploring it. Say, suppose now, you can explore the entire world that these are the things which I want to sell it. Definitely the people will come and salute you and touch your feet to take these products. Catharanthus roses, such an easy plant where a pilot scale agemenicin they have extracted out of this. You can see the roots. Within no time, the roots, they're able to get it in the Catharanthus roses. You can see here, just simple, like in the bioreactors also, 4 or 5 types are there. So what we use it is bubble gum. Otherwise, there are many bioreactors. This type of bioreactors, only the nutrients will be sprayed from the top. It will just spray and the root starts producing it. So these are advantageous roots. Then many plants, even there is a secondary metabolite from the organ cultures. As I said in marigold, say suppose now you culture the marigold petal and you can get the lutein out of it. So like that, Fritillaria species where they could able to get from the bulb. The bulb they could able to get it from the bioreactor, and the alkaloids has been extracted from the bulb. So from bulb they got it, and bulbs are produced in the bioreactors. So there are as I said,
sir, can you sir try to complete, sir?
Yeah. If you want me to stop, I stop here only. Shall I stop?
Complete madi, sir. Try to complete.
Okay. I have okay. See, if that is a problem with seminars. Okay. Factors affecting the root. There are many factors which affect the multiplication of advantage roots. So these are the factors. Okay. elicitation is the one which is very important. I have a lot of other slides where we worked, where these are the problems where, the low cell productivity, culture parameter, genetic instability, slow growth. These are the problems where research students can also take up. There are, of course, other than the secondary metabolites, we have worked a lot on the production of secondary material, the planting material also, which your organizers don't want to give me. So I just go very fast. So this is oil palm, then Chinese, then strawberry, of course, in banana, probably Uma might have told. Then papaya multiplication, Dr. Payas might have told. Papaya already you have done. Potato. So this is in our lab. We could able to multiply the anthurium in a large number using the bioreactors, and then we have even the Cadila is buying this technology, and we have standardized the phalaenopsis. We have standardized the lilium bulbs. Otherwise, lilium bulbs every year 1 million bulbs are imported. So we could able to standardize the lilium bulbs in here lilium bulbs. We have made a sort of commercial protocol. So then chrysanthemum we could able to make gerbera. So there are, for youngsters, there are a lot of path to ahead and a lot of style improvements. Then either you can do research or you can even go for a commercial product. So if you want to really change, it is not easy, but take a chance to change and get it done. So I feel my the PowerPoint is will well fit into our beloved Prime Minister's Skill India program, where you can really take up vocal for local, because most of our medicinal plants are local. Then Atmanirbhar, now we have to start exporting and rather stop the importing, and that is what is. Thank you very much. I have no time.
Sir.
Yeah.
Ashwath sir, thank you for a wonderful presentation. And
give time, sir. You ask us.
Sorry.
No. So I am very sorry. I am very sorry to say that this is the type of seminars where we have to ignite people. Just for a namesake, don't do seminars, please. See, we have to allow.
Okay, I'll communicate to the organizer.
I prepared this for last 15 days. You asked me to just scroll slides within 10 minutes or 15 minutes. It is not.
No. You have been given 25 minutes, sir. You crossed it. Thank you, sir. Thank you for a nice presentation, and especially the challenges in terms of secondary metabolites you have highlighted. And definitely, again, it's another area where you can take it to the level of industry. So thank you very much, sir. Any questions to the speaker?
Sir, am I audible?
Yeah.
Ashwath sir, thank you very much for such a wonderful presentation. A quick question to you. You have a quantum of knowledge in bioreactor-based phytochemical production. So is it possible to produce lipoproteins or fatty acids through bioreactors? Maybe using algae or something.
All these days, I mean, like we have worked on more of using the plant, either one of the plant part, like a leaf or stem or a root. Okay, where the already secondary metabolites are there. Secondary metabolites are the one which plant probably one to one we can discuss. I'll come back to you. Okay.
So thank you, sir. Thank you very much, sir, for your nice presentation. So I call upon the next speaker, Dr. N. Bharati. So she'll be talking from Bamboo, scientist and founder director, so Grow More Biotech. So she'll be speaking on advances in micropropagation of bamboo as a case study for crop improvement and sustainable productivity.
It's he, sir, not she.
He, sorry, okay, sorry. He, okay, sorry. My apologies for that. So I request Dr. N. Bharati, please.
Sir, most probably is not there.
Okay.
So you can go ahead.
Fine. So the Dr. N. Bharati is not available. So we can now go to the oral presentation session, where we have totally 7 oral presenters are there. So I'll call upon the first speaker, Dr. Santosh Savardekar, Professor and in charge Biotechnology Center, Dr. B.S. Konkan Krishi Vidyapeeth, Dapoli, and he'll be speaking on standardization of callus development technique in Sarpagandha for isolation of secondary metabolites. Sir, you have 10 minutes presentation, so kindly reduce your introduction part. Thank you.
Sir, good afternoon, all of you. Sir, myself, Dr. Savard from Konkan Krishi Vidyapeeth. So now I am speaking with the standard on the callus development technique in the Sarpagandha, that is Raulpia serpentina for isolation of the secondary metabolites. Sir, sir, may I audible?
Yes, sir. Yes, sir. You go ahead, sir.
Hello.
Yes, sir. We can hear you,
You're
sir.
audible,
Yes.
sir.
Audible.
Audible. Shades are also visible.
Yes, sir. Yes, sir. Thank you. Thank you. So, if you see the particularly in it is called as the Sarpagandha. So, right now we have heard about the Ashwagandha from Dr. Aswasya, but we are trying to develop some protocol for the metabolite. As far as this Raulpia serpentina is concerned, it is important position in the pharmaceutical world, and because of this immense anti-hypertensive properties, and it is having a rich serpanido in the oleoresin fraction of the roots. So this another thing, this Raulpia serpentina is written as extinction in the India because of its indiscriminate collection, and for the collection is by tribal peoples, particularly from of the Maharashtra region. So if you want to protect it, so there is a need to develop. First one is to develop that protocol so that we can able to protect the endangered species. So our objective study to study and the regeneration technique in the Sarpagandha, that is Raulpia serpentina. And another one is that is the qualitative analysis and the quantitative estimation. So I'm directly switching over the so bring all this material from the outside. There is a need to standardize the surface sterilization technique. So that we have observed, we have taken the number of events, and among this, the carbon as in at 0.1% for the 10 minutes, and 36% 5 minutes. We have developed near about 4% of septic culture. We have taken that and we observed that the medium have the highest culture for the 95%, followed by the lip disc and the leaf node. So there are some of the another thing is that the it is also found significant as far as the lip disc is concerned. If you see the frequency of the callus with the different combinations, DAP, they have produced the maximum percent of the callus that there are the and follow series. So you see in this table, if you increase the concentration of the 2,4-D, we have after that, so they are decreasing in the frequency of this that endogenous level is the most important we observed in this study. So the average number of the days, because it is also important because the first generation was important, and I observed that near about 15 or 14 days, we can say we have got the size of the no, and this next step have produced lithium produced the maximum it has produced the 14 as compared to the last. So the weight of the callus after the 21 days number of the callus that is near about the 2.06 grams other, and in the mean of all these three, we observed that 189 so if nature has the variety callus, the green so that the callus is important. So we have tried to develop, we have been given the consent and
I think Dr. Santosh got disconnected because of internet issue.
Yeah, what has happened to the speaker? Some problem?
He might have got some disturbance in the internet, sir.
Okay.
Sir, you can call upon next person, next speaker, sir.
Sure. Yeah. I'm sorry, Santosh, I think. Hello?
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 Hello, may I audible?
Yes, he has come
You're
back.
audible. The point is your slides are not visible.
Sir, sorry, because of the connectivity
Okay,
problem.
okay, fine.
Yeah,
Sure.
because of the connectivity problem.
Sure, sure. Continue, please.
Yeah. So we got this security. So the secondary metabolic. So we observed there is the
Sir, once again he got disconnected.
Okay, I think Dr. Prabhuling is ready. So
yes,
can
sir.
I request him?
Yes, sir. Yes, sir.
Right. Yeah, sir, I request you to kindly, you can start presentation. I will inform the audience that Dr. Prabhuling G, Professor Biotechnology, College of Horticulture, Bagalkot. So he is going to speak on direct organogenesis in banana and cultivar Rajapuri Vale, that is Musa species. So I request him, sir, you can cut down your introduction part and you have 10 minutes.
Okay.
Prabhuling,
Please.
sir, something else got into screen. Kindly check it. It is your slide or what?
No, it's not his slide, sir. It's only a schedule. Okay. Sir, you can start.
Okay. Slides are coming now, sir?
Yeah, certainly.
Yes, sir.
You're audible also.
Yes, sir. Yes, sir. Sir, good afternoon, Chairman, sir, and Co-Chairman, sir, and my dear delegates. I'm here to present my research findings on diet organogenesis in banana cultivar Rajapuri Vale. No, banana basically is a triploid seedless. Most of the edible bananas are parthenocarpic in nature, and as Uma Madam was mentioning, there were about 20 commercial cultivars available in India. So one of the important cultivar is Rajapuri Vale, which is very popular in our northern part of Karnataka, especially in Belgaum, Balkot, and Bijapur district. This is very hardy crop, and it withstand very well to the wind damage. And it fetches a very premium price in the market because of its very good blend of a sweet and acid taste. In fact, upon ripening, it gives very skin turn to golden yellow color, very attractive color. So almost it fetches 3 times more the price than the Grand 9 variety, and almost the prices are on par with the Eleki Bade or other local cultivars. Banana is the most intensively micropropagated crop rather than any other one. In commercial, because of the various advantages associated with the micropropagation, banana is commercially multiplied through tissue culture using a shoot tip culture method. This method ensures the genetic stability of the plant. In tissue culture, the plantlet produced should be two type, and they should preserve all the original characteristics of the varieties. So normally, commercial tissue culture relies upon the shoot tip culture. Unfortunately, this system is unable in accumulator group like Grand 9 and Red Banana. Most of the local cultivars of banana, including Rajapuri, they are highly recalcitrant to tissue culture through shoot tip culture. These are different series of micropropagation involved in the shoot tip culture, where you can see very few number of shoots induced, hardly 2 or 3 maximum here. So this is one of the major problem. Even though there is a lot of demand for this elite cultivar, planting material for this elite cultivar, there is a very less, or you can say no availability of the tissue culture plants in this variety, in this cultivar. So as I said, the major problem why the companies, tissue culture companies are not multiplying, very few companies are involved in it, is because of the recalcitrant nature of this variety. Less number of shoots are produced, and there's a more problem of contamination, difficulty in excision, and so many other problems associated with this method. So in fact, we tried to optimize the protocol through shoot tip culture in the earlier our studies, and we could able to produce about 3 shoots in Rajapuri Bade itself using the so-called highly effective hormone TTZ at 0.2 milligram per liter. We're able to increase only three number per explant. And in another study also about 3 shoots here also with the BAP level of 4 mg per liter in the same cultivar. So these results clearly indicate that it's highly recalcitrant with the traditional conventional method of tissue culture using shoot tip culture. And we try to find out the literature whether there are any other available options, alternative are there or not. We found that alternative to shoot tip culture, male flowers, we say male flower birds, can be a potential source of explant. So in banana, in the inflorescence paradix, we found these male flowers towards the terminal part of the inflorescence. These male flowers are located in the axils of the bracts, each bracts. If you see the structure of the male flowers here, this is the individual flower. These are the individual flowers. You can see the anthers, filaments, and these anthers, no, they are devoid of the viable pollens. They are sterile. And this basal portion, it is receptacle. And these flowers are placed in the cluster. So this is the bract. In the axil, you can see the location of the male flower. These are in the cluster, placed in the cluster. So these are called male flower birds. So for commercial tissue culture, for usage as these as explant, these are scooped out from the peduncle, and they are cultured on the media. Here you can see. So these are the upper parts which withered and almost dried. They are the anthers and the filaments. The basal portion which you are seeing, which is turning now green and bulging. So that is the receptacle part from which we get the aseptic culture. And there are a lot of advantages are associated with this novel method of male flower culture, male flower bud culture. Here we can break the recalcitrancy and produce large number of the plants here from each inflorescence. And the contamination problem is very, very less in this case. And these are very easy to acquire because in most of the cases, after the complete formation of the hands, farmers will discard them and throw them away. So instead of wasting, they can be used for a tissue culture process, and transportation is also very easy because tissue culture industry needs these suckers in conventional method in thousands. So transportation is very difficult from one place to another place, and you can easily remove it from the mother plants sterilization process is very, very minimum here in this male flower, because this is in the aerial, in the air portion, unlike the suckers which are attached to the soil, which carries a lot of contaminants. So there's less harm to the environment also if you use the male bud flowers instead of suckers, because the process of sterilization is very simple, very, very simple. We use only few chemicals. So it's safety to very less harm to the environment. So a lot of advantages are associated with this male flower bud cultures. And these are the different steps involved in the male flower. You can see here, as Madam was mentioning, Uma Madam. So these are the male flowers, flower bud we say. These are excised from these axils. At the axils there will be, axil of the bracts there will be a male flower bud. These will be scooped out, and they will be cultured on the media. So which produces the buds directly on the explant, and which these buds we say the adventitious buds directly induced on the explant, and they produce a shoot directly without any intervening callus. So you can see the huge number of shoot produced here, by using a male flower culture technique, we have three ways of regeneration: direct regeneration, indirect organogenesis, and somatic embryogenesis. See, for a commercial tissue culture, we always rely upon the protocol which ensures stability, besides the good number of plant which yields. So here, besides the shoot tip culture, the best regeneration pathway which we can adopt here is direct organogenesis, as it ensures stability besides increasing the number of plants. So here you can see these birds are directly induced on the explant here. Histological study reveals that the protrusion of the epidermal layer resulting in the formation of the birds here you can see. So these birds are induced directly on the explant without any intervening callus phase. That is very, very important in commercial tissue culture, which ensures stability of the plant. You can see huge number of these shoots coming out from the explant. Okay, this is the indirect organogenesis where it involves callus formation. Of course, there is a large number of plants can be produced, but stability of the plant is some question is always there here. And another method, very efficient and highly rapid method, is the embryogenesis. Here you can produce lakhs of plants, but here the stability of the plant is also is questionable, and most of our local cultivars are not amenable to these somatic embryogenesis, as Madam was mentioning in her speech. So we resorted to the next best option of regeneration, that is direct organogenesis, which involves different stages. These are the different steps involved in the direct tissue culture multiplication through male bud culture, where the birds are induced directly on the explants. These birds we called as protocomb-like bodies, or PLBs, or cauliflower-like bodies, which grows into shoots directly without intervening callus or any embryo formation. So here, first in our study, we try to optimize the protocol for standardization of the establishment of the aseptic culture. So we isolated the male flowers, the inflorescence paddocks, and we scooped out the male flowers, male flower buds, which are of uniform size 3 centimeter, and we transferred them onto the multiplication medium, fortified with different levels of BAP. All these media were also supplied with NaA 1 milligram per liter, and also tender coconut water at the rate of 150 ml per liter. So we could see here with media containing 5 milligram per liter BAP with the coconut water 150 ml per liter, resulted in a very higher level of establishment of aseptic culture, almost 100% you can say, and even very high in case of 4 milligram per liter BAP also. And we could see also the better greening and bulging of the explant, and also less intensity of browning with this treatment. And of course, there is no contamination problem involved with this treatment, and also in other treatment also the contamination is very, very meager. So this is what most desirable parameter we look into while establishment of aseptic culture. Here you can see this is the treatment, 5 mg per liter. You can see the very good bulging and greening of the explant here. So this is the most ideal treatment for inducing the establishment of aseptic culture. See, when you isolate the male bud from the inflorescence, we come across different sizes of the explant. Say, normally when you begin to isolate, we start with, say, when we isolate, first one will be around very bigger, about 5 centimeter, then 4 centimeter in length, then 3, 2, 1, like that. Further, we cannot isolate beyond this because it's very difficult to see them. So this is last remaining will be this whole inflorescence of 5 centimeter size. And this is before excision of these male birds, this is how it looks. So we cultured all these explants to see the effect on the establishment of perceptive culture. And we have seen that the explant of the size of 3 to 4 centimeters in length. So these two, and of course even 1 centimeter also. And this whole inflorescence, we have 5 centimeter size, were found very good in terms of establishment of perceptive culture, beginning bulging, and also lesser browning intensity. Of course, the contamination is also meager, very meager in this. Because these they are placed deep inside the inflorescence, so chance of entry of the bacteria or fungus is very less here. So that's why we are encountering very less contamination, which is a major problem in case of shoot tip culture. And this is one of the major advantages of the male flower culture technology, besides enhancing the shoot proliferation. So here you can see the very good see the growth of the basal pores and receptacle. What we see, this upper portion dried the filament and anthers, which we remove, excise and remove, and we take this basal part only. So here you can see good establishment of perceptive culture in an intact inflorescence of 5 centimeter size. So these 4 centimeter, 3 centimeter, even 2 centimeter, 1 centimeter also, they are ideal for culturing for established perceptive culture. From each male flower, we get male inflorescence, we get about 15 to 20 such culturable explants, which yields very good perceptive culture. So
time, you please look at the time.
Yes, sir. So these established perceptive culture were transferred onto the proliferation medium. We were able to see the induction of birds at early time, and also good number of birds and a higher number of shoot with BAP 4 mg per liter, along with NAA and also coconut water. And we could also see other better growth of the shoot and also number of leaves with this treatment. And also the thickness of shoot is very important when we take them for hardening. So better thickness of the shoot was also good with 4 mg BAP per liter, and we could not see, this is a desired one. We not observed any callus induction, that is most desired. Birds were induced directly into the explant without intervening callus. And see here, these are the higher rate of proliferation. You can see this 4 mg, then also 5 mg BAP per liter, very good proliferation, and almost the proliferation rate is almost 12 on an average, 10 to 12 per explant, almost 3 to 4 times higher than the shoot tip culture method. And after complete shoot formation, weeks, which produce root as well on the same media, very good root formation with the higher number of roots and length of the roots, and plantlets were normally they phenotypically they're normal. So these are the regional plantlets which is just separated from the clump. So you can see the plantlet development was very normal without any abnormal. These small plantlet is because of the difference in the treatment, which turned normal after hardening. See, always there is a question when we use male flowers. Many questions are intact. What will be the ploidy level? So we check the ploidy level, and the ploidy level of our test plants obtained through the male bud culture were triploid as that of the mother plant. So there is no problem of the ploidy level, even though we use the male flower, because these are basically sterile flowers, and regeneration occurs from the basal part of the explant. And previous studies also indicated that there was no variation not observed among the plants originated from the male flower bud culture. And we planted them in the farmer's field and observed a very good desirable phenotypic, the vegetative as well as reproductive growth. And you can see the very good formation of the bunch and also fingers, almost 12 to 15 kg each plant bunches were weighing. They are all normal. So there is no issue of any variation or the change in the ploidy level. So with this technique, we are able to increase the multiplication by 3 to 4 fold almost. So this technology can be used by entrepreneurs, small entrepreneurs who are having the production capacity of 1 lakh or 2 lakh per annum, because they cannot go for the suspension culture or other methods which are very high end and costly. So of course, with this technology, we can scale up the production and we can make available the tissue culture plants available to the farmers in these local cultivars. But still there is a scope for enhancing the multiplication through, as Madam was mentioning, through indirect organogenesis or embryogenesis, which we need to work. We are working on that. While dealing with that, we need to consider the risk of somoclonal variation, as it is chance of getting the variation in these methods. If this protocol is optimized through these high-end somatic embryogenesis in our local banana, really we can revolutionize the tissue culture production and banana production in our state or in other local cultivars also. I'm very much thankful to the funding agency. In fact, the KBITS, now it is called KITS, Department of Science and Technology, Government of Karnataka, for funding. It is in fact a huge fund we got. It is one of the experiment in that project. We are thankful to Genome Bio, Pune, who analyzed our samples for FlyD testing. And I'm very much thankful to my MSc student, Ajmal Habibi, who carried out this work. And I'm thankful to the advisory members, Prakash, Raghavendra Babu, and Tika Chitapur for their help. Thank you, sir.
Yeah, my sincere thanks to Dr. Prabhulinga. A wonderful technology you have developed and wonderful way of looking banana tissue culture. If any questions, quickly, one or two questions. If you have any, audience, please. Okay, sir. Thank you so much.
Thank you, sir.
Next, yes, sir. Next, I call upon Dr. Achyut Barrows, Associate Professor, VD College of Agriculture, Biotechnology, Latur. So he's going to speak on in vitro regeneration of pomegranate, especially cultivar the Bhagava from different explants. Hello? No.
Sir, hello.
Some problem with link.
Yes, now you select your PPT, sir.
Fine. Sir, you will be given 10 minutes. So try to reduce your introduction part. You can spend more on your materials method and also technical things.
Good afternoon, everybody. Myself, Dr. Bharat Singh, Associate Professor from Vasantara Naik Agricultural University, Parbhani, Maharashtra. I'm audible?
Yes, sir, you are audible,
Today,
but
I will
slides are
talk
not
about the in vitro
open.
regeneration.
Hello, slides not yet shared. No.
Is it not open?
No.
Just shared.
Not yet,
Okay,
sir.
just a minute, sir. Just a minute. Now?
Not yet, sir.
Now is it?
No, sir, we are not able to.
Not yet?
No, sir.
Resume sharing.
I think it's uploading. It's under the process of opening.
Otherwise, you give some other person a chance, sir. Later, let him present. That would be better, actually.
Okay, fine. Again, I'll come back to Dr. Achyuta in case if you're ready afterwards. I'll call upon Dr. S.K. Ranjitam, Associate Professor, Department of Plant Pathology, TNU Coimbatore. So we'll be speaking on in vitro bacterialization of micropropagated banana plantlets with enhanced resistance to. Hello?
Hello, sir.
Manoranjitam?
I think Achyut Barros, sir.
Yeah, Achyut Barros, right. You ready?
Next speaker.
He is Manoranjitam.
He's available, sir. Is
Yeah,
there
ask him
in
to load. It's the case.
Dr. S.K. Manoranjitam.
You are available?
Sir, hello. Dr. S.K. Manoranjitam.
Okay,
Then
we will
we
move
can,
on to the next person. Yogesh Bhagat.
Yes, sir, I'm there, sir. Shall I share the screen?
Fine. Yeah, great. So for the sake of audience, Dr. Yogesh Bhagat is Assistant Professor, VD College of Agriculture, in Biotechnology, Latur. Going to speak on the establishment of an efficient callus induction system for anther and gynoecium culture of pigeon pea, Kejanus kejan. Please, sir. Again, 10 minutes.
Am I
Try
audible,
to
sir?
introduction part, sir.
yes, sir. We can hear you, sir.
Okay, sir. Am I?
Continue. Yes, sir.
Sure.
Okay. All of you, respected chairman, co-chairman, reporters, and organizing secretary, Dr. Fakhruddin sir, for giving me a chance to present in this conference. So I have to raise the establishment of an efficient callus induction system for anther and gynoecium culture of pigeon pea. So just I will introduce you, just have a brief introduction. So here we can see the pigeon pea, as all of you know, that it is an important legume crop and it is often cross-pollinated crop. So that the maintaining the homozygote for this, and has chromosome number 22, genome size is around 858 megabase pairs. And as far as concerned, so it is the second important pulse crop cultivated next to the pigeon pea, and it is mostly in the rain-fed region. And it has an cultivable area that is around 3.88 million hectare with production of 2.84 million tons, and productivity is 7 to 8 quintal per hectare. And the most important thing is that India is the largest producer as well as consumer in the world for the pigeon pea. So you can see here the area, production, and activity of pigeon pea for the last 60 years. You see the blue line that is indicate the cultivable area. So from last 60 years, the pigeon pea, the cultivable area under the pigeon pea is getting increased, slightly getting increased. Similarly, the production is also slightly getting since last 60 years. But if you see the yield, that is the productivity is still stagnant. So we are having a chance to increase the productivity of pigeon pea. So here we can see the haploids. So haploids, as you know, they are plants which are having the gamete chromosome number. Then this potential of haploidy for the plant breeding, it is arose in 1964 when Guha and Maheshwari, they have successfully done the in vitro culture of Datura anthers. And these haploids for plants, they originate from the gametes or gametes like cell, and they can be referred as a maternal or paternal haploids. If the haploid plants has been generated from the gynoecium or ovule or ovary, then we can call them as an gynogenic haploids. Whereas if the haploid plants are generated from the anthers or pollen, then we can call them as an androgenic haploids. So what are the implications of this anther or gynoecium culture? So the anther and gynoecium culture are very, very much useful to get the haploids or double haploids, and that can be widely used for the cytogenetic studies like monosomics, nullisomics, and aneuploid stocks. They are also useful for the identification of the dominant and recessive mutations, and also to study the various factors that control the pollen or pollen embryogenesis. And they can be also used, this anther and gynoecium can be also used to develop and double haploid lines, then which can be used as a mapping population, and which is important part for the functional genomics research, for the QTL mapping as well. And these haploids are also useful for transferring the alien genes. Then we can also convert these haploids to the double haploids, and these double haploid lines can be used as a cultivar. So this is the implications of the anther and gynoecium cultures. And if we see the previous efforts for these anther and gynoecium cultures in pigeon pea, there is paucity of information for that. Only 4 scientists has tried, and they also not get that much success. So here we can see the Bajaj in 1980, then Foggart in 1992, then Vishikumar in 2000. So they have tried the anther culture of pigeon pea, but they have not got that much success. So with this background, the objective of the study were framed as to optimize the media with different hormone concentration to induce the callus from anther and gynoecium in pigeon pea genotypes, that is BDN 711 and BDN 716, and also to study the effect of various genotypes and culture media on callus induction. So coming to the materials and methods, so let's see the materials. So in this study, we have used the 2 popular pigeon pea varieties, that is BDN 711 and BDN 716, which has been developed by Pulse Research Station of our university, that is located at Badnapur. And these 2 varieties are resistant to the fusarium wilt as well as sterility mosaic disease. So, and we took the seeds from them, and we have raised the pigeon pea plants from that of that variety in the aseptic condition. Then the unopened flower buds of that aseptically maintained plants we have collected, and we have sterilized it with the mercury chloride, and then we have followed by the washes of sterile distilled water. Then these explants such as anther and gynoecium, they are excised from that unopened flower buds, and they have kept in the callus induction medium, which has been supplemented with the various concentration of the plant growth hormones. And finally, these cultures were maintained at an temperature 25 degrees Celsius with relative humidity of 60% and 16 hours of photoperiod. So this is the medium we have used. We have used the anther and gynoecium culture. So we have used the combination of the auxin and cytokinins, and we have used the combination of auxin and cytokinins like 2,4-D and BAP, then 2,4-D and kinetin, and also the NAA and BAP. So let's see the methodology. So here the anthers were kept on the medium, different 12 treatments that we have MS medium containing the various different concentrations of the growth hormones. Then the gynoecium cultures. Then the gynoecium has taken from these 2 different varieties of pigeon pea, popular variety of pigeon pea, and we kept it in MS medium supplied with the various concentration of the plant growth hormones. And let's see the results, that is the effect of growth hormones on the callus induction from the anther explants of pigeon pea genotypes. So here we can see the MSC4 treatment that contains the 2,4-D at 2,4-D 1 milligram per liter, and the 2 milligram per liter, and BAP at the rate of 2 milligram per liter. So it has shown the promising results. So here we can see around 11.3% of callousing anthers has been obtained in MS4 treatment in case of BDN 711. Similarly, in case of BDN 716, 90.7% of the callousing anthers has been obtained. And if you look at the all treatment, so the MS4 treatment that is having the 2,4-D and BAP, it is given the good response, that means significantly good response in both the irrespective of the genotypes. So that we can suggest that the MS4 treatment is very well that has the 2,4-D concentration at the rate of 1 milligram per liter, and BAP concentration is at the rate of 2 milligram per liter. And we also did the t-test independent analysis. So for that, after the analysis, we have also seen that BDN 716 has given the significantly higher response as compared to the BDN 711 for the anther culture. Then similarly, here we can see the MS4 media containing the 2,4-D and BAP, that is 2,4-D at the rate of 1 milligram per liter and BAP at the rate of 2 milligram per liter. So they have given the significantly higher response in case of BDN 711 as well as BDN 716. Similarly, we have also tried the gynosia culture, and in the gynosia culture also we have used the two. Gynosia has been obtained from the two different pigeon pea cultivars, and they have kept on the media containing the different growth hormones. So MS4 treatments are again have shown the promising results with the 58% of the callousing anthers, and the MS3 treatment that also contain the 2,4-D and BAP, which has shown the 50% of the callousing anthers. And when we did the analysis, when we compared the two genotype, that is BDN 711 and BDN 716, so we couldn't find the significantly, they couldn't differ significantly for the gynosia culture. So here we can see the MS4 medium that contains the 2,4-D and BAP, which is giving the good response, and MS3 media that contains the 2,4-D and BAP at the rate of 1 milligram per liter. So which has also shown the significant results. Later on, we have also analyzed the morphological characteristics of the calli. So when we where we have studied the texture of the calli, size of the calli, and color of the calli. So here we can see the MS4 treatment, which contains the 2,4-D and BAP, which has shown the compact calli. The callus size was significantly higher in case of BDN 711 and BDN 716, and color was the greenish, so it has the chlorophyll. So this MS4 treatments again has shown the good response. And so finally, coming to the conclusion, so I can conclude that whatever the sterilization procedure we have followed, we didn't get any contaminations. Similarly, MS4 treatment that contains the 2,4-D and BAP MS4 treatment that is at the rate of 1 milligram has shown the significantly better results, and this haploid lines can be converted to the double haploid lines, and that can be a that can be used as an inbreed lines for the development of hybrids. And this haploids and double haploids will have the immense value in the pigeon pea breeding in reducing the time required for developing inbreed lines. And I'm very happy to say that, and this is the first report for the gynosia culture of the pigeon pea. In case of BDN 7, by taking the cultivars BDN 711 and BDN 176, which are resistant to the fusarium and sterility mosaic diseases. And later on, these double haploid lines can be used as an cultivars, so that and they can be used as a genetic stocks to study the various. That means they can use as an uneuploid stock to do the various cytogenetic study. So with this, I'm very much thankful for the DBT department. Grant to carry out this research at our plant. Thank you to the organizer for giving me this short time for presentation of this work. Very much presentation of this work. So thank you very much.
Sir, thank you very much for a wonderful technique that you have developed about using the gynoecium and also andrecium culture in pigeon pea. So next I call upon the speaker, Dr. Shiviyogi Ravalada.
Yes, sir.
Professor CDV, UHS Bagalkot.
Sir, small for sir.
Echo ಆಗ್ತಾ ಇದೆ. Echo ಆಗ್ತಾ ಇದೆ.
Lokesh.
Sir, madam.
Sir, SK Manoranjitam, she wanted to.
Fine. Okay. Sorry. Okay. I'll come back to you, madam. You can. I can call upon Dr. SK Manoranjitam, Associate Professor.
Sir.
Department of Plant Pathology, TNAU Coimbatore. she'll be speaking on in vitro
Hello.
bacterialization
Am I
of
audible,
micropropagated bacteria.
Madam.
Hello?
Yeah, madam,
Yes,
please.
madam.
Am I audible?
Yes, madam.
Yeah,
We
you're
can
audible,
hear
madam.
you, madam. Wait for a while, madam. Sir is giving introduction.
Okay. So she'll be speaking on in vitro bacterialization of micropropagated banana plantlets with enhanced resistance to a particular fungus. Okay, madam, please. Hello?
Yeah.
Hello?
Good afternoon, sir. Respected
Yeah.
chairman. Sir, please share my slide. Madam, please share my slides. Madam, please share my slides.
Yes, madam. I am opening. Madam, it has come. Just check.
No, it's not mine.
We're not able.
Somebody else.
No, this is
It's not
next.
mine, madam.
Oh, okay. Sorry, madam.
Next speakers.
Sorry. I'm sharing. Now, are you able to see this?
No, madam, not yet. Not yet.
It's shared,
Or
madam.
can I share?
Not yet, madam. Slides are not coming.
Now we are able to see here, madam. You are not able to see this?
No, it's not visible here.
Not me also, madam.
It's shared on. Madam, can you try it from your end? Madam, please.
Okay, I will try, madam.
Madam, wait.
Visible now?
Yeah, visible,
Yeah,
madam.
it's visible now for me, but it is not in.
It is visible, visible.
Praveen?
Madam, it has come to screen. Just check, madam.
Please shift to. Yeah. Can you start?
Sharing is from my side. I did share it. I only shared it.
Okay,
I will move on.
But it is not in. Okay. I can able to see it.
Yes,
please change to slide share mode.
Yes, it has been changed, madam. Just check.
Okay. Yes. So good afternoon, respected chairman, co-chairman, and my topic is on bacterialization of micro-propagated banana plantlet with enhanced resistance to bacterium, that is Pectobacterium carotororum. Next. Not moving. Madam, it's not moving. Yeah, please. So Erwinia rot of banana, it is a major problem in case of tissue culture grown banana plantlets. Up to 5 months, we can able to see the more incidents, that is up to 35%. When we do planting in the month of January, the incidence will be more, and we can record up to the month of May. So in the incidence is more in TC plants compared to sucker grown plants. The infection can be seen from the first month onwards, and the infection starts from the crown region, and the pathogen enters and it is systematically invades the plant tissue. And here are some of the symptoms I have depicted, and here you can see so complete death will be seen. And in grown-up plant, that is in 5-month-old plant, when we push the plant, it can easily fall down, leaving the suckers inside. So since the variety which is highly susceptible is Gran 9. This is the majority, as yesterday's speaker, Dr. Bakirani Madam mentioned that Gran 9 is grown up to 60% is 60% of the area is covered by Gran 9. So this disease mostly recorded in the Gran 9 variety. So what we have tried, the TC plants has to be enhanced with the disease resistance by inoculating with PGPR as well as endophytes. So next slide, please. The hotspot area is Theni district, and in Theni, majority of the area is the Gran 9 is grown, and some of the hotspot area of Ersanaikanoor, Muttalapuram, Kanniserupatti, and Sinna Ovulapuram, Vaikalpatti, this area we have did surveyed, and we have collected the samples and did isolation from the infected pseudostem part as well as the rhizome. And next, please. And the culture was isolated. Next slide, please. And we have done the molecular characterization also for the Erwinia pathogen. This Erwinia pathogen is producing pectinolytic enzyme. So what we did is, due to the production of pectinolytic enzyme, the tissues get macerated, and the tissue can, this is also called as soft rot. So we aimed for the PEL genes, that is a pectinolytic gene, and we could amplify from the culture around 464 base pair amplicon from the cultures. And we have confirmed this bacterium as Pectobacterium, and this bacterium can be easily grown in the crystal violet pectate medium. In this medium, it can able to produce some cavities, that is, it will because of the degradation of the pectin which is present inside the medium. So from that also we could identify this bacterium, and similarly we have done we have aimed for biocontrol agents. So we have collected some of the biocontrol agents from the rhizosphere soil as well as some endophytes from the resistant varieties. Some of the PIA varieties we have collected the endophytes, and these endophytes as well as biocontrol agents were tested against the bacterial pathogen, and we could able to identify 3 endophytes which is promising. Next, please. So molecular characterization of the all collected isolates we have carried out, and some of the isolates were Pseudomonas, and few isolates were recorded as Bacillus subtilis. And using ITS primer, we could able to amplify. Then next, please. After confirmation of the molecular characterization, and we have done the in vitro screening by using paper disc assay as well as agar method. In this method, the paper discs in the cultures, and we have plated this paper disc inoculated with the bacterium and placed over the media. This media can able to solidify, that is, some of the bacteria around the discs. So from this we could able to confirm that this bacteria is effective against Erwinia. Next. Agarwal method also, we have inoculated the biocontrol agent inside the well, and the remaining is seeded with the Erwinia previously, so that this from the agar well, this biocontrol agent can able to that is able to degrade the bacterium around the well. So from this we can able to confirm that this bacteria were effective against the Erwinia rot pathogen. Please next, please. Madam, please move to the next slide.
Madam, is this okay, madam?
It is not moving. Yeah, please. Yeah, it's moving. Yeah, no.
This one is this one.
So this yeah, we have confirmed the molecular characterization of the promising biocontrol agents. That is 3 bacterial endophytes we have detected. That is namely PP, then NB10 and CL3. These 3 bacterial endophytes were molecular characterized, and we could able to deposit the sequence in the NCBI database with the following the accession numbers. And these bacteriums were identified as Bacillus. and these bacterium were further subjected for biotization. So next. Next slide, madam. So in this biotization, normally we used to raise the plants using shoot tip culture. So here we have inoculated with the bacteria. In this, the uninoculated control, this without bacterium, that is pathogen, the plant can able to well, whereas the pathogen inoculated media, the plant is unable to grow well. So for challenge inoculation, we have taken this task. After that, please next, please. So what we did is next. So we have raised the shoot tip culture normally followed in the tissue culture program. After getting the shoots, the shoots were transferred before rooting, before transferring into the rooting media. The shoots were dipped in the bacterial suspension for 5 minutes. After 5 minutes, these bacterial, biotized TC plants were transferred to the rooting media. Next, please. And here also I have shown the 3 different cultures what we have used for biotization with this efficient strains. Next, please. And here we have observed that some growth was very good growth was noticed in case of control because there was no inoculation with the bacteria, whereas the biocontrol inoculated plants somewhat it is a little some physiological stress was there, but even though it can able to grow well. So we have kept these plantlets for 25 days in this media. After that, this was transferred for primary hardening stage. In this, before doing primary hardening, again these rooted plants were dipped in the bacterial suspension for half an hour. After half an hour, these were subjected for primary hardening. So in the after primary hardening stage, in the primary hardening stage, we could able to identify some growth compared to control. Some growth was there. Later, the same in this later it was subjected for secondary hardening. Next, please. So secondary next, please. After secondary hardening, we have taken these plants to pot cultures. In the pot culture also, we have inoculated the talc-based formulation of Bacillus subtilis. And this Bacillus subtilis can able to enhance the growth of the plant. And also, it's after that we have inoculated the bacteria because this pathogen, the pathogen can able to colonize during the primary hardening stage. So that stage we have inoculated, challenge inoculated with the pathogen. So it can able to resist the bacterial, this biocontrol agents can able to give some resistance against the Erwinia pathogen. And based on the glasshouse studies, we have taken the same another set of plants to the field studies also. Please, next, please. So in the field studies, what we have done is we have inoculated with Bacillus subtilis. After secondary hardening, these plants were subjected to the field screening. Here we have added with some trichoderma as well as a VAM to give enhanced resistance against the rhizome rot, because since the consortia will give more protection against the plants. So these plantlets, we have analyzed the presence of isoforms, whereas the inoculated plant showed some of the increased PPO activity, whereas in uninoculated control, the isoform incidence is very less. And because shortage of time, I have not included the field trial data. In the field trial data, we could able to observe that this can able to give important resistance against the Erwinia pathogen, and followed by the VAM added plants. So from this, this is a new technology we have published in Scientia Horticulture. I thank DST sir for funding this project, and thank TNAU for carrying out this research work. So biotization is a new novel technology that can impart resistance against the upcoming pathogen in TC plants. Thank you, sir.
Yeah, thank you, madam, for a wonderful technology that you have developed with respect to banana plantlets are concerned.
I got one
So,
comment here.
Yeah, please.
Yeah, this is you are
Thank you,
referring
sir.
to in aspect that when you are telling about
Sir, it's not audible. Sir?
Yeah, you can repeat question now. Thomas, please. You can repeat the
Sir?
question.
Can you hear? In vitro bacterialization.
Yeah, please.
When you are inoculating the organism in the tissue culture medium,
Yeah.
are the plants able to survive, or the bacteria is outdoing the culture?
Sir, the plants, I have shown the slide. It showed some
Yeah, but
decrease
then
in the
that
growth, but even though after that we could able to gain the after the additioning, after we added some inoculants in the pot culture studies. But in TC, it shows somewhat growth reduction.
Yeah, in vitro bacterialization, I would not be a.
Sir, please.
Yeah. Can you hear? And also, I would like to mention that if you are
Please, sir.
Also, you are adopting any slides from the net, you acknowledge that also. Okay.
Can I do one question? Can I?
Yeah, sure. Yeah.
Ma'am, your presentation is very good and is very lubricating. But my one of my question is, are you scale the disease index according to your study? What scale, under which scale you're actually you can detect the disease up to which scale? Either it is from 1 to 4 scale method or on to 5 scale method. If you know there are different type of scaling to know the disease forming efficiency of any fungi or any types of plant pathogens. Am I audible, ma'am?
Yeah. For fungi and all, we do scaling, sir. For the Cervinia, sir, can you able to hear me?
Sure. Yes, madam. Go ahead.
Yes, obviously.
Hello.
Yes,
Go ahead,
yes. Yes, ma'am.
For in case of bacteria, we don't have any scales, but we have just analyzed percent disease. That is number of plants infected divided by total number of plants into 100. That is the percentage. Because total death was noticed in case of Erwin erot incidents.
Yes, but no disease scaling was done, right, ma'am?
Thank you, sir.
No disease scaling was done, right, ma'am?
Yeah. Disease scaling, because normally it will confuse. No, disease scaling was not done. Only this rating, that is a percent incidence was taken.
Okay. Another question is, ma'am, are you do the?
It is total.
Are you perform the defense enzyme study of any of the pathogen inoculant or PGPR inoculant plant? Comparative study of disease in the disease defense enzymes.
Yeah. That the one slide I have shown PPO activity I have shown. Remaining
Polyphenol
work
oxidase.
I have not included here. The remaining
Polyphenol
biochemical
oxidase.
assay, everything was carried out. Yeah. Everything was carried out.
Right.
It was there. No. It is in the Science Horticulture paper.
Okay. Thank you, ma'am. For your answer.
Sir, I have one question to Dr.
Sure, madam.
Yeah, this is Saraswati from NRC Banana.
Yeah, please.
You have how did you after treating? No, did you do any sterilization again, or you have directly put into the rooting medium? You have done your priming first biotization. That is your.
No, madam. Yeah. That's what I have shown you, madam. No.
Yeah, you
that
have
after
shown me.
biotization, we have straight away. There is a reduction in
because
the growth,
you have done your
but
priming
it can
at
able
3
to
stages.
survive. It can able to survive.
You have done your priming thrice. Once is before rooting, putting it in the rooting media.
Additional.
So before putting it in the rooting media, did you do treatment?
1 minute only.
Give any treatment that is for sterilization or something, or directly you have put soaked it?
No, madam.
Then how did you transfer?
After removing the sterilization, the root was cleared. Straight away it was transferred, madam, but the inoculum was there. That's what I shown you. Some reduction in the growth, the plants are not able to survive, like control.
The growth was not as good as it was in control. Yeah, that you have shown. And I have my another question. Another question I have.
Yeah. That's what.
Because while putting
Yeah.
it in the pot or in the field, I
please.
don't remember. You have added BAM and trichoderma in addition to your own bio agent, whatever it is.
Yeah.
But if you add those two, you
Yeah.
know
Yes, madam.
whether it
Yes,
is the
madam.
exact
Because
impact of your
this
thing or it is because of BAM and trichoderma. You're able to get my question?
No. Actually, we had 14 treatments. I have not shown here.
Okay.
Yeah. I can able to get you. We have 14 treatments in the field without this PGPR inoculants, only BAM and trichoderma. Along with this, these 3 strains. In this PP strain performed well, along with trichoderma as well as with BAM.
So you mean to say that was alone,
what I
different
feel
treatments?
Is, some good.
I think we can have.
Yeah. 14 treatments we have laid out with trichoderma, with mycorrhizae, with PP strain like that.
Okay, that's fine. Thank you very much.
How was, madam? One more question.
Thank
I'm Loganathan
Thank you.
from NRC Banana.
I think we should continue the discussion in the chat.
Wait, Thomas, one second. Yeah, please. I think somebody wanted a question.
Yeah, myself, Loganathan from NRC Banana. Yeah, I would like to get some clarification.
Yes, please.
Whether that bioperming done by PPO isolates, that's something you have told endophytes. Is it not?
Yeah, bacillus.
How the effect was that endophyte alone, bacillus alone?
So when it was alone, the incidence was somewhat 50% incidence we could able to notice.
50% reduction.
Whereas if it is added with the trichoderma plus 50% reduction, 50% reduction was there in the sick clot.
Okay.
Hello.
Yeah, I'm getting it, madam.
So when it was added with trichoderma plus BAM, we can able to enhance the disease resistance. If we could identify some more best strain than this PP, that will help us in future, sir. That is my opinion.
Brother.
Okay, fine. Thank you, madam, for a wonderful presentation.
Thank you.
And a good technique. So now I call upon again, I'll go back to Dr. Achyut Rao. Achyut Ashok Rao Bharos, Associate Professor from Latur. Hello. Yeah, you are available, right? Hello, Achyut, you ready? Hello.
Sir, Achyut sir, please unmute your speaker.
Already they have loaded the slides. Share ಮಾಡಿದ್ದಾರೆ. You ready?
We are seeing, sir, your slides are available on the screen.
You have to hurry up. Okay, Achyut. You have to complete in 8 minutes, okay? Yeah.
Something got wrong, sir.
I made you know PPT.
Shall I take up that one, sir? Wait, sir, I'm taking. Now, can you able to see this?
Yes, ma'am.
Display?
Share it. Okay.
Now it's shared, sir.
Shall I start?
Yes,
sure.
sir, you can start.
Actually, I'm not able to see PPT, ma'am.
Yes, sir, I will put, sir. Which slide I have to go, sir, now?
Madam, put it to slideshow mode. Right, okay.
It's on.
Achyut, please, skip all the introduction part. Hello?
Earlier, sir has presented few slides, sir. In between, he got stuck. Now
Okay.
we need to
You
go
can go
ahead.
to that slide.
Huh.
You can skip introduction, madam. You can go to the straight away. Go to the.
Achit sir, are you there, sir?
Madam, it looks like I think is not getting. So shall we go
Sir.
ahead with the next speaker?
Yes.
Okay.
Yes,
Yeah,
sir.
Doctor.
We can hear you.
This is the morphology of pomegranate.
You can skip introduction now, Achyut.
Yeah. Yes, sir.
Yeah. So you can save time.
Okay, this is all the additional value. Here we are focused on the bacterial blight. Just down, skip bacterial blight. Yes, it is there. Actually, it's called treating the problem in obtaining the economic yield. The bacterial blight, generally you can call it as a oily spot. It is caused by Xanthomonas actiniporus. Next, please. The cultivation method of propagation for pomegranate, hardwood cutting, sucker, and ear laying. These are from IHR. Then drawbacks: 2 type plant progeny is not obtained. Require stocks in large number. Lot of space in greenhouse. Require much time and environment conditions. Next, please. Difficulties related to breeding are that conventional breeding of woody fruit trees is often difficult and slow because of high level of heterozygosity and long generation time between successive crosses. Here, to reduce the drawbacks, overcome the disease, and breeding regeneration protocol for pomegranate through indirect regeneration. When we're talking about indirect regeneration, just we go to the callus, the one more stage, the additional. The callus is nothing but an unorganized, growing and dividing mass of cell. Callus is usually composed of unspecialized trichoma cells. This is the plant tissue culture media, what we are using here. These are the, next please, vitamins. Next, plant growth regulators. This is the what of the steps we carried out for regeneration of pomegranate. First one, establishment of seedlings, then establishment of callus, then callus differentiation, shoot bud initiation, and shoot regeneration, then in vitro rooting and accumulation of the plantlets. Next, please. When establishing of the seedlings, we have germinated the seeds on MS basal media so as to get a 2 type or prepare seedlings within 30 days after inoculation. We have worked on the cultivar Bhagwa, which is well known. This is what the regenerated seedling, and we have used this plant for our explant as explant. Then why we go for indirect regeneration? As we all know that indirect regeneration offers advantage for genetic improvement and involve genotype development as they show variation in offspring plant from the mother plant and can be used to produce the somatic variants. And this we can have for our purpose. Then 2 types of explant were used. One is hypocotyle and another is leaves. Hypocotyle we have used after 11 to 17 days. Hypocotyles we are excited from septic seedlings that we have grown on the media from the seeds, cut into 5 to 7 mm segment longitudinally and dissecting explant. Whereas for leaves, we choose a fresh leaves and we just crack the bottom below portion of the leaves and cut it into 2 to 3 mm size pieces. Then for the callus induction from the leaves, we have the 9 different resins or media resins on which we have grown the leaves, and we found that 5th resin, the full MS plus 2,4-D 0.5 mg per liter was very efficient and good for producing the callus. Next, please. Then we go on for the hypocotyle with the same resin, but here we got the good response from full MS plus 2,4-D 0.5 mg per liter plus kinetin 0.5 mg per liter. This is what the picture what we obtained after 21 days after the transfer to the callus inducing media. Then we go for shooting. Here we applied 5 different resins, out of which MS plus BAP 2 mg per liter have given a good response. Then this is what the picture, how we obtain the multiple shoots. And this was started within 20-25 days after the transfer on the shoot initiation media. This is another picture. We got a long stem of the pomegranate. Then after that, we shifted to the rooting media. Again, we have the 5 different resins, of which half MS plus activated charcoal 500 mg per liter have good response for rooting. This is the picture of rooting after 15 days after transfer to the rooting media. And then go for hardening. Hardening using plastic cups with cocoa peat. And then after, we go for earthen pot containing soil, cocoa peat and sand, 1:2 proportion. And these are the some outcomes. Just we have discussed. We have differentiated of best we have used. Leaves we got a good callousing. Then on the full MS media plus 2,4-D 0.5 mg per liter and full MS plus 2,4-D 0.5 mg per liter plus kinetin 0.5 mg per liter for hypocotyle. The shoot formation was observed more in MS plus BAP 2 mg per liter, whereas rooting in half MS plus activated charcoal 500 mg per liter. So I'm very glad to say that we have sent the developed plantlet for screening for bacterial blight, and some of them were found to be resistant, and it is still in development process. The present study indicate that regeneration media has been standardized for cultivar Bhagwa, which is well known famous in Maharashtra for its appearance, which will be helpful for developing oily spot resistant variety of pomegranate through somatic variants, or it will be used for in vitro protocol, may be used for transformation experiment. With this, I complete my work and thank you for organizing secretary for giving me the chance. I'm sorry for inconvenience from my side so that I cannot able to offer much.
Okay, thank you, Achyut. Yeah, that's a good attempt made to generate callus and also regeneration through different explants in pomegranate. So, any questions, audience, please, quickly? Okay, if there are no questions, then I'll move on to request Dr. Shivayogi Ravalad, Assistant Professor CD NEET, UHS Bagalkot. He'll be speaking on development of encapsulated somatic embryos and evaluation for seed quality parameters in papaya.
Shivayogi sir, shall I share your?
Am I audible, madam?
Yes,
Yeah,
sir.
yeah, you're
Your
audible.
slides are also visible, sir. You can put on the presentation mode, sir.
Shall I start, sir?
Yeah.
Yes, sir. Yes, sir. You go.
You can
Good
start,
evening,
please.
Chairman, co-chairman of the session, and myself, Dr. Shivayogi Rao, Assistant Professor, UHS Bagalkot. And here I'm going to present my research work on development of encapsulated somatic embryos and evaluation for seed quality parameters in papaya. Papaya is decidedly a tropical fruit, important fruit crop over the years for its nutritional and pharmaceutical values. Besides its quick and continuous yielding habit generating early income to the growers, day by day, the papaya area is increasing in the country. But papaya cultivation is hindered due to the inherent heterozygosity and dioecis in nature. Production of non-true to type seeds of open-potted flowers exhibit considerable variation in shape, size, and flavor of the fruit, and highly susceptible to papaya ring spot virus and color rot. Normal seed product encounters some problems like hybrid varieties, vegetatively propagated crops, transgenic plants, costlier. As we all know, the 10-gram papaya seeds cost about around more than 3,000 rupees, and it's lack in timely supply of seeds. To overcome all these, the alternate method is the production of synthetic seeds. Some of the desirable attributes of synthetic seeds is non-damaging to the somatic embryos, durable during storage, transportation, and planting. And this protect the embryos while allowing for germination and conversion, contain nutrients, microorganisms necessary for germination, and is also help for preservation of germplasm and convenience in germplasm exchange. Large production of identical embryos in short time, it can be produced throughout the year. There is no season specific. With all these benefits, the study was taken with these objectives to know the influence of growth regulators on somatic embryo production or induction, to standardize the conditions for production of synthetic seeds in papaya. The present investigation was conducted at Tissue Culture Laboratory, Department of ITBT, UHS Bagalkote, and the source of plant material was used Arka Surya, which is released by Indian Institute of Horticulture Research, Bangalore, from the cross between Sunrise Solar and Pink Plus Switch. All the in vitro cultures work was carried out aseptically in a laminar air flow chamber, and a commonly used medium for all the experiment was MS medium. And these are the amount of stock solution added to the media, and sucrose was used as a carbon source at a rate of 30 gram per liter. These are my research outline, preparation of experiments. The shoot tip from the 40 to 60 days old plants we have taken from the nursery in MHRC Bagalkoti, and the surface sterilization was done by using various chemicals like mercury chloride, sodium hypochlorite, tin-20, bavistine, streptomycin, etc. And once sterilization is taken, the sterilized explant was inoculated, and regeneration protocol, somatic embryogenesis, encapsulation of somatic embryos, and evaluation of seed quality parameters. These are the results, and we have taken 2,4-D different concentration of 2,4-D and BAP for growth and survival of callus in culture medium. These are the different concentration along with different concentration of BAP we used. And the observation taken, enlargement of callus was done by usual observation by grading no response, very poor response, and good response. When you see the data, the results were shown. The treatment number 8, the 2,4-D at a rate of 4 mg per liter along with 0.2 mg of BAP was shown maximum callus growth at the end of 28 days after inoculation, that is 1.47 gram. And these are some photographs of inoculation in the growth media, initiation of callus and fully grown callus. And influence of 2,4-D on somatic embryo in this. Once we have got sufficient quantity of callus, then the callus was subcultured for somatic embryogenesis by using different concentration of 2,4-D. Here we are seeing the observation taken, quality tolerance of somatic embryos production from callus will be graded based on visual observations such as embryo induction, spongy tissue, no response, delayed somatic embryo, and somatic embryo induction after every 14, 21, 28, and 42nd days of inoculation. And these are the results. When we see the results, the 2,4-D at a rate of 2.5 milligram per liter was shown maximum number of somatic embryo induction from the callus, that is 35.25% compared to all other treatment. And whereas in 2,4-D at 1 gram and this 3 gram is less compared to this. And quality tolerance of somatic embryo formation, 2,4-D at 2 mg per liter and 2,4-D at 2.5 mg per liter was shows embryo induction, but 2,4-D at 2 mg per liter is a delayed response. With respect to 1 mg, 1.5 mg, there is no response with respect to somatic embryo product formation. And again, that callus was subcultured in for the somatic production in the BAP, benzylamine purine, with a different concentration. Here also we taken quality tolerance of somatic embryos formation from callus will be taken at every 7, 14, 21, and 28 days after inoculation. The BAP at a rate of 0.3 mg per liter shows maximum number of somatic embryos at the end of 28 days after inoculation, that is 50.75 percentage. And quality of somatic embryo formation, same with 0.2 mg and 5.3 mg embryo induction was we seen, but at 0.2 mg the delayed induction was seen. And with respect to control, that is 0 mg BAP and 0.1 mg, there is no response and very delayed response. And at 1 mg and 2 mg of BAP, there is no response. And this is the influence of kinetin on production of somatic embryos. Here we use a different concentration of kinetin for somatic embryo production. Kinetin at the rate of 0.2 mg per liter shows maximum number of somatic embryos at the end of 28 days after inoculation, compared to all other concentration of kinetin. And this is a qualitative analysis. There is no response with respect to control. And embryo induction was seen in 0.1, 0.2 and 0.3 mg. But, and also the 0.4 mg, 0.5, 1 and 2 mg, there is no response with respect to embryo formation. And these are the some photographs: somatic embryo induction, subculturing of somatic embryos in different growth trimester, embryogenic calli surrounded by water, cotyledonal stage and torpedo stage. Once you got fertile sufficient quantity of these somatic embryos, then the somatic embryos are encapsulated with 4% sodium alginate and with using complexing material, that is 100 micromolar calcium chloride. After encapsulating, the synthetic seed was tested for their viability by using tetradolium test. And we have taken germination test for synthetic seeds. The 80% of germination was found in MS nutrient media, and 30% at sterilized peat plus nutrient media. And in other directly soil, there is no germination even in directly to peat. And these are the uncontaminated seeds of synthetic seeds of papaya, cultivar Arca surya, and bacterial contaminated seeds, fungal contaminated seeds. And this is the capability of the encapsulated seeds to germinate on MS medium and on peat soil. These are the synthetic seed grown seedlings. With this, I conclude my presentation. The work carried suggests that by employing synthetic seeds, the tissue culture raised plants can be regenerated on a simplified medium, eliminating subculture, thus reducing the cost of operation. Development of number of plants can be produced in tissue cultures through embryogenesis, multiple shoot cultures. Their delivery is cumbersome. Embryos or shoots have to be separated singly and transferred for rooting to achieve root shoot balance, and the plants have to be hardened in greenhouse before field planting. Direct sowing of synthetic seeds in the soil does not need acclimatization, often enabling flexibility in handling the and transport as compared to large parcels of seedlings. Future line lock, synthetic seeds are a poor storer even in under low temperature, so there has to be need to study in the complexing material which is used for encapsulation to increase storability of synthetic seeds. Need to study the fortification of somatic embryos with encapsulating materials, which is to helpful in problematic soils. Thank you, HSP and organizer. Thank you, sir. Thank you very much.
Yeah, thank you, sir. Thank you, Dr. Shivayogi. Good technique you tried to develop somatic embryogenesis and also encapsulated somatic embryogenesis. So it's a good technology that you have developed. Any questions from the audience, please? Quick questions. Okay. In that case,
I've got a question.
Thomas, you have okay, sure.
Like
Sir,
commercial commercial. Why don't you take it to commercial scaling up?
Sir?
Why don't you go for the commercial scaling up?
Commercial scaling, sir?
Commercial scaling. You are telling about synthetic seed production in papaya.
Yes, sir.
To the commercial scale production.
We are planning, sir. We are planning. We are taking this not yet published, sir. This research work, And another also because this is a poor store, when we produce the synthetic seeds only under controlled condition at 8 degrees centigrade and 4 degrees centigrade, it meant we stored only for 4 to 5 days only.
So how many plants could you raise?
Research, it is poor storage.
How many plants could you generate through this technique so far?
Around 9 to 10 plants, sir.
9 to 10 plants.
Yeah. Under open conditions, sir.
Okay.
Yes, sir.
Thomas, okay, fine.
Yeah, that's fine.
All right. Yeah, thank you, Shivogi, for your presentation. The last speaker for under oral presentation is Dr. Pooja Bohra from Andaman Nicobar Islands. She's a scientist, food science, Division of Horticulture and Forestry, ICR, Central Island Agriculture Research Institute, Port Blair. So she'll be speaking on first report on in vitro regeneration in Musa species Damascensis, an endemic wild banana species from Andaman Nicobar Islands. Madam, your time starts now.
Thank you, Madam, I shared your slide. Can you able to see it?
Yeah,
Yeah.
I'm able to see it.
Okay, great.
Can you make it full
Go ahead.
screen, please?
Yes, Madam, I made it. I think it's getting in.
Good evening, everyone. I'll be talking on my work on in vitro regeneration in Musa indamanensis, which is an endemic wild banana species from Andaman and Nicobar group of islands. As the name suggests, it's from Indian islands of Andaman group of islands, so the name indamanensis. Next slide, please. There are around 37 wild taxa of Musa, which are distributed in different parts of our country, ranging from Western Ghats to Northeastern India, and then to Andaman and Nicobar group of islands. So Andaman and Nicobar group of islands, which have around 300 endemic species of different flora. Previous slide, please. So this species is one of those, and the species was recently discovered in the year 2014 by the Botanical Survey of India. However, it faces conservation threats as it has issues of anthropogenic pressure as well as its consumption by elephants, monkeys, and parrots. So the species, within 5 years of its discovery, it has been classified as a threatened species. In fact, now it is in a critically endangered category by IUCN criteria. And it is one of the wild relative of cultivated banana as it belongs to the same genus. Coming to the morphology of this species, the species is unique in nature as it has green colored flower bracts, which is in contrast to red or pink colored flower bracts in case of other banana species. And this is a photograph of its mature bunch. And each bunch consists of around 10 to 12 hands, and each hand has 22 fingers. And the below side, first photograph is that of a ripe fruit of Musa indamanensis, and middle photograph is that of completely seeded fruits of Musa indamanensis. And pulp color is golden yellow. So when we estimated the content, it was 2,640 microgram per 100 gram of fresh fruit pulp. This was total carotenoid content in terms of beta carotene equivalent. And last photograph shows the asymmetrically shaped seeds of Musa indamanensis. Now the question is, species has large number of seeds, that is around 200 to 250 seeds per fruit. And despite this large number of seeds, the species is in endangered category. Why? The reason was that it is a very shy suckering species. And when we explored natural habitats of the species, there was no natural regeneration. Means naturally regenerated seedlings were not present below the canopy. So it meant that the species had issue of natural regeneration. It was having some issues. So we decided to take it up scientifically. And considering its shy suckering habit, each fruiting plant will have only one or maximum two suckers in its mat, which is really, really a low number considering its diversity. So it was having the issues. Next slide, please. As we found only one fruiting bunch that was half eaten by birds and animals, so somehow we rescued it, and it is very, very limited. So when we surveyed Little Andaman Islands, we found limited number of fruits with us. So it was our 3 years work. In these 3 years of surveys, its distribution was highly, highly fragmented, and we were able to find a fruiting bunch only once. So we decided to divide our seeds into two categories, one for in vitro regeneration and one for ex vitro seed germination. So in case of in vitro seed germination, we did not find much success. While in case of ex vitro seed germination, another lot which was parallelly used for studies, we found that combination of suitable pre-treatment, that was 0.1% potassium nitrate for 24 hours, and best combination of substrate, it gave germination percentage of 91.67%, which is one of the highest rate of ex vitro seed germination within 2 months period in any wild banana species reported till date. So it was one of the very successful regeneration technique. And we used these as a limited number of seeds were limited number of suckers were present with us. So we used these seedlings as source of explant. And it had added advantage because using seedlings will retain variability present in the species and will have advantage of large number of explants with us. So in this slide, the best treatment of aseptic culture has been displayed. Though we tried different treatments for aseptic culture establishment, this was the best treatment giving response, and 90% success was achieved using this particular protocol for aseptic culture establishment. Next slide, please. When aseptically established cultures were available with us, so we wanted to try the cytokinins to induce shoot multiplication. We tried 6 different cytokinins at different concentrations, and we were surprised to see that very high level of BAP, that is 6 to 8 mg BAP, was also unable to induce shoot multiplication in case of Musa indonemanensis, which is highly unexpected considering the amount of work done in other Musa species. And surprisingly, roots were present in all the cultures, except in case of TDZ. So we decided that TDZ, this preliminary work, it clearly indicated that TDZ would be a suitable cytokinin for our studies. So in our next phase of our study, we decided to estimate the best possible concentration of TDZ for supplementation of our media. And in this particular study, when MS media was supplemented with 60 milligram of adenine sulfate along with different concentration of TDZ and 0.2 milligram per liter of naphthalene acetic acid, sucrose was used as a sugar source. We found that supplementation of TDZ definitely improved shoot multiplication in our cultures. So in subculture 1, shoot multiplication varied from 2.5 to 6.7, which and 2-bud inoculum was used initially. So it was quite promising. When we went for a second subculture, we found that there was shoot multiplication as high as 18 shoots per inoculum in subculture 2. And however, it was not significantly different with that in TDZ 0.2 mg per liter. So so it was apparently clear that 0.2 to 0.5 mg concentration was best, and seeing the economy, 0.2 mg per liter concentration was best for multiplication of Musa indonemanensis. However, supraoptimal concentration of 1 and 2 mg per liter was discarded. Next. So these are the slide. These are the photographs of our cultures. First photograph is that of an established culture which is in proliferation stage, and second photograph is that of successfully multiplied cultures obtained. Next slide, please. This slide shows multiplied cultures obtained in best treatment, that is 0.2 mg per liter of TDZ. Next slide, please. When obtained microshoots were used for in vitro rooting, so supplementation of indole 3 butyric acid at 1 mg and 2 mg concentration improved rooting percentage from 80% to 100%. However, there was no difference in root length which was observed. Previous slide, please. But number of roots increased in this when IBA concentration was increased. Next slide. So this micro shoots which were obtained from different media during in vitro rooting, when they were forwarded for primary hardening. So after primary hardening also there was difference in their performance, and we found that there was no difference in root length and number of leaves and root length. However, number of roots and shoot length improved with increase in concentration of IBA. So IBA definitely had role to play in rooting as well as further survival of the plantlets. Next slide. These are the photographs of primary hardening and secondary hardening of our regenerated seedlings. Next slide. So these successfully hardened seedlings, I'm very happy to share that we were successful in hardening around 1,000 seedlings or 1,000 shootlets of this particular plant, this particular species. So these plantlets were used for various activities. Since it is an endangered species, it's a critically endangered species. So it was very, very important to conserve it in maximum ways possible. That's why we went for both the things. Previous slide. Yeah, ex situ conservation as well as in situ conservation. For ex situ conservation, we established an exclusive germplasm block of this particular species in our garden. And since these are seedling regenerated progenies, means seedling explants were used, so definitely natural variability is present in seedlings, and in coming days, with suitable characterization studies, definitely we will be able to come up with something very wonderful. In case of in situ conservation, this photograph particularly is of Chidiyatapu Biological Park, which is one of the selected biological parks of our country. It is located in South Andaman Island. And besides these, beside this biological park, we also planted the plants in forest nurseries, botanical gardens, community areas, and colleges, which are located in different parts of our Andaman group of islands. And since we have multiplying cultures available with us, and we have received IC number for the same, so we will be able to share the material for research purpose. So anybody who wishes to work on this particular species for further research and other kinds of studies, they can write to our director, and through proper channel, we will be able to share the material with you. So in this way, plant tissue culture technique was practically employed for conservation of a species, for saving a species from extinction, which is at present in critically endangered status. But definitely, I'm sure with this work, and since we already have multiplied seedlings, as and when we wish, we can harden it and again take our conservation efforts to the place where it is required. So thank you so much for this opportunity, and also I acknowledge the financial support given by DBT for conducting this research work. Thank you.
Yeah, thank you, Dr. Pooja. It's a wonderful attempt you have made to conserve the species, and that too a wild species in the Andaman instance, Musa species. And I was about to ask a question. Did you reintroduce them to the wild? Already you have an answer for that. So anyway, congratulations for a wonderful attempt.
Thank you so
Quickly,
much,
if
sir.
any questions are there to Madam, my audience? Thomas,
We
do
have
you have
another
any question?
session, sir, please.
That's fine. Very good. I think it's
Okay, thank you, Madam.
Thank
And
you,
one the announcement I had to make first is that technical session, the talk which was scheduled today, that is Dr. Carolina from Singapore, has been shifted to 2:30 tomorrow. So this is what organizers have told me. So we had a very good session, started with Dr. Uma, giving a very bang on the, especially bioreactors, and the power of bioreactors, especially in case of banana and all the vegetable crops. There were two invited talks, one by Mohanty and also Ashwath. Unfortunately, Dean Bharati could not participate. Rest all oral presentations have been made. So I thank all the speakers for wonderful slides, wonderful information covered, and wonderful technology that they have developed. And my thanks to organizers, especially Dr. Fakhruddin and also Alesh. My thanks to Dr. Pias Thomas to act as co-chairman in the session. And my thanks also to rapporteurs, that is Dr.
Sir, I would like to request Dr. Pias Thomas, CEO
I'll be calling. I will ask him to. Okay.
Comment, sir.
Yeah, please.
sorry.
Thomas, please. You have comments?
Yes, sir.
Yeah, we had a very wonderful, useful session. Dealing with the commercial application from lab to in commercial scaling up, particularly from Dr. Uma. It has been very successfully demonstrated with banana. I hope this would be a role model for other crops also. And also there was such good presentation from Dr. Mohanty and Dr. Ashwath, which are keynote invited speakers to this session. Also, we had a very good participation, presentation from different participants on different crops and related aspects. I congratulate all the participants and also thank all the delegates for patient listening and attending. Thank you, Anand. And
Okay.
also thank the chairman and the organizers for this opportunity to co-chair the session. Thank you.
Okay. Dear delegates and participants, I would like to thank Dr. R. Lokesh, Dean, College of Agriculture, B. Gudi, University of Agricultural Sciences, Raichur, for chairing the session. And also I would like to thank Dr. Pius Thomas, CEO of Thomas Biotech, and also the former principal scientist of Indian Horticulture Research, Srigata, for nicely handling this session, that is advanced in vitro plant tissue culture and micro propagation. Thank you, sir. Thank you very much.
Thank you.
Thank you so much.
Okay, dear delegates and participants, now let's move to another session. The theme is advances in molecular biology and abiotic stress tolerance. Molecular biology of abiotic stress tolerance. For this session, Dr. K.S. Shivshankar, Principal Scientist and Head of the Division, Basic Sciences, ICR, IHR, Esrigata, Bangalore, will be chairing this session. And Dr. Y.A. Nanjareddy, Professor and Head, Department of Crop Physiology, University of Agricultural Sciences, Bangalore, will be co-chairing this session. That is advances in molecular biology of abiotic stress tolerance. Let me introduce this chair and co-chairs. Dr. K.S. Shivshankar, after completion of this undergraduation, post-graduation, and doctoral degree programs, he has been selected for the post of scientist in ASRB. Okay, he had put 29 years of experience in research in IHR. Presently, he is heading the Division of Basic Sciences in IHR Esrigata. He has handled many projects funded by DBT, DST, ICR, RKVY, and he has completed his postdoctoral fellow in Tsukuba at Japan, which was funded by UNU Kirin Fellowship. He has been trained in Agri-Food Canada, which was funded by NAIP Fellowship, and he has been awarded Fellow of International College of Nutrition, Alberta, Canada. He has guided 3 PhD students, 1 MSc students. He has got more than 80 publications in national and international journals. He has edited 4 books. He has got 12 book chapters. He has attended more than 26 seminars and symposia. His expertise is in metabolite profiling for abiotic and biotic stress tolerance, flowering physiology, post-harvest physiology. Thank you, sir. I would like to introduce Dr. Y.A. Nanjareddy, co-chair of this session. Dr. Y.A. Nanjareddy, after completion of his undergraduation, post-graduation, and doctoral degree program in University of Agricultural Sciences, Bangalore, so he had joined Rubber Board. At later period of time, he joined as assistant professor at College of Sericulture, Chintamani. Later, he moved to US Harvard for the post of associate professor. He came back to US Bangalore for the professor post. He has guided 10 MSc students and also many PhD students. He has handled several projects which were funded by ICR, IDRC, DBT, and Indo-Swiss collaboration on biotechnology. He has put more than 25 years of experience. So he has got his expertise in physiological basis of yield improvement in small millets, identification of germplasm accessions for abiotic stress tolerance in particular to temperature and drought, and nutritional studies related to biofortification and evaluation of germplasm lines, further nutrient composition in small millet. I once again welcome Dr. K. Shivshankar and Dr. Y.A. Nanjareddy as chairman and co-chairman for this session.
Thank you.
Please.
Thank you, Halesh.
Thank you, Dr. Halesh, for a nice introduction. And I have the pleasure of having a very good co-chair as well as 2 rapporteurs, Dr. Mallikarjun Avati and also Dr. Babu A.G. Without taking much of your time, I would like to invite Dr. Sheshashai for invited talk. We have 2 invited talks in this session as well as 6 oral presentations and also 1 keynote address. 2 keynote address. I heard that 1 keynote address is not being given because Dr. Shetty is indispensable, and Dr. Robert E. Sharp is giving the another keynote address. So without wasting much time, I would like to invite Dr. Sheshashai, who is a professor in Department of Crop Physiology, UAS GKVK. He is my friend and my colleague at UAS GKVK when I was doing my PhD. I have the pleasure of inviting him to give his keynote invited talk. He has got vast experience in molecular breeding for drought tolerance with special emphasis on marker-resisted selection for water use efficiency. And he is also highly experienced in identifying the traits related to drought tolerance. And he is, I think, one of the physiologists who have one variety to his credit. So with this brief introduction, I would like to invite Dr. Sheshashai to give his invited talk. Dr. Shesha, please.
Thank you, Shiv Shankar, and thank you, Dr. Anchareddy, and thanks for this opportunity. This is an interesting conference, really, though it has taken some time.
Hello, sir. Shesha, sir.
Hey, Halesh, thank you so much for coming. And I know all of you are friends, and it's a very interesting situation that all the guys who are sitting there in the chairing the session and the rapporteurs are my very close friends, and they're all colleagues in the department. Now, that apart, I want to ask the chairperson, how much time do we have? Though Dr. Fakhruddin mentioned to me that because Dr. Chetty is not available, maybe we can use the time, and then he asked me to prepare a little longer lecture, but I can manage it to shorten that if there is less time.
Yeah, 30 minutes for invited talk, and I think 10 minutes each for the oral presentations.
All right.
So you can take your own time, no problem.
Yeah, you can remind me to stop for after 30 minutes. All right. So let me share my slides and go ahead. Thanks for the kind words and introduction. I hope this is visible.
Yes, sir.
Is the full screen visible? Yes.
It's getting on, sir.
Yeah. Okay. So there you go. So as Dr. Shiv Shankar was introducing me, he said that we were colleagues, when we started all this work quite some time back. And from the beginning, we were interested in understanding how the plants respond to drought and what is it that we need to do as physiologists to sort of improve this further. Though this work started a long time back, and there were my dear friends, colleagues at that time, though they were not directly involved with me as students, but then Dr. Natarajan, who is now an associate professor in Karaikal, I hope he is fine with all the rainfall that is going on there. So we started that work quite some time back. As he said, that we started looking at the markers to look for this complex physiological traits. Now, to understand how we go about this, I have a very, I could have 2 kind of things. One is an elaborate one and a short one. Maybe we will make it somewhere in between these 2 things. And there is one thing that I always sort of amused by this. We are not supposed to talk something very vulgar in the public, but then you look at this, Indian population is going to be the highest in the world, and we are going to overtake China. Nobody can beat us in this kind of a thing for sure. But the question is, if you look at the graph that we have here, this was a paper published in 2015 or 16 in Science. I'm not very sure of the year, but then the story is very, very clear. If you look at this, this is the productivity that you have for rice, wheat, and maize in 3 panels. And you have something like 8 or 9 tons per hectare produced in, let's say, the developed countries like United States or Australia or some of those very fertile places. But what is appalling to me, at least, is that the Indian productivity is always at the bottom. Like its rice is something like 2.5 to 3 tons per hectare even now. And then, if you put the graph for pulses, it is actually crawling on the x-axis. So it's something which is terrible to now look at, especially because we have so many people to feed. And therefore, it is all that more important for us to now ask this question, though we are self-sufficient now, are we going to face the challenge in terms of feeding the people that we are going to have, where we will be the most populous country in the world? Thanks to the breeding activity, I'm sure there are a number of plant breeders in the audience as well as and most of the collaborators that I have in the country as well as outside most of them are geneticists and breeders. The question that we always ask for each other is, why is it that the yield has not been, we have not been able to break this yield barrier here for the past 15, 20 years. And though there was a significant increase, and then if you can see this curve which is going down, it is the contribution of agriculture to the GDP of the country. And this is a real serious problem. And if you look at agriculture as an industry, it's not going to be remunerative at all. Now, what's the question? Why is it that, we have only two choices. We address this issue or we die and then, don't do anything at all about it. But if you ask this question, the major problem seems to be is that 54%, if not more than that, in India is actually suffering in high to extremely high water stress conditions. So we have to really look at how is it that we can address this situation. Now, besides breaking this yield barrier, I said, where we are reaching some kind of a plateau, we need to break that. Now, we have two real challenges that we need to sort of address. One is to save the water from the irrigated fields, because water is becoming scarce. I think some people also talked about it in the morning and in the afternoon as well, that water is going to be the biggest problem. And then when we look at the kind of crops that we have, about 70% of Indian agriculture happens in the rain-fed conditions. Now, how do we sustain the yield there? And one of the biggest culprit that we did talk about it in the morning today when we had a students' colloquium with Dr. Nanjaredi. Now, if you are consuming rice, you are consuming a lot of water along with it. Is this a viable crop at all? I mean, do we have to eat rice or do we cultivate rice with less water? Now, are these questions viable? So I have several questions on my card actually to ask and do some experiments with our whatever that resources that we have. Can we cultivate rice with less water? Now, is it possible that we can save water as well as sustain productivity? And of course, we are trying to increase the yield under all sorts of good conditions. So we come to this question as to now, how do we meet this goal of asking whether the food requirement that we have for the growing population can be addressed? Now, with that comes to the question of asking whether we are capturing the resources that are there and whether the resources are used very efficiently at all. That's the reason why we consciously started looking into working on how the plants can be addressed from the water use efficiency. We have the light, and that's not going to change even if there is climate change. But of course, the radiation is going to be a problem if you have a lot of aerosols in the atmosphere, a lot of pollutants in the atmosphere. The amount of radiation that is coming down to the Earth surface where our biosphere exists is going to be, it is decreasing actually. And of course, water and nutrients are the real major threats, and this is a problem. And now what I did was to sort of bifurcate the yield. If you ask the breeder, he looks for the yield, and that's as the kind of a strategy to select. Now we have two questions. One is the potential yield when resources are sufficient, like let's say the irrigated or well taken care of farms. We have potential yields there. But we have other kind of yields where we talk about when the resources are not used and when they are limiting, we call them as a realized yield. It's a realized yield which is a problem actually. Therefore, we need to now address this question. I'll come to answer this trades business a little while later. But then when we ask this question of the realized yields and the resources are not very good, none of these resources are going to be sufficient. How is it that we can improve the productivity at all under water or under resource limiting conditions? Now, if you look at rice, and the question that we had was that can I grow rice with less water so that we can sustain the resources at least? But then the question would be the yield and definitely decreases when the water is limiting, for sure. Whether the water limitation occurs in the initial vegetative phase, we call it as a GS1 or the growth stage 1, and then during the flowering stage or during the grain filling stages. Now, when we did some experiments recently with one of my PhD students who works in association at the Wageningen University, but we did the experiments here in Bangalore, and we saw that the yield decrease in rice can happen either because of the reduction in the leaf area if it occurs in the initial stages, or because of an increased spikelet sterility during the flowering period or anthesis period, and the reduction in the canopy photosynthesis when the stress occurs in the grain filling period. In any case, there's a tremendous amount of reduction in the yield under water limiting conditions, and that's exactly where we need to now understand. But if you look at these three genotypes, there are some genotypes which can sustain. Like, for example, if you have these three varieties, like, there is a, this is called as the upland rice, and then Apo is a tolerant variety, and then of course, the light, the water-loving IR64. IR64 yield reduction is the highest, so that's where the problem appears. And therefore, we need to now look at the other genotypes and then understand how we can now do this. But if you look at how the breeders did this job in the past, they selected for high absolute yields under stress. This is the kind of thing I think today morning Dr. Ravindran was also mentioning about this, and then he was mentioning about the work that was done by the ERI and then the ERI unit in India, Dr. Arvind Kumar and a number of others, and AK Singh in IARI. They were all looking for selecting for high yield under stress. It has worked for sure, no doubt about it. Progress has been very rapid, but if you now look at the kind of high yielding varieties, the variability that you find among them is narrow. And then there's a very high genotype to environment interaction for yield, and therefore it is less stable and heritable. But on the other hand, if I ask the plant physiologist, and I'm ready to confuse you more and more now, before we really clarify and come back to some consensus and see whether we can really do this. But if I improve specific drought adaptive trait, is it going to be better? The progress is going to be painfully slow, no doubt about it, but the advantages are the traits as such. If I look at some specific traits, I will come back and tell you, is it very easy to look for traits or not? They have less genotype to environment interaction. We have data on that. And because of that, they are very highly stable when you do the breeding programs. And that's the reason why perhaps an analytical approach or a trait-based approach we call these days, or it is also called the physiological breeding approach, is it going to be a preferable one? And in the next few minutes, maybe about 15 minutes, I'm going to tell you, try to convince you if this trait-based breeding can really be a successful thing. But before we go there, let's confuse each other and ask this question, which trait are we going to use? So the moment I ask a plant physiologist, I will tell something, Shushankar will tell something, Nadaredi will tell something, and all of us will tell a million things, and therefore the breeder is not interested in us. But let me clarify this. If I have to now have the drought adaptation as a mechanism, I look at three very important things. Something that happens at the morphological mechanisms or morphological traits, or we look at the physiological traits, or we look at something that happens at the cellular level. We call it the cellular level tolerance or acquired tolerance traits. These are something which are not there under normal conditions, get expressed. I'll come back to that also. Now, when you look at the physiological traits or morphological traits or cellular level traits, you have again, I will just put a very few, but there can be a large number of these things. Now, each of them is a breedable trait. Now, this is the problem. Now, which one will you use? How will you use? How will you measure? These are the questions. But if you look at each of these things, there is one thing that they are all sort of coming and converging into one, and they appear to be carbon metabolism and water relations. Therefore, all these mechanisms that we talk about, morphological, physiological, or cellular, they will always point towards maintenance of metabolism and maintenance of water relations. Either you maintain the water relations or you maintain the metabolism even when the water relations are low. I mean, this particular work that we did, which we did as a review with some of my students like Preeti and Pratibha, this was very highly recognized and it is highly being cited these days, which we published in Frontiers in Chemistry a couple of years ago. And then what we did was to now go ahead and now see if we can simplify this entire thing. And we said we follow those, a very classical model which was proposed by John Paciora quite some time back in the 1970s, mid-1970s. And he said, any genotype that uses water and uses it efficiently will be the one that has a better productivity. There is no doubt about it. We keep talking about some of these things. Any person who's intelligent and works hard will be the first ranked person. There is no doubt about it, isn't it? So this is simple logic that he said. When we look at the water mining, we look at the roots to now absorb water. When we look at water use efficiency, we look at metabolism, especially the carbon metabolism, and then of course when we have the waxes. But then these are some of these traits that I would say are constitutive and they will be doing well only when the cellular level tolerance or the acquired tolerance levels are better. Now, when we go ahead with this kind of a thing, and then go into the literature and find out if people have done these kind of things, you can see a large number of information, and there is no doubt at all about how whether or not the roots are important. And this is one of those where one of our colleagues, which all of us know, Dr. Jagdish Rane, was also a part of this paper in Nature, which was published by Uga and his team in 2017. They did a wonderful job to find out a gene called DRO. DRO is the deep root gene. And they identified an allele and they fine mapped it, and this is a fantastic work that they have done. we did two kinds of things from, we had started this before, but then we got the kind of courage to do this further. We developed a mechanism to phenotype for the roots. We identified large variations both in germplasm as well as in mutants. What we did was to now identify some of these traits and see if we can do some molecular biology with it, or we can do some physiology with it. And of course, one of the major things that we wanted to do was see if it can be improved in crop improvement programs to incorporate them into higher yielding types under water limited conditions. Now what we have was, we identified from a large number of mutants, which Dr. Ravindran was mentioning this morning. And a part of that we used here for looking for the traits that we are interested in. We had high root types and we had bad root fellows. These are the knockdown mutants, and we have the gain in function mutants. And if you can see that the high root mutant like 392 has much higher photosynthetic rate under stress conditions, much better stromatal conductance under stress conditions. There's not much of a reduction. So these are the guys who can maintain the water relations, is what I'm saying. So water relation traits help in maintain metabolism, is something which was very interesting, and we published that recently. But the problem comes, the plant physiologists are very good in telling you this is what happens, but but then the questions are something else. If I increase the root under water limiting conditions, it actually exhausts the water from deeper layers faster. That means we have large number of information in the literature to suggest that if these plants are grown under conditions when there is residual moisture stress is going to be the problem, then these plants are certainly going to now encounter or experience end of season stress, which will generally come with flowering or the grain filling period, and thereby decreases the yield. The good roots, though are good, are not good actually. Now this is the problem. Then we shift the emphasis on understanding whether the water use efficiency has anything to do with it at all. All right. So when we look at this, we have two questions in it. Now, how do we capture the genetic variability in these traits? Like, let's say it's root or water use efficiency, and then ask a question whether this particular variability that we find in these are breedable at all. Okay. Now, when we look at this, there's one small thing that we have a large number of information that we have generated on this. We have a national facility to measure the stable isotopes. But what I want to tell you very quickly is that the stable isotopes, they're stable, not radioactive. Please be aware that these things can be used as fantastic, they are called the tracers for understanding some of our metabolism. And what we did was, you can see that the carbon, for example, the lighter isotope, which has atomic mass of 12, and another heavy isotope of carbon, which has an atomic mass of 13. And the 13 isotope is much less abundant in the atmosphere as CO2. But if you look at the composition of these isotopes in all the different organic and inorganic substances, there's tremendous amount of variations that you find. And this variation comes because of biophysical or biochemical processes that are altering the isotopic composition. Now, if you believe photosynthesis and transpiration, which are the two physiological parameters simply used for calculating the water use efficiency, are both physiological or biochemical and biophysical processes. So if I use this carbon isotope ratios, or another thing I will come back and tell you for the oxygen isotopes, can they be used to infer the process that we are talking about? And lo and behold, yes, we have a large number of data on very many different species, including some of those perennials like cashew and coffee. And we have done that in much larger number of genotypes these days than what is published here. And then the relationship exists very strongly between water use efficiency and carbon isotope. And the carbon isotope ratio, therefore, becomes a very important surrogate for water use efficiency. Now, this is not the only thing that we did. I was glad that I worked for my postdoc with this great man called Graham Farquhar in Australian National University 20 years ago. And then at that time, they were developing this. I was also a part of this when we say that the isotope discrimination can be used as a strategy to achieve higher yields under water limited conditions. Excellent. And then we established this facility. Graham Farquhar and Richard Richards, who are our collaborators, visited us here in Bangalore. And when we now have this is a facility that I said, we don't have to spend time on that. But again, as a physiologist, I'm very happy with this but and all those kind of things. When I have a but, breeding for water use efficiency alone did not help at all. See, we said that the roots are not good alone, and then water use efficiency, now I am saying, is not alone, is a good thing, because the yield advantage disappears when the rainfall or the water availability during the cropping season increases. So the relative advantage of the yield, this is a fantastic work that we did together, and to understand whether the water use efficiency is a good one at all. And that led to a large number of publications that came in the world at that time to understand this water use efficiency is not a good take at all, especially from some of these scientists, physiologists like Abraham Blum. Okay, but what we wanted to do was to increase the water use efficiency without decreasing the transpiration. That's where the challenge was. And we needed to have a mechanism to know, identify genotype that had. And we developed an oxygen isotope signature as a strategy to understand this. And we have fantastic data to now show that the isotope of oxygen is a surrogate for transpiration rate and the mean transpiration rate in the field. And then when we use this data, we can use it for estimating the differences in the root biomass. Therefore, oxygen isotope helps as a proxy to root rates, water use rates. Now, if I have these two strategies to measure the water use as well as water use efficiency, I have a mechanism to now determine what is the best way to find out the differences in water use efficiency. But the question this exists again, does water use efficiency have the potential as a drought adaptive trait at all? So this is where the question was. And the answer is yes. For me, I would say yes, definitely yes, because we have done this, and Anthony Condon and Graham Furco and others have published this. We have our data to show water use efficiency is indeed a parameter that can help. But a lot of data also says no, it is not useful. Because when I have the regression between water use efficiency and the total biomass or yield, you only get a scatter. The breeders are not interested in this. Breeder wants some kind of, if I do this, can I increase the yield? That's the question that we have. And we analyzed this data very thoroughly. We had large number of germplasm that we had from germplasms of groundnut and the germplasm of rice. And the rice germplasm, we got it from CRRI at that time from Qatar. And then the germplasm of groundnut was accessed from ICRISAT with Rajiv and HD Upadhyaya. And what we did was to now analyze the water use efficiency and biomass and analyze the big data that we got. We have large data to now show them. Now, what is coming out here is that when we look at the water use efficiencies, we compared them. We have the low delta 13C types and the low water use efficiency types here, and in both the cases. But what we said was, then when we classified them for the root, the bad root genotypes always had low delta 13C, which indicates that the bad root genotypes are more efficient in water use. This is the problem. We don't want them to conserve water, but they want the plants to use water, but then use them efficiently. That means the high water use efficient genotypes are always low root types, and the low root types don't do well under water limiting or any other conditions. And that's the reason why whenever you selected for high water use efficiency or low delta 13 carbon, that resulted in low yield. This was something which was amazing. But when we now took care of the variations in total leaf area or the total water that they use, or the kind of water that was the plants use, then when we now plotted the carbon isotope ratio with total biomass or with yield, there was a much stronger relationship in both groundnut and rice genotype. This was something which was a brilliant thing, that gave me a lot of energy and courage to the entire team that we can go ahead and do this. Then, I mean, in this case, what we are saying is when water use efficiency alone is not good enough, root rates or water use is alone not good enough, but is it possible that we can combine them? This is the question. We tried doing that by using genetic approaches. We identified a genotype which had high water use efficiency called Tanu, and then crossed it with a high root but low water use efficiency genotype from the germplasm called IET 15963. And this we developed into this was the work that I started with Timpa and Natarajan long back, and it was about 15 years back. And now we have understood that trade, and I think we can do this much faster these days. But then we developed the recombinant inbred lines. We identified some of the lines which had both the traits. That means when you cross them, the genetics says that there can be transgressive segregations as well as trait introgressions. So when we wanted to look at the trait introgressive lines, one of the varieties that Shiv Shankar was telling before we started, we named it as Daksha. One of the variety which we named it as KMP 175, Karnataka Mandya Paddy number 175, appeared to have something which which was amazing for us. It had better root system, as well as, like for example, you have it here. This is a Tanu, which was a bad rooter, and the root donor line is here. And when you combine them, several of them were like this, but one of them we released as a variety, and that variety is really doing very, very well now. And this has been released and notified at the national level. But what is interesting for me also to find out whether the physiological traits improved, we call them as a trait introgress. The line has better photosynthesis, better stomatal conductance, and better water use efficiency despite everything, and the yield is higher even under stress. This was the best part of it. Even when we put them to the aerobic condition, then this particular variety is doing very well. A lot of people who are working, I mean, there was one person in Magadi Taluk who had grown this. He didn't irrigate at all this particular season. The rainfall that we got, which was good this year, was good enough for him to get up to 25 quintals of rice per acre. I mean, that was brilliant. That was amazing. You know, for me to say that it gives you around 6 tons per hectare, but without irrigation. That's where the problem. That's where the beauty was. But we were not happy with this Daksha alone. We wanted to check if there can be something else that can be done. But we said that both root and water use efficiency are constitutive traits, and these constitutive traits will not do very well without the kind of acquired traits. Acquired traits are those who don't exist under normal conditions but are significantly upregulated when the plants experience stress, and they get primed by this, and then they will that will give them a big advantage for the constitutive traits to do their job, and that's where we harness the kind of thing. So what we did was to develop a methodology which was done a long time back when we were doing our masters in our department. One of my classmates called Uma, who's a postdoc and is a scientist in the United States now, developed a temperature induction response technique. One of my students recently, Dr. Pushpa, and screened a large number of germplasm for the induction tolerance. And we took some 150 germplasm lines from there and did an association analysis. And then we found that there is a very, very nice way, a high throughput way for the physiologists to look for the genetic traits. And then we identified some of them, and we don't have to go into these details here. We got some of these lines which were very, very, very significantly different from one another in acquired tolerance. And these acquired tolerance traits, we did a metabolomic approach, and then we identified some of the metabolism. Pushpa did this excellent work in association with National Chemical Laboratory in Pune, NCL Pune, with Tulsi Ram. And then she developed this data. And one of our, I think he's there in the audience. He was also, her husband, he was also responsible for, Raghavendra was responsible for analyzing this data very, very well. And using this data, we mapped it. We mapped it using association genetics, and we found one particular place where the reduction in the recovery growth was associated with two markers which were 1 centimorgan apart. That was brilliant. And we identified the number of genes that are there inside that, and Pushpa could map them to find out that there are two fantastic mechanisms, one responsible for the ABA associated things, and the other one metabolite that was there, which was an osmotic substance. So there was a place where the acquired tolerance traits associated with the genome was related to, or which was controlling some of the mechanisms were with water relations, as well as sustaining the metabolism under water limited conditions, because those genes that we talk about, GABA and others, were responsible for reactive oxygen species scavenging, and these metabolites led us to identify some of the lines, and then we got some of these lines like AC39020, and these lines have better root, better water use efficiency, and they are all in different individuals. Now the question was, I have a genotype which is giving me a much better induction response. There's another genotype which is good in root. There's a third genotype which is better in, let's say, the water use efficiency traits. Now the question was, how do we combine them? We can ask our children to go study hard and then become intelligent. Becoming intelligent was my problem, because my son cannot be intelligent if I am not. That's the genetics. But how is it that we can train our plants to do this? We can get them. What we did was another amazing story which was started by Mohan, Shashidhar, Parsi, and a lot of people have continuously worked on these things for their PhD with us. We had a panel of about 175 indica accessions, which we obtained from Sierra Raikata. and we genotyped them with SSR markers at that time. We can do the whole genome sequencing these days. And then we phenotyped them extensively for carbon isotopes, oxygen isotopes, root and water use efficiency, yield, and every trait. And then we did an association mapping. We had tremendous diversity for sure. Sorry, and then we did a population structure analysis and then associated them and then calculated all the false positives and false positive discoveries and all those kind of things which Raju was able to publish this in Molecular Breeding some 4 years ago. After that, what we did was to now do the job actually that is necessary for us to do. We did an elaborate program of crossing, and I should thank my breeder friend Dr. Rajana, who was associating with me through and through always. And then we developed a marker-assisted backcross breeding strategy for this. And that was the kind of thing which was very new when we started it. Now we do what is called as MAGIC, multi-parent advanced generation intercross. I'm sure geneticists like Lokesh and all are listening, they will definitely understand this. So we have a mechanism of doing this. What we did was to cross the high root donor line with a mega variety called IR64, which is a water-loving plant I told you in the beginning. And we crossed a we crossed a genotype, which is a water use efficiency donor line to IR64. How many minutes I have? Sure. And then we combined these crosses and developed an elaborate backcross breeding. We backcrossed it with IR64 and we used the markers for both foreground as well as the background selection. And then we ultimately got what we wanted is a trait introgress line which had about 98% of the genome of IR64. So the trait introgression lines looks exactly like the IR64, but it has better root like this genotype. Look at the grain size, grain type. You don't have the grain type at all in the introgress line. Neither this nor the root donor line we don't have. Therefore, we eliminated the bad qualities and we only have the good traits that we have in terms of water use and water use efficiency. And the moment we get that, we see this, that this genotype, the trait introgression line here, the gray bar, maintains better spikelet fertility under both well-watered and water-limited conditions. It maintains better water use efficiency, both under well-watered and water-limited condition. And therefore, it maintains much higher yield under water-limited conditions as well as well-watered conditions. This is something which is amazing, and we are trying to now look at that. Another student of mine recently, she has submitted her PhD thesis. Pooja worked on this analysis further. We put them in several places, and then we have the data on.
Dr. Shesha, another 5 minutes. Can you sum it up?
Sure. I need only 3 minutes.
Oh, okay. Thank you.
Thank you, sure. Thank you so much. So we have these different things that have been screened in various places to now understand what is the QTL environment interaction and what is the QTL interaction among these trait introgression lines. I mean, I should say this one word here. When we now did this, we had about 10,000 BC3F2 lines. And from that 10,000 BC3F2 lines, we have now developed about 260 lines, out of which 6 of them we have studied very extensively. And then it comes us to the feeling that most of the work that we did, which was painstakingly done over the past 12, 15 years, can be done very easily if you had a very, very novel, nice phenotyping facility where things can be very rapidly done and accurately done. And we developed that facility in our department, thanks to all our colleagues and seniors who are a part of this, who developed this. We have this, and you're welcome, whoever is wanting to come and see can come. And then we designed this completely indigenously, and this is a very cost-effective mechanism, and it's about, I mean, it will not cost the way it does in the one that we have, and the best part is it is outside. You know, the natural conditions can change here and all that. And I have a 2-minute video. I don't know whether it works. Is it working?
Yeah, yes, it is working.
Okay, so what we have is these are the high-end things that we talk about, and these high-end facilities are very costly and they need to be placed in very precise conditions. But what this is a laminar tech facility. I think IHR has one, IARI has, but we wanted to make it a simpler one. We developed a hybrid platform. And then this was supported by the ICR under the Niche Area of Excellence. We developed this very simple thing. What we have here is every container with a plant is sitting on a load cell balance, and we have an automated rainout shelter, and every individual is weighed. And then there is an automated irrigation facility and maintains the water level always constant, irrespective of the differences in transpiration. There is a real-time display of data, and then we can calibrate it at any time we want. All automated facilities are there. And it's all the data through Wi-Fi, Internet of Things. And then we also have a device called the Plant Eye, which is a 3D imaging system, and we can impose stress at will, and we can have cycles of stress to mimic outside. And this is very ideal for maintaining the specific status of water, and we can make measurements on anything. We can see my colleague Mohan Raju and Professor Uday Kumar. And then we can use it for many crops. Other than rice, we have done that in groundnut as well as other leguminous crops. And that led us to this kind of things, like to find out the varieties that we have been talking about for phenotyping and the trait in progress line that I told you about just now. So this appears to be the need of the scientists now that we are talking about. And to summarize the whole thing, I have now very clear, all of us have very clear understanding of the physiological basis of drought adaptation, and that will surely help crop improvement is our very strong belief. But phenotyping has been a challenge for physiology especially, but now with these kind of phenotyping platforms, I think phenotyping under natural conditions is now very much possible. And then the genetic resources such as the germplasm, the mapping populations, mutants and all that is available with the Indian teams now for sure. And the genomic based crop improvement approaches can be very, very strategically adapted is our hope. And then, looking into these kind of things, we and Dr. Anja Reddy and I, we had a big program to sequence a large number of germplasms of ragi. We have the genomic sequences, we have the phenotyping mechanism. I think why should anybody stop us from doing physiological breeding? And everything was funded by a large number of projects that we have been receiving in the department. I should thank the Department of Biotechnology, ICAR, and the Science and Technology. And recently the Karnataka government also gave us some funds for establishing the facility. And to all my national and international collaborators, I think TNAU, National Institute of Plant Biotechnology, like Amita and Ravindran gave talks today. We have Rohini, Prashant and all that in our National Fund project. And we have IARI. Look at this. Most of them that we are talking about are breeders here. And that's the reason why it looks very interesting for us. And I should really thank my group of students and who have been very dedicated and working very, very hard. I think we hate each other when the hard work is there. And a number of international collaborators like Shinyu and Paul Struth, which is very, they've been very, very good in terms of photosynthesis and other things. We have Kentaro and Masomi Hatakayama from ETH Zurich in Switzerland. And of course, Graham Furco and Richard Richards from Australian National University. And my dear friend Rachi Vasne, who's been very, very kind in supporting us all through. Thank you very much for your time, and thanks to the organizers and chairperson, and the co-chair. I'll be very happy if there are some questions. Thank you so much.
Yeah, thank you, Dr. Shesha, for a wonderful talk. And I know that you are very much interested in, probably we would like to hear you for more than 1 hour. I know you have the capacity to go on and on and on water stress and especially molecular breeding for, drought tolerance. That is your pet area. And trait introgression is again specific to you. And I am very happy. Actually, I would like to hear you more, but due to lack of time, we cannot have. So
yes.
And that is really interesting that your idea for trait integration with high water use efficient types with similar leaf area and then transpiration, then only it is correlated with the total biomass. So that is really very good. And if there are any questions, maybe a couple of questions, only 2 questions I think we can have because of a lack of time. People can ask. Anybody, please. Yeah, I think everybody has understood very good, and it is late also. Probably people are tired. So thank you, Dr. Shesha, for wonderful
Thank you so
talk.
much.
And
Thank
now,
you very much. Thanks.
Okay, bye-bye, sir.
Thank you.
Can I introduce the next speaker, Dr. Halesh? Is it okay? Yeah. Next speaker is Dr. Rahul Latte. He is from UPSC Sweden. That is Umeå Plant Science Center. He is working in the Department of Forest Genetics and Plant Physiology. Before joining Umeå, Sweden, he was in Max Planck Institute of Plant Molecular Biology, Molecular Plant Physiology. So he is very well versed with the molecular plant physiology. Today, he is going to talk on a tale of DUF 1068 family genes in cell formation in Arabidopsis thaliana and potential application in abiotic stress response. Dr. Rahul Latte, please. Hello, Dr. Rahul.
Can you hear me?
Yeah, we can hear.
Am I audible?
Yeah, audible. Go ahead.
Yeah. Thank you for nice introduction and organizers for giving me this opportunity. It is always a pleasure to listen to Shesha sir, and it was like thanks for stimulating talk, which makes my job a bit tough. But I have to admit that my talk will be a bit different than my previous speakers, and in the end, I will try to do justice to the theme in which I have been selected. So basically, I'm a part of this Umeå Plant Science Center, which is a joint venture of Umeå University and Swedish University of Agricultural Sciences. So basically, we are based at Umeå, which is like a capital city of a like Västerbotten County in the northern Sweden. We are like just 300, 400 kilometers south of Arctic Circle, which makes the climate very temperate. So I feel that there are only two things that we do here. One is mountain, like hiking in the mountains in the summertime, or like go to the mountains in the winter to see the beautiful northern lights. And apart from that, like it is just a job or work. So perfect place for our colleagues. Thanks to Emanuela, like who received this year's Nobel Prize in Chemistry for her discovery of CRISPR-Cas technique, which she did during her stay at MIMS under the Umeå University. And this is the only good news that we have so far in the Umeå in 2020. So coming to the science, like over a period of time, I have been involved in several projects, and these projects are mainly in the direction of understanding the cell wall formation, identification of new players, and I'm currently like applying that knowledge to understand how the differential growth occurs across the apical hook during the seedling emergence. But today I would like to talk about like the project which is like very interesting. And today I would like to talk like, you know, how we identified this gene family and how we characterized it for its role in cell wall formation, and why do I feel that it has a potential to unravel a new mechanism of stress or ion stress response. So I would like to begin with the basics that, as we know, the plant cells are surrounded by relatively thin but mechanically strong matrix polysaccharides, which are deposited outside the plasma membrane. And this cell wall actually imparts the structural strength, and it regulates the anisotropic growth of a cell by regulating the turgor pressure created by vacuoles, and plays an important role in defense mechanism, pattern formation, and cell-to-cell adhesion. So loss of proper cell wall leads to a defect in the growth, which can be seen here in the full-grown plant of the cellulosynthase mutant. Also the development of pollen, suggesting the importance of cell wall in growth and development. And it has been like a great raw source for like various industries like fiber, wood, jelly, and biofuel industry. So what is cell wall made up of? Basically, the cell wall is mostly made up of polysaccharides and to some extent proteins. And the content of the cell wall is a very tissue and cell specific. So as we know, there are two types of cell walls. Like one is the primary wall, which is deposited in the growing cells. When the growth is ceased, then we have a deposition of secondary cell wall in certain cell types. So coming to like a primary wall, it is mainly consist of 3 different polysaccharides: cellulose, hemicellulose, and pectins. So in the left side, you can see the scale model of primary wall of Arabidopsis leaf epidermal cell, and the different components that are shown in this diagram are in the proportion to the amount of cellulose microfibrils shown. So cellulose is the most abundant biopolymer available on Earth, and it is the main component which provides the tensile strength to the plant cell and determines the shape of the cell. And this cellulose is made up of like different microfibrils of like beta-1,4-glucan chains. And these cellulose microfibrils are synthesized by these hexameric rosette-like structures, which are present at the plasma membrane, can be seen by immunogold labeling. And the individual subunit of these cellulose synthase complex is the cellulose synthase A family proteins. Arabidopsis genome has 10 genes which encodes these family genes, family proteins. And primary wall cellulose synthase, cellulose is synthesized by the SESA 1, 3, and 6. And I would like to briefly talk about the secretion of these different components. As I said, like the cellulose synthase, which I will be calling hereafter SESAs, are believed to be assembled in the Golgi apparatus, which are then packed into the Golgi-derived vesicles. And then the one of the important component of the cytoskeleton that is actin plays an important role in its delivery to the plasma membrane. And at plasma membrane, another component of the cytoskeleton, microtubule, guides its insertion and then subsequent activity in the synthesis of the microfibrils. So this process is called secretion, whereas the internalization of the cargoes from the plasma membrane is called as the endocytosis. Unlike cellulose, the other matrix polysaccharides such as the pectin and hemicellulose are believed to be synthesized in the Golgi apparatus, which are subsequently modified, and this modified in the sense like especially pectins undergo methyl esterification, and these highly methyl esterified pectins are secreted to the plasma where they undergo a demethyl esterification, and then this matrix polysaccharide and the cellulose microfibril forms a complex structure. So the role of microtubule in the directional deposition of the cellulose has been studied in past, mostly using the basically the role of microtubule in the cellulose synthesis was mainly studied using the electron microscopy based experiments. But this theory was recently revisited in the David Erhardt's lab, where they subunits of SESAs and microtubule and co-express simultaneously. What they observed is that the SESAs localized to the plasma membrane, and it is observed that hides the cellulose. It was like a so 3 groups basically, the one of the group and even the Martin Brinkman from our group have shown that these 2 components are linked by a linker protein called a CSI1 or POM2, which interacts with the primary wall cellulose synthase complexes as well as binds to the microtubule in vitro. And in the absence of this linker protein, the SESA trajectories deviate from the underlying cortical microtubules, showing the essential role of CSI1 in microtubule-based SESA activity. So CSI1 shows a cellulose synthesis related deficiency, cellulose deficiency related phenotypes such as the reduction in the cellulose content and other phenotypes. And apart from that, they also show a twisting of the cell files in the hypocotyl as well as in the roots. And then the full-grown plant shows an altered phyllotaxis. So what is the phyllotaxis? It's a regular arrangement of lateral organs around a plant stem is called as a phyllotaxis. And Arabidopsis exhibit a spiral phyllotaxis. That means the divergence angle between the 2 subsequent calyx are centered around 137 degree, which is also called as a golden angle. So basically, we first characterized this phenotype in collaboration with Olivier Hamon's lab at INRA Lyon, and I just would like to highlight the findings in the graphical abstract. As I said, CSI1 act as a linker between the cellulose synthase complex and microtubules. In the absence, the CSI trajectory deviates from the underlying microtubules, which leads to a post-meristematic right-hand torsion of around 2.8 degree in the internodal region. And as we know, the new organs are initiated in the shoot apical meristem. So if the right-hand twist, if the twist and the pattern of organ initiation are in the same direction, we would expect a phyllotaxis of 180 degree. Whereas if they go in the opposite direction, we would expect a phyllotaxis of 90 degree. So basically, like then we calculated divergence angle of the silix from the population of CSI1 or a POM2, and we saw that like it has two peaks, one at 90 degree and another one as 180 degree, whereas the wild type had a peak at around 137 degree, which is like a golden angle for spiral phyllotaxis. So why I told you about this story is because, like, in recent years, the co-expression network and genetic screen have enabled us to identify new players in identification for their role in the related pathways. So this is a co-expression network of CSA3, which is derived from the RNet developed by Marek Mutwil from our lab. And if we have a closer look, what we can see is that the CSA3 is co-expressed with other cellulose synthase subunits, such as like CSA5, 6, 9, 2, which are involved in the which are a part of this hexameric rosette-like structure. And apart from these subunit like genes encoding these subunits, there are certain genes which are also co-expressed with them, such as the CSI1, which about which I just spoke, POM2 or chitinase, chitinase like 1, CC, that is a companion of cellulose synthase. So these genes have been characterized characterized and have shown that they have an important role in the cellulose synthesis along with the cellulose synthase complexes. This shows the robustness of the co-expression network. So if we have a look at this network, there are certain genes which are unknown genes. And for example, there are certain genes with a domain of unknown function 1068, which are co-expressed with these primary wall CSAs. So the objective was to like characterize these genes with a simple hypothesis that functionally related genes tend to be co-expressed together. So we began with in silico analysis, and as the name suggests, like nothing was known about these gene families. So these proteins are very plant specific, and the family contains just uncharacterized genes or proteins. And Arabidopsis genome has 6 genes which encode proteins with the DUF1068 domains. And the one that is co-expressed with CSA3 is we now call it as a NKS1, because like the mutant of this gene shows the sensitivity to sodium and potassium ions. So, but I will come back later, like how we can, why this gene has a potential in the stress tolerance later. So the NKS1 has a close homologue. We call it as a NKS2, with which it shares the similarity of 85%. And if we look at the protein structure, it has just N-terminal transmembrane domain with a huge unknown domain. So as CSAs, CSAs are expressed throughout the different developmental stages. NKS1 also showed an expression throughout the different developmental stages and the tissues, which was seen in the GUS reporter assay when the GUS was expressed under the NKS1 promoter, and the histochemical assay shows the ubiquitous expression in the both light grown as well as dark grown seedlings. And then we began our wet lab experiment with identification of mutant alleles for these genes. So we ordered 2 tDNA insertions from the public database, and then we did a RT-PCR and a QRT-PCR. And the NKS1 transcript was significantly reduced in these 2 alleles, and these 2 alleles were subsequently used for phenotypic characterization. So cell expansion is one of the important feature that we consider to screen any mutants which are involved in the cellulose synthesis. So basically, what we observed is that 3-day-old and 6-day-old seedlings show a reduction in the hypocotyl elongation as well as the root growth. And if you have like a closer look at the apical region, so what we observed apart from this cell elongation, the mutants also display a shorter hypocotyls and the with fully open cotyledons. That means they have a hook defect. So these mutants were subsequently complemented with NKS1 fused with GFP, both C-terminal and N-terminal, and we could able to complement this phenotype, suggesting that the NKS1 is not a hypothetical protein, but a functional protein. So as it is co-expressed with the SSA3, so we wanted to study the dynamics of the cellulose synthase, that is the SSA3, in the NKS1 mutant background. So basically we studied the insertion of the SSAs at the plasma membrane, the density of the SSAs at the plasma membrane, and the velocities of the SSAs at the plasma membrane. And surprisingly, like we did not find any change in the like any significant change or like reduction in the velocity, density, as well as insertion of the SSA particles at the plasma membrane, even though the mutant showed more slow-moving SSAs compared to the wild type. And there was no significant reduction in the cellulose content in the mutant. It was a bit disappointing, but yeah. So the cellulose and pectin related mutants also show another phenotype of like a reduction or a defect in the secretion of the seed mucilage. And the seed mucilage are rich in pectin secreted from the seed epidermal cells. So upon hydration and shaking, the mucilage, the inner layer of mucilage is released, which is acidic in nature. So ruthenium red stains this mucilage, and we can see that Arabidopsis has an halo-like structure around the seeds, whereas the mucilage was completely absent in the NKS1 mutant background. So it shifted our focus from cellulose to the pectin, and also like the epidermal cells of hypocotyl show a cell adhesion defect. And this cell adhesion defect is mainly due to the cell adhesion is basically mediated by homogalactrons like the pectins, especially the homogalactron calcium, the bridge, which plays an important role in the adhesion. So any changes in the pectin content, modification or demethylesterification leads to a defect in the cell adhesion. And then subsequently, we estimated the uronic acid content in the mutants, and what we observed that the uronic acid was drastically reduced in the mutant compared to the wild type. So then we revisited this co-expression network using NKS1 or a DUF1068 gene as a bait, and we observed that it not only co-expressed with cellulose synthesis related genes, but also with the pectin and some trafficking related genes. So what is pectin? So it's another matrix polysaccharide, but you have to believe me that like it is one of the most complex matrix polysaccharide, which consists of like different diverse and complex polysaccharides. The backbone of the pectin is a linear chain of alpha-1,4-D linked galactronic acid. And there are and this pectin plays a very important role during the cell wall integrity during the stress response. And there are three different types like homogalactrons, rhamnogalactron 1 and rhamnogalactron 2. And the rhamnogalactron 2 is a most complex structure. Basically, it has like, it has like a side chain of 12 different glycol residues linked together with at least 21 different linkages. And any small change in this structure of RG2 has a severe effect, defect or like effect on the cell wall growth. Then we made use of like cell biological tools. So we studied the NKS1 localization, and we used this GFP tagged version, both C-terminal and N-terminal, and using spinning disk confocal microscopy. What we observed is that unlike SSAs, the NKS1 was localized to spherical bodies with a cytoplasmic streaming. And when we did the Z-stack imaging, we did not see any signal at the plasma membrane. So it was clear that it localizes to the endomembrane system with some cytoplasmic streaming. So what are the endomembrane organelles? Like in plants, we have different compartments with distinct functions, such as the endoplasmic reticulum, Golgi apparatus, transgolgi network, prevacuolar compartment, recycling endosomes, vacuoles, and plasma membrane. So in order to find its precise localization, we crossed the fluorescently tagged NKS1 with different compartment-specific markers, and the dual lines expressing these markers were studied for co-localization. And what we observe is that NKS1 co-localizes to the Golgi apparatus, as well as it associates with trans-Golgi network. Even though Golgi and trans-Golgi network may associate with each other, but they have a different function. So how do you find out then? So we make use of like a drug called brifildin A, which blocks the secretion from trans-Golgi network, and it like upon a BFA treatment, the TGN or like endosomal proteins form the aggregates called as the BFA bodies. So we used like a dual line of trans-Golgi network and the Golgi crossed with NKS1. And what we observed is that the TGN marker was a part of BFA aggregate, whereas the NKS1 was showed a ring-like structure surrounding this BFA body, and it was very clear that it co-localizes with the Golgi marker upon a BFA treatment, suggesting that NKS1 is a Golgi protein and may be associated with the trans-Golgi network. So then we studied the like endocytosis, that is the internalization of proteins from the plasma membrane, and we made use of a dye called FM464, which is the lipophilic, and it follows the normal route. And en route, like, you know, it stains a different compartment before it reaches to the vacuole. So we treated the Arabidopsis roots with a BFA for 30 minutes and then stained with FM464 for 5 minutes. And we estimated the ratio of fluorescence intensity of BFA body to the plasma membrane to deduce the rate of endocytosis. And what we observed is that rate of endocytosis was not, or like endocytosis is not affected in the NKS1. You know, like while doing this experiment, we somehow forgot to remove the seedlings, and we stained them like over a long period of time with FM464. And what we observed is that the vacuolar structure in the NKS1 mutant is disrupted or like changed compared to the wild type. And then we studied the secretion of proteins, and we tried like different markers, but I have a result of PIN2, which is an auxin efflux carrier, which localizes to the Arabidopsis roots in a polar fashion. So upon a BFA treatment, the plasma membrane signal is internalized, and we see the PIN2 signal in the BFA bodies. We did this treatment for both wild type as well as in the NKS1 mutant background. And then we did a washout experiment that for 60 minutes and 120 minutes. So what happens after 2 hours is that most of the signal from the from the internal compartments is recycled back to the plasma membrane in the Arabidopsis wild type. But whereas these mutants, there was like a defect in the recycling of the PIN2 to the plasma membrane. It was very interesting for us because there are certain genes or like mutants which showed such kind of defects, especially like echidna and other the mutants which are, or the corresponding genes which are involved in the secretion of pectin secretion to pectin to plasma membrane. And then like finally, like what we did is like we did transmission electron microscopy, and what we observed is the two important observations. One was the cell wall was deformed in the mutant compared to the wild type. And the Golgi ultrastructure was totally changed in the mutant. For example, in wild type, we have stacks, whereas the mutant showed the round Golgis surrounded by small vesicle-like structures. So taken together, all these data, what I can say is that co-expression could be a robust approach to find new genes which are involved in the related processes. And NKS1 has less or no role in the cellulosynthesis. And it is like ubiquitously expressed and localizes to the Golgi apparatus. The mutant shows the defect in the etiolated seedlings growth. It has the reduced pectin content and also defect in the cell adhesion. And like basically the cell wall and the Golgi morphology is altered. The endocytosis is not affected, but the recycling is delayed in the mutants. So currently, like I have listed some experiment as an outlook, but basically we are in the middle or about to finish this experiment. So basically, like we are characterizing like the hook phenotype of the NKS1. We are studying the genetic interaction of NKS1 with some of the proteins like that we know that involved in the secretion of pectin to the plasma membrane. And we did some yeast-to hybrid to identify some interactors. And we are currently doing a biopsy to decipher the precise function of the NKS1. So why do I say that it has a potential role in abiotic stress tolerance? Because when we were like characterizing this gene family, there was a publication where they identified the mutant of this gene showed an increased sensitivity to actually, in fact, it increases the sensitivity of SOS3 allele to NaCl. Because what if I understand, like, you know, when I read this paper, what they were trying to do is like they were trying to do an genome-wide screening in the background of SOS3. So they characterized several, they were looking for a tDNA insertion in the SOS3 background, which shows increased sensitivity or like suppression to the NaCl. And then what they observed is that NKS1 shows it enhances the sensitivity of SOS3 mutant allele. So, and then the conclusion from their paper is that, like, you know, unlike SOS mutants, the NKS1 shows the sensitivity to sodium and potassium, whereas the SOS genes, as the mutants or alleles, shows the sensitivity to sodium and lithium ions. They showed that it is localized to the ER based on the transient expression, but now we know that it is a Golgi localized and has something to do with pectin synthesis. And they propose that there could be like, there is a connection between the ion stress tolerance and the endomembrane, which is not known. And but like, then how do we proceed, or like, how do we use this gene? So there was like a recent publication where from the Husaydineni's lab, where they show the role of Feronia in maintaining the cell wall integrity during the salt stress. So Feronia is a receptor-like kinase which sense the change in the, which sense the salt stress, and this further activates the downstream signaling, which leads to, like, you know, activation of calcium transient, which eventually leads to like repair of the cell wall, which triggers the repairing of the cell wall. So yeah, with these two like background, I propose that NKS1 could be involved in this maintaining cell wall integrity during stress response, but a long way to go. And with this, I would like to thank my supervisor Stefan for giving me an opportunity to work on these projects, Rene and Heather for doing TEM-related experiment, Rishi for allowing me to continue that work, Neil Snood, like a graduate student who is helping me with BIFC, and our collaborators, especially Olivier and Berit, for doing cell wall analysis, Jose Alonso, Malcolm Bennett, and Bruno on another project. I thank the Green Team, Media Kitchen, Microscopy Unit, and IT section. And I thank you all for your kind attention, and I will be happy to take suggestions and answer questions if you have any. Thank you.
Thank you, Dr. Rahul, for the nice basic science related talk. It is really very much, it is really interesting to have certain basic science knowledge to, do such work. And I congratulate you for the wonderful work, and especially highlighting the NKS involvement in cellulose synthesis and the ionic balance as well as pectin synthesis. We know that ionic balance is very important for abiotic stress tolerance. So it may be involved in sodium, potassium, ionic balance also. So thank you very much for the wonderful lecture. And if any questions are there, one or two questions, please, audience are welcome.
Person, can I ask? I'm Shesha.
Yes, go ahead, Shesha.
Rahul, thanks very much. Very good. Well done. And it's a good data that you have shared. And I have some, this question that I have. We've been looking at two things. One is the root growth during stress, for which the cell wall has to loosen and then the root has to expand. Now, this is not the absolute growth, but then the responsive growth. The moment you put them to stress, the roots are longer. And the same thing happens when there is first phase starvation. First phase starvation also makes the root to grow longer. Now, I have two things. One is this pectins as well as the cellulose loosening and then building it once again perhaps can help in the cell expansion and the growth of the root, number one. But when it comes to the tolerance to abiotic stresses like drought or salinity that you're talking about, more than the cell wall, the cell membrane will have a number of ways to go and sort of respond and then manage the stress at the cellular level. What's your opinion on that?
Yes, sir. Yeah, maybe like this is a new field for me, but like what I say is that based on my knowledge, the cell wall integrity, like the sensing, because like upon a salt stress, it is a like wall which is first affected. So basically, the cell wall undergoes like repair mechanism or like, and then the plasma membrane localized receptor kinases sense these changes in the like wall, and then they activate like a downstream mechanism which are involved in the which activates the cell wall integrity maintenance. So I feel that like, okay, I mean, like we don't know it because, but based on a few studies like Feronia, which clearly shows that like maintaining the cell wall integrity is also very essential during the salt stress.
Okay.
Thank you. If no more questions, I thank Dr. Rahul for the very inspiring lecture. Thank you very much.
Thank you, sir. Thank you, sir.
Now we move to the oral presentations. I hope everybody is there. The first presentation is by Dr. Sengutuvel. He is a scientist from ICR, Indian Institute of Rice Research, Hyderabad.
Sir, Shivshankar sir,
Yeah,
instead
please.
of Senaguttuvel, his student Mr. Madhusudan will present. He is there, he has logged in.
Okay, no problem. Yes, no problem. His topic is improvement of APM-S6B for phosphorus use efficiency by introduction of PUP1 and evaluation under phosphorus starved field conditions in rice. Please.
Excuse me, sir. Where can I? My file is on desktop. Share screen.
Yeah, please.
From the Zoom, you select the share screen option, which is in a green color.
Okay.
And now you choose the PPT from your desktop.
Madam, it is showing basic advanced files.
Next Have you opened it, sir? Open it, your PPT open on the screen and make it minimize. And later come to the share screen option where you can choose.
First open your PPT.
Okay, sir, I opened.
Yeah, then go to the share screen.
It's coming.
Yeah.
Yes,
put it in slide mode.
Yeah, sir.
Yes, good, very good. Go ahead.
Sir, due to my advisor, Dr. P. Sangitwal was busy with the GHMC election Hyderabad. So instead of him, I'm going to present. He's student, PhD student. It is my part of research work. So myself, Dr. Madhusudan, I worked in DRR Hyderabad hybrid rice section. So we worked on improvement of APMA 6P for phosphorus use efficiency by introduction of PUP1, that is phosphorus uptake 1 gene, and evaluation under P-star field condition in rice. So rice is one of the most staple food crop in South Asia, and the heterosis phenomena is well exploited in hybrid rice, but the still the area under hybrid rice is only 6% of the total rice area, that is 3 million hectare. So we know that hybrids in almost all the crops, they are well response to high input fertilizers. So India is making is giving more subsidy for the phosphatic fertilizers every year. So and India is the largest producer and consumer of fertilizer. So among them, the phosphatic fertilizer is one of the fertilizer where there is no alternative fertilizer for phosphorus. So for it is a non-renewable source for phosphorus. So hence, in Indian soils are low to medium phosphorus condition in soils. So phosphorus deficiency in Indian soils is very high due to high fixation in both acidic and alkaline soil conditions. So phosphorus deficiency in rice leads to stunted growth and reduced tillers, and the flowering will be delayed and maturity will be delayed. So in rice there are a lot of genetic variability for phosphorus use efficiency has been identified and exploited. So there will be a scope for genetic improvement, and by ERI people, they identified the QTL controlling the low tolerance to low phosphorus. So they identified the phosphorus uptake 1 gene. So and they also characterized the PUP1 QTL, and they identified the gene specific markers for the PUP1 QTL. So this help, and also so many workers they integrated into the elect rice varieties, and also in India by DRR in Sundaram sir lab where they transferred him to MTU 1010 variety. So they introgressed lines with PUP1 are reported. So the candidate gene for QTL has been cloned by ERA, and they identified the gene that is P-stall1, that is a phosphorus starvation gene, which is responsible for root growth under the low P conditions for phosphorus uptake efficiency. So in our study, our main objective is to introduce the PUP1 QTL in the genetic background of improved APMS 6B, which is a maintainer line for DRRH3 hybrid, and evaluation of elite introgressed lines with PUP1 QTL under low phosphorus soil condition and normal soil phosphorus condition. So we used the recipient parent improved APMS 6P, which is an essential derived variety of APMS 6P for DR, which is a parental maintainer line for DRH3, which is very MS grain type, that is medium slender grain type indica rice variety, which is excellent cooking quality, which is well known for excellent cooking quality, and it has having 2 bacterial blight genes has been introduced, that is XA21, XA33. That was developed in DRR only under LASR lab. So it is a response very well in hybrid seed production. And we use a donor parent as a backcross 1F3 population derived from the cross between APMS6P and Kasalat. The Kasalat is Indian ash type variety where they identified, Erie people has identified the Papuan QTL in Kasalat. So our crossing plan we developed backcross 1F3 population, 46 lines by conventional backcross breeding. So we genotype the backcross 1F3 population in low phosphorus soil condition, and we identified the tolerant one in low phosphorus with phenotypically and genotypically we identified them. So we used them in the intercross with the improved APMS6B for developing the intercross F3 population. So these are the molecular markers we used in marker backcross, and the POP1 gene which is located on chromosome 12 of the rice, and the XA38 gene which is located on chromosome 4, and XA21 which is located on chromosome 11. So the backcross 1F3 lines derived from the APMS6P and Kasalat were screened genotypically and phenotypically under low P soil condition, and simultaneously they are genotyped for RF4 and RF3 markers, which are negative selection was employed. So in that one, the best and early flowering ATR 594-1 was used as a donor to intercross with the improved APM6B. And these backcross 1F3 population also we characterized, we crossed with the APM6A female line to check whether they are sterile or fertile. So we came to know that they are sterile, though, so hence they are well good maintainer. So these are the gel pictures we screen for POP1 specific markers, and also the RF4 and RF3 markers. So we found among 43 intercrossed 43 derived lines, we found 11 were positive for POP1, and among them one was used as a donor to intercross with the improved APMS6B. We developed 25 intercross F1s. So we genotyped them, and we found 9 were true F1s. So these 9 true F1s are we did parental polymorphic survey with 111 polymorphic markers. We found that the BP10 one true F1 was found 88.29% of the background genome recovery. We found that line was forwarded to the intercross F2. This is the GGT graph for background genome recovery percentage. So in intercross F2, we saw shown the intercross F2 among 252 plant population size. So we inoculated the inoculum for bacterial blight gene. So from the DRR lab, pathology lab, using IXO pathogen cultivar. So we found the lines which are stronger resistance with BLB score of 1. We selected them for genotyping with POP1 markers, and which are all showing positive for POP1 markers. For them, we did a recombinant selection. So from that recombinant selection, we also did check for the presence of XA21 and XA38 gene. So we found the four lines which are having positive for all the 3 genes, and we evaluated them under low phosphorus condition in DRR, where the soil phosphorus condition is less than 1.5 ppm. This plot was maintained for 15 years without adding any phosphatic fertilizers. And also we planted in normal soil phosphorus condition, where the soil phosphorus was more than 20 to 25 ppm range. With Wolfson P method, we calculated the soil phosphorus availability of soil phosphorus. So, we did with RCBD design with three replication. We found the significant differences with the introgressed lines for productive tillers and spikelet fertility. So, and also in yield, we found good compared to recurrent parent improved APMS 6P under low P soil condition. So we did root length and root volume study by uprooting the plants at a boot lift stage, where with mass by volume discharge. So we identified the root volume was calculated, and we found good root length and root volume compared to the improved APMS 6P. And we did the stress susceptibility index and percent yield reduction, and we found that the BP10-5 was the line which showing highest less yield reduction and highest stress tolerance index. So we thought that that will be a good index for the selecting for low phosphorus soil condition.
Dr. Madhusudan, can you,
Yeah, sir.
1 minute.
So, and we so the conclusion, we thought that development of heterotic hybrids with phosphorus use, with high phosphorus use will greatly reduce the application of P fertilizers, that is 3 to 4 times once in after 3 to 4 growing season. So it will reduce the cost of cultivation, and identified maintainer and derived heterotic hybrids with puffin and bacterial blight will be a great helpful for higher yield under bacterial blight prone areas, and in acidic and alkaline soils and water limited conditions where P availability is very poor. And I'm acknowledging my research work with the my professor at PGTSU and the scientist Sundaram sir and Subramaniam sir and Ananta sir and Krishnamurthy sir at DRR, and all the lab colleagues at Sundaram sir and hybrid rice section and ICR and DRR. Thank you, sir.
Oh, thank you, Dr. Madhusudan, for the wonderful work. And definitely it will help growing these type of varieties under phosphorus deficient conditions as well as under water limiting conditions. Thank you very much. And I have time, I think, maybe for one question or 1 or 2 questions, if anybody wants to ask. Yeah, if there are no questions, thank you, Dr. Madhusudan, for the wonderful presentations. We move on to the next speaker. You please stop slide sharing. Next speaker is Dr. Anjani Kumar. He is a scientist, Crop Production Division, ICR, National Rice Research Institute, Cuttack. And he will be talking on path modeling approach for assessing effects of elevated carbon dioxide and water deficit stress on nitrous oxide emission and soil enzyme activities in tropical rice soil. Dr. Anjani Kumar, you have 10 minutes, please.
Thank you, sir. And sir, am I audible?
Yeah, audible. Yeah, go to the slide mode.
Yes, sir.
Slide.
Yes, sir.
Oh, thank you.
Okay, sir.
Okay, fine. Go ahead.
Good evening to chairman and co-chairman of this session and my listeners. Myself, Anjani Kumar, scientist at National Rice Research Institute, Cuttack. And today I will be presenting on the topic path modeling approach for assessing effects of elevated carbon dioxide and water deficit stress on nitrous oxide emission and soil enzyme activities in tropical rice soil. And here we see the Indian rice scenario, that rice is grown by around 56% of farmers, and it is a livelihood of around 40% of the rural people, and it feeds around 65% of the global population, and the annual value of rice crop is around $53 billion, and it emits around 18% of the greenhouse gas from agriculture. This slide shows the importance of rice in Indian economy as well as the global economy. And we all know that the water availability is decreasing, and under that condition, we have to grow rice to feed the tummy of all the population. And here we can see that we are already in water stress situation, and we all know that rice consumes around 80% of irrigation water. Its water requirement is around 1500 to 2000 millimeter, and under this condition, we have to grow rice with limited water, and we don't want to compromise with the yield. In such a situation, aerobic rice may be a possible solution, and we have the challenge of maximize water productivity. We also know that rice fields emits around 18% of the total global greenhouse gas, and there are irrigation methods like SRI, alternate dry and wet, mid-season drainage, and intermittent drainage. Reduces the methane emission, but at the same time they enhances the nitrous oxide and carbon dioxide emission. Therefore, in such conditions, the major challenge is how to minimize the greenhouse gas emission from rice fields, and also to reduce the overall global warming potential. And under the increasing elevated CO2, under the increasing atmospheric CO2 condition, we can see in this graph that the CO2 concentration has risen from 280 to 400 ppm, and by the 2050 it will rise up to 500 ppm, and by end of 21st century it will go around 700 ppm. We all know that rice is a C3 plant, and its CO2 saturation point is 1000 ppm. Thus, there is a potential, there is a chance that under the anticipated elevated atmospheric CO2 condition, the rice grain yield can enhance. And this was a paper published by our group. And here we can see that under elevated CO2 condition, there is a decrease in nitrified population, and it also decreases the soil ammonical nitrogen content, nitrogen mineralization, and microbial biomass nitrogen. At the same time, elevated CO2 increases the denitrified population, and it increases the nitrate nitrogen condition. And under this leads to the that under elevated CO2 condition, it promotes anoxic condition, stimulates denitrification, and hence increases the nitrous oxide emission. And keeping these things in mind, we conducted our experiment in split plot design. Under main plot, we have taken two water treatments. One was continuously flooded, where 5 centimeter of standing water was maintained, and the other plots where water deficit is stress, and tensiometers were put in that plot, and irrigation was given when the tensiometer reading showed minus 60 kilopascal. And there were 3 nitrogen treatments, that no nitrogen, nitrogen at the rate of 100 kg per hectare, and nitrogen higher dose 150 kg per hectare. And this experiment was conducted under open top chambers where there was a facility of CO2 elevation. And coming to the results, we can see that there was a decrease in nitrified population under elevated CO2 condition, and nitrified population increases with the nitrogen dose, and it was higher under well-watered condition. Similarly, there was an increase in denitrified population under elevated CO2 condition, but it increased with nitrogen dose, and it was higher under well-watered condition. And coming to the microbial biomass nitrogen, it increased with the CO2 concentration, and it also increased with the nitrogen doses. And similarly, the ammonical and nitrate nitrogen, it decreased with the increase in CO2 concentration, and it increases with the increasing nitrogen dose, and it was higher under well-watered condition. This is the slide of nitrate nitrogen content. And we also calculated the nitrogen mineralization and nitrification potential. And we found that it was lowest under elevated CO2 condition, but it increased with increasing nitrogen dose, and it was higher under well-watered condition as compared to the water deficit stress condition. We also examined the soil soil enzymes like dehydrogenase, FDA, beta glucosidase, and urease enzymes. and they all were higher under elevated CO2 concentration, and they increased with increasing nitrogen dose, and they were higher under well-watered condition. And then we also estimated the nitrous oxide emission from the rice field. We found that under elevated CO2 condition, the emission of nitrous oxide was higher. And this is the nitrogen samples were taken at an interval of 5 days, and this is the daily flux. Here we can see that it was higher under elevated CO2 condition, and it was again higher under higher dose of nitrogen, and it was higher under a well-watered condition. This M1 is well-watered condition, this blue line. And then here we also see that the seasonal emission of nitrous oxide was highest at both the growth stage, that is maximum tilling stage and panicle initiation stage. At C3 stage, it means the elevated CO2 stage. And again, the seasonal emission was also highest. Seasonal emission is the cumulative emission. It was highest for a higher dose of nitrogen, and again it was higher under well-watered condition as when it was compared with the water deficit stress condition. Then the PCA bioplot, we constructed the PCA bioplot using the scale data set, and it explained, the PC1 explained 66% and PC2 explained around 22% for ambient CO2 conditions, and around the PC1, and for ambient CO2 conditions, the PC1 explained around 61% and PC2 14% of the total variance. Under ambient CO2 concentration, nitrous oxide emission was higher under panicle initiation stage, and it was positively correlated with nitrogen mineralization, nitrifier, and denitrifier population. And under elevated CO2 condition, that is a 550 ppm, the parameters demonstrating higher positive correlations were MOA gene, nitrifier, and denitrifier population under panicle initiation stage. With further increase in CO2 concentrations, the highly positive correlated parameters under panicle initiation stage were nitrifier and denitrifier population. And at 700 ppm CO2 stage, we found that NP, that is nitrogen nitrifying potential, and nitrogen mineralization decreased with increase in CO2 concentration, which was accompanied by decrease in ambient and soil nitrogen fractions. We did the PLS-PM model, and we found that with increasing concentration of atmospheric CO2, the influence of microbe on N2O emission and N-min increased, whereas the influence of N-min on nitrogen mineralization or nitrification potential decreased. And under elevated CO2 conditions, the influence of different forms of nitrogen like nitrate and ammonical form on nitrous oxide emissions showed a negative trend with increase in CO2 concentration, which might have affected the denitrification rate under elevated CO2 levels. And this PLS-PM model further revealed that microbes, the nitrifier and denitrifier, directly affected the nitrous oxide emission from the rice soils, and the effect was more pronounced under elevated CO2 concentration as compared to ambient CO2 levels. We also worked out the prediction models for nitrous oxide emission, and under maximum tilling state, the model involved these parameters like CO2 concentration, nitrification potential, nitrogen mineralization, FDA enzyme, nitrate nitrogen, ammonical nitrogen with these coefficients. And similarly for panicle initiation stage, the equation was like this. And we found R square for the equation at maximum tilling stage 0.79, and the R square for panicle initiation stage was 0.62. From the predictive models, we found that the impact of FDA on nitrous oxide emission was relatively higher as compared to other variables under both maximum tilling and panicle initiation stage. This model suggests that with rise in 1 unit of CO2 concentration, the nitrous oxide emission is increased by 0.002 unit at maximum tilling stage and 0.001 unit at PI stage. This suggests that the maximum tilling stage of rice is going to be affected more under the anticipated elevated CO2 concentration for nitrous oxide emission. And the higher coefficient values of different parameters like N-min, FDA, N-nitrate, and ammonical nitrogen provides evidence that these parameters had greater influence on nitrous oxide emission as compared to CO2 levels. And then coming to the grain yield under combined effect of water deficit stress and elevated CO2, we could publish this data in field crop research. And we all know that under elevated CO2 condition, there is a reduction in the stomatal opening. This reduction is followed by the increased net photosynthesis, lower transpiration, and improved water use efficiency. And here we can see here that under well-watered condition, under ambient CO2 condition, there is a yield reduction of around 33% when the moisture condition here, this is well-watered condition and this is water deficit stress condition. And ambient CO2 condition, there is a reduction in yield. And here under elevated CO2, this is well-watered condition under elevated CO2, and this is water deficit stress condition under elevated CO2. Here again there is reduction in yield, but the reduction is lower as compared to the ambient CO2 condition. Thus, we can conclude here that under elevated CO2 condition, the effect of water deficit stress on rice grain yield is going to be curtailed. And here we can see the water productivity. Under well-watered condition, the water productivity was like this, and the CO2 effect was 29%. But under water deficit stress condition, the CO2 effect on water productivity was 46.8.
Dr. Anjani Kumar, can you conclude in 1 minute?
Yes, sir. Yes, sir. And then we come to the biochemical responses of rice under elevated CO2. Here we can see the electrolyte leakage increased under water deficit stress as compared to the well-watered condition, and the relative water content was also increased under elevated CO2 condition, and leaf water potential also followed the same route. And here we can see the proline content. We can observe here the increase in osmolyte concentration under water deficit condition as compared to well-watered regime at all crop growth stages, and the increase in proline content under water deficit regime was significantly lower under elevated as compared to ambient CO2. And similarly, the same pattern was followed by catalase and peroxidase concentration in rice crop when it was subjected to water deficit stress and elevated CO2. And our salient findings are the atmospheric CO2 enrichment condition has the capacity to mitigate the negative effect of water deficit stress on rice soil. The increase in gaseous loss of nitrogen under nitrous oxide under elevated CO2 environment was significantly correlated with the increased denitrifier and decreased nitrifier population. The predictive models for N2O emission developed in this study provides evidence that the effect of varying CO2 levels was more pronounced at vegetative stage. And these are the research leads for future research work. And the breeders can identify appropriate donor which harbored higher nitrifier population in the rhizosphere soil so that the N2O emission can be minimized from the rice fields. And there is a need of further research for enhanced understanding of the mechanism of soil-plant microbe interaction. And I am thankful to my director, and thanks to all of you.
Thank you, Dr. Anjani Kumar, for sticking on to the time given to you. And it's a wonderful work with respect to greenhouse gases. Definitely, we have to look forward to reduce the greenhouse gases. Now we know that the global temperature is increasing at a very fast rate. So in that direction, your work is really very good. And I invite maybe 1 or 2 questions. Anybody from the audience, please. Yeah, if there are no questions, then I thank Dr. Anjani Kumar. Thank you very much for your presentation.
Thank you, sir.
Now the next speaker is Dr. Senthil Kumar, KM Scientist, ICR Central Tuber Crops Research Institute, Trivandrum. And his topic is genomoid identification, classification, collinearity, and expression analysis of MEHSP 70 family genes in cassava. Dr. Senthil Kumar, please.
Thank you, sir. Respected sir, co-chair, conference delegates, good evening to all. I'm going to present my research work on genomoid identification, classification, collinearity, and expression analysis of HSP 70 family genes in cassava. Cassava is a perennial woody shrub. It is mainly cultivated as an annual crop for its starch-rich edible storage roots. It is an important staple food for around 800 million people. Still, it remains as an essential part of diet in many tropical countries, including India. For instance, in southern states like Kerala, you can find in many households, people used to consume cassava at least one servings per day. It is third biggest source of carbohydrates after rice and maize in tropical regions. Cassava is a hardy plant. It has ability to grow under drought condition where other crops fail to grow. So it is considered as an important reserve of carbohydrates to relieve global famine. The starch obtained from cassava remains as ideal feedstock for various industries, which include food industries, biomaterials, animal feeds, biopesticide, and bioenergy. For instance, the sabudana, sago, which is used in making payasam and kheer, is made from cassava starch. Government of India has included cassava as a biofuel crop under National Biofuel Policy 2018. Because of this, the crop is also gaining importance as a biofuel crop. So identification of agronomically important genes are highly imperative for development of climate resilient cassava genotypes. HSP70s are ubiquitous molecular chaperones that plays important role in various cellular functions. It affects all aspects in the life of proteins, starting from synthesis to degradation of the proteins. These are all some of the roles of HSP70 in the cellular pathways. For instance, during protein synthesis, HSP70 bind to the nascent proteins. It protects the nascent protein from misfolding. It is also involved in oligomerization of the protein complexes, and it is also play a main role in protein translocation between the organelles. For instance, from cytoplasm to mitochondria, as well as vice versa. And it is also playing role in determining the fate of non-native stage of native proteins. It may decide as per the needs whether it has to be refolded or degraded. And it is also involved in the process of apoptosis. And it is also playing main role in heat shock response. HSP70 is a main player for induced heat shock response by regulating the heat shock transcription factor HSF1. It is also playing an important role in unfolded protein response. HSP70 has three functionally conserved domains, which include highly conserved nucleotide binding domain, and C-terminal substrate binding domain, and the linker domain, which links both the domains. In HSP17 proteins, nucleotide binding domain plays a role in binding of ATP, whereas substrate binding domain plays a role in binding of the substrate. Several classes of HSP70 genes are known to play important role in plant growth and development in many plant crops, which includes Arabidopsis, rice, wheat, and maize. Minimal efforts have been made to study these genes in cassava. Thus, understanding the function of these genes in cassava will help to develop climate resilient cassava genotypes. This is the workflow we have followed for identification of HSP70 genes in cassava. Cassava genome was sequenced in 2018. We have downloaded the genome from the public domain. We had used in-house built profile HMO search for identification of putative domains, which is having HSP70 proteins. Further, cluster omega as well as SMART tool was used to remove the pseudogenes. Finally, our analysis led to identification of 22 HSP70 genes in cassava. This is the chromosomal distribution pattern of these genes in cassava. Out of 18 chromosomes, HSPs are distributed in the 11 chromosomes. Chromosome number 7 has the maximum of 5 HSP70 genes in cassava. To classify HSP70 genes, we had did phylogenetic analysis of these genes in malpighial genomes. So far, around 4 genomes has been sequenced from malpighial orders. We did genome-wide analysis to identify HSP70 genes in all the 4 genomes. Our analysis led to identification of 13 HSP70 genes in castor, 28 HSP genes in salix, 25 HSP70 genes in populus. All the malpighials HSP70 genes, along with Arabidopsis, has been used for construction of phylogenetic tree. Phylogenetic tree construction has led to grouping of these family genes into 2 groups and 8 subgroups. Here you can see the conserved pattern of these genes among different plant genomes. And if you see the domain structure, malpighial genomes HSP70 genes is highly conserved as compared to the Arabidopsis HSP70 genes. This shows that these genes are highly conserved across all the plant genomes, and most probably the function is also highly conserved. So whatever function that was discovered in other crops might play in cassava also. So we have also studied the synteny relationship of these genes within cassava. Out of 18 chromosomes studied, 5 gene pairs had shown synteny blocks in different chromosomes. Similarly, we have also studied the synteny blocks among different genomes. For example, figure A shows the synteny relationship of cassava with Arabidopsis. Similarly, B shows the synteny relationship with Brachypodium. C shows the synteny relationship of cassava with Populus. D image shows the synteny relationship of cassava with Oryza. Whereas E shows the synteny relationship of Dysphoria, another tuber crops. Interestingly, when the genomes are closer evolutionarily, the synteny blocks content was higher. For instance, you see figure number C, the synteny relationship is much higher compared to other distance genomes. This shows that HSP70 genes are evolved through duplication and divergence in cassava. We also studied the 3D homology structure of HSP70 genes through online tools. Our analysis showed all the 22 genes has conserved nucleotide binding domain, linker domain, substrate binding domain in all the 22 genes. We also studied the promoter cis-regulatory elements in all the 22 genes. Interestingly, there is a diverse functional diversity of existence in the promoter cis elements, which includes biotic, abiotic, light responsive, cell cycle responsive elements. This was the functional diversity of HSP70 genes in cassava. We also studied the expression pattern of HSP70 genes in different tissues using RNA-seq datasets. For this, we have downloaded the RNA-seq datasets from 11 tissues, which includes friable embryogenic callus, or organogenic embryogenic structure, root apical meristem, shoot apical meristem, leaf tissue, midbrain tissue, petiole tissue, stem, lateral bud, fibrous, as well as storage roots. Our analysis showed there is the existence of constitutive as well as tissue-specific expression pattern of these genes in cassava. For instance, HSP76 as well as HSP72 is expressed in most of the tissues, whereas few genes like BAP1, BAP2 were expressed only in RAM tissue. Likewise, there is a tissue-specific expression pattern of these genes. Similarly, we have also studied the expression pattern of these genes under different biotic as well as abiotic stresses. Our analysis showed 4 genes were upregulated in both drought and cold conditions. Whereas only 5 genes had upregulated in drought condition, and 1 gene had upregulated both in cold and virus infection. This shows there is a functional role of these genes in biotic and abiotic stresses in cassava. We also studied the relationship of microRNAs and HSP70 genes. Our analysis showed around 19 HSP genes remains as a target for 24 microRNAs. This was the functional complexity of these genes in cassava. Today, in morning session, our speakers has been showed the importance of this microRNA diversity in plants. Interestingly, these HSP70 genes were targeted by many microRNAs, which includes microRNA 396, which is mainly known for biotic and abiotic stress responses, and vegetative and reproductive phase transition microRNAs like microRNA 159 and microRNA 172 also remains active on HSP70 genes of cassava. Coming to the summary, our analysis led to identification of 22 HSP70 family genes in cassava. The genes are distributed out of 11 chromosome, in 11 chromosome out of 18 chromosome. Promoter analysis showed there is a existence of tissue-specific biotic, abiotic, and cell cycle responsive regulatory elements. Genomic analysis led to identification of 18, 25, and 28 HSP70 genes from castor, propolis, and salix. Expression analysis also revealed the presence of constitutive as well as tissue-specific induction of HSP70 genes in cassava. This analysis gives an idea about better understanding of complexity of HSP70 genes in cassava, and it acts as a basis for further functional characterization of these genes in cassava. We are planning to selectively validate some of the HSP70 genes to functional values in cassava. I take this opportunity to thank my director and HOD for providing me all the facilities for doing this work. I thank the conference organizer for giving me opportunity to present. Thank you.
Thank you, Senthil Kumar, for completing within the allotted time. And it was really very interesting to know that HSP families and their regulatory elements in cassava. Really, even though it is a drought tolerant crop, we need to understand the basic of heat tolerance. So it is a wonderful work. Thank you very much. And we have time for maybe one or two questions if anybody is having, please. Yeah, I don't think there are any questions. Thank you, Dr. Senthil Kumar, for your wonderful lecture. Now I call upon Dr. Abhijit Arun Deshput. He is Assistant Professor, College of Agricultural Biotechnology, Mahatma Phule University, Mahatma Phule Krishi Vishwavidyalaya, Rahuri. And he is going to give a talk on molecular characterization of aluminum responsive gene expression in pigeon pea. Dr. Abhijit.
Yes, sir.
Okay, please go ahead with your PowerPoint.
Yes, sir. Document. Video. Good evening, all of you. Am I audible?
Yeah, audible. Go ahead.
Yeah, thank you, sir. So my research topic, I am presenting on the molecular characterization of aluminum responsive gene expression in pigeon pea. So coming to the introduction, pigeon pea is sixth most important grain legume of the world, and it has high nutritive value and high protein source for like 20 to 30% protein for more than a billion people. Approximately 50% world arable land is negatively impacted by the aluminum due to the acidic soil. Near about 24 million hectare soil have pH below the 5.5. In the if pH gets below the 5.5, aluminum inhibits the root growth, and it's a major limiting factor for the plant productivity in acid soil. So the use of liming to detoxify aluminum in subsurface soil is common practice, but it is not suitable for subsoil acidity, and it's not always economically feasible. So some plants have already developed some mechanism to minimize the harmful effect of the aluminum toxicity, such as this is the one of the mechanism, that is secretion of organic acid from the root, and the various plant species that secretes various organic acids such as citrate, malate, and oxalate. It's depend on the plant species. The secretion of organic acid is different. So there are several transporter gene responsive for this organic acid secretion, such as the MET transporter protein, that is multi-drugs and toxic compound exclusion protein, that is responsive for citrate secretion. Then the ALMT1 aluminum activated malate transporter, that is responsive for the malate secretion, and the another one transporter gene, that is aluminum, its function is still unknown. This transmembrane protein is controlled by the transcription factor, that is sensitive to stop on rhizotoxicity stop on protein. So first we started our experiment on pigeon pea with the root growth inhibition. So initially we grown pigeon pea plants in 3 days in hydroponic assay, and pre-grown plant we subjected for the aluminum stress for 24 hours, 48 hours, and 72 hours with various concentration of aluminum, such as 20, 50, and 100 micromolar aluminum compared to the also the control. In the 50 micromolar concentration during the 48 hours and 72 hours, we have found that there is almost 50% in the root growth reduction. So this study shows that the aluminum has a positive impact, and it reduces the root growth of pigeon pea. So our next aim is to identify the which kind of organic acid that secretes from the pigeon pea roots, because of the there are various studies, like for example Arabidopsis can secrete the malate. That malate is major organic acid that secretion in during the exposure of aluminum. So we are interested to find out which kind of organic acid secretes from the pigeon pea during aluminum stress. We use the enzymatic method, that NAD and NADH coupled assay, to identify the organic acid secretion. Same here, we use the hydroponic assay, and we use different time course as well as the dose dependent experiment, and we observed that the citrate, that is major organic acid secretion, secreted from the pigeon pea roots during the aluminum exposure. So same, it's a time course dependent assay, also shows the citrate is released from the root when time is increasing, and also during the dose dependent experiment assay shows that increasing the concentration also increase the secretion of organic acid. So now our next aim is to find out the which kind of the transporter or transmembrane protein that responsive for the citrate secretion, that is MET gene. So next we isolated and characterized the MET1 gene. So we use degenerate PCR followed by the 3' and 5' rays to isolate the full length of CC MET gene from pigeon pea, and the cluster W assay and the phylogenetic analysis shows that the isolated the MET gene from pigeon pea, that is CC MET1 gene, that is very similar to the previously identified MET gene from the various plant species. Then we checked the expression analysis by using real-time PCR, and we observed that the increasing the concentration of the aluminum, like 10, 50, and 100 micromolar aluminum, enhance the expression of the MET gene compared to the control. Also in the time course experiment, the increasing the time points also enhance the expression of the MET gene. So next we characterize the MET gene, whether isolated MET gene is really can overcome the toxic effect of aluminum in the plants. So here we use the tobacco, transgenic tobacco lines already that develop in our laboratory, that is stop on knockdown suppressed by RNAi lines, which is suppress the root growth. So we use same material to characterize the isolated MET gene from the pigeon pea, and we use hairy root approach. We develop first construct with MET gene fusion by the GFP. Because of this will be easy to selection, because the both the construct that already have the same kanamycin resistant gene. So based on the antibiotic, it cannot be possible to select the gene. So we use this approach, and also we want to localize the gene. So that's why we use the fusion construct for transformation. We transform this construct into agrobacterium rhizogenesis by using electroporation and develop the hairy roots. So after development of hairy roots, we studied under the confocal laser microscope, and found that this CC MET1 is localized only in the plasma membrane, compared to the control GFP that is localized everywhere in the cell compartments, like nucleus as well as the plasma membrane. So from this study, we conclude that this CC MET1 gene it's a plasma membrane localized transporter gene. Further, we study it can overcome the effect of aluminum in the transgenic transgenic tobacco. So we observed that it can also enhance the root growth compared to the control in the transgenic tobacco lines. Next, we identified the secretion of the organic acid. The tobacco, when we introduce this CC1 gene into the tobacco knockdown with the stop one, so when we expose these hairy roots, we observe that this can enhance the citrate secretion compared to the control transgenic plants. Also, we found that this can only enhance the cytosecretion, but not only the mallet. So this can from this study we concluded that so isolated stop one has potential role in the aluminum tolerance in pigeon pea. Next, our aim is to isolate the identify the transcription factor that regulates the mate gene. So next, we isolated the stop one gene that regulates, that is transcription factor that regulates the downstream gene. So here also we use degenerate PCR assay as, followed by the 3' and 5' race. We isolated full length gene. We cluster analyze the isolated sequences. This shows that there are 4 conserved zinc finger domain, C2H2 type 4 zinc finger domain. Phylogenetic analysis shows that the isolated CC stop one gene is also highly similar to the previously isolated stop one gene. Then we use transient assay to identify the localization of the stop one. So we developed the construct fusion with the GFP, synthetic GFP fused with the CC stop one, and transform into Agrobacterium tumefacius, and done the transient assay. And we observed this the tobacco leaf under the fluorescent microscope, and we found that this isolated stop one that is localized only in the nucleus compared to the control GFP that is localized everywhere in the cell compared to the nucleus as well as the all compartments of cell. From this study, we concluded that this stop one, it's a nuclear localizing transcription factor. Next, our aim is to characterize the stop one, whether this isolated stop one has potential role in the pigeon pea. So we use here we use loss of function analysis of CC stop one by using RNAi in pigeon pea hairy roots. So suppression of the pigeon pea stop one by using RNAi suppression in the pigeon pea hairy roots, we found that by using the real-time expression analysis, that the stop one suppression lines also suppress their downstream gene, that is CC mate1, as well as the ALS3 gene expression. So suppression of this stop one suppression, this suppress the CC mate as well as ALS3 gene, also can accumulate more amount of the aluminum in the root. That is we observed by using the aluminum responsive probe, that is Morin, and we observed this root under the fluorescent microscope. Then we found that the stop one suppression hairy roots can be give more fluorescent compared to the control plants. So next, we also identified the stop on binding sites on the CC met promoter. So we identified
Dr. Abhijit,
approximately 1.5 kb upstream sequence from the starting point, starting sequence of the mate gene.
Dr.
And
Abhijit,
by using in vitro binding assay, we confirm that the CC stop one has the sequence, the binding sequence on the CCMate1 promoter.
Dr. Abhijit.
Conclusion is this in this study. So we successfully isolated CC stop one gene from the pigeon pea, and this can regulate the downstream gene, that CC ALS3 as well as the CCMate1 gene that is responsive for cytosecretion. So this study can be helpful for the further pigeon pea breeding that can enhance the aluminum tolerance in pigeon pea. So this is the publication of my study, and here I would like to acknowledge my professor, PhD professor, that Professor Hiroyuki Koyama, also my master professor Dr. Fakhruddin sir, and the co-workers for their kind supports during the work. Thank you, sir.
Thank you, Dr. Abhijit, for completing maybe 1 minute extra you have taken. Otherwise, it is wonderful. You have maintained the time. And aluminum toxicity is really very important. In terms of that, you studied on the molecular characterization of aluminum responsive gene expression in PGNP. Thank you very much. If there are any questions, one question, please.
No, you can
Yeah,
go ahead.
if there are no questions, then I have the pleasure of welcoming Dr. Robert. He has just joined our session, but anyway, he will give his keynote address after another two speakers. We have two more speakers, but I would like to mention that he is distinguished professor from University of Missouri, USA.
It's a pleasure to be here, but you want me to wait until after another two talks, right? That's fine.
Thank you, Professor.
Hello. Dr. Anand.
Yes, sir. Hello.
Yeah, it's audible.
Yeah, yes, sir. Audible, sir.
Yeah, please go ahead.
Thank you, sir. I will share my slides. Sir, my slides are visible, sir?
Yeah.
So good evening to everybody. So respected chairman, co-chairman and participants. So I would like to thank the organizers for giving the opportunity to present my research work on allele mining strategy to identify the low phosphorus tolerance intervention lines with superior alleles in rice. So we all know that phosphorus is an essential nutrient for most of the agricultural crops, and it is very important for the major developmental processes and reproduction in plants. So as we know, almost 20 to 30% of the applied P will only be taken up by the plants. So rest of the thing either it will be fixed in the soil or sometimes it will be it will not be utilized and it will be in soil itself. So phosphorus deficiency mainly causes the massive reduction in growth. So as here we can see in rice, this is IR26. So this is phosphorus was not applied here, and recommended dose of phosphorus was applied here. So drastic reduction in yield we can see here, and also sometimes curly leaves will be formed, and number of tillers will drastically reduce. Sometimes it will not flower at all. So whatever the other nitrogen and potassium fertilizers and all favorable growth conditions we provide, so this will not come to flower. So this is the major effect of phosphorus in rice. So if you see the global scenario for soil phosphorus status, so around 30% of the crop area in the world, so it is a deficit in phosphorus. So this tells the need for going for the work on phosphorus reduction in agriculture without reducing the yield. If we see the scenario in India, So whatever the red patches we are seeing, these are all deficit in phosphorus. So yellow color is moderately deficit. So so this will also tells that 90% of the districts in India, they are medium to low in phosphorus fertility. So with this brief introduction, what I'm going to speak about the variability present in phosphorus, and also how this variability or genome information can be utilized for rice improvement. So here you can see the picture. So this is the phenotypic facility available with our institute, Indian Institute of Rice Research, where rice genotypes can be grown at very low phosphorus condition. So nearly since 20 years, we have not applied phosphorus in this facility, and very much clear-cut genotypic difference we can observe. So with this screening the genotypes in this facility. So Dr. earlier Krishnamurthy was there, and also earlier workers like Sundaram sir and Badana sir, they created this one and they well maintained, and that we have been using. So here, just to show the phenotypic difference in the same P0 plot, we will tell, or low phosphorus plot, we will tell. So this is the real populations. So with same level of nitrogen and potash and phosphorus we have not applied, but residual phosphorus is already there in this P0 plot also. So this is the vast variation we can see in this phenotypic screening. So, and earlier at IRRI, International Rice Research Institute, they identified one QTL called PUP1, phosphorus uptake 1, for giving the tolerance to low phosphorus soil conditions. So it was working out very well. And when they screened, many of the Indian germplasm or varieties were also, mega varieties were also having this PUP1. So with this one, so Dr. Sundaram led the work, and they they transferred this PUP1 from this Varna. It's a mega variety in India, and they transferred to M21010, IR64, and even improved Samba Musuri also. So this earlier Dr. Madhusudan has presented this information. So wherein this PUP1 loci is present in chromosome number 12. So in this one, what I'm presenting is so variation we saw in the sequence when we amplify this K46-2 when we amplify this region with this marker K46-2. So here material was taken was 192. Earlier we started with 192 introgression lines. So these were derived from crossing Basmati DPT 5204 with 10 Rupi Pogan varieties. So we know that Varaiza Rupi Pogan is a wild species in this one, and 10, these are the 10 variety accessions we have taken, and it was crossing was made, and 192 BC1 F3 population was obtained. So this is the initial work. Pre-breeding was work was done by Dr. Girish. So, and from here BC1 F3 onwards, we have taken those 197 lines for screening under low phosphorus condition, and F3 F4, BC1, F4, we evaluated under low phosphorus condition, and selected lines we again evaluated in this same low condition in F4 and F5 and F6. This one. So here we can see the phenotypic variation for this selected line BC3 here, F3, BC1, F3, F4. When they grown in control and low phosphorus condition, lot of variation was observed when we grow these things in control and low P. So these are all the other root rates. So these are all the markers we have used for knowing these 38 lines, selected lines, along with the checks we have used. So this is the 46-2 marker amplification. So this is the result of when this 5 markers associated with PUP1 loci screened for different introduction lines, along with this one, wherever this positive is there, where they are given the amplification. So this is the phenotypic difference we can see here. So these are all the susceptible one under low phosphorus in this study, introduction lines, and tolerant lines. You can see very good growth. Everything is very good. So this is the 45 days when we take out the plants at 45 days. This is the variation we could find. So this is the other one. This is Swarna, so which is having POP1, and these are the interrogation lines. So when we saw the marker trait association, so this root shoot dry weight and root dry weight, they are associated with these primers. So for this 46-1, this is the R square value 15%, and for root dry weight, this is 15.14. These are having very good phenotypic variance association with these root parameters. So initially, what actually POP1 gives is it increases the root biomass, and indirectly it will help to take up more phosphorus so from the soil. So when we amplify this K46-2, this region with this primer, we got this the sequence we got. So we sequenced that amplicon, and these are the indels we got and SNPs we identified. So here we can see these are all the three indels we could see with this interrogation lines, and in comparison with the Kasalat. So this is the one in the interrogation line 52-55, wherein it has got this ATCG, wherein none of the other either checks or none of the other interrogation lines, they have these regions. And if you see SNP variation, so these are all the 6 regions where we could see the variation for this nucleotides SNPs in this region. So this is the when the cluster analysis was done using the SNP variation. So this Kasalat and another one, they got separated as the other clusters, and the rest they were grouped into 3 clusters: clad A, B, C, and C, Rasi and 83-3, 6 were grouped into this one. So once this has been done, this haplotype analysis of this sequence was done. So totally 4 haplotypes were observed. So in this small sequence, because if you see Kasalat POP1 QTL, it's a long QTL. So in this one only small portion which that 46-2 amplified. So with this one we could identify 4 haplotypes. Haplotype 1, which is Kasalat type, so which is not having any resemblance with any of the complete resemblance. So partial resemblance is with, available with the interrogation lines, but totally it is different, and it is a group. That's why it is only one genotype is grouped in this haplotype. So second haplotype, so total 6 genotypes were there, and the third haplotype 10 genotypes were there, and the fourth one only 1 is there. So these are the variations we could see in this one, and further haplopheno analysis is further pending. So what I want to conclude from this small work study is, so this genotypic variation is not present in this rice. So for low phosphorus tolerance, so in coming days we have to get more yield with less inputs. So especially this major NPK, we have to reduce the use of those things, and at the same time we have to maintain the yield. So all whatever the genotypic information is available with us is really good, and it's helpful in maintaining the sustainable yield in rice. So at the end, this is the work of PhD student Vannappa is there. So Dr. Diwan from US Raichur also helped a lot in conducting these experiments and all. So at the end, I would like to thank our director, ICR, IIRR, and also co-workers, Dr. R.M. Sundaram sir, Dr. Girish, and everyone who helped to conduct this experiment. Thank you, sir. Thank you very much, sir.
Thank you, Dr. Anand, for a wonderful presentation on phosphorus tolerant interrogation lines and allele mining strategy we have used to identify these lines. And it has led to identification of some of the lines with POP1 interrogation. So I hope you will take it forward and come out with some varieties with phosphorus tolerant character.
Thank you, sir.
I have, I think, 1 minute for some questions if anybody is having. I think there are no questions. Then thank you, Dr. Anand.
Thank you, sir.
Now the next speaker is Mrs. Javeria Uzma, Research Scholar, Department of Biotechnology, Telangana University. And she's going to talk on assessment of salt tolerance, deploying antioxidant defense systems, in 4 different genotypes of Gerbera, an important ornamental plant. Dr. Javeria, please. Is she available? Dr. Javeria? Organizers?
Sir, 1 minute, they are checking, sir. Just a minute. So they are not there.
Not there?
Not there.
Okay. Then we will move on to the keynote address. Yeah, I welcome Dr. Robert E. Sharp. He is a distinguished professor from University of Missouri, USA. I have the pleasure of introducing him to this seminar. And he is a recipient of many awards and recognitions. He works in Division of Plant Sciences, Interdisciplinary Plant Group, University of Missouri, USA. He is a fellow of American Society of Plant Biologists. He was interim director in Division of Plant Sciences from 2005 to 2008. And he is also recipient of Frederick Mumford Outstanding Faculty Award, College of Agriculture, Food and Natural Resources, University of Missouri. And he is a fellow of American Association of Advancement of Science. So like this, I think it takes a lot of time to introduce all the awards, whatever he has received. He is also in the editorial board of Journal of Experimental Botany, and he is in the editorial board of Plant Physiology also. So he has got wonderful credentials to his credit. And on this occasion, I welcome Dr. E. Sharp to give his lecture on physiological controls of root growth maintenance under water deficits, contrasting responses of maize lateral and nodal roots. It is very interesting to hear to you, Dr. Robert. Please.
Okay, thank you very much. Can you hear me?
Yeah, you can hear. Go ahead.
Okay. Thank you very much for the kind introduction and for the invitation to participate in your conference. And it's great to say hello to some friends for many years, Dr. Fakrudin and Dr. Shashida. I've known for a long time, and it's a shame we can't be together in person, but anyway, it's a pleasure to be part of this conference. And so what I'd like to do today is tell you about some newer, more recent studies in my lab, focusing on the responses of lateral roots and nodal roots of maize to drought. We all know that root growth and development is a critically important aspect of how plants respond to drought. This is emphasized by some work from colleague Richard Richards at CSIRO in Australia, who published this review 10 years ago, listing, in his opinion, the 10 most important plant traits for improving crop performance under drought. And this includes improved seedling establishment, root vigor, and rooting depth, all of which relate to the work that I'll talk about today. However, in most cases, the physiology of root growth under water deficits remains poorly understood. Now, this is to a large extent because roots and root systems are difficult to study experimentally, and that's partly because they're very developmentally complex. The overall response of a maize root system to drought is illustrated on the left here from a classic figure from Jim Weaver, published in 1926. And this overall response encompasses a large range of responses of different types of roots that are responding to different severities of water stress at different depths within the soil profile. There is an overall deeper rooting that you can see here, but also encompassed in this figure is enhanced lateral or branch root development, which I'll talk about in the first half of the talk. And I've also focused for many years on the ability and necessity of some types of roots to grow through dry soil in order to reach moisture in deeper soil layers. And this is true of the primary root of seedlings, where it's critical for seedling establishment. and also for the nodal or crown roots in maize and other grasses. And I'll move on to that topic in the second half of the presentation. So just so we're all familiar with the types of roots we're talking about here, in grasses, there is the primary and seminal root system that grows from the seed and is critical for seedling establishment. But then as the plant matures, there are the stem-borne nodal or crown roots that grow from the base of the stem, and these form the framework of the mature root system and are critical for water uptake as the plant matures. And then both from the primary root and from the nodal roots, there's the development of the lateral or branch roots that are in fact responsible for the majority of the water uptake from the soil. So I've worked for many years on primary root growth maintenance, and I'm just going to briefly review that first before moving on to our newer work on lateral root development and nodal root development. So the reason we focused on the primary root is because it provides an experimentally simple system for us to focus on and learn about fundamental mechanisms of how roots grow under water deficit conditions. So it's a simple system that allows an extremely high level of reproducibility. Simply, after germination, we transplant seedlings to a media. We use vermiculite, which has been pre-adjusted to a range of specific water potentials. And then we grow the plants under conditions of minimal evaporation that prevents further water loss from the media and allows the seedlings to exhibit steady and reproducible responses. And as you can see from the main figure and also from the photograph, under severe water deficits, the primary root has the ability to continue growing at much lower water potentials than those that then inhibit the growth of the shoot. In fact, at this water stress level here, minus 1.6 megapascals, the primary root is essentially the only part of the plant that continues to grow. Now, that reproducibility of the system allows us to conduct detailed genetic diversity studies, and results of a paper we published in Crop Science back in 2011 are illustrated here for mild water deficits and severe water deficits with root elongation rate plotted as a percentage of the well-watered control. And from these data, we selected 2 lines for more detailed studies of mechanistic underlying mechanisms of root growth response. That is FR697 that shows a superior response, and the standard maize genotype B73 that shows a much lesser ability to maintain growth under water stress. Now, we've worked on the primary root system for many years, and our most recent comprehensive review was actually published now 10 years ago now in Plant Soil Environment. And this is a review of the physiological and molecular mechanisms as we understood them at that time. The following figures come from that review, just to briefly summarize a few of the key aspects of how primary roots continue to grow under water deficits. First, we know way back, actually, this is my first ever paper from my PhD thesis back in 1979. We know the importance of osmotic adjustment and particularly the role of pearline accumulation in that process. And that was a focus of a study back in 1991 here. I've done a lot of work on hormone interactions, particularly focused on abscisic acid or ABA, and its interaction with ethylene, and also more recently with auxin. A review of the ABA ethylene interactions was again published in Plant Soil Environment in 2002. And we've also done quite a bit of work on a modification of cell wall properties, and particularly cell wall extensibility. There was a question asked about this a few talks ago in this conference about the importance of modification of cell wall extensibility for root growth under water deficits. We did some work originally back in the mid-1990s showing that there is an increase in cell wall extensibility in the root growth zone that helps the roots grow under water deficit conditions. And we published work on both cell wall loosening proteins, including expandins and XTH or XET. And more recently, from a cell wall proteomics paper that we published in Plant Phys in 2007, this proteomics work led to some more recent studies of the accumulation of apoplastic reactive oxygen species in the cell wall that appears to have a role both in regulating cell wall extensibility and which was expected, but also more surprisingly, a role of the cell wall reactive oxygen species in regulating cell production in the meristem. And this is a recent paper that we published earlier this year, and I'm going to return to that point a little bit later in the talk. So what I'd like to do today is address some newer work, both on the responses of lateral and nodal roots, to ask whether we can study these roots with similar level of precision that we've been able to study the primary root. As I'll explain, both of these types of roots are more difficult to study than the simple system that we use for the primary root. Now, focusing first on lateral root development, we published a couple of papers in the last 2 years in plant cell environment. This is work of Tyler Dowd, a recent graduate student of mine. And this was to characterize an enhanced elongation response of lateral root development in response to water deficits. This has actually been known for decades that one can see enhanced lateral root development, but the mechanisms underlying this response have not been studied in detail before. And as you can see in this picture from FR697, you see this enhanced lateral root elongation. And this is a specific response to mild water stress. And as such, the experiments require very precise control of soil water deficit conditions. So to do this, we developed a system that allows us to study for a more extended period light-grown transpiring plants to look at the effect of mild water deficits on lateral root development over a period of 9 days. To do this, we transplant to Promix H, or grow the plants in Promix HP, which is a peat-based potting mix that holds a lot of water. And we, similar to the seedling system, pre-adjust the media to a range of specific soil water potentials, and then grow the seedlings in a high humidity environment to reduce transpirational water loss. And this allows us to achieve almost steady state water potentials over the course of 9 days, allowing assessment of the lateral root growth response to a range of specific and highly reproducible mild water stress conditions. And the results are shown here. For the 2 genotypes I've already mentioned, FR697 that was selected for having a superior primary root response. And as you can see, it also has a superior ability to enhance lateral root elongation under mild water deficits. About a 27% increase in total lateral root length from the primary root system. Interestingly, specifically from the primary root system and not from the seminal roots that show no response. In complete contrast, in B73, you can see that there's absolutely no enhancement of lateral elongation from the primary system and an inhibition from the seminal root system. If we take a closer look at this and focus in on a specific class of lateral roots, here we're focusing on the 10 longest lateral roots from FR697 and B73 under either well-watered or mild water stress conditions. And again, when we focus on those specific root class, you can see again about a 35% increase in the average length of the individual lateral roots in FR697, and again, absolutely no response in B73. So there's quite striking genetic diversity between these 2 lines in the ability of the lateral roots to show enhanced elongation in response to stress. Now, an increase in root length, I should go back a slide, this is taken at the end of these 9-day experiments. So this response of increased length could have various causes. It could involve an actual increase in root elongation rate. It could involve earlier emergence of the lateral root initials from the root axis. But what it turns out to be caused by is actually an extended duration of growth or delayed determinacy. As you will be aware, in some plant organs, including lateral roots, the meristems are genetically encoded to stop production of new cells at a particular age. And this results in a determinate growth program, a specific limited duration of growth. And the explanation for these longer lateral roots in FR697 is because the growth duration is extended. And this is illustrated in this slide. So this is root length over time, over 11 days of growth under either well-watered conditions, shown by the black line, or mild water stress conditions, shown in red. And you can see that in both FR697 on the left and B73 on the right, the lateral roots have about an 11 or 12-day growth duration. It's a bell-shaped growth profile where they increase to a maximum and then decelerate and stop. And you can see in B73 on the right, there's absolutely no impact of water stress on that growth profile or duration. But in contrast, in FR697, whereas the well-watered roots begin to decelerate after about 5 days, the water-stressed roots continue to grow at a maximum rate for a period of several days longer, and by the end of the experiment at 11 days are still growing at a substantial rate and obviously are nowhere close to cessation of growth yet. And so, so this is an interaction of water stress with the determinate growth program, overcoming that normal deceleration of growth that occurs after 5 days and allowing the roots to continue to grow for a more extended period. Now, as we're all aware, organ growth, root growth in this case, is a product of two basic factors. One is the production of cells in the meristem, and secondly, the elongation of those cells as they're displaced through the growth zone. And to assess whether one or both of these processes are involved in this continued root elongation over time, we conducted a kinematic analysis or spatial analysis of cell expansion patterns based on cell length profiles using techniques developed by Wendy Silk at the University of California, Davis back in 1989. These first few slides show the effect of water stress on the cell flux over the course of the 11-day experiment. This first slide shows the well-watered controls of FR697. Now, the cell flux is calculated, as shown here, by dividing the total root elongation rate by the final cell length, and it gives you a calculation of the number of cells that are leaving the elongation zone per unit time. And under steady conditions, this approximates the number of cells leaving the meristem, so the overall cell production rate from the meristem. And you can see in the well-watered FR697 roots, after a maximum at 6 days, there's a dramatic deceleration of meristematic activity, so the cell flux declines almost to zero by 11 days. If we compare this with the water-stressed plants, you can see that cell flux or meristematic activity is maintained at a substantially higher rate throughout the experiment, and so that by 11 days, the cell flux is about 4 times the level that occurs in the well-watered controls. So there's a substantial interaction of water deficits with maintenance of meristematic activity. In complete contrast, in B73, you see virtually identical data between the well-watered and the water-stressed plants, showing no interaction of water stress with meristem function. If we look at cell expansion, again, we can study this by looking at the cell length profile through the root elongation zone. Now, remember, we're talking about lateral roots in maize, so they have a very short elongation zone of about 4,000 micrometers or 4 millimeters. So there's a rapid increase in cell expansion over the first 2 millimeters, and then a slowing down and cessation of growth at about 4 millimeters. Now, this is for the well-watered FR697, and you can see, to begin with, there's no change. It's a constant profile over the first 7 days. But as the roots approach determinacy, you see the beginning of slowing down or earlier cessation of cell expansion and reduced final cell length. And this is further reduced by 11 days with about a 25% decrease in final cell length. And this is absolutely completely prevented in the water-stressed plants, where you see the anatomical profile is identical throughout the experiment. So water stress is delaying root determinacy, both by prolonging activity of cell production in the meristem and prolonging the normal complete cell expansion, reaching a same final cell length throughout the experiment. Again, in B73, the effect is very different, that we see the same reduction in final cell length at the end of the growth zone over the course of the experiment. And this is completely unaffected by water stress in B73, so that the final cell length is reduced just the same as in the well-water control. So in summary of this initial lateral root studies, in FR697, but not in B73, water stress interacts with the normal determinant growth program to delay determinacy, and this allows for maintenance of rates of lateral root elongation, ultimately giving longer roots. And to our knowledge, this is the first report that water stress can act to promote root elongation via delaying root determinacy. And as I've shown you, this involves maintenance of both cell production and cell elongation processes. Now, we haven't started studying those processes in detail in the lateral root system yet, but I would draw your attention to the paper that I mentioned earlier on, as is in the primary root system. But this is recent work of Priya Vupaluru, who was a previous graduate student of mine, published earlier this year in Frontiers in Plant Science. And it was a study of the role of this increase in apoplastic reactive oxygen in the primary growth zone. And the results showed, as I've already mentioned, that increasing apoplastic reactive oxygen has a fundamental role in regulating not only root elongation, but specifically cell production rate in the meristem under both well-watered and water stress conditions. Now, that was in the primary root, but it will be interesting to see whether this water stress response that occurs in the growth zone of the primary root, whether this has a role in regulating determinacy in the lateral root system. Now, I'd like to for the rest of the talk, switch gears and focus on our work on nodal root growth. And in contrast to the mild water deficit response of lateral roots, the important aspect of how nodal root growth has to respond to drought conditions is the necessity for these roots to have to grow through very dry soil. As I've already mentioned, nodal roots are produced from the base of the stem. And so, as illustrated in this photograph, under drought conditions, when the surface soil can become very, very dry and also hot and hard, these nodal roots produced from the base of the stem grow directly into perhaps the driest part of the profile. Now, I've mentioned they form the framework of the mature root system, so it's essential that they manage to grow through this harsh upper soil environment in order to sustain water and nutrient uptake as the plant matures. And also, of course, these roots are important for plant support, and the later generations are often referred to as brace or prop roots. But as I've already mentioned, under drought conditions, they need to grow through this dry soil that they emerge into in order to find moisture at depth. And therefore, it's very critical that they avoid tissue desiccation from water loss into that soil. Now, I also point out, as indicated by the blue arrows, there's a unique aspect to the direction of water transport in these roots. We generally think of roots as taking up water from the soil, and the water is then moved to the shoot via transpiration. In this case, water is being taken up from roots that are already at greater depth from the primary and seminal seedling root system, and that water is moving up the shoot to support transpiration, but is also moving down these developing nodal roots to the apex. And again, as I've said, it's essential to avoid tissue desiccation from water loss surrounding dry soil. If the nodal roots are unable to continue growth, the plants develop what is termed rootless corn syndrome, and this can occur when the surface soil is excessively dry, hot or hard under drought conditions, causing complete failure of the nodal roots to develop. In this situation, the plant loses the ability to take up the majority of its water and nutrient uptake as the plants mature, and also makes the plants very vulnerable to lodging or falling over if a windstorm was to come through. So it's not surprising that nodal roots have been shown to have a superior ability to maintain growth under dry soil conditions. And actually, this was as the work that I've already mentioned that I conducted in my own PhD studies back in 1979, and also a subsequent paper from John Boyer's lab that was published in 1985, from which this figure comes from. And so this is a plot of the elongation rate of different tissues in the maize plant as a function of increasing water deficits. And you can see that the nodal roots have a superior ability to maintain growth compared to the leaves, stems, and reproductive structures. Now, surprisingly, given how important these roots are, as far as I know, these remain the only papers on physiological mechanisms underlying the ability of nodal roots to continue growth. And both of these are studies of osmotic adjustment. So we have, in my lab, for the last couple of years, been studying nodal root responses in much greater detail. Now, to do so, we had to develop a new model system because simply withholding water has difficulties which are illustrated here. Because the nodal roots are produced in different whorls a few days apart over the course of shoot development, you can see if you simply did a dry down experiment, the different nodes of nodal roots would be experiencing different severities of water stress, both spatially and as the soil dries over time. And that makes reproducibility a challenge, and also comparison across different genotypes very difficult. So Cara Casey, a recent graduate student of mine, developed this divided root system approach where we grow the seedling root system, the primary and seminal roots, in a container, which we can keep watered, and that water is taken up by the seedling system, supplies the water for transpiration and shoot development, and also supplies the water for maintaining nodal root growth into an outer container that we can fill with predetermined specific water potential environments. As such, the outer container is analogous to the seedling system I mentioned earlier on. The water, it's a covered outer container, so the water is not lost by evaporation during the course of the experiment. And so the nodal roots grow into defined, uniform, and reproducible water deficit conditions and obtain their water from the deeper seminal and primary roots, similarly to the situation in the field. This is the finished product. The system is about 1 meter deep. This is the inner container and the outer container. And on the right hand is an illustration of young plants that have been grown in such a system where the primary and seminal root system is separated from the developing nodal roots. Now, our initial studies of this system reveal an unexpected complexity in the response of nodal root growth. So these data are at the end of experiments for node 1 after 7 days, and for node 2, 3 days later, such that the total length of the well-watered roots is about 25 centimeters at the time of harvest. Now, of course, our interest in nodal root growth under water deficits is in the maintenance of root growth, and that certainly is true. Here we have roots, nodal roots that have been growing into that outer container at a soil water potential of -2 megapascals, very dry soil, and here in node 1, in FR697 particularly, root growth is about 50% of the well water control. And in node 2, you can see an even greater ability. In fact, statistically, the nodal root length after 10 days of growth at -2 megapascals is actually statistically the same as the well water control in FR697. So yeah, nodal roots do maintain growth through very dry soil, as has been published many years ago. But when we look at the response to a range of steady state water potentials, we see there's surprising complexity, such that nodal root growth is actually quite sensitive to the mild water stress range in both genotypes, and particularly in node 2 in B73. But then there is a change in the response, so that as the soil water potential decreases all the way from about 0.6 megapascals down to -2 megapascals, we see no further inhibition of growth in either node 1 or in node 2. Now, the explanation for this profile of response over the range of soil water potential is explained by the behavior of the root tip water potential itself. This is data plotting root length against the soil water potential that the roots are growing through. The next slide shows the same experiment, the same data in the upper part of the figure, but with the lower part of the figure showing the root length against the root tip water potential, or the root growth zone water potential. And you can see a very interesting data now that explains this biphasic growth response. Over this initial mild range of water stress, there is actually a substantial reduction in root tip water potential. But as the soil dries from 0.6 down to -2 megapascals here, you can see that there's no further reduction in root tip water potential in FR697, whereas the more sensitive genotype B73 shows a significantly greater reduction in root tip water potential as the soil gets drier. And these same trends are true in node 2, again, with no further decline in root tip water potential in FR697 as the soil dries from about -1 to -2 megapascals. There's no further change in root tip water potential. Now, these same data are plotted again on the next slide, where we simply are comparing the response of root tip water potential compared to the soil water potential that the roots are growing through. And this further reveals the very interesting behavior that's occurring. If the root was simply reflecting, if the water status of the root was simply reflecting the water status of the soil that it's growing through, we would have a one-to-one relationship, which is illustrated by this dashed black line. And you can see that this reasonably represents the behavior of B73 roots in both node 1 and node 2. Now, you can already see that FR697 has a different slope. In other words, as the soil dries, the root water potential is decreasing less in FR697 than it does in B73. And as we extend the experiment out to much drier conditions, you can see in FR697 that there's no further decline in root tip water potential in both node 1 and node 2. So this indicates some form of induction of mechanisms that prevents the root tissue, the root tip tissue, from further dehydration, even though the soil water potential is decreasing all the way down to -2 megapascals. So if you like this, it suggests some form of waterproofing of the roots as they're growing through very dry soil. An induction of mechanisms that facilitates water transport from the base of the stem to the root growth zone, and or a retention of the water in the root growth zone and preventing water loss to the surrounding dry soil. And whatever these processes are obviously quite genetically variable and particularly occurring in FR697 compared to B73. So these observations took me right back to my PhD studies. As I said, I was studying osmotic adjustment in maize nodal roots. And this model comes from my PhD thesis based on data in that original paper of osmotic adjustment in 1979. And we were speculating, I was speculating, that nodal roots that are growing through soil that's drier than the roots themselves are vulnerable to the development of steep water potential gradients, because again, the water has to flow all the way from the base of the stem down these elongated, the axes of these elongating roots. And there is always the potential for water loss to the surrounding dry soil. So by the time you get to the root tips, that they may be vulnerable to dehydration and desiccation. So it's important to have mechanisms involved that allow the root to, again, be somewhat waterproofed as it grows through this very dry soil environment. And we're focusing on this at the moment by transcriptomic and anatomical studies, focusing on potential waterproofing mechanisms, for example, superization of the root axis to prevent water loss to the soil, and also water transport processes within the root growth zone itself. For example, aquaporin function and water delivery from the phloem, because the phloem, of course, develops all the way down to the very apex of the root to deliver sugars and other growth-supporting materials. And those sugars are dissolved in water. And so flow and delivery of sugars also delivers water right down to the root apex. And there is some evidence that water stress can modify phloem development in the root tip to enhance water delivery. So we're continuing these studies as part of a major grant from the National Science Foundation in the US that we have at the University of Missouri that continues on for another year or so. We're using a multi-omic approach to gain a comprehensive understanding of the mechanisms of response to water deficits that are occurring in the root growth zone. Focused on transcriptomics, both in using the model system in the lab, but also extending out to the field, using mobile rainout shelters to control drought imposition, and then also focused on plasma membrane proteomics and metabolomics, and then integrating all of this data bioinformatically to develop candidate genes for testing further. Now, this is a non-high throughput system, and so we've also been developing a higher throughput dry down system in growth chambers that allows us to study progressive nodal root development over a period of about 5 weeks. I don't have time to show you data today, but our initial studies of genetic diversity of nodal root development under drought indicate that there is a large variation in the ability of nodal root systems to develop under drought, such that after 5 weeks of soil drying, the range of response as the nodal system as a percentage of the total root system in terms of dry weight can range between 80% to 30%. So this is a huge range of response of nodal root development to soil drying. And so we'll be continuing to study the mechanisms involved in that in more detail. So to summarize what I've tried to convey to you today, just focus on the contrasting responses of primary, lateral, and nodal roots to water deficits. In all cases, root growth is either maintained or even enhanced in the case of lateral roots, but the mechanisms that are involved appear to be fundamentally different. We've done a lot of work, as I said, on the primary root, and we know quite a bit about the physiological and molecular mechanisms involved in primary root maintenance. But what I've focused on today are the novel findings of how lateral roots and nodal roots maintain growth under water stress. In the case of lateral roots, by delaying determinate development, and in the case of nodal roots, emphasizing the critical importance of maintaining the water status of the root growth zone as these roots grow through very dry soil. I'd like to thank the people who've been involved in this work, particularly the 2 grad students I mentioned already, and the team working on the NSF Plant Genome Grant. And I'd just like to take one final minute to bring to your attention the 11th meeting of the International Society of Root Research, which is going to be hosted at the University of Missouri at the end of May in 2021. Of course, this was originally going to be a in-person meeting. Now, because of the pandemic, the meeting is going to be held virtually. But we're taking advantage of this situation in order to link to a second meeting in the United Kingdom, the 9th International Symposium on Root Development, which will be hosted at the University of Nottingham. We're all organizing the joint program to allow live streaming of speakers and sessions, maximizing opportunities for participation from around the world. And there'll be a number of special issues and other shared activities. I'm the chair of the organizing committee for the ISRR 11 in Missouri, and Malcolm Bennett is the chair of the Rooting 2021 meeting in the UK. So I look forward to hosting these meetings and welcoming some of you to participate next May. So with that, thank you very much.
Thank you, Dr. Robert, for a wonderful lecture on the underground world of roots. We thank you very much for exposing us to the underground world. I also feel that the answer for many abiotic stresses are lying underground, below the ground level. Instead, we try to focus on the above ground, because which we can see. So it's very difficult to study the underground world, but you have made a wonderful effort in analyzing the primary roots, lateral roots, as well as the nodal roots. So congratulations for that, and it's really a wonderful lecture. I think you can take a couple of questions.
Sure.
Okay. If anybody is having any questions, please ask. Yeah, if there are no questions, Dr. Robert, I have one or two questions. Yeah, this nodal roots, they grow into the dry soil, but does it have any adaptive mechanism so that it provides, mechanism for the plants to withstand the stress? Does it provide water through the nodal roots?
Well, sure. As I emphasized, the nodal roots in grasses like maize or wheat or sorghum are the framework of the mature root system. So the primary and seminal root system, the support seedling establishment, has a sort of a finite depth, maybe 1 or 2 feet. But as the plant matures, it's the nodal roots are the roots that grow 2 meters down into the soil in the case of maize. And so particularly in drought conditions, a typical drought where the surface soil dries more, but there may be moisture at depth. Obtaining that moisture at depth is critically important to sustain plant development under drought conditions. And so it's essential that the nodal roots manage to grow through the dry surface part of the soil profile in order to get down at depth to maintain water uptake as the plant develops. So they play a particularly important role in water uptake, especially under drought conditions. But in order to do so, they also, because of the way they're produced from the base of the stem, they're in a very vulnerable environment. And as I showed in that picture, there's corn syndrome. If the surface soil is dry enough, or not necessarily just dry, but hot and hard, failure of the nodal root system to develop can lead to complete failure of the crop. So it's an interesting situation where they're both vulnerable to surface soil, but necessary to through surface soil in order to find water at depth.
Oh, thank you. Now it is over to the host to take over. I also thank our co-chairman, Dr. Nanjay Reddy, and also rapporteurs, Dr. Mallikarjun Avati and Dr. Babu. I hope that they have noted down some of the important findings of these sessions. So I request our co-chair, Dr. Y.N. Nanjay Reddy, to give his remarks.
Thank you, Dr. Srishankar. So I have noted a few points, good points, which may be needed from Sheshasai and some others who spoke, that is of the oral presentation. Why there is a stagnation in the yield is a major question which I am interested. Dr. Sheshasai and another person, probably Madhusudan, have spoke about this. I have an answer or clarification for me. What I need is, anything will have a certain stage up to which it can grow, further it is not. Okay, we have stagnated yields of many cereal crops now. Probably if you give any food or something at the early stages, there is a chances of the little bit more growth. Because we have a stature, we have stagnated stature, we cannot, beyond that we cannot grow. So probabilities of at the childhood, at the small plant stature level, if you can provide something, there are chances, one. The second question is the irrigation it has come. Aerobic possibilities of the aerobic rice, where aerobic rice or the decrease in the water or the irrigation will reduce the yield of the rice. But I have seen in the maize as well as in the rice, reduction in the irrigation up to 50% or 75% can survive well better with the aerobic paddy up to flowering. Flowering onwards, if you can maintain the 90% of the irrigation, then it will do better, no problem for the even for the quality. Now we have at the GKVK one experiment is going on where 50% irrigation to maize is excellent. It is showing very well, 50 to 75% of the field capacity. The third, which I could see, is the spiral phyllotaxy given by the Rahul really good. That's in the one graph. For us, it will be useful to present to the students sometimes. And then the other speaker, that is MSS Sheshasai and Anjani Kumar, also said about the rise, about the field capacity. 75% field capacity could be the best for most of the cereals. Then Papuan gene is good in two topics which they gave. Excellent, the topic delivered by the Robert, which is of the physiology in nature. Majority of us are physiologists. We are really very happy. And we have to turn now our face towards the root morphologies and the root structure and root related to the drought functions. I'm working on Ragi as millet, so which I am interested now. Really, it's a very enthusiastic talk and for the specialist, especially for the physiology guys. So I thank you and thank the organizers, Fakhruddin and Dr. Halesh, both have given me the opportunity to be coach at the session. And I wish the best for the Madhusudan, Anjani Kumar, Anant, Senthil Kumar and Abhijit. So all of you did well and really wonderful, very good. And many people are here, even though it is the web, there are 62 people are here to view even at this time. Really very, very happy to everyone. Thank you one and all. Shivu, thank you.
Thank you, Dr. Reddy. We had a really wonderful and thought-provoking session, and we had one keynote address by Dr. Robert and two invited lectures by Dr. Sheshasai and by Dr. Rahul. And also we had 5 oral presentations. I think Dr. Reddy has really summarized the overall whatever presentation has given us today. So I thank you, Dr. Reddy. And it is over to the host, Dr. Halesh, please.
Dear delegates, participants, ladies and gentlemen, really I would like to thank our chairman of this session, Dr. K. Shivshankar, Principal Scientist, Division of Basic Sciences, ICR, IIHR, Fasrigata, Bangalore, and the co-chairman, Dr. Y.A. Nanjareddy, Professor in Head, Department of Crop Physiology, University of Agricultural Sciences, Bangalore, as you have really managed this session excellently, sir. I am grateful to you people. Thanks, sir. Thank you very much.
Thanks from both.
Okay, sir.
Thank you.
Right. Thank you.
Thank you very much.
Okay.